The conceit of hindsight.
History doesn't repeat itself, but it rhymes. Mark Twain. History repeats itself. That's one of the things that's wrong with history. Clarence Darrow.
History has been described as one damn thing after another. The remark can be seen as a warning against a pair of temptations, but, duly warned, I shall cautiously flirt with both. First, the historian is tempted to scour the past for patterns that repeat themselves, or at least, following Mark Twain, to seek reason and rhyme for everything. This appetite for pattern affronts those who insist that, as Mark Twain will also be found to have said, history is usually a random, messy affair, going nowhere and following no rules. The second connected temptation is the vanity of the present, of seeing the past as aimed at our own time, as though the characters in history's play had nothing better to do with their lives than foreshadow us. Under names that need not trouble us, these are live issues in human history, and they arise with greater force and no greater agreement on the longer time scale of evolution. Evolutionary history can be represented as one damn species after another. But many biologists will join me in finding this an impoverished view. Look at evolution that way, and you miss most of what matters. Evolution rhymes, patterns recur. And this doesn't just happen to be so. It is so for well-understood reasons, Darwinian reasons mostly, for biology, unlike human history or even physics, already has its grand unifying theory, accepted by all informed practitioners, though in varying versions and interpretations. In writing evolutionary history, I do not shrink from seeking patterns and principles, but I try to be careful about it. What of the second temptation, the conceit of hindsight, the idea that the past works to deliver our particular present? The late Stephen Jay Gould rightly pointed out that a dominant icon of evolution in popular mythology, a caricature almost as ubiquitous as lemmings jumping over cliffs, and that myth is false too, is a shambling file of simian ancestors rising progressively in the wake of the erect, striding, majestic figure of Homo sapiens sapiens, man as evolution's last word. And in this context, it always is man rather than woman. Man as what the whole enterprise is pointing towards, man as a magnet, drawing evolution from the past towards his eminence. There is a physicist's version which is less obviously vainglorious and which I should mention in passing. This is the anthropic notion that the very laws of physics themselves, or the fundamental constants of the universe, are a carefully tuned, put-up job, calculated to bring humanity eventually into existence. It is not necessarily founded on vanity. It doesn't have to mean that the universe was deliberately made in order that we should exist. It need mean only that we are here, and we could not be, in a universe that lacked the capability of producing us. As physicists have pointed out, it is no accident that we see stars in our sky, for stars are a necessary part of any universe capable of generating us. Again, this does not imply that stars exist in order to make us. It is just that without stars, there would be no atoms heavier than lithium in the periodic table, and a chemistry of only 3 elements is too impoverished to support life. Seeing is the kind of activity that can go on only in the kind of universe where what you see is stars. But there is a little more that needs to be said. Granted the trivial fact that our presence requires physical laws and constants capable of producing us, the existence of such potent ground rules may still seem tantalizingly improbable. Depending upon their assumptions, physicists may reckon that the set of possible universes vastly outnumbers that subset whose laws and constants allowed physics to mature via stars into chemistry and via planets into biology. To some, this means that the laws and constants must have been deliberately premeditated from the start, although it baffles me why anybody regards this as an explanation for anything, given that the problem so swiftly regresses to the larger one of explaining the existence of the equally fine-tuned and improbable premeditator. Other physicists are less confident that the laws and constants were free to vary in the first place. When I was little, it was not obvious to me why 5 times 8 had to give the same result as 8 times 5. I accepted it as one of those facts that grown-ups assert. Only later did I understand, perhaps through visualizing rectangles, why such pairs of multiplications are not free to vary independently of one another. We understand that the circumference and the diameter of a circle are not independent, otherwise we might feel tempted to postulate a plethora of possible universes, each with a different value of pi. Perhaps, argue some physicists such as the Nobel Prize-winning theorist Steven Weinberg, the fundamental constants of the universe, which at present we treat as independent of one another, will in some grand unified fullness of time be understood to have fewer degrees of freedom than we now imagine. Maybe there is only one way for a universe to be. That would undermine the appearance of anthropic coincidence.
Other physicists, including Sir Martin Rees, the present Astronomer Royal, accept that there is a real coincidence in need of explanation, and explain it by postulating many actual universes existing in parallel, mutually incommunicado, each with its own set of laws and constants. Obviously we, who find ourselves reflecting upon such things, must be in one of those universes, however rare, whose laws and constants are capable of evolving us. But that was a digression about the physicist's version of the conceit of hindsight. The biologist's version is easier to dismiss since Darwin, though harder before him, and it is our concern here. Biological evolution has no privileged line of descent and no designated end. Evolution has reached many millions of interim ends, the number of surviving species at the time of observation, and there is no reason other than vanity, human vanity as it happens, since we are doing the talking, to designate any one as more privileged or climactic than any other.
This doesn't mean, as I shall continue to argue, that there is a total dearth of reasons or rhymes in evolutionary history. I believe there are recurring patterns. I also believe, though this is more controversial today than it once was, that there are senses in which evolution may be said to be directional, progressive, and even predictable. But progress is emphatically not the same thing as progress towards humanity, and we must live with a weak and unflattering sense of the predictable. The historian must beware of stringing together a narrative that seems, even to the smallest degree, to be homing in on a human climax. A book in my possession, in the main a good book, so I shall not name and shame it, provides an example. It is comparing Homo habilis, a human species probably ancestral to us, with its predecessors, the Australopithecines. What the book says is that Homo habilis was
considerably more evolved than the Australopithecines.
More evolved? What can this mean but that evolution is moving in some pre-specified direction? The book leaves us in no doubt of what the presumed direction is.
The first signs of a chin are apparent.
First encourages us to expect second and third signs towards a complete human chin.
The teeth start to resemble ours.
As if those teeth were the way they are, not because it suited the Haberline diet, but because they were embarking upon the road towards becoming our teeth. The passage ends with a telltale remark about a later species of extinct human, Homo erectus.
Although their faces are still different from ours, they have a much more human look in their eyes. They are like sculptures in the making, unfinished works.
In the making, unfinished? Only with the unwisdom of hindsight. In excuse of that book, it is probably true that were we to meet a Homo erectus face to face, it might well look to our eyes like an unfinished sculpture in the making. But that is only because we are looking with human hindsight. A living creature is always in the business of surviving in its own environment. It is never unfinished, or in another sense it is always unfinished. So, presumably, are we. The conceit of hindsight tempts us at other stages in our history. From our human point of view, the emergence of our remote fish ancestors from water to land was a momentous step, an evolutionary rite of passage. It was undertaken in the Devonian period by lobe-finned fish, a bit like modern lungfish. We look at fossils of the period with a pardonable yearning to gaze upon our forebears, and are seduced by a knowledge of what came later, drawn into seeing these Devonian fish as halfway towards becoming land animals, everything about them earnestly transitional, bound into an epic quest to invade the land and initiate the next big phase of evolution. That is not the way it was at the time. Those Devonian fish had a living to earn. They were not on a mission to evolve, not on a quest towards the distant future. An otherwise excellent book about vertebrate evolution contains the following sentence about fish which
ventured out of the water onto the land at the end of the Devonian period and jumped the gap, so to speak, from one vertebrate class to another to become the first amphibians.
The gap comes from hindsight. There was nothing resembling a gap at the time, and the classes that we now recognize were no more separate in those days than two species. As we shall see again, jumping gaps is not what evolution does. It makes no more sense, and no less, to aim our historical narrative towards Homo sapiens than towards any other modern species, Octopus vulgaris, say, or Panthera leo, or Sequoia sempervirens. A historically minded swift, understandably proud of flight as self-evidently the premier accomplishment of life, will regard swift-kind, those spectacular flying machines with their swept-back wings, who stay aloft for a year at a time and even copulate in pre-flight, as the acme of evolutionary progress. To build on a fancy of Steven Pinker, if elephants could write history, they might portray tapirs, elephant shrews, elephant seals, and proboscis monkeys as tentative beginners along the main trunk road of evolution, taking the first fumbling steps, but each, for some reason, never quite making it, so near yet so far. Elephant astronomers might wonder whether, on some other world, there exist alien life forms that have crossed the nasal Rubicon and taken the final leap to full proboscitude.
We are not swifts nor elephants, we are people. As we wander in imagination through some long-dead epoch, it is humanly natural to reserve a special warmth and curiosity for whichever otherwise ordinary species in that ancient landscape is our ancestor. It is an intriguingly unfamiliar thought that there is always one such species. It is hard to deny our human temptation to see this one species as on the main line of evolution, the others as supporting cast, walk-on parts, sidelined cameos. Without succumbing to that error, there is one way to indulge a legitimate human centrism while respecting historical propriety. That way is to do our history backwards, and it is the way of this book. Backward chronology in search of ancestors really can sensibly aim towards a single distant target. The distant target is the grand ancestor of all life, and we can't help converging upon it no matter where we start, elephant or eagle, swift or salmonella, Wellingtonia or woman. Backward chronology and forward chronology are each good for different purposes. Go backwards, and no matter where you start, you end up celebrating the unity of life. Go forwards, and you extol diversity. It works on small timescales as well as large. The forward chronology of the mammals, within their large but limited timescale, is a story of branching diversification, uncovering the richness of that group of hairy warm-bloods. Backward chronology, taking any modern mammal as our starting point, will always converge upon the same unique archimammal, shadowy, insectivorous, nocturnal contemporary of the dinosaurs. This is a local convergence. A yet more local one converges on the most recent ancestor of all rodents, who lived somewhere around the time the dinosaurs went extinct. More local still is the backward convergence of all apes, including humans, on their shared ancestor, who lived about 18 million years ago. On a larger scale, there is a comparable convergence to be found if we work backwards from any vertebrate, an even larger convergence working backwards from any animal to the ancestor of all animals. The largest convergence of all takes us from any modern creature, animal, plant, fungus, or bacterium, back to the universal progenitor of all surviving organisms, probably resembling some kind of bacterium.
I used convergence in the last paragraph, but I really want to reserve that word for a completely different meaning in forward chronology. So for the present purpose I shall substitute confluence, or for reasons that will make sense in a moment, rendezvous. I could have used coalescence, except that, as we shall see, geneticists have already adopted it in a more precise sense, similar to my confluence, but concentrating on genes rather than species. In a backward chronology, the ancestors of any set of species must eventually meet at a particular geological moment. Their point of rendezvous is the last common ancestor that they all share, what I shall call their concestor, the focal rodent or the focal mammal or the focal vertebrate, say. The oldest concestor is the grand ancestor of all surviving life. We can be very sure there really is a single concestor of all surviving life forms on this planet. The evidence is that all that have ever been examined share, exactly in most cases, almost exactly in the rest, the same genetic code, and the genetic code is too detailed in arbitrary aspects of its complexity to have been invented twice. Although not every species has been examined, we already have enough coverage to be pretty certain that no surprises, alas, await us. If we now were to discover a life form sufficiently alien to have a completely different genetic code, it would be the most exciting biological discovery in my adult lifetime, whether it lives on this planet or another. As things stand, it appears that all known life forms can be traced to a single ancestor which lived more than 3 billion years ago. If there were other independent origins of life, they have left no descendants that we have discovered. And if new ones arose now, they would swiftly be eaten, probably by bacteria.
The grand confluence of all surviving life is not the same thing as the origin of life itself. This is because all surviving species presumably share a concestor who lived after the origin of life. Anything else would be an unlikely coincidence, for it would suggest that the original life form immediately branched, and more than one of its branches survive to this day. Current textbook orthodoxy dates the oldest bacterial fossils at about 3.5 billion years ago, so the origin of life must at least be earlier than that. The grand confluence, the last common ancestor of all surviving creatures, could predate the oldest fossils, it didn't fossilize, or it could have lived a billion years later. All but one of the other lineages went extinct. Given that all backward chronologies, no matter where they start, culminate in the one grand confluence, we can legitimately indulge our human preoccupation and concentrate upon the single line of our own ancestors. Instead of treating evolution as aimed towards us, we choose modern Homo sapiens as our arbitrary, but forgivably preferred, starting point for a reverse chronology. We choose this route out of all possible routes to the past because we are curious about our own great-grandcestors. At the same time, although we need not follow them in detail, we shall not forget that there are other historians, animals and plants belonging to other species, who are independently walking backwards from their separate starting points on separate pilgrimages to visit their own ancestors, including eventually the ones they share with us. If we retrace our own ancestral steps, we shall inevitably meet these other pilgrims and join forces with them in a definite order, the order in which their lineages rendezvous with ours, the order of ever more inclusive cousinship.
Pilgrimages join forces with pilgrims. Yes, why not? Pilgrimage is an apt way to think about our journey to the past. This book will be cast in the form of an epic pilgrimage from the present to the past. All roads lead to the origin of life, but because we are human, the path we shall follow will be that of our own ancestors. It will be a human pilgrimage to discover human ancestors. As we go, we shall greet other pilgrims who will join us in strict order as we reach the common ancestors we share with them. The first fellow pilgrims we shall greet, some 5 million years ago, deep in Africa, where Stanley memorably shook hands with Livingstone, are the chimpanzees. The chimpanzee and bonobo pilgrims will already have joined forces with each other before we greet them. and here we have a little linguistic trickiness which I must face at the outset before it dogs us any further. I placed before in inverted commas because it could confuse. I used it to mean before in the backward sense, before in the course of the pilgrimage to the past. But that, of course, means after in the chronological sense, the exact opposite meaning. My guess is that no reader was confused in this particular case, but there will be other instances where the reader's patience may be tested. While writing this book, I tried the experiment of coining a new preposition tailored to the peculiar needs of a backward historian, but it didn't fly. Instead, I shall adopt the convention of before in inverted commas.
The next pilgrims with whom we shall rendezvous as we push back along our journey are gorillas, then orangutans, quite a lot deeper into the past, and probably no longer in Africa. Next we shall greet gibbons, then Old World monkeys, then New World monkeys, then various other groups of mammals, and so on, until eventually all the pilgrims of life are marching together in one single backward quest for the origin of life itself. As we push on back, there will come a time when it is no longer meaningful to name the continent in which a rendezvous takes place. The map of the world was so different because of the remarkable phenomenon of plate tectonics. And further back still, all rendezvous will take place in the sea. It is a rather surprising fact that we human pilgrims pass only about 40 rendezvous points in all before we hit the origin of life itself. At each of the 40 steps we shall find one particular shared ancestor, the concestor, which will bear the same labelling number as the rendezvous. For example, concestor 2, whom we meet at rendezvous 2, is the most recent common ancestor of gorillas on the one hand, and humans plus chimpanzees and bonobos on the other. Concestor 3 is the most recent common ancestor of orangutans and humans plus chimpanzees and bonobos plus gorillas. Concestor 39 is the grand ancestor of all surviving life forms. Concestor 0 is a special case, the most recent ancestor of all surviving humans.
We shall be pilgrims then, sharing fellowship ever more inclusively with other pilgrim bands, which also have been swelling on their own way to their rendezvous with us. After each meeting, we continue together on the high road back to our shared Archean goal, our Canterbury. There are other literary allusions, of course, and I almost made Bunyan my model and Pilgrim's Regress my title. But it was to Chaucer's Canterbury Tales that I and my research assistant Yan Wong kept returning in our discussions, and it seemed increasingly natural to think of Chaucer throughout this book. Unlike most of Chaucer's pilgrims, mine do not all set out together, although they do set off at the same time, the present. These other pilgrims aim towards their ancient Canterbury from different starting points, joining our human pilgrimage at various rendezvous along the road. In this respect, my pilgrims are unlike those who gathered in London's Tabard Inn. Mine are more like the sinister canon and his understandably disloyal yeoman, who joined Chaucer's pilgrims at Bourton-under-Blee, 5 miles short of Canterbury. Following Chaucer's lead, my pilgrims, which are all the different species of living creature, will have the opportunity to tell tales along the way to their Canterbury, which is the origin of life. It is these tales that form the main substance of this book. Dead men tell no tales, and extinct creatures such as trilobites are deemed not to be pilgrims capable of telling them. But I shall make exceptions of 2 special classes. Animals such as the dodo, which survived into historical times and whose DNA is still available to us, are treated as honorary members of the modern fauna, setting off on pilgrimage at the same time as us and joining us at some particular rendezvous. Since we are responsible for their so recent extinction, it seems the least we can do. The other honorary pilgrims, exceptions to the rule that dead men tell no tales, really are men or women. Since we human pilgrims are directly seeking our own ancestors, fossils that might plausibly be considered candidates for being our ancestors are deemed members of our human pilgrimage, and we shall hear tales from some of these shadow pilgrims, for example the handyman, Homo habilis. I decided it would be twee to let my animal and plant tale-teller speak in the first-person singular, and I shall not do so. Save for occasional asides and prefatory remarks, Chaucer's pilgrims don't either. Many of Chaucer's tales have their own prologue, and some have an epilogue too, all written in Chaucer's own voice as narrator of the pilgrimage. I shall occasionally follow his example. As with Chaucer, an epilogue may serve as a bridge from one tale to the next. Before his tales begin, Chaucer has a long general prologue in which he sets out his caste list, the professions, and in some cases the names, of the pilgrims who are about to set off from the tavern. Instead, I shall introduce new pilgrims as they join us. Chaucer's jovial host offers to guide the pilgrims and encourages them to tell their tales to while away the journey. In my role as host, I shall use the general prologue for some preparatory remarks about methods and problems of reconstructing evolutionary history, which must be faced and solved whether we do our history backwards or forwards. Then we shall embark on our backwards history itself. Although we shall concentrate on our own ancestors, noting other creatures usually only when they join us, we shall from time to time look up from our road and remind ourselves that there are other pilgrims on their own more or less independent routes to our ultimate destination. The numbered rendezvous milestones, plus a few intermediate markers necessary to consolidate the chronology, will provide the scaffolding for our narrative. Each will mark a new chapter, where we halt to take stock of our pilgrimage, and maybe listen to a tale or two. On rare occasions, something important happens in the world around us, and then our pilgrims may pause briefly to reflect on it. But for the most part, we shall mark our progress to the dawn of life by the measure of those 40 natural milestones, the trysts that enrich our pilgrimage.
The General Prologue. How shall we know the past, and how date it? What aids to our vision will help us peer into theatres of ancient life and reconstruct the scenes and the players, their exits and their entrances of long ago? Conventional human history has 3 main methods, and we shall find their counterparts on the larger timescale of evolution. First, there is archaeology, the study of bones, arrowheads, fragments of pots, oyster shell middens, figurines, and other relics that survive as hard evidence from the past. In evolutionary history, the most obvious hard relics are bones and teeth, and the fossils that they eventually become. Second, there are renewed relics, records that are not themselves old, but which contain or embody a copy or representation of what is old. In human history, these are written or spoken accounts handed down, repeated, reprinted, or otherwise duplicated from the past to the present. In evolution, let us propose DNA as the main renewed relic, equivalent to a written and recopied record. Third, there is triangulation. This name comes from a method of judging distances by measuring angles. Take a bearing on a target. Now walk a measured distance sideways, and take another. From the intercept of the 2 angles, calculate the distance of the target. Some camera rangefinders use the principle, and map surveyors traditionally relied upon it. Evolutionists can be said to triangulate an ancestor by comparing 2 or more of its surviving descendants.
The pilgrimage begins. It is time to set off on our pilgrimage to the past, which we can think of as a journey in a time machine in quest of our ancestors. For the first few tens of thousands of years of our backwards quest, our ancestral genes reside in individuals who look the same as us. Well, that is obviously not literally true, because we don't look exactly the same as each other. Let me rephrase it. For the first tens of thousands of years of our pilgrimage, the people we meet as we step outside our time machine will be no more different from us than we today are different from each other. Bear in mind that we today includes Germans and Zulus, Pygmies and Chinese, Berbers and Melanesians. Our genetic ancestors of 50,000 years ago would have fallen within the same envelope of variability as we see around the world today.
If not biological evolution, then what changes shall we see as we go back through tens of millennia as opposed to hundreds or thousands of millennia? There is an evolution-like process, orders of magnitude faster than biological evolution, which in the early stages of our time machine's journey will dominate the view from the porthole. This is variously called cultural evolution, exosomatic evolution, or technological evolution. We notice it in the evolution of the motor car, or of the necktie, or of the English language. We mustn't overestimate its resemblance to biological evolution, and it will in any case not detain us long. We have a 4-billion-year road to run, and we shall soon have to set the time machine into a gear too high to allow us more than a fleeting glimpse of events on the scale of human history.
But first, while our time machine is still in bottom gear, travelling on the timescale of human rather than evolutionary history, a pair of tales about two major cultural advances. The farmer's tale is the story of the agricultural revolution, arguably the human innovation that has had the greatest repercussions for the rest of the world's organisms. And the Cro-Magnon's tale is about the great leap forward, that flowering of the human mind which, in a special sense, provided a new medium for the evolutionary process itself.
The Farmer's Tale. The agricultural revolution began at the wane of the last ice age, about 10,000 years ago, in the so-called Fertile Crescent between the Tigris and the Euphrates. Agriculture also arose, probably independently, in China and along the banks of the Nile, and completely independently in the New World. An interesting case can be made for yet another independent cradle of agricultural civilization in the astonishingly isolated highland interior of New Guinea. The agricultural revolution dates the start of the new Stone Age, the Neolithic. The transition from wandering hunter-gatherers to a settled agricultural lifestyle may represent the first time people had a concept of a home. Contemporaries of the first farmers in other parts of the world were unreconstructed hunter-gatherers who wandered more or less continuously. Indeed, the hunter-gatherer lifestyle, hunter can include fisher, has not died out. It is still practiced in pockets around the world, by Australian Aborigines, by San and related tribes in southern Africa, wrongly called Bushmen, by various Native American tribes, called Indians after a navigational error, and by the Inuit of the Arctic, who prefer not to be called Eskimos. Hunter-gatherers typically do not cultivate plants and do not keep livestock. In practice, all intermediates between pure hunter-gatherers and pure agriculturalists or pastoralists are found. But earlier than 10,000 years ago, all human populations were hunter-gatherers. Soon, probably none will be. Those not extinct will be civilized, or corrupted, depending on your point of view. Colin Tudge, in his little book, Neanderthals, Bandits and Farmers, How Agriculture Really Began, agrees with Jared Diamond, the third chimpanzee, that the switch to agriculture from hunting and gathering was by no means the improvement we, in our complacent hindsight, might think. The agricultural revolution did not, in their view, increase human happiness. Agriculture supported larger populations than the hunter-gatherer lifestyle that it superseded, but not in obviously improved health or happiness. In fact, larger populations generally harbour more vicious diseases, for sound evolutionary reasons. A parasite is less concerned to prolong the life of its present host if it can easily find new victims. Nevertheless, our situation as hunter-gatherers cannot have been a utopia either. It has lately become fashionable to regard hunter-gatherers and primitive agricultural societies as more in balance with nature than us. This is probably a mistake. They may well have had a greater knowledge of the wild simply because they lived and survived in it, but, like us, they seem to have used their knowledge to exploit and often overexploit the environment to the best of their abilities at the time. Far from being in balance with nature, pre-agricultural hunter-gatherers were probably responsible for widespread extinctions of many large animals around the globe. Just prior to the agricultural revolution, the colonization of remote areas by hunter-gatherer peoples is suspiciously often followed in the archaeological record by the wiping out of many large and presumably palatable birds and mammals. We tend to regard urban as the antithesis of agricultural, but in the longer perspective that this book must adopt, city dwellers should be lumped in with farmers as opposed to hunter-gatherers. Almost all the food of a town comes from owned and cultivated land, in ancient times from fields round about the town, in modern times from anywhere in the world, transported and sold on through middlemen before being consumed. The agricultural revolution soon led to specialization. Potters, weavers, and smiths traded their skills for food which others grew. Before the agricultural revolution, food was not cultivated on owned land, but captured or gathered on unowned commons. Pastoralism, the herding of animals on common land, may have been an intermediate stage.
Whether it was a change for better or worse, the agricultural revolution was presumably not a sudden event. Husbandry was not the overnight brainwave of a genius and Neolithic turnip Townsend. To begin with, hunters of wild animals in open and unowned country might have guarded hunting territories against rival hunters, or guarded the herds themselves while following them about. From there it was a natural progression to herding them, then feeding them, and finally corralling and housing them. I dare say none of these changes would have seemed revolutionary when they happened. Meanwhile, the animals themselves were evolving, becoming domesticated by rudimentary forms of artificial selection. The Darwinian consequences on the animals would have been gradual. Without any deliberate intention to breed for domestic tractability, our ancestors inadvertently changed the selection pressures on the animals. Within the gene pools of the herds, there would no longer be a premium on fleetness or other survival skills of the wild. Successive generations of domestic animals became tamer, less able to fend for themselves, more apt to flourish and grow fat under feather-bedded domestic conditions. Some idea of the accidental genetic consequences of domestication is given by some interesting Russian work on silver foxes. D.K. Belayev and his colleagues took captive silver foxes, vulpes vulpes, and set out systematically to breed for tameness. They succeeded dramatically. By mating together the tamest individuals of each generation, Belayev had, within 20 years, produced foxes that behaved like border collies, actively seeking human company and wagging their tails when approached. That is not very surprising, although the speed with which it happened may be. Less expected were the byproducts of selection for tameness. These genetically tamed foxes not only behaved like collies, they looked like collies. They grew black and white coats with white face patches and muzzles. Instead of the characteristic pricked ears of a wild fox, they developed lovable, floppy ears. Their reproductive hormone balance changed, and they assumed the habit of breeding all the year round, instead of in a breeding season. Probably associated with their lowered aggression, they were found to contain higher levels of the neurally active chemical serotonin. It took only 20 years to turn foxes into dogs by artificial selection. I put dogs in inverted commas because our domestic dogs are not descended from foxes, they are descended from wolves. No doubt the original story of the evolution of dogs from wolves was similar to the new one simulated by Belayev with foxes, with the difference that Belayev was breeding for tameness deliberately. Our ancestors did it inadvertently, and it probably happened several times, independently, in different parts of the world. Perhaps initially, wolves took to scavenging around human encampments. Humans may have found such scavengers a convenient means of refuse disposal, and they may also have valued them as watchdogs, and even as warm sleep comforters. If this amicable scenario sounds surprising, reflect that the medieval legend of wolves as mythic symbols of terror coming out of the forest was born of ignorance. Our wild ancestors living in more open country would have known better. Indeed, they evidently did know better, because they ended up domesticating the wolf, thereby making the loyal, trusted dog.
From the wolf's point of view, human camps provided rich pickings for a scavenger, and the individuals most likely to benefit were those whose serotonin levels and other brain characteristics, propensity to tameness, happened to make them feel at home with humans. Several writers have speculated, plausibly enough, about orphaned cubs being adopted as pets by children. Experiments have shown that domestic dogs are better than wolves at reading the expressions on human faces. At the same time, we read their faces, and dog facial expressions have become more human-like than those of wolves, because of inadvertent selection by humans. This is presumably why we think wolves look sinister while dogs look loving, guilty, soppy, and so on. The Russian fox experiment demonstrates the speed with which domestication can happen, and the likelihood that a train of incidental effects would follow in the wake of selection for tameness. It is entirely probable that cattle, pigs, horses, sheep, goats, chickens, geese, ducks, and camels followed a course which was just as fast and just as rich in unexpected side effects. It also seems plausible that we ourselves evolved down a parallel road of domestication after the agricultural revolution towards our own version of tameness and associated byproduct traits. In some cases, the story of our own domestication is clearly written in our genes. The classic example, meticulously documented by William Durham in his book Coevolution, is lactose tolerance. Milk is baby food, not intended for adults and originally not good for them. Lactose, the sugar in milk, requires a particular enzyme, lactase, to digest it. Young mammals switch off the gene that produces lactase after they pass the age of normal weaning. It isn't that they lack the gene, of course. Genes needed only in childhood are not removed from the genome, not even in butterflies, which must carry large numbers of genes needed only for making caterpillars. But lactase production is switched off in human infants at the age of about 4 under the influence of other controlling genes. Fresh milk makes adults feel ill, with symptoms ranging from flatulence and intestinal cramps to diarrhea and vomiting.
All adults? No, of course not. There are exceptions. I am one of them, and there's a good chance that you are too. My generalization concerned the human species as a whole, and by implication, the wild Homo sapiens from which we are all descended. It is as if I had said, wolves are big, fierce carnivores that hunt in packs and bay at the moon, knowing full well that Pekingeses and Yorkshire Terriers belie it. The difference is that we have a separate word, dog, for domestic wolf, but not for domestic human. The genes of domestic animals have changed as a result of generations of contact with humanity, inadvertently following the same sort of course as the genes of the silver fox. The genes of some humans have changed as a result of generations of contact with domestic animals. Of course, nobody deliberately changes their genes. Science is only now beginning to work out how to do that. As usual, the job was done for us by natural selection, and it happened millennia ago. I don't know exactly by what route natural selection produced adult lactose tolerance. Perhaps adults resorted to baby food in times of desperation, and the individuals that were most tolerant of it survived better. Perhaps some cultures postponed weaning, and selection for survival of children under these conditions spilled over, gradually, into adult tolerance. Whatever the details, the change, though genetic, was culture-driven. The evolution of tameness and increasing milk yields in cattle, sheep, and goats paralleled that of lactose tolerance in the tribes that herded them. Both were true evolutionary trends in that they were changes in gene frequencies in populations, but both were driven by non-genetic cultural changes. Is lactose tolerance just the tip of the iceberg? Are our genomes riddled with evidences of domestication affecting not just our biochemistry, but our minds? Like Beliav's domesticated foxes, and like the domesticated wolves that we call dogs, have we become tamer, more lovable, with the human equivalents of floppy ears, soppy faces, and wagging tails? I leave you with a thought, and move hastily on. While hunting was sliding into herding, gathering presumably followed a similar slide into cultivation of plants. Again, it was mostly inadvertent. No doubt there were moments of creative discovery, as when people first noticed that if you put seeds in the ground, they make plants like those from which they came, or when somebody first observed that it helps to water them, weed them, and manure them. It was probably more difficult to work out that it might be a good idea to keep back the best seed for planting, rather than follow the obvious course of eating the best and planting the dross. My father, as a young man fresh out of college, taught agriculture to peasant farmers in Central Africa in the 1940s, and he tells me that this was one of the hardest lessons to get across. But mostly, the transition from gatherer to cultivator passed unnoticed by those concerned, like the transition from hunter to herder.
Many of our staple food crops, including wheat, oats, barley, rye, and maize, are members of the grass family, which have become greatly modified since the dawn of agriculture by inadvertent and later deliberate human selection. It is possible that we, too, have become genetically modified over the millennia to increase our tolerance of cereals in a way parallel to our evolution of tolerance of milk. Starchy cereals, such as wheat and oats, cannot have featured prominently in our diets before the agricultural revolution. Unlike oranges and strawberries, cereal seeds do not want to be eaten. Passing through an animal's digestive tract is no part of their dispersal strategy, as it is of plum and tomato seeds. On our side of the relationship, the human digestive tract is not able, unaided, to absorb much nutriment from seeds of the grass family, with their meagre starch reserves and hard, unsympathetic husks. Some aid comes from milling and cooking, but it also seems conceivable that in parallel with the evolution of tolerance to milk, we might have evolved an increased physiological tolerance to wheat compared to our wild ancestors. Wheat intolerance is a known problem for a substantial number of unfortunate individuals who discover, by painful experience, that they are happier if they avoid it. In any case, coevolution between animals and their food plants was nothing new. Grazing animals had been exerting a kind of benevolent Darwinian selection on grasses, guiding their evolution towards mutualistic cooperation for millions of years before we started domesticating wheat, barley, oats, rye, and maize. Grasses flourish in the presence of grazers, and they probably have been doing so for most of the 20 million years since their pollens first announced them in the fossil record. It's not, of course, that individual plants actually benefit by being eaten, but that grasses can withstand being cropped better than rival plants can. My enemy's enemy is my friend, and grasses, even when grazed, thrive when herbivores eat, along with the grasses themselves, other plants that would compete for soil, sun, and water. Grasses became ever more able to thrive in the presence of wild cattle, antelopes, horses, and other grazers, and eventually lawn mowers, as the millions of years went by. And the herbivores became better equipped, for example, with specialized teeth and complicated digestive tracts, including fermentation vats with cultures of microorganisms, to flourish on a diet of grass. This isn't what we ordinarily mean by domestication, but in effect it's not far from it. When, starting about 10,000 years ago, wild grasses of the genus Triticum were domesticated by our ancestors into what we now call wheat, it was in a way a continuation of what herbivores of many kinds had been doing to the ancestors of Triticum for 20 million years. Our ancestors accelerated the process, especially when we later switched from inadvertent accidental domestication to deliberate, planned, selective breeding, and very recently scientific hybridization and genetically engineered mutations. That is all we need to say about the origins of agriculture. Now, as our time machine leaves the 10,000-year mark and heads for rendezvous zero, we briefly pause, one more time, around 40,000 years ago. Here human society, entirely consisting of hunter-gatherers, underwent what may have been an even larger revolution than the agricultural one, the cultural Great Leap Forward. The tale of the Great Leap Forward will be told by Cro-Magnon Man, named after the cave in the Dordogne where fossils of this race of Homo sapiens were first discovered.
The Cro-Magnon's tale. Archaeology suggests that something very special began to happen to our species around 40,000 years ago. Anatomically, our ancestors who lived before this watershed date were the same as those who came after. Humans, sampled earlier than the watershed, would be no more different from us than they were from their own contemporaries in other parts of the world, or indeed than we are from our contemporaries. That's if you look at their anatomy. If you look at their culture, there is a huge difference. Of course, there are also huge differences between the cultures of different peoples across the world today, but this wasn't true if we go back much more than 40,000 years. Something happened then. Many archaeologists regard it as sudden enough to be called an event. I like Jared Diamond's name for it, the Great Leap Forward. Earlier than the Great Leap Forward, man-made artifacts had hardly changed for 1 million years. The ones that survive for us are almost entirely stone tools and weapons, quite crudely shaped. Doubtless wood, or in Asia bamboo, was a more frequently worked material, but wooden relics don't easily survive. As far as we can tell, there were no paintings, no carvings, no figurines, no grave goods, no ornamentation. After the leap, all these things suddenly appear in the archaeological record, together with musical instruments such as bone flutes, and it wasn't long before stunning creations like the Lascaux cave murals were created by Cro-Magnon people. A disinterested observer taking the long view from another planet might see our modern culture, with its computers, supersonic planes, and space exploration, as an afterthought to the Great Leap Forward. On the very long geological timescale, all our modern achievements, from the Sistine Chapel to special relativity, from the Goldberg variations to the Goldbach conjecture, could be seen as almost contemporaneous with the Venus of Willendorf and the Lascaux caves, all part of the same cultural revolution, all part of the blooming cultural upsurge that succeeded the long lower Paleolithic stagnation. Actually, I'm not sure that our extraplanetary observer's uniformitarian view would stand up to much searching analysis, but it could be at least briefly defended. Some authorities are so impressed by the Great Leap Forward that they think it coincided with the origin of language. What else, they ask, could account for such a sudden change? It's not as silly as it sounds to suggest that language arose suddenly. Nobody thinks writing goes back more than a few thousand years, and everyone agrees that brain anatomy didn't change to coincide with anything so recent as the invention of writing. In theory, speech could be another example of the same thing. Nevertheless, my hunch, supported by the authority of linguists such as Steven Pinker, is that language is older than the leap. If not language itself, perhaps the Great Leap Forward coincided with the sudden discovery of what we might call a new software technique, maybe a new trick of grammar, such as the conditional clause, which, at a stroke, enabled what-if imagination to flower. Or maybe early language before the leap could be used to talk only about things that were there on the scene. Perhaps some forgotten genius realized the possibility of using words referentially as tokens of things that were not immediately present. It is the difference between that waterhole which we can both see and suppose there was a waterhole the other side of the hill. Or perhaps representational art, which is all but unknown in the archaeological record before the leap, was the bridge to referential language. Perhaps people learned to draw bison before they learned to talk about bison that were not immediately visible. Much as I would like to linger around the heady time of the Great Leap Forward, we have a long pilgrimage to accomplish, and we must press on backwards. We are approaching the point where we can start looking for Concestor Zero, the most recent ancestor of all surviving humans. Rendezvous Zero, all humankind. The Human Genome Project has reached completion, hailed by a justly proud humanity. We might pardonably wonder whose genome has been sequenced. Has an illustrious dignitary been singled out for the honor, or is it a random nobody pulled off the street, or even an anonymous clone of cells from a tissue culture lab? It makes a difference because we vary. I have brown eyes while you perhaps have blue. I can't curl my tongue into a tube, whereas it's 50-50 that you can. Which version of the tongue-curling gene makes it into the published human genome? What is the canonical eye colour? I raise the question only to draw a parallel. This book traces our ancestors back through time. But whose ancestors are we talking about, yours or mine, a Bambuti Pygmies or a Torres Strait Islanders? If we go sufficiently far back, everybody's ancestors are shared. All your ancestors are mine, whoever you are, and all mine are yours, not just approximately, but literally. This is one of those truths that turns out, on reflection, to need no new evidence. We prove it by pure reason, using the mathematician's trick of reductio ad absurdum. Take our imaginary time machine absurdly far back, say 100 million years, to an age when our ancestors resembled shrews or opossums. Somewhere in the world at that ancient date, at least one of my personal ancestors must have been living, or I wouldn't be here. Let us call this particular little mammal Henry. It happens to be a family name. We seek to prove that if Henry is my ancestor, he must be yours too. Imagine for a moment the contrary. I am descended from Henry, and you are not. For this to be so, your lineage and mine would have to have marched side by side, yet never touching, through 100 million years of evolution to the present, never interbreeding, yet ending up at the same evolutionary destination, so alike that your relatives are still capable of interbreeding with mine. This reductio is clearly absurd. If Henry is my ancestor, he has to be yours too. If not mine, he cannot be yours. Without specifying how ancient is sufficiently, we have just proved that a sufficiently ancient individual with any human descendants at all must be an ancestor of the entire human race. Long-distance ancestry of a particular group of descendants, such as the human species, is an all-or-nothing affair. Moreover, it is perfectly possible that Henry is my ancestor, and necessarily yours, given that you are human enough to be reading this book, while his brother, Eric, is the ancestor of, say, all the surviving aardvarks. Not only is it possible, it is a remarkable fact that there must be a moment in history when there were two animals in the same species, one of whom became the ancestor of all humans and no aardvarks, while the other became the ancestor of all aardvarks and no humans. They may well have met, and may even have been brothers. You can cross out aardvark and substitute any other modern species you like, and the statement must still be true. Think it through, and you will find that it follows from the fact that all species are cousins of one another. Bear in mind, when you do so, that the ancestor of all aardvarks will also be the ancestor of lots of very different things besides aardvarks. In this case, the entire major group called Afrotheria, which we shall meet at Rendezvous 13, and which includes elephants and dugongs, hyraxes, and Madagascan tenrecs. My reasoning was constructed as a reductio ad absurdum. It assumed that Henry lived long enough ago for it to be obvious that he begat either all living humans or none. How long is long enough? That's a harder question. 100 million years is more than enough to assure the conclusion we seek. If we go back only 100 years, no individual can claim the entire human race as direct descendants. Between the obvious cases of 100 years and 100 million, what can we say about unobvious intermediates, such as 10,000, 100,000, or 1 million years? The precise calculations were beyond me when I explained this reductio in River Out of Eden, but happily a Yale University statistician called Joseph T. Chang has now made a start on them. His conclusions and their implications form the Tasmanian's tale, a tale of particular relevance to this rendezvous because Concestor Zero is the most recent common ancestor of all living humans. It is more elaborate versions of calculations like Chang's that we need to do in order to date Rendezvous Zero. The conclusion we shall come to from these calculations is that Rendezvous Zero is surprisingly recent, maybe only a few thousands or tens of thousands of years. Rendezvous Zero is the time when, on our backwards pilgrimage, we first meet a common human ancestor. But, according to our reductio, there is a point not much further in the past when every individual that we encounter with our time machine is either a common ancestor or no ancestor at all. And although no one ancestor can be singled out for attention at this more distant milestone, it is worth a nod as we go by, because it marks the point where we can stop worrying about whether it is your ancestors we trace or mine. From that milestone on, all my readers march shoulder to shoulder in a phalanx of pilgrims towards the past.
Ape Men. The popular literature on human fossils is hyped up with alleged ambition to discover the earliest human ancestor. This is silly. You can ask a specific question like, which was the earliest human ancestor to walk habitually on two legs? Or, which was the first creature to be our ancestor and not the ancestor of a chimpanzee? Or, which was the earliest human ancestor to have a brain volume larger than 600 cc? Those questions at least mean something in principle, although they are hard to answer in practice, and some of them suffer from the vice of erecting artificial gaps in a seamless continuum. But who was the earliest human ancestor means nothing at all. More insidiously, the competition to find human ancestors means that new fossil discoveries are touted as on the main human line whenever remotely possible. But as the ground yields up more and more fossils, it becomes increasingly clear that during most of hominid history, Africa housed several species of hominids simultaneously. This has to mean that many fossil species now thought of as ancestral will turn out to be our cousins. At various times since Homo first appeared in Africa, it shared the continent with more robust hominids, perhaps several different species of them. As usual, their affinities and the exact number of species are hotly disputed. Names that have been attached to various of these creatures are Australopithecus robustus, Australopithecus boisei, and Australopithecus ethiopicus. They seem to have evolved from more gracile apes, gracile being the opposite of robust. The gracile apes are also placed in the genus Australopithecus, and we too almost certainly emerged from among the gracile Australopithecine ranks. Indeed, it is often difficult to distinguish early Homo from gracile Australopithecines. Let's look at some of the gracile fossils. Mrs. Ples lived about 2.5 million years ago. Her nickname comes from the genus Plesianthropus, to which she was originally assigned before people decided to incorporate her into Australopithecus, and from the fact that she was thought, perhaps erroneously, to be female. Individual fossil hominids often pick up pet names like this. Mr. Ples, naturally, is a more recently discovered fossil from Sterkfontein, who is in the same species as Mrs. Ples, Australopithecus africanus. Other fossils with nicknames include Dear Boy, a robust Australopithecine also known as Zinge, because he was originally named Zinjanthropus boisei. Little Foot, who's yet to be properly described and named, and the famous Lucy, Australopithecus afarensis, who lived about halfway between our time and that of Concestor I, our common ancestor with chimpanzees.
Rendezvous 1. Chimpanzees. Between 5 and 7 million years ago, somewhere in Africa, we human pilgrims enjoy a momentous encounter. It is Rendezvous 1, our first meeting with pilgrims from another species. Two other species, to be precise, for the common chimpanzee pilgrims and the pygmy chimpanzees or bonobo pilgrims have already joined forces with each other some 4 million years before their rendezvous with us. The common ancestor we share with them, Concestor I, is our 250,000 great-grandparent, an approximate guess this, of course, like the comparable estimates that I shall be making for other concestors. As we approach Rendezvous 1, then, the chimpanzee pilgrims are approaching the same point from another direction. Unfortunately, we don't know anything about that other direction. Although Africa has yielded up thousands of hominid fossils or fragments of fossils, not a single fossil has ever been found which can definitely be regarded as along the chimpanzee line of descent from Concestor I. This may be because they are forest animals and the leaf litter of forest floors is not friendly to fossils. Whatever the reason, it means that chimpanzee pilgrims are searching blind. Their equivalents of the Turkana boy of 1470, of Mrs. Pless, Lucy, Little Foot, Dear Boy, and the rest of our fossils have never been found. Nevertheless, in our fantasy, the chimpanzee and bonobo pilgrims meet us in some Pliocene forest clearing, and their dark brown eyes, like our less predictable ones, are fixed upon Concestor 1, their ancestor as well as ours. Time for a tale.
The Bonobo's Tale. The bonobo, Pan paniscus, looks pretty much like a common chimpanzee, Pan troglodytes, and before 1929 they were not recognized as separate species. The bonobo, despite its other name of pygmy chimpanzee, which should be abandoned, is not noticeably smaller than the common chimpanzee. Its body proportions are slightly different, and so are its habits, and that is the cue for its brief tale. The primatologist Frans de Waal put it neatly. The chimpanzee resolves sexual issues with power, the bonobo resolves power issues with sex. Bonobos use sex as a currency of social interaction, somewhat as we use money. They use copulation or copulatory gestures to appease, to assert dominance, to cement bonds with other troop members of any age or sex, including small infants. Paedophilia is not a hang-up with bonobos. All kinds of philia seem fine to them. De Waal describes how, in a group of captive bonobos that he watched, the males would develop erections as soon as a keeper approached at feeding time. He speculates that this was in preparation for sexually mediated food sharing. Female bonobos pair off to practice so-called GG, genital rubbing. The Haight-Ashbury image of free-loving bonobos has led to a piece of wishful thinking among nice people who perhaps came of age in the 1960s. The wishful thinking is that we are more closely related to bonobos than to common chimpanzees. The Margaret Mead in us feels closer to this gentle role model than to the patriarchal monkey-butchering chimpanzee. Unfortunately, like it or not, we are exactly equally close to both species. This is simply because P. troglodytes and P. paniscus share a common ancestor, which lived more recently than the ancestor they share with us. It does not follow from this that we resemble chimpanzees and bonobos equally. If chimpanzees have changed more than bonobos since the shared ancestor, Concestor 1, we might be more like bonobos than chimpanzees, or vice versa, and we shall probably find different things in common with both our pan cousins, perhaps in roughly equal measure. They are equally closely related to us because they are linked to us via the same shared ancestor. This is the moral of the bonobo's tale, a simple moral and a very general one, which we shall meet again and again at other junctures of our pilgrimage.
Rendezvous 2. Gorillas. The molecular clock tells us that Rendezvous 2, where the gorillas join us, again in Africa, is only 1,000,000 years further into our pilgrimage than Rendezvous 1. Unfortunately, there are no fossils to bridge the gap between Concestors 2 and 1, nothing to guide us in deciding whether Concestor 2, which is perhaps our 300,000 great-grandparent, was more like a gorilla, or more like a chimpanzee, or indeed more like a human. My guess would be chimpanzee, but this is only because the huge gorilla seems more extreme and less like the generality of apes. Don't let's exaggerate the unusualness of gorillas, however. They are not the largest apes that have ever lived. The Asian ape Gigantopithecus, a sort of giant orangutan, would have stood head and massive shoulders over the largest gorilla. It lived in China and went extinct only about 500,000 years ago, overlapping with Homo erectus and archaic Homo sapiens. This is so recent that some enterprising fantasists have gone so far as to suggest that the Yeti, or abominable snowman of the Himalayas, but I digress. Apes, perhaps especially gorillas, have long been potent generators and victims of human myths. The gorilla's tale considers our changing attitudes to our closest cousins.
The Gorilla's Tale. The rise of Darwinism in the 19th century polarized attitudes towards the apes. Opponents who might have stomached evolution itself balked with visceral horror at cousinship with what they perceived as low and revolting brutes, and desperately tried to inflate our differences from them. This was nowhere more true than with gorillas. Apes were animals, we were set apart. Worse, where other animals such as cats or deer could be seen as beautiful in their own way, gorillas and other apes, precisely because of their similarity to ourselves, seemed like caricatures, distortions, grotesques. Darwin never missed an opportunity to put the other side, sometimes in little asides such as his charming observation in The Descent of Man that monkeys smoke tobacco with pleasure. T.H. Huxley, Darwin's formidable ally, had a robust exchange with Sir Richard Owen, the leading anatomist of the day, who claimed, wrongly as Huxley showed, that the hippocampus minor was uniquely diagnostic of the human brain. Nowadays, scientists not only think we resemble apes, we include ourselves within the apes, specifically the African apes. We emphasize, by contrast, the distinctness of apes, including humans, from monkeys. To call a gorilla or a chimpanzee a monkey is a solecism. It has not always been so. In former times, apes were frequently lumped with monkeys, and some of the early descriptions confused apes with baboons or with Barbary macaques, which indeed are still known as Barbary apes. More surprisingly, long before people thought in terms of evolution at all, and before apes were clearly distinguished from each other or from monkeys, great apes were often confused with humans. Agreeable as it would be to approve this apparent prescience of evolution, it unfortunately may owe more to racism. Early white explorers in Africa saw chimpanzees and gorillas as close kin only to black humans, not to themselves. Interestingly, tribes in both Southeast Asia and Africa have traditional legends suggesting a reversal of evolution as conventionally seen. Their local great apes are regarded as humans who fell from grace. Orangutan means man of the woods in Malay. A picture of an orangutan by the Dutch Dr. Bontius in 1658 is, in T.H. Huxley's words,
nothing but a very hairy woman of rather comely aspect and with proportions and feet wholly human.
Also very human are the pictures made a century later by Linnaeus's pupil Hoppius, 1763. One of his creatures has a tail, but is otherwise wholly human, bipedal, and carries a walking stick. Pliny the Elder says that the tailed species have even been known to play at draughts, American checkers. One might have thought such a mythology would have prepared our civilization for the idea of evolution when it arrived in the 19th century, and might even have accelerated its discovery. Apparently not. Instead, we come back to the racism which until relatively late in the 20th century was endemic in our culture. Early explorers often assigned the native peoples of the forest a closer affinity with chimpanzees, gorillas, or orangs than with the explorers themselves. In the 19th century, after Darwin, evolutionists often regarded African peoples as intermediate between apes and Europeans on the upward path to white supremacy. This is not only factually wrong, it violates a fundamental principle of evolution. Two cousins are always exactly equally related to any outgroup because they are connected to that outgroup via a shared ancestor. For the reasons given in the Bonobo's tale, all humans are exactly equally close cousins to all gorillas. Racism and speciesism and our perennial confusion over how inclusively we wish to cast our moral and ethical net are brought into sharp and sometimes uncomfortable focus in the history of our attitudes to our fellow humans and our attitudes to apes, our fellow apes.
Rendezvous 3 to 9. The orangutan's tale at Rendezvous 3 suggests that the Asian apes, originally from Africa, returned to Africa, where apes had meanwhile died out and eventually evolved into the modern African apes, including ourselves. At Rendezvous 4, the gibbon's tale introduces some more advanced techniques not covered in the general prologue. It does so by discussing an evolutionary analysis of the original and copied manuscripts of Chaucer's Canterbury Tales. At Rendezvous 5, none of the Old World monkeys tells a tale, but at Rendezvous 6, where we join the New World monkeys, the howler monkey's tale shows how true colour vision, having been lost by most mammals in their long nocturnal phase during the reign of the dinosaurs, was independently rediscovered by howler monkeys and by Old World monkeys and apes. Rendezvous 7, with tarsiers, and 8, with lemurs, lorises, and bush babies, are followed by the Aye-Aye's tale, a tale of Madagascar and its unique fauna born of ancient isolation. Rendezvous 9, Colugos and Tree Shrews, yields a salutary tale of how uncertain we still are about some of the splits in life's family tree.
Rendezvous 10. Rodents and Rabbit Kind. Rendezvous 10 occurs 75 million years into our journey. It is here that our pilgrims are joined, overwhelmed rather, by a teeming, scurrying, gnawing, whisker-quivering plague of rodents. For good measure, we also greet at this point the rabbits, including the very similar hares and jackrabbits, and the rather more distant pikas. Consistor 10 is approximately our 15 million greats-grandparent. It is the latest ancestor we share with a mouse, but the mouse is connected to it through a very much larger number of greats because of short generation times. Rodents are one of the great success stories of mammaldom. More than 40% of all mammal species are rodents, and there are said to be more individual rodents in the world than all other mammals combined. Rats and mice have been the hidden beneficiaries of our own agricultural revolution, and they have travelled with us across the seas to every land in the world. They devastate our granaries and our health. Rats and their cargo of fleas were responsible for the Great Plague. Traditionally, but now controversially, the Black Death may also have been bubonic plague. They have spread typhus and have been blamed for more human deaths in the second millennium than all wars and revolutions put together. When even the 4 horsemen are laid low by the apocalypse, it will be rats that scavenge their remains, rats that will swarm like lemmings over the ruins of civilization. And by the way, lemmings are rodents too, northern voles, who for reasons that are not entirely clear, build up their populations to plague proportions in so-called lemming years, and then indulge in frantic, though not wantonly suicidal, as is falsely alleged, mass migrations. Rodents are gnawing machines. They have a pair of very prominent incisor teeth at the front, perpetually growing to replace massive wear and tear. The gnawing masseter muscles are especially well developed in rodents. They don't have canine teeth, and the large gap or diastema that separates their incisors from their back teeth improves the efficiency of their gnawing. Rodents can gnaw their way through almost anything. Beavers fell substantial trees by gnawing through their trunks. Mole rats live entirely underground, tunnelling not with their front paws like moles, but purely with their incisor teeth. Different species of rodents have penetrated the deserts of the world, gundis, gerbils, the high mountains, marmots, chinchillas, the forest canopy, squirrels, including flying squirrels, rivers, water voles, beavers, capybaras, rainforest floor, agoutis, savanna, maras, spring hares, and Arctic tundra, lemmings. Most rodents are mouse-sized, but they range up through marmots, beavers, agoutis, and maras to the sheep-sized capybaras of the South American waterways. Capybaras are prized for meat, not just because of their large size, but because, bizarrely, the Roman Catholic Church traditionally deemed them honorary fish for Fridays, presumably because they live in water. Large as they are, modern capybaras are dwarfed by various giant South American rodents that went extinct only quite recently. The giant capybara, Protohydrochaeros, was the size of a donkey. Telecomis was an even larger rodent the size of a small rhinoceros which, like the giant capybara, went extinct at the time of the Great American Interchange, when the Isthmus of Panama ended South America's island status. These two groups of giant rodents were not particularly closely related to each other and seem to have evolved their gigantism independently.
A world without rodents would be a very different world. It is less likely to come to pass than a world dominated by rodents and free of people. I have a post-Armageddon vision. We and all other large animals are gone. Rodents emerge as the ultimate post-human scavengers. They gnaw their way through New York, London, and Tokyo, digesting spilled larders, ghost supermarkets, and human corpses, and turning them into new generations of rats and mice, whose racing populations explode out of the cities and into the countryside. When all the relics of human profligacy are eaten, populations crash again, and the rodents turn on each other and on the cockroaches scavenging with them. In a period of intense competition, short generations, perhaps with radioactively enhanced mutation rates, boost rapid evolution. With human ships and planes gone, islands become islands again, with local populations isolated, save for occasional lucky raftings, ideal conditions for evolutionary divergence. Within 5 million years, a whole range of new species replace the ones we know. Herds of giant grazing rats are stalked by saber-toothed predatory rats. Given enough time, will a species of intelligent, cultivated rats emerge? Will rodent historians and scientists eventually organize careful archaeological gnaws through the strata of our long compacted cities and reconstruct the peculiar and temporally tragic circumstances that gave ratkind its big break?
The mouse and the beaver each have a tale to tell at Rendezvous 10. The beaver's tale explains the meaning of the phrase extended phenotype. The mouse's tale follows.
The mouse's tale. Of all the thousands of rodents, the house mouse, Mus musculus, has a special tale to tell because it has become the second most intensively studied mammal species after our own. Much more than the proverbial guinea pig, the mouse is a main staple of medical, physiological, and genetic laboratories the world over. In particular, the mouse is one of very few mammals apart from ourselves whose genome has so far been completely sequenced. Two things about these recently sequenced genomes have sparked unwarranted surprise. The first is that mammal genomes seem rather small, of the order of 30,000 genes, or maybe even less. And the second is that they are so similar to each other. Human dignity seemed to demand that our genome should be much larger than that of a tiny mouse. And shouldn't it be absolutely larger than 30,000 genes anyway? This last expectation has led people, including some who should know better, to deduce that the environment must be more important than we thought, because there aren't enough genes to specify a body. That really is a breathtakingly naive piece of logic. By what standard do we decide how many genes you need to specify a body? This kind of thinking is based on a subconscious assumption which is wrong, the assumption that the genome is a kind of blueprint, with each gene specifying its own little piece of body. As the fruit fly's tale tells us, it is not a blueprint, but something more like a recipe, a computer program, or a manual of instructions for assembly. If you think of the genome as a blueprint, you might expect a big, complicated animal like yourself to have more genes than a little mouse, with fewer cells and a less sophisticated brain. But, as I said, that isn't the way genes work. Even the recipe or instruction book model can be misleading unless it is properly understood. My colleague Matt Ridley develops a different analogy, which I find beautifully clear, in his book Nature via Nurture. Most of the genome that we sequence is not the book of instructions or master computer program for building a human or a mouse, although parts of it are. If it were, we might indeed expect our program to be larger than the mouse's. But most of the genome is more like the dictionary of words available for writing the book of instructions, or, as we shall soon see, the set of subroutines that are called by the master program. As Ridley says, the list of words in David Copperfield is almost the same as the list of words in The Catcher in the Rye. Both draw upon the vocabulary of an educated native speaker of English. What is completely different about the two books is the order in which those words are strung together. When a person is made, or when a mouse is made, both embryologies draw upon the same dictionary of genes, the normal vocabulary of mammal embryologies. The difference between a person and a mouse comes out of the different orders with which the genes, drawn from that shared mammalian vocabulary, are deployed, the different places in the body where this happens, and its timing. All this is under the control of particular genes whose business it is to turn other genes on in complicated and exquisitely timed cascades. But such controlling genes constitute only a minority of the genes in the genome. Don't misunderstand order as meaning the order in which the genes are strung out along the chromosomes. With notable exceptions discussed in the fruit fly's tale, the order of genes along a chromosome is as arbitrary as the order in which words are listed in a vocabulary, usually alphabetical, but, especially in phrase books for foreign travel, sometimes an order of convenience, words useful in airports, words useful when visiting the doctor, words useful for shopping, and so on. The order in which genes are stored on chromosomes is unimportant. What matters is that the cellular machinery finds the right gene when it needs it, and it does this using methods that are becoming increasingly understood. What distinguishes a mouse from a man is mostly not the genes themselves, nor the order in which they are stored in the chromosomal phrasebook, but the order in which they are turned on, the equivalent of Dickens or Salinger choosing words from the vocabulary of English and arranging them in sentences. In one respect, the analogy of words is misleading. Words are shorter than genes, and some writers have likened each gene to a sentence. But sentences aren't a good analogy, for a different reason. Different books are not put together by permuting a fixed repertoire of sentences. Most sentences are unique. Genes, like words but unlike sentences, are used over and over again in different contexts. A better analogy for a gene than either a word or a sentence is a toolbox subroutine in a computer. The computer I happen to be familiar with is the Macintosh, and it is some years since I did any programming, so I am certainly out of date with the details. Never mind, the principle remains, and it is true of other computers too. The Mac has a toolbox of routines stored in ROM, read-only memory, or in system files permanently loaded at startup time. There are thousands of these toolbox routines, each one doing a particular operation, which is likely to be needed over and over again in slightly different ways in different programs. For example, the toolbox routine called Obscure Cursor hides the cursor from the screen until the next time the mouse is moved. Unseen to you, the Obscure Cursor gene is called every time you start typing and the mouse cursor vanishes. Toolbox routines lie behind the familiar features shared by all programs on the Mac, and their imitated equivalents on Windows machines. Pull-down menus, scroll bars, shrinkable windows that you can drag around the screen with the mouse, and many others. The reason all Mac programs have the same look and feel, that very similarity famously became the subject of litigation, is precisely that all Mac programs, whether written by Apple or by Microsoft or by anybody else, call the same toolbox routines. If you are a programmer who wishes to move a whole region of the screen in some direction, say following a mouse drag, you would be wasting your time if you didn't invoke the scroll rect toolbox routine. Or, if you want to place a check mark by a pull-down menu item, you would be mad to write your own code to do it. Just write a call of check item into your program, and the job is done for you. If you look at the text of a Mac program, whoever wrote it, in whatever programming language and for whatever purpose, the main thing you'll notice is that it consists largely of invocations of familiar, built-in toolbox routines. The same repertoire of routines is available to all programmers. Different programs string calls of these routines together in different combinations and sequences. The genome, sitting in the nucleus of every cell, is the toolbox of DNA routines available for performing standard biochemical functions. The nucleus of a cell is like the ROM of a Mac. Different cells, for example liver cells, bone cells, and muscle cells, string calls of these routines together in different orders and combinations when performing particular cell functions, including growing, dividing, or secreting hormones. Mouse bone cells are more similar to human bone cells than they are to mouse liver cells. They perform very similar operations and need to call the same repertoire of toolbox routines in order to do so. This is the kind of reason why all mammal genomes are approximately the same size as each other. They all need the same toolbox.
Rendezvous 11. Laurasiatheres. 85 million years ago, in the hothouse world of the Upper Cretaceous, we greet Consister 11, approximately our 25 million greats-grandparent. Here we are joined by a much more diverse band of pilgrims than the rodents and rabbits who swelled our party at Rendezvous 10. Zealous taxonomists recognize their shared ancestry by giving them a name, Laurasiatheria, but it is seldom used because in truth this is a miscellaneous bunch. The rodents are all built to the same toothy design and have proliferated and diversified, presumably because it works so well. Rodents, therefore, really means something strong. It unites animals that have much in common. Laurasiatheria is as awkward as it sounds. It unites highly disparate mammals which have only one thing in common: their pilgrims all joined up with each other before they join us. They all hail originally from the old northern continent of Laurasia. And what a diverse crew these Laurasian pilgrims are! Some of them flying, some of them swimming, many of them galloping, half of them nervously looking over their shoulder for fear of being eaten by the other half. They belong to 7 different orders: the Pholidota, pangolins; carnivora, dogs, cats, hyenas, bears, weasels, seals, etc.; the Perissodactyla, horses, tapirs, and rhinos; Cetartiodactyla, antelopes, deer, cattle, camels, pigs, hippos, and, well, we'll come to the surprise member of this group later. Microchiroptera and Megachiroptera, respectively small and big bats, and Insectivora, moles, hedgehogs, and shrews, but not elephant shrews or tenrecs. We have to wait for Rendezvous 13 to meet them. Of the Laurasiatheres, 2 contribute tails: the seal, about what makes males different from females, and the hippo.
The hippo's tail. When I was a schoolboy studying Greek, I learned that hippos meant horse and potamos, river. Hippopotamuses were river horses. Later, when I gave up Greek and read zoology, I was not too disconcerted to learn that hippopotamuses weren't close to horses after all. Instead, they were classified firmly with pigs in the middle of the even-toed ungulates, or artiodactyls. I have now learned something so shocking that I'm still reluctant to believe it, but it looks as though I'm going to have to. Hippos' closest living relatives are whales. The even-toed ungulates include whales. Whales, needless to say, don't have hooves at all, whether odd or even-toed. Indeed, they don't have toes, so it might be less confusing if we adopt the scientific name artiodactyls, which is actually just the Greek for even-toed, so the change doesn't help much. For completeness, I should add that the equivalent name for the horse order is Perissodactyla, Greek for uneven-toed. Whales, it would now seem from strong molecular evidence, are artiodactyls. But since they previously had been placed in the order Cetacea, and since Artiodactyla was also a well-established name, a new composite has been coined, Cetartiodactyla. Whales are wonders of the world. They include the largest organisms that have ever moved. They swim with up and down movements of the spine derived from the mammalian gallop, as opposed to the side-to-side wave motion of the spine of a swimming fish or a running lizard. The front limbs are used for steering and stabilizing. There are no externally visible hind limbs at all, but some whales have small vestigial, pelvic and leg bones buried deep in their bodies. It would not be too hard to believe that whales are closer cousins to even-toed ungulates than they are to any other mammals. A bit strange, perhaps, but not shocking to accept that some remote ancestor branched to the left and went to sea to give rise to the whales, while it branched to the right to give all the even-toed ungulates. What is shocking is that, according to the molecular evidence, whales are deeply embedded within the even-toed ungulates. Hippos are closer cousins to whales than hippos are to anything else, including other even-toed ungulates such as pigs. On their backward journey, the hippo pilgrims and the whale pilgrims unite with each other before the two of them join the ruminants and then the other even-toed ungulates such as pigs. Whales are the surprise inclusion that I coyly referred to when I introduced the cetartiodactyls at Rendezvous 11. It is known as the Whippo hypothesis. All this supposes that we believe the testimony of the molecules. What do the fossils say? To my initial surprise, the new theory fits quite nicely. Most of the great orders of mammals, though not the subdivisions within them, go a long way back into the age of dinosaurs. Rendezvous 10, with the rodents and rabbits, and Rendezvous 11, the one we've just reached, both took place during the Cretaceous period at the height of the dinosaur regime. But mammals in those days were all rather small, shrew-like creatures, whether their respective descendants were destined to become mice or hippos. The real growth of mammal diversity started suddenly after the dinosaurs went extinct 65.5 million years ago. It was then that the mammals were able to blossom into all economic trades vacated by the dinosaurs. Large body size was just one thing that became possible for mammals only when the dinosaurs were gone. The process of divergent evolution was swift, and a huge range of mammals of all sizes and shapes roamed the land within 5 million years of liberation. 5 to 10 million years later, in the late Paleocene to early Eocene epoch, there are abundant fossils of even-toed ungulates. Another 5 million years later, in the early to middle Eocene, we find a group called the Archaeocetes. The name means old whales, and most authorities accept that among these animals are to be found the ancestors of modern whales. An early one of these, Pachycetus from Pakistan, seems to have spent at least some of its time on land. Later ones include the unfortunately named Basilosaurus. Unfortunate not because of basil, but because saurus means lizard. When first discovered, Basilosaurus was thought to be a marine reptile, and the rules of naming rigidly enforce priority, even though we now know better. Basilosaurus had an immensely long body, and would have been a good candidate for the giant sea serpent of legend, if only it were not long extinct. Around the time that whales were represented by the likes of Basilosaurus, the contemporary hippo ancestors may have been members of a group called the Anthracotheres, some reconstructions of which make them look quite like hippos. Returning to the whales, what are the antecedents of the Archaeocetes before they reinvaded the water? If the molecules are right, that whales' closest affinities are with hippos, we might be tempted to seek their ancestors among fossils which show some evidence of herbivory. On the other hand, no modern whale or dolphin is herbivorous. The completely unrelated dugongs and manatees, by the way, show that it is perfectly possible for a purely marine mammal to have a purely herbivorous diet. But whales eat either planktonic crustacea, baleen whales, fish or squid, dolphins and most toothed whales, or large prey such as seals, killer whales. This has led people to look for whale ancestors among carnivorous land mammals, beginning with Darwin's own speculation, sometimes ridiculed, though I've never understood why.
In North America, the black bear was seen by Hearn swimming for hours with widely open mouth, thus catching, like a whale, insects in the water. Even in so extreme a case as this, if the supply of insects were constant, and if better adapted competitors did not already exist in the country, I can see no difficulty in a race of bears being rendered, by natural selection, more and more aquatic in their structure and habits, with larger and larger mouths, till a creature was produced as monstrous as a whale.
As an aside, this suggestion of Darwin illustrates an important general point about evolution. The bear seen by Hearn was evidently an enterprising individual, feeding in an unusual way for its species. I suspect that major new departures in evolution often start in just such a way, with a piece of lateral thinking by an individual who discovers a new and useful trick and learns to perfect it. If the habit is then imitated by others, including perhaps the individual's own children, there will be a new selection pressure set up. Natural selection will favor genetic predispositions to be good at learning the new trick, and much will follow. I suspect that something like this is how instinctive feeding habits such as tree hammering in woodpeckers and mollusk smashing in thrushes and sea otters got their start. For a long time, people looking over the available fossils for a plausible antecedent to the Archaeocetes have favored the Mesonychids, a large group of land mammals that flourished in the Paleocene epoch just after the extinction of the dinosaurs. The Mesonychids seem to have been largely carnivorous or omnivorous like Darwin's bear, and they fit with what we all, before the coming of the hippo theory, thought a whale ancestor ought to be. An additionally nice thing about the Mesonychids is that they had hooves. They were hoofed carnivores, perhaps a bit like wolves, but running on hooves. Could they then have given rise to the even-toed ungulates as well as to the whales? Unfortunately, the idea doesn't fit with the hippo theory specifically. Even though the Mesonychids seem to be cousins of today's even-toed ungulates, and there are reasons for believing this over and above their hooves, they are no closer to hippos than they are to all the rest of the cloven-hoofed animals. We keep coming back to the molecular shocker. Whales are not just cousins of all the artiodactyls, they are buried within the artiodactyls, closer to hippos than hippos are to cows and pigs. Gathering all this together, we can sketch a forward chronology as follows. Molecular evidence puts the split between camels plus llamas and the rest of the artiodactyls at 65 million years, more or less exactly when the last dinosaurs died. Don't imagine, by the way, that the shared ancestor looked anything like a camel. In those days, all mammals looked more or less like shrews. But 65 million years ago, the shrews that were going to give rise to camels split from the shrews that were going to give rise to all the rest of the artiodactyls. The split between pigs and the rest, mostly ruminants, took place 60 million years ago. The split between ruminants and hippos took place about 55 million years ago. Then the whale lineage split off from the hippo lineage not long afterwards, say about 54 million years ago, which gives time for primitive whales, such as the semi-aquatic Pachycetus, to have evolved by 50 million years ago. Toothed whales and baleen whales parted company much later, around 34 million years ago, around the time when the earliest baleen whale fossils are found. Perhaps I was exaggerating a little when I implied that a traditional zoologist like me should be positively upset at the discovery of the hippo-whale connection. But let me try to explain why I was genuinely disconcerted when I first read about it a few years ago. It wasn't just that it was different from what I had learned as a student. That wouldn't have worried me at all. In fact, I would have found it positively exhilarating. What worried me, and still does to some extent, was that it seemed to undermine all generalizations that one might wish to make about groupings of animals. The life of a molecular taxonomist is too short to allow a pairwise comparison of every species with every other species. Instead, what one does is take 2 or 3 whale species, say, and assume that they are representative of whales as a group. It is tantamount to the assumption that the whales are a clade, sharing a common ancestor which is not shared by the other animals with which one is making the comparison. It is assumed not to matter, in other words, which whale you take to stand for all. Similarly, lacking the time to test every species of rodent, say, or artiodactyl, we might take blood from a rat and from a cow. It doesn't matter which artiodactyl you take to compare with the representative whale, because yet again we assume that the artiodactyls are a good clade, so it doesn't make any difference whether we take a cow, a pig, a camel, or a hippo. But now we are told that it does matter. Camel blood and hippo blood really will give a different comparison with whale blood, because hippos are closer cousins to whales than they are to camels. See where this lands us. If we can't trust the artiodactyls to hang together as a group, represented by any one of their number, how can we be sure that any group will hang together? Can we even assume that hippos hang together? such that it doesn't matter whether we choose a pygmy hippo or a common hippo for comparison with whales? What if whales are closer to pygmy hippos than to common hippos? Actually, we probably can rule that out, because fossil evidence suggests that the two hippo genera split apart about as recently as our split from chimpanzees, and that really does leave too little time to evolve all the different kinds of whales and dolphins. It is more problematical whether all the whales hang together. On the face of it, the toothed whales and the baleen whales might well represent two entirely separate returns to the sea from the land. Indeed, that very possibility has often been advocated. The molecular taxonomists who demonstrated the hippo connection very wisely did take DNA from both a toothed whale and a baleen whale. They found that the two whales are indeed much closer cousins to each other than they are to a hippo. But again, how do we know that the toothed whales hang together as a group, and the same for the baleen whales? Maybe all the baleen whales are related to a hippo except the minke whale, which is related to a hamster. No, I don't believe that, and I really do think the baleen whales are a united clade, sharing a common ancestor which is not shared by anything that is not a baleen whale. But can you see how the hippo whale discovery shakes the confidence? We could regain our confidence if we could think of a good reason why whales might be special in this respect. If whales are glorified artiodactyls, they are artiodactyls that suddenly took off, evolutionarily speaking, leaving the rest of the artiodactyls behind. Their closest cousins, the hippos, remained relatively static as normal, respectable artiodactyls. Something happened in the history of the whales that made them flip into evolutionary overdrive. They evolved so much faster than all the rest of the artiodactyls that their origin within that group was obscured, until molecular taxonomists came along and uncovered it. So, what is special about the history of the whales? When you write it down like that, the solution leaps off the page. Leaving the land and becoming wholly aquatic was a bit like going into outer space. When we go into space, we are weightless. Not, by the way, because we are a long way from Earth's gravity, as many people think, but because we are in free fall, like a parachutist before he pulls the ripcord. A whale floats. Unlike a seal or a turtle, which still comes on land to breed, a whale never stops floating. It never has to contend with gravity. A hippo spends time in the water, but it still needs stout, tree-trunk-like legs and strong leg muscles for the land. A whale doesn't need legs at all, and indeed it doesn't have any. Think of a whale as what a hippo would like to be if only it could be freed from the tyranny of gravity. And of course, there are so many other odd things about living the whole time in the sea that it comes to seem far less surprising that whale evolution should have spurted as it did, leaving hippos behind, stranded on land and stranded in the middle of the artiodactyls. This suggests that I was unduly alarmist a few paragraphs back. Much the same thing happened in the other direction, 300 million years earlier, when our fish ancestors emerged from the water onto the land. If whales are glorified hippos, we are glorified lungfish. The emergence of legless whales from within the middle of the artiodactyls, leaving the rest of the artiodactyls behind, should not seem more surprising than the emergence of four-legged land animals from one particular group of fish, leaving those fish behind. That, at any rate, is how I rationalize the hippo-whale connection and recover my lost zoological composure.
Rendezvous 12. Xenarthrans. Rendezvous 12, about 95 million years ago, in the time of our 35 million great-grandparent, is where we meet the Xenarthran pilgrims from South America, which at that time had fairly recently torn itself away from Africa and was a very large island, just the thing for fostering the evolution of a unique fauna. The Xenarthrans are a rather odd group of mammals consisting of the armadillos, sloths, and anteaters, and their extinct relatives. Their name means alien joints, referring to the peculiar way their vertebrae join onto each other. They have extra articulations between their lumbar vertebrae, which strengthen the backbone for the digging that so many of them go in for. Among anteaters, only South American ones are xenarthrans. Other mammals, such as pangolins and aardvarks, also eat ants and are called, respectively, scaly anteaters and ant bears. All anteaters, by the way, might just as well be called termite eaters. They are very fond of termites. The armadillo's tail is the tail of South America in the age of mammals. It is the tail of a gigantic raft, like Madagascar, Australia, and India, cut adrift by the break-up of Gondwana. Madagascar is the subject of the aye-aye's tail, Australia that of the marsupial mole's tail. India would have been a fourth raft experiment, except that it travelled north so fast that it reached Asia rather early, and so its fauna became integrated with that of Asia during the latter half of the age of mammals. Africa, too, was a gigantic island during the rise of the mammals, not so isolated as South America and not for so long, but long enough for a large and very diverse group of mammals to go their own way in isolation, closer cousins to each other than to the rest of the mammals, though you'd never guess it to look at them. These are the Afrotheria, and we meet them at Rendezvous 13.
Rendezvous 13. Afrotheres. The Afrotheria are the last placental mammals to join our pilgrimage. They originated in Africa, as their name suggests, and they include the elephants, the elephant shrews, the dugongs and manatees, also known as sea elephants or sea cows, the hyraxes, the aardvarks or ant bears, and probably the tenrecs of Madagascar and the golden moles of southern Africa. The next pilgrims we shall greet will be our far more distant cousins, the marsupials, so the Afrotheria, all of them equally, are our most distant non-marsupial cousins. Concestor 13 lived 105 million years ago and was our 45 million great-grandparent, or thereabouts. Once again, it looked similar to Concestor 12 and Concestor 11, all pretty much like shrews.
Rendezvous 14. Marsupials. Marsupium means pouch in Latin. Anatomists use it as a technical term for any pouch, such as the human scrotum. But the most famous pouches in the animal kingdom are those in which kangaroos and other marsupials keep their young. Marsupials are born as tiny embryos equipped only to crawl for their tiny lives through the forest of their mother's fur into the pouch where they clamp their mouths to a teat. Of approximately 270 surviving species of marsupial in the world, about 3/4 are Australasian. The rest are all American, mostly opossums, plus a few other species, such as the enigmatic Dromiceops, the Monito del Monte. The 200 or so Australasian species have branched to fill the whole range of trades formerly occupied by the dinosaurs and independently occupied by other mammals in the rest of the world. The marsupial mole's tail goes through some of these trades, one by one, showing how similar each one is to its more familiar opposite number.
Rendezvous 15. Monotremes. Rendezvous 15 takes place approximately 180 million years ago in the half-monsoonal, half-arid world of the Lower Jurassic. The southern continent of Gondwana was still just about connected to the great northern continent of Laurasia, the first time on our backwards journey that we find all major landmasses collected into a contiguous Pangaea. In forward time, the split of Pangaea would have momentous consequences for the descendants of Concestor 15, perhaps 120 million great-grandparent. Our rendezvous is a rather one-sided affair. The new pilgrims that join all the rest of the mammals here represent only 3 genera: Ornithorhynchus anatinus, the duck-billed platypus, which lives in eastern Australia and Tasmania; Tachyglossus aculeatus, the short-beaked echidna, which lives all over Australia and New Guinea; and Zaglossus, the long-beaked echidna, which is confined to the highlands of New Guinea. Collectively, the 3 genera are known as monotremes.
The duckbill's tail. An early Latin name of the platypus was Ornithorhynchus paradoxus. It seems so weird when first discovered, that a specimen sent to a museum was thought to be a hoax, bits of mammal and bits of bird stitched together. Others have wondered whether God was having a bad day when he created the platypus. Finding some spare parts left over on the workshop floor, he decided to unite rather than waste them. More insidiously, because they aren't joking, some zoologists write the monotremes off as primitive, as though sitting around being primitive was a full-time way of life. To question this is a purpose of the duckbill's tale. Since their common ancestor with us, platypuses have had exactly the same time to evolve as the rest of the mammals. There's no reason why either group should be more primitive than the other. Primitive, remember, precisely means resembling the ancestor. Monotremes might be more primitive than us in some respects, such as laying eggs, but there is no reason at all why primitiveness in one respect should dictate primitiveness in another. There's no substance called essence of antiquity that pervades the blood and soaks into the bones. A primitive bone is a bone that has not changed much for a long time. There's no rule that says the neighbouring bone has to be primitive too, not even a faint presumption in that direction, at least unless a further case is made. There's no better illustration than the eponymous duckbill itself. It has evolved far, even if other parts of the platypus have not. The platypus bill seems comic. Its resemblance to that of a duck made the more incongruous by its relatively large size, and also because a duck's bill has a certain intrinsic laughableness, perhaps borrowed from Donald. But humour does an injustice to this wondrous apparatus. If you want to think in terms of an incongruous graft, forget all about ducks. A more telling comparison is the extra nose grafted onto a Nimrod reconnaissance aircraft. The American equivalent is AWACS, more familiar but less appropriate for my comparison in that the AWACS graft is on top of the fuselage rather than at the front like a bill. The point is that the platypus bill is not just a pair of jaws for dabbling and feeding as in a duck. It is that too, though it is rubbery rather than horny like a duck's bill. But far more interestingly, the platypus bill is a reconnaissance device, an AWACS organ. Platypuses hunt crustaceans, insect larvae, and other small creatures in the mud at the bottom of streams. Eyes aren't much use in mud, and the platypus keeps them tight shut while hunting. Not only that, it closes its nostrils and its ears as well. See no prey, hear no prey, smell no prey. Yet it finds prey with great efficiency, catching half its own weight in a day. If you were a skeptical investigator of somebody claiming a sixth sense, what would you do? You'd blindfold him, stop his ears and his nostrils, and then set him some task of sensory perception. Platypuses go out of their way to do the experiment for you. They switch off 3 senses which are important to us, and perhaps to them on land, as if to concentrate all their attention on some other sense. And the clue is given by one further feature of their hunting behaviour. They swing the bill in movements called saccades, side to side, as they swim. It looks like a radar dish scanning. One of the first scientific descriptions of the platypus, Sir Everard Hume's publication in The Philosophical Transactions of the Royal Society for 1802, was far-sighted. He noticed that the branch of the trigeminal nerve that innervates the face is
uncommonly large. We should be led by this circumstance to believe that the sensibility of the different parts of the bill is very great, and therefore that it answers the purpose of a hand, and is capable of nice discrimination in its feeling.
Sir Everard didn't know the half of it. It's the reference to a hand that tells. The great Canadian neurologist Wilder Penfield published a famous picture of a human brain, together with a diagram showing the proportions given over to different parts of the body. The striking thing about the Penfield homunculus is the huge prominence given to the hand. The human brain is hand-heavy. The distinguished Australian neurobiologist Jack Pettigrew and his colleagues prepared a platypunculus, the platypus equivalent of a Penfield homunculus. Again, there is an orderly spatial mapping between each part of the body and the corresponding part of the brain. The hands of the platypunculus are approximately in proportion to the body itself, unlike the case of the Penfield homunculus with its vast hands. What is not in proportion in the platypunculus is the bill. Where the human brain is hand-heavy, the platypus brain is bill-heavy. Sir Everard Hume's guess is looking good. But as we shall see, in one respect the bill is even better than a hand. It can reach out and feel things that it's not touching. It can feel at a distance. It does it by electricity. When any animal, such as a freshwater shrimp, which is a typical platypus prey, uses its muscles, weak electric fields are inevitably generated. With sufficiently sensitive apparatus, these can be detected, especially in water. Given dedicated computer power to handle data from a large array of such sensors, the source of the electric fields can be calculated. Platypuses don't, of course, calculate as a mathematician or a computer would, but at some level in their brain the equivalent of a calculation is done, and the result is that they catch their prey. Platypuses have about 40,000 electrical sensors distributed in longitudinal stripes over both surfaces of the bill. A large proportion of the brain is given over to processing the data from these 40,000 sensors. But the plot thickens. In addition to the 40,000 electrical sensors, there are about 60,000 mechanical sensors called push rods scattered over the surface of the bill. Pettigrew and his co-workers have found nerve cells in the brain that receive inputs from mechanical sensors, and they've found other brain cells that respond to both electrical and mechanical sensors. So far, they've found no brain cells that respond to electrical sensors only. Both kinds of cell occupy their correct position on the spatial map of the bill, and they are layered in a way that is reminiscent of the human visual brain, where layering assists binocular vision. Just as our layered brain combines information from the two eyes to construct a stereo percept, Pettigrew suggests that the platypus might be combining the information from electrical and mechanical sensors in some similarly useful way. How might this be done?
He proposes the analogy of thunder and lightning. The flash of lightning and the crack of thunder happen at the same moment. We see the lightning instantaneously, but the thunder takes longer to reach us, travelling at the relatively slow speed of sound, and incidentally the bang becomes a rumble because of echoes. By timing the lag between lightning and thunder, we can calculate how far away the storm is. Perhaps the electrical discharges from the prey's muscles are the platypus's lightning, while the thunder is the waves of disturbance in the water caused by the prey animal's movements. Is the platypus brain set up to compute the time lag between the two, and hence calculate how far away the prey is? It seems likely. As for pinpointing the prey's direction, this must be done by comparing the inputs from different receptors all over the map, presumably aided by the scanning side-to-side movements of the bill, just as a man-made radar uses the rotation of the dish. With such a huge array of sensors projecting to mapped arrays of brain cells, the platypus very likely forms a detailed three-dimensional image of any electrical disturbances in its vicinity. When you think of a platypus, forget duck, think Nimrod, think AWACS, think huge hand feeling its way by remote pins and needles, think lightning flashing and thunder rumbling through the watery mud of Australia. The sting in the tail of the platypus is actually in the hind claws of the male. True venomous stings with hypodermic injection are found in various invertebrate phyla and in fish and reptiles among vertebrates, but never in birds or mammals other than the platypus, unless you count the toxic saliva of selenodons and some shrews that make their bites slightly venomous. Among mammals, the male platypus is in a class of its own, and it may be in a class of its own among venomous animals too. The fact that the sting is found only in males suggests rather surprisingly that it is aimed not at predators, as in bees, nor at prey, as in snakes, but at rivals. It is not dangerous, but is extremely painful, and is unresponsive to morphine. it looks as though platypus venom works directly on pain receptors themselves. If scientists could understand how this is done, there is a hope that it might give a clue to how to resist the pain caused by cancer. This tale began by chiding those zoologists who call the platypus primitive, as though that were any kind of explanation for the way it is. At best, it is a description. Primitive means resembling the ancestor, and there are many respects in which this is a fair description of a platypus. The bill and the sting are interesting exceptions. But the more important moral of the tale is that even an animal that is genuinely primitive in all respects is primitive for a reason. The ancestral characteristics are good for its way of life, so there is no reason to change. As Professor Arthur Kane of Liverpool University liked to say, an animal is the way it is because it needs to be.
What the star-nosed mole said to the duck-billed platypus. The star-nosed mole, who had joined the pilgrimage along with the other Laurasiatheres at Rendezvous 11, listened to the duck-bill's tale with close attention and with growing recognition in what was left of his vestigial pinprick eyes. "Yes," he squeaked, too high for some of the larger pilgrims to hear, and he clapped his spades with excitement. "That's just the way it is for me. Well, sort of."
No, it won't do. I wanted to follow Chaucer in having at least one section devoted to what one pilgrim said to another, but I'll limit it to the heading and first paragraph, and now revert to my practice of telling the tale itself in my own words. Bruce Vogel, 101 Questions Your Dog Would Ask Its Vet, or Olivia Judson, Dr. Tatiana's Sex Advice to All Creation, might get away with it, but not me.
The star-nosed mole, Condylura cristata, is a North American mole which, in addition to burrowing and hunting for worms like other moles, is a good swimmer too, hunting for underwater prey. It's also more at home above ground than other moles, where it still prefers damp, soggy places. It has large, spade hands, like other moles. What sets it apart is the remarkable nose that gives it its name. Surrounding the two forward-pointing nostrils, there is an extraordinary ring of fleshy tentacles like a baby sea anemone with 22 arms. The tentacles are not used to grasp things, nor are they an aid to smelling, which is the next hypothesis that might occur to us. Nor are they an electrical radar like that of the platypus. Their true nature has been beautifully worked out by Kenneth Catania and John Cass at Vanderbilt University, Tennessee. The star is a touch-sensitive organ, like a super-sensitive human hand, but lacking the grasping function of the hand and emphasizing its sensitivity instead. But it isn't just any ordinary touch-sensitive organ. The star-nosed mole pushes the envelope of touch beyond our dreams. The skin of its nose is more sensitive than any other area of skin anywhere among the mammals, not excluding the human hand. If the star is not an electrical sensor, whence the empathy with the platypus with which this section began? Catania and Cass constructed a schematic model of the relative amount of brain tissue given over to different parts of the body surface. It is a molunculus, by analogy with Penfield's homunculus and Pettigrew's platypunculus. And it turns out that a vast proportion of the brain map is given over to, you've guessed it, the star nose. The star-shaped brain map dramatically displays where the star-nosed mole's priorities lie. It gives us a feel for the world of the star-nosed mole, and feel is the right word. This animal lives in a tactile world, dominated by the tentacles of the nose, with a subsidiary interest in the large spade hands and the whiskers, which are also prominently represented in the brain map.
What is it like to be a star-nosed mole? I am tempted to propose the star-nosed counterpart to an idea I once offered for bats. Bats live in a world of sound, but what they do with their ears is pretty much the same as what, say, insect-hawking birds like swallows do with their eyes. In both cases, the brain needs to construct a mental model of a three-dimensional world to be navigated at high speed, with obstacles to be avoided and small moving targets to catch. The model of the world needs to be the same whether it is constructed and updated with the aid of light rays or sound echoes. my conjecture was that a bat probably sees the world, using echoes, in pretty much the same way as a swallow or a person sees the world using light. I even went so far as to speculate that bats hear in colour. The hues that we perceive have no necessary link with the particular wavelengths of light that they represent. The sensation that I call red, and nobody knows if my red is the same as yours, is an arbitrary label for light of long wavelengths. It could equally well have been used for short wavelengths, blue, and the sensation that I call blue used for long wavelengths. Those hue sensations are available in the brain for tying to whatever in the outside world is most convenient. In bat brains, those vivid qualia would be wasted on light. They are more likely to be used as labels tied to particular qualities of echo, perhaps textures of surfaces on obstacles or prey. My conjecture now is that a star-nosed mole sees with its nose, and my speculation is that it uses those same qualia that we call colour as labels for tactile sensations. Similarly, I want to guess that duck-billed platypuses see with the bill and use the qualia we call colour as internal labels for electrical sensations.
Could this be why platypuses close their eyes tight shut when they're hunting electrically with the bill? Could it be because the eyes and the bill are competing in the brain for internal qualia labels, and to use both senses at once would lead to confusion?
Rendezvous 16. Sauropsids. Rendezvous 16 is where a huge throng of new pilgrims join us, the sauropsids. The sauropsids are by far the largest contingent of newcomers we have yet had to deal with along our pilgrim's way. For most of the years since Concestor 16 lived, sauropsids in the form of dinosaurs dominated the planet. Even today, with the dinosaurs gone, there still are more than 3 times as many sauropsid species as mammals. At Rendezvous 16, approximately 4,600 mammal pilgrims greet 9,600 bird pilgrims and 7,770 pilgrims from the rest of the reptiles: crocodiles, snakes, lizards, tuataras, turtles. They are the main group of land vertebrate pilgrims. The only reason I'm regarding them as joining us, rather than we joining them, is that we arbitrarily chose to see the journey through human eyes. The sauropsid pilgrims who now join us include the turtles, lizards and snakes, crocodiles and birds, together with the huge concourse of shadow pilgrims, the pterosaurs in the air, the ichthyosaurs, plesiosaurs and mosasaurs in water, and above all, the dinosaurs on land. Focused as this book is on pilgrims from the present, it is not appropriate to expatiate on the dinosaurs who dominated the planet for so long, and who would dominate it yet, but for the cruel, no, indifferent, meteorite that laid them low. It seems added cruelty to treat them now so indifferently. They do survive, after a fashion, the special and beautiful fashion of birds, and we shall do them homage by listening to tales of birds. But first, in memoriam, Shelley's well-known Ode to a Dinosaur.
I met a traveller in an antique land who said, "2 vast and trunkless legs of stone stand in the desert. Near them, on the sand, half sunk, a shattered visage lies, whose frown and wrinkled lip and sneer of cold command tell that its sculptor well those passions read which yet survive, stamped on these lifeless things, the hand that mocked them, and the heart that fed. And on the pedestal these words appear: My name is Ozymandias, King of Kings. Look on my works, ye mighty, and despair. Nothing beside remains. Round the decay of that colossal wreck, boundless and bare, the lone and level sands stretch far away."
Prologue to the Galapagos Finch's Tale. The human imagination is cowed by antiquity, and the magnitude of geological time is so far beyond the ken of poets and archaeologists it can be frightening. But geological time is large not only in comparison to the familiar timescales of human life and human history. It is large on the timescale of evolution itself. This would surprise those from Darwin's own critics on who have complained of insufficient time for natural selection to wreak the changes the theory requires of it. We now realize that the problem is, if anything, opposite. There has been too much time. If we measure evolutionary rates over a short time and then extrapolate, say, to 1 million years, the potential amount of evolutionary change turns out to be hugely greater than the actual amount. It is as though evolution must have been marking time for much of that period. Or, if not marking time, wandering around this way and that, with meandering fluctuations drowning out in the short term whatever trends there might be in the long. To repeat an analogy I've used before, think of a cork bobbing about off the Atlantic coast of America. The Gulf Stream imposes an overall eastward drift in the average position of the cork, which will eventually be washed up on some European shore. But if you measure its direction of movement during any 1 minute, buffeted by waves and eddies and whirlpools, it will seem to move west as often as east. You won't notice any eastward bias unless you sample its position over much longer periods. Yet the eastward bias is real, it is there, and it too deserves an explanation. The waves and eddies of natural evolution are usually too slow for us to see in our little lifetimes, or at least within the short compass of a typical research grant. There are a few notable exceptions. The school of E.B. Ford, the eccentric and fastidious scholar from whom my generation of Oxford zoologists learned our genetics, devoted decades of research to tracking the year-by-year fortunes of particular genes in wild populations of butterflies, moths, and snails. Their results in some cases seem to have straightforward Darwinian explanations. In other cases, the noise of buffeting waves drowns out the signal of whatever Gulf streams may have been tugging the undertow, and the results are enigmatic. The point I am now making is that such an enigma is to be expected by any mortal Darwinian, even a Darwinian with a research career as long as Ford's. One of the main messages Ford himself drew from his life's work was that the selection pressures actually to be found in nature, even if they don't always pull in the same direction, are orders of magnitude stronger than anything dreamed of by the most optimistic founders of the neo-Darwinian revival. And this again underlines the point. Why doesn't evolution go much faster than it does?
The Galapagos Finch's Tale. The Galapagos archipelago is volcanic and no more than 5 million years old. During that brief existence, a spectacular quantity of diversity has evolved, most famously among the 14 species of finches, widely, though perhaps wrongly, believed to have been Darwin's principal inspiration. The Galapagos finches are among the most thoroughly studied wild animals in existence. Peter and Rosemary Grant have devoted their professional lives to following the year-by-year fortunes of these small island birds. And in the years between Charles Darwin and Peter Grant, who himself bears a pleasing facial resemblance to Darwin, the great but clean-shaven ornithologist David Lack also paid them a perceptive and productive visit, which culminated in his book, Darwin's Finches. The Grants and their colleagues and students have been returning yearly to the Galapagos Islands for more than a quarter of a century, trapping finches, individually marking them, measuring their beaks and wings, and more recently taking blood samples for DNA analysis to establish paternities and other relationships. There's probably never been a more complete study of the individuals and genes of any wild population. The Grants know in minute detail exactly what is happening to the bobbing corks, which are the finch populations, as they are tossed this way and that in the sea of evolution by selection pressures that change every year. In 1977 there was a severe drought, and the food supply plummeted. The total number of individual finches of all species on the small island of Daphne Major dropped from 1,300 in January to less than 300 by December. The population of the dominant species, Geospiza fortis, the medium ground finch, dropped from 1,200 to 180. The cactus finch, G. scandens, fell from 280 to 110. Figures for other species confirmed that 1977 was a finch annus horribilis. But the Grant team didn't just count the numbers of each species dying and living. Being Darwinians, they looked at the selective mortality figures within each species. Were individuals with certain characteristics more likely to survive the catastrophe than others? Did the drought selectively change the relative composition of a population?
Yes, it did. Within the G. fortis population, the survivors were on average more than 5% larger than those that succumbed, and the average beak after the drought was 11.07 millimeters long compared with 10.68 millimeters before. The mean depth of beak had similarly gone up from 9.42 millimeters to 9.96 millimeters. These differences may seem tiny, but within the skeptical conventions of statistical science, they were too consistent to be due to chance. But why would a drought year favor such changes? The team already had evidence that larger birds with larger beaks are more efficient than average birds at dealing with the big, tough, spiky seeds, such as those of the weed tribulus, which were just about the only seeds to be found during the worst of the drought. A different species, the large ground finch, G. magnirostris, is the professional when it comes to handling tribulus seeds. But Darwinian survival of the fittest is all about the relative survival of individuals within a species, not the relative survival of one species compared to another. And within the population of medium ground finches, the largest individuals with the largest beaks survived best. The average G. fortis individual became a tiny bit more like G. magnirostris. The Grant team had observed a small episode of natural selection in action during a single year.
They witnessed another episode after the drought ended, which pushed the finch populations in the same evolutionary direction, but for a different reason. As with many species of bird, G. fortis males are larger than females, and they have larger beaks, which presumably equip them to survive the drought better. Before the drought, there were about 600 males and 600 females. Of the 180 individuals who survived, 150 were male. The rains, when they finally returned in January 1978, unleashed boom conditions which were ideal for breeding. But now there were 5 males for every female. Understandably, there was fierce competition among the males for the scarce females, and the males who won these sexual competitions, the new winners among the already larger than normal surviving males, again tended to be the largest males with the largest beaks. Once again, natural selection was driving the population to evolve larger body size and larger beaks, but for a different reason. If large size is such an advantage, why weren't the birds just larger in the first place? Because in other years, non-drought years, natural selection favors smaller individuals with smaller beaks. The Grants actually witnessed this in the years following 1982-83, when there happened to be an El Niño flood. After the flood, the balance of seeds changed. The large, tough seeds of plants such as tribulus became rare in comparison with the smaller, softer seeds of plants like cacabus. Now smaller finches with smaller beaks came into their own. It wasn't that large birds couldn't eat small, soft seeds, but they needed more of them to maintain their larger bodies. So smaller birds now had a slight edge, and within the population of medium ground finches, the tables were turned. The evolutionary trend of the drought years was reversed. The differences in beak size between the successful and the unsuccessful birds in the drought year seem awfully small, don't they? Jonathan Weiner quotes a telling anecdote about this from Peter Grant.
Once, just as I was beginning a lecture, a biologist in the audience interrupted me. "How much difference do you claim to see," he asked me, "between the beak of a finch that survives and the beak of a finch that dies?" 1/2 of a millimeter on average, I told him. "I don't believe it," the man said. "I don't believe 1/2 of a millimeter really matters so much." "Well, that's the fact," I said. "Watch my data and then ask questions." And he asked no questions.
Peter Grant calculated that it would take only 23 bouts of 1977-style drought on Daphne major to turn Geospiza fortis into G. magnirostris. It wouldn't literally be magnirostris, of course, but it is a vivid way to visualize the origin of species and how rapidly it can happen. Darwin little knew, when he met them and failed to label them properly, what powerful allies his finches would eventually turn out to be.
The dodo's tale. Land animals, for obvious reasons, have a hard time reaching remote oceanic islands such as the Galapagos archipelago or Mauritius. If, through the much-invoked freak accident of rafting inadvertently on a detached tree, they do happen to find themselves on an island like Mauritius, an easy life is likely to open up. This is precisely because it is hard to get to the island in the first place, so the competition and the predation are usually not so fierce as on the mainland left behind. As we have seen, this is probably how monkeys and rodents arrived in South America. If I say it is hard to colonize an island, I must hasten to forestall the usual misunderstanding. A drowning individual may try desperately to reach land, but no species ever tries to colonize an island. A species is not the kind of entity that tries to do anything. Individuals of a species may happen, by luck, to find themselves in a position to colonize an island previously uninhabited by their kind. The individuals concerned can then be expected to take advantage of the vacuum, and the consequence may be that their species, with hindsight, is said to have colonized the island. The descendants of the species may subsequently change their ways over evolutionary time to accommodate the unfamiliar island conditions. And now here is the point of the dodo's tale. It is hard for land animals to reach an island, but it is a lot easier if they have wings, like the ancestors of the Galapagos finches, or the ancestors of the dodo, whoever they were. Flying animals are in a special situation. They don't need the proverbial raft. Their wings carry them, perhaps as a freak accident, blown on a gale to a distant island. Having arrived on wings, they find that they no longer need them, especially because islands often lack predators. This is why island animals, as Darwin noted on the Galapagos, are often remarkably tame. And this is what makes them easy meat for sailors. The most famous example is the dodo, Rhaphus cuculatus, cruelly named Didus ineptus by Linnaeus, the father of taxonomy. The very name dodo comes from the Portuguese for stupid. Stupid is unfair. When Portuguese sailors arrived on Mauritius in 1507, the abundant dodos were completely tame and approached the sailors in a manner which cannot have been far from trusting. Why would they not trust? For their ancestors had not encountered a predator for thousands of years. Alas for trust! The unfortunate dodos were clubbed to death by Portuguese and later Dutch sailors, even though they were deemed unpalatable. Presumably, it was sport. Extinction took less than 2 centuries. As so often, it came about through a combination of killing and more indirect effects. Humans introduced dogs, pigs, rats, and religious refugees. The first 3 ate dodo eggs, and the last planted sugarcane and destroyed habitats. Conservation is a very modern idea. I doubt that extinction and what it means entered anybody's head in the 17th century. I can hardly bear to tell the story of the Oxford dodo, the last dodo stuffed in England. Its owner and taxidermist, John Tradescant, was induced to bequeath his large collection of curios and treasures to the infamous, some say, Elias Ashmole, which is why the Ashmolean Museum in Oxford is not called the Tradescantian, as some say it should be. Ashmole's curators, some say probably falsely, later decided to burn as rubbish all of Tradescant's dodo, except the beak and 1 foot. These are now in my place of work, the University Museum of Natural History, where they memorably inspired Lewis Carroll. Also, Hilaire Belloc.
The dodo used to walk around and take the sun and air. The sun yet warms his native ground. The dodo is not there. The voice which used to squawk and squeak is now forever dumb. Yet you may see his bones and beak all in the museum.
The ancestors of the dodos had wings. Their forebears were flying pigeons who arrived on the Mascarene Islands under their own muscle power, perhaps aided by a freak wind. Once there, they had no need to fly any more, nothing to flee, and so lost their wings. Like Galapagos and Hawaii, these islands are recent volcanic creations, none of them more than 7 million years old. Molecular evidence suggests that the dodos probably arrived on the Mascarene Islands from the east, not from Africa or Madagascar, as we might otherwise have supposed. Why bother to lose the wings? They took a long time to evolve. Why not hang on to them in case one day they might come in useful again? Alas, for the dodo, that is not the way evolution thinks. Evolution doesn't think at all, and certainly not ahead. If it did, the dodos would have kept their wings, and the Portuguese and Dutch sailors would not have had sitting targets for their vandalism. The late Douglas Adams was moved by the sad case of the dodo. In one of the episodes of Doctor Who that he wrote in the 1970s, the aged Professor Chronotis's college room in Cambridge serves as a time machine. Because of a strike at the BBC, this episode of Doctor Who was never broadcast, and Douglas Adams later recycled the story in his novel Dirk Gently's Holistic Detective Agency. Professor Chronotis uses his time machine for one purpose only, his secret vice. He obsessively and repeatedly visits 17th century Mauritius in order to weep for the dodo. Call me sentimental, but I must pause for a moment, for Douglas and for Professor Chronotis and what he wept for.
Evolution, or its driving engine, natural selection, has no foresight. In every generation, within every species, the individuals best equipped to survive and reproduce contribute more than their fair share of genes to the next generation. The consequence, blind as it is the nearest approach to foresight that nature admits. Wings might be useful 1,000,000 years hence when sailors arrive with clubs, but wings will not help a bird contribute offspring and genes to the next generation in the immediate here and now. On the contrary, wings, and especially the massive breast muscles needed to power them, are an expensive luxury. Shrink them, and the resources saved can now be spent on something more immediately useful, such as eggs. Immediately useful for surviving and reproducing the very genes that programmed the shrinkage. That's the kind of thing natural selection does all the time. It is always tinkering, here shrinking a bit, there expanding a bit, constantly adjusting, putting on and taking off, optimizing immediate reproductive success. Survival in future centuries doesn't enter into the calculation, for the good reason that it isn't really a calculation at all. It all happens automatically, as some genes survive in the gene pool and others don't.
The sad end of the Oxford dodo, Alice's dodo, Belloc's dodo, is mitigated by a happier sequel. A group of Oxford scientists in the laboratory of my colleague Alan Cooper obtained permission to take a tiny sample from inside one of the foot bones. They also obtained a thigh bone of a solitaire, the related species from the neighbouring island of Rodriguez. These bones yielded enough mitochondrial DNA to allow detailed letter-by-letter sequence comparisons between the two extinct birds and a wide range of living birds. The results confirm that, as long suspected, dodos were modified pigeons. It is also no surprise that, within the pigeon family, the closest relative of the dodo is the solitaire and vice versa. What is less expected is that these two extinct flightless giants are nested deep inside the pigeon family tree. In other words, dodos are more closely related to some flying pigeons than those flying pigeons are to other flying pigeons, despite the fact that, to look at them, you'd expect all the flying pigeons to be more closely related to each other, with the dodos out on a limb. Among pigeons, the dodos are most close to the knickerbocker, Calenus nickobarica, a beautiful pigeon from Southeast Asia. In turn, the group consisting of the Nicobar pigeon and the dodos is most closely related to the Victoria crowned pigeon, a splendid bird from New Guinea, and Didunculus, a rare Samoan tooth-billed pigeon, which looks quite like a dodo, and whose name even means little dodo. The Oxford scientists comment that the nomadic lifestyle of the Nicobar pigeon makes it ideally suited to invade remote islands, and Nicobar-type fossils are known from Pacific islands as far east as the Pitcairns. These crowned and tooth-billed pigeons, they go on to point out, are large, ground-dwelling birds who rarely fly. It looks as though this whole subgroup of pigeons habitually colonize islands and then lose their power of flight and become larger and more dodo-like. The dodo itself and the solitaire have pushed the trend to extremes.
Something like the dodo's tail has been repeated on islands all over the world. Many different families of birds, most of which are dominated numerically by flying species, have evolved flightless forms on islands. Mauritius itself had a large flightless rail, Aphanapteryx bonacea, also now extinct, which may on occasion have been confused with the dodo. Rodriguez had a related species, A. leguati. Rails seem to lend themselves to dodo-tail island hopping followed by flightlessness. In addition to the Indian Ocean forms, there is a flightless rail in the Tristan da Cunha group in the South Atlantic, and most of the Pacific islands have, or had, their own species of flightless rail. Before man ruined the Hawaiian avifauna, there were more than 12 species of flightless rail in that archipelago. More than a quarter of all the world's 60-odd living species of rail are flightless, and all flightless rails live on islands. Perhaps as many as 200 species have gone extinct on tropical Pacific islands since human contact. In all these cases, the evolutionary story is almost certainly a version of the dodo's tale. Ancestral flying birds are carried by their wings to a remote island where an absence of mammals opens up opportunities for making a living on the ground. Their wings are no longer useful in the way that they were on the mainland, so the birds give up flying, and their wings and costly wing muscles degenerate. There is one notable exception, one of the oldest and the most famous of all the groups of flightless birds, the ratites, the ostrich order. The evolutionary story of the ratites is very different from all the rest of the flightless birds, and they have a tale of their own.
The elephant bird's tale. From the tales of the Arabian Nights, the image that most stirred my childish imagination was the roc encountered by Sinbad the sailor, who at first thought this monstrous bird was a cloud come over the sun.
I had heard aforetime of pilgrims and travellers how in a certain island dwelleth a huge bird called the roc, which feedeth its young on elephants.
The legend of the roc, Ruck or Ruch, surfaces in several stories of the Arabian Nights. It is mentioned by Marco Polo as living in Madagascar, and envoys from the king of Madagascar were said to have presented the Khan of Cathay with a roc feather. Michael Drayton, 1563–1631, invoked the monstrous bird's name to contrast it with the proverbially tiny wren.
All feathered things yet ever known to men, from the huge ruck unto the little wren.
What is the origin of the roc legend? And if it is pure fantasy, whence the recurrent connection with Madagascar? Fossils from Madagascar tell us that a gigantic bird, the elephant bird Epeornis maximus, lived there perhaps until as late as the 17th century, although more probably around AD 1000. The elephant bird finally succumbed, perhaps partly through people stealing its eggs, which were up to 1 meter in circumference and would have produced as much food as 200 chicken eggs. The elephant bird was 3 meters tall and weighed nearly half a ton, as much as 5 ostriches. Unlike the legendary roc, which used its 16-meter wingspan to carry Sinbad aloft as well as elephants, the real elephant bird could not fly, and its wings were relatively small, like an ostrich's. But though a cousin, it would be wrong to imagine it as a scaled-up ostrich. It was a more robust, heavy-set bird, a kind of feathered tank with a big head and neck, unlike the ostrich's slender periscope. Given how legends readily grow and inflate, Epeornis is a plausible progenitor of the roc.
The elephant bird was probably vegetarian, unlike earlier groups of giant carnivorous birds, such as the Phorus rhacoid family of the New World. These could grow to the same height as Epeornis, with a fearsomely hooked beak which, as if in justification of their nickname of feathered tyrannosaurs, looks capable of swallowing a medium-sized lawyer whole. These monstrous cranes seem at first sight better casting for the role of the terrifying roc than Epeornis, but they went extinct too long ago to have started the legend, and in any case Sinbad, or his real-life Arab counterparts, never visited the Americas. The elephant bird of Madagascar is the heaviest bird known to have lived, but it was not the longest. Some species of moa could reach a height of 3.5 meters, but only if the neck was raised, as in some museum mountings. In life, it seems, they normally carried the head only a little way above the back. But the moa cannot have generated the rock legend, for New Zealand too was well beyond Sinbad's ken. About 10 moa species existed in New Zealand, and they ranged in size from turkey to double-sized ostrich. Moas are extreme among flightless birds in that they have no trace of wings at all, not even buried vestiges of wing bones. They thrived in both the North and South Islands of New Zealand until the recent invasion by the Maori people about AD 1250. They were easy prey, no doubt for the same reasons as the dodo. Except for the extinct Haast's eagle, the largest eagle ever to have lived, they'd known no predators for tens of millions of years, and the Maoris slaughtered them all, eating the choicer parts and discarding the rest, belying, not for the first time, the wishful myth of the noble savage living in respectful harmony with his environment. By the time the Europeans arrived, only a few centuries after the Maoris, the last moa was gone. Legends and tall stories of sightings persist to this day, but the hope is forlorn. In the words of a plaintive song to be sung in a mournful New Zealand accent,
No moa in old Aotearoa. Can't get 'em, they've et 'em. They've gone, and there ain't no moa.
Elephant birds and moas, but not the carnivorous Phorus rhacoids nor various other extinct flightless giants, were ratites, an ancient family of birds which now includes the rheas of South America, the emus of Australia, the cassowaries of New Guinea and Australia, the kiwis of New Zealand, and the ostrich, now confined to Africa and Arabia but previously common in Asia and even Europe.
I take delight in the power of natural selection, and it would have given me satisfaction to report that the ratites evolved their flightlessness separately in different parts of the world, conforming to the message of the dodo's tail. In other words, I would have liked the ratites to have been an artificial assemblage, driven to superficial resemblance by parallel pressures in different places. Alas, this is not so. The true tale of the ratites, which I am attributing to the elephant bird, is very different. And I must say that it eventually turns out to be, in its way, even more fascinating. The elephant bird's tail is a tale of Gondwana and of continental drift, or, as it is now called, plate tectonics.
The ratites are a truly natural group. Ostriches, emus, cassowaries, rheas, kiwis, moas, and elephant birds really are more closely related to each other than they are to any other birds. And their shared ancestor was flightless too. Probably it originally lost its wings, following good dodo's tail reasons, after flying to some long-forgotten island off Gondwana. But that was before the ratites divided into separate forms, whose descendants we now find on the different southern continents and islands. Moreover, the split of the ratites from the rest of the birds is extremely old. The ratites are a genuinely ancient group in the following sense. Surviving birds fall into two groups. On the one hand are the ratites and the tinamus, a group of South American birds which can fly. On the other hand are all the rest of the surviving birds put together. So if you are a bird, either you are a ratite tinamu or you are in with the rest, and the division between these two categories is the oldest split among surviving birds. The ratites, then, are a natural group with a common ratite ancestor that was also flightless. This is not to deny that an earlier ancestor of all the ratites flew. Of course it did, for why else would they, most of them, have vestigial wings? But the last common ancestor of all the surviving ratites had already reduced its wings to stubby vestiges long before its descendants branched into the various groups of ratites we see today. This deprives us of our accustomed dodo's tail of ancestors flying across the sea to distant lands, and then each independently losing its wings. The ratites reached their present separated homelands without benefit of flight. How did they get there?
They walked all the way. How is this possible? It is the whole point of the elephant bird's tail. The sea wasn't there. There was nothing to cross. What we now know as separate continents were joined together, and the great flightless birds walked dry-shod. When I was a small child in Africa, my father regaled my little sister and me with bedtime stories as we lay under our mosquito nets and marvelled at his luminous wristwatch, about a bronchosaurus who lived far away in a place called Gonwonkyland. I forgot all about this until much later, when I learned about the great southern continent of Gondwanaland. 150 million years ago, Gondwanaland, or Gondwana, consisted of everything that we now know as South America, Africa, Arabia, Antarctica, Australasia, Madagascar, and India. The southern tip of Africa was touching Antarctica and tilted to the right. There was therefore a triangular gap between the east coast of Africa and the north coast of Antarctica, but it wasn't really a gap because it was filled by India. India was in those days separated from the rest of Asia, Laurasia, by an ocean, the Tethys, whose centre roughly corresponds today to the modern Indian Ocean, and whose westernmost reaches turned into today's Mediterranean Sea. Madagascar nestled between India and Africa, joined on both sides. Australia with New Guinea and the embryonic New Zealand were also joined to Antarctica, further round the coast from India. But Gondwana was about to break up. You can see where the tale is going. When the ratite birds first roamed Gondwana, they could walk from any of the places where they later dwelt to any other. Ratite fossils have even been found in Antarctica, which we know from plant fossils to have been covered with warm subtropical forest at the time. Ancestral ratites wandered freely over the whole continent of Gondwana, with no inkling that their homeland was destined to be broken up into chunks, separated by thousands of miles of ocean. When it did break up, the ratites went too. They rafted all right, but their raft was not the proverbial fragment of vegetation. It was the very ground beneath their feet, and there was plenty of time for them to evolve their separate ways on their separately retreating land masses.
The break-up happened rather suddenly and explosively by the standards of geological time. About 150 million years ago, India, still with Madagascar attached, started to break away from Africa. As the gap widened between Africa and India-Madagascar, by 140 million years ago, clear water began to open up between the other side of India and Antarctica and between Australia and Antarctica. A little later, South America began to draw away from the west side of Africa, and by 120 million years ago an immensely long and narrow angled channel separated the two. The last place you could walk across was where West Africa just hung on by a thread to what is now Brazil. By then a similarly long, narrow channel had opened between Antarctica and the new south coast of Australia. Around 80 million years ago, Madagascar split off from India and remained approximately in its present position, while India began a spectacularly fast migration north, eventually to crunch into the south coast of Asia and raise the Himalayas. During the same period, the other fragments of Gondwana had continued to drift apart, each bearing its manifest of ratite passengers. Ancestral rheas on the new South American continent, ancestral elephant birds on India-Madagascar, ancestral emus on Australia, ancestral ostriches on... But no, let's postpone that matter.
Plant fossils tell us that Cretaceous Antarctica was subtropical, lush with vegetation, and a fine place for animals to live. The dearth of fossils that have actually been found cannot reflect a corresponding dearth of animals. A rich vegetation like that must have supported an equally rich fauna. As I've already mentioned, among the few fossil animals that have been found are large ratites, some as large as moas, and it seems likely that these birds were abundant in Cretaceous Antarctica. If not necessarily ratite Grand Central Station, Antarctica provided a clement and ratite-friendly land bridge linking Africa and South America on one side of the world to Australia and New Zealand on the other, and India-Madagascar too. From the point of view of a wandering ratite ancestor, what matters is not when the great bulk of its particular continent separated from the rest of Gondwana. What matters is the last moment when it could still have walked across the gap. For example, by 100 million years ago, Africa was widely separated from Antarctica in the south and from India, Madagascar in the east. From these points of view, Africa was already an island. It was also widely separated from South America, almost all the way along its west coast. But there was still a lingering bridge between the southern margin of the West African bulge and a part of what is now Brazil. This was the last moment of contact between the ancestors of the Rhea and the rest of what had once been Gondwana. We have other dates of last contact between the various elements of the Gondwanan continental diaspora.
Is there a coincidence between the times at which the various continents and islands split apart geographically and the times at which, on molecular genetic evidence, the corresponding ratite bird lineages split apart in evolution? Or if this is too much to ask, are the two sets of timings at least compatible with each other? Yes, they are. And with the exception of the kiwi, and in an interesting sense which we shall come to, the ostrich, they are incompatible with the alternative hypothesis that the ratites distributed themselves among their present land masses after those land masses separated from one another. Alan Cooper and his colleagues at Oxford, whom we met in the Dodo's Tale, have compared the molecular genetics of all the ratite birds. Doing this for surviving birds is easy. You just take blood from zoo specimens of ostriches, emus, and the rest. Indeed, lots of sequences have already been published in the technical literature. But the Cooper team achieved the additional coup of sequencing mitochondrial DNA from two genera of moas and an elephant bird, for which they had only old bones borrowed from museums. Remarkably, the team managed to piece together the entire mitochondrial genome of both genera of moas, although they were at least 700 years dead. The material from elephant birds was less well preserved, but still they managed to sequence some elephant bird DNA. These ancient DNA sequences could then be compared with each other and with the sequences of the surviving ratites. The molecular clock technique allowed them to put approximate dates on the evolutionary divergences among the ratites. Here's the best guess at what happened. Think of Antarctica as the unit from which the other continents split. Of course, the continents split from each other as well, but it helps to have a reference point, and Antarctica is conveniently central, which makes it easy to visualize. What is more, as we have seen, during the period that matters for our tale, in the Cretaceous period spanning roughly the 40 million years on either side of the 100 million year mark, Antarctica was by no means the frozen waste it is now. Is this because Antarctica was at a more clement latitude? No, it was only somewhat north of its present position. It was warm because coastal shapes in those days happened to direct warm currents from the tropics to far southern latitudes, in a more dramatic version of the way the Gulf Stream fosters palm trees in western Scotland today. One of the consequences of the breakup of Gondwana was that the warm current was no longer directed south. Antarctica reverted to the icy climate appropriate to its latitude, and it has been cold ever since. So there were plenty of ratites in Antarctica at just the right time. The rest of the tale is straightforward. South America was already well populated with the ancestors of the Rheas. New Zealand broke away from Antarctica about 70 million years ago, carrying the ancestral moas as cargo. The molecular data suggest that the moas had already diverged from the other ratites about 80 million years ago. Australia lost contact with Antarctica around 56 million years ago. This fits with the molecular evidence that the moas split off from other ratites earlier, 82 million years, than the Australian ratites, the emu and the cassowary, who diverged from one another around 30 million years ago. The kiwis are probably the one exception to the rule that the ratite birds walked everywhere. They are not closely related to the moas. Their affinities are with the Australian ratites, and they presumably island-hopped from Australia to New Zealand via New Caledonia. As for the elephant bird, it stayed in Madagascar after India broke away 75 million years ago and remained there until the arrival of man.
I said I would return to the ostrich. From about 90 million years ago, it was no longer possible to cross by land between Africa and any other part of the former Gondwana. This therefore constitutes the last moment at which, one might have thought, the the ostrich, being an African bird, could have diverged from the rest of the ratites. In fact, however, the molecular evidence suggests that the ostrich line diverged later, around 75 million years ago. How can this be? The argument is a little intricate, so let me repeat the problem. The geographic evidence suggests that Africa was already separated from the rest of the former Gondwana by about 90 million years ago, yet the molecular evidence suggests that the ostrich split off from the other Gondwana birds around 75 million years ago. Where were the ancestors of the ostrich during the intervening 15 million years? Presumably not in Africa, for the reason we've just seen. They could have been anywhere in the rest of Gondwana, because all the other bits, South America, Australia, New Zealand, and India-Madagascar, remained connected to each other, if only via Antarctica, and if only by lingering land bridges. How then do modern ostriches manage to end up in Africa? Alan Cooper has an ingenious theory. India-Madagascar remained connected to Antarctica via a large land bridge called the Kerguelen Plateau, now submerged, until 75 million years ago, when what is now Sri Lanka pulled away. Up till that moment, the ancestors of the ostrich and the elephant bird were still in contact with Antarctica, and hence with the rest of Gondwana, except Africa, which had earlier separated. Cooper believes that the ancestors of the ostrich and the elephant bird were in India-Madagascar at this time of separation. On this hypothesis, we can regard 75 million years as the last moment the ostrich and elephant bird lineages could have split away from the other ratites, and this fits well with the molecular data. Then, about 5 million years later, India detached itself from Madagascar, taking what were to become ostriches along for the ride, and leaving what were to become the elephant birds behind. So far, so good. But we are still left with the riddle which began this part of our tale. If the ancestors of the ostrich were ensconced in the then island of India, how did they finally get to Africa? Now we come to the climax of Cooper's theory. India, you will remember, after parting from Madagascar, sped off north to its present position as part of Asia. Cooper believes that it carried with it the ostrich ancestors, who took advantage of the collision to spill out into Asia. Then, once in Asia, the ostrich line fanned out in a great loop to the north. There are still ostriches in Arabia, and there are ostrich fossils in Asia, including India, and even in Europe. At that time, as now, Africa was connected to Asia via Arabia, and by this route the ostriches finally arrived, perhaps 20 million years ago, in Africa, where we now find them. According to Cooper, the ancestral ostriches were by no means the only animals to catch the India ferry to Asia. He suggests that India's cargo of Gondwanan animals played a major part in the recolonization of Asia after the catastrophe that killed the dinosaurs.
The legend of the roc, the fabulous great bird with the strength to shift elephants, is a wonder of childhood. But isn't the true story of how the very continents themselves are shifted through thousands of miles an even greater wonder, more worthy of the adult imagination?
Rendezvous 17. Amphibians. 340 million years ago, in the early Carboniferous period, only about 30 million years beyond the great milestone of Rendezvous 16, we amniotes, the name that unites mammals with reptiles and birds, meet our amphibian cousins at Rendezvous 17. Concestor 17, in the vicinity of our 175 million great-grandparent, is the ancestor of all surviving tetrapods. Tetrapod means 4 feet. We who don't walk on 4 feet are lapsed tetrapods, recently lapsed in our case, much less recently in the case of birds, but we are all called tetrapods. More to the point, Concestor 17 is the grand ancestor of the huge throng of land vertebrates. Despite my earlier strictures on the conceit of hindsight, the emergence of fish onto the land was a major transition in our evolutionary history. 3 main bands of modern amphibian pilgrims have joined forces long before they meet up with us amniotes. They are the frogs, and toads, the distinction is not a zoologically helpful one, the salamanders and newts, which are those species that return to the water to breed, and the caecilians, moist, legless burrowers or swimmers with a superficial resemblance to earthworms or snakes. The narrow-mouthed frog's tail is about how species separate in evolution. The axolotl's tail is about how evolutionary novelty sometimes takes off from the juvenile stages of an ancestor rather than the adult. The salamander's tail is a good example of my principle that a tale may be only partly about the teller. It may broaden out into a more general lesson, in this case what I've called the tyranny of the discontinuous mind.
The salamander's tail. Names are a menace in evolutionary history. It is no secret that paleontology is a controversial subject in which there are even some personal enmities. At least 8 books called Bones of Contention are in print. And if you look at what 2 paleontologists are quarrelling about, as often as not it turns out to be a name. Is this fossil Homo erectus, or is it an archaic Homo sapiens? Is this one an early Homo habilis or a late Australopithecus? People evidently feel strongly about such questions, but they often turn out to be splitting hairs. Indeed, they resemble theological questions, which perhaps gives a clue to why they arouse such passionate disagreements. The obsession with discrete names is an example of the tyranny of the discontinuous mind. The salamander's tail strikes a blow against the discontinuous mind.
The Central Valley runs much of the length of California, bounded by the coastal range to the west and by the Sierra Nevada to the east. These long mountain ranges link up at the north and the south ends of the valley, which is therefore surrounded by high ground. Throughout this high ground lives a genus of salamanders called Ensatina. The Central Valley itself, about 40 miles wide, is not friendly to salamanders, and they are not found there. They can move all round the valley, but normally not across it, in an elongated ring of more or less continuous population. In practice, any one salamander's short legs in its short lifetime don't carry it far from its birthplace. But genes, persisting through a longer time scale, are another matter. Individual salamanders can interbreed with neighbours whose parents may have interbred with neighbours further round the ring, and so on. There is therefore potentially gene flow all around the ring. Potentially. What happens in practice has been elegantly worked out by the research of my old colleagues at the University of California at Berkeley, initiated by Robert Stebbins and continued by David Wake.
In a study area called Camp Wallahi, in the mountains to the south of the valley, there are 2 clearly distinct species of Ensatina which do not interbreed. One is conspicuously marked with yellow and black blotches. The other is a uniform light brown with no blotches. Camp Wallahi is in a zone of overlap, but wider sampling shows that the blotched species is typical of the eastern side of the Central Valley, which here in Southern California is known as the San Joaquin Valley. The light brown species, on the contrary, is typically found on the western side of the San Joaquin. Non-interbreeding is the recognized criterion for whether 2 populations deserve distinct species names. It therefore should be straightforward to use the name Ensatina eschscholzii for the plain western species and Ensatina clauberi for the blotched eastern species. Straightforward, but for one remarkable circumstance, which is the nub of the tail. If you go to the mountains that bound the north end of the Central Valley, which up there is called the Sacramento Valley, you'll find only one species of Ensatina. Its appearance is intermediate between the blotched and the plain species, mostly brown with rather indistinct blotches. It is not a hybrid between the 2, that is the wrong way to look at it. To discover the right way, make 2 expeditions south, sampling the salamander populations as they fork to west and east on either side of the Central Valley. On the east side they become progressively more blotched until they reach the extreme of clauberi in the far south. On the west side, the salamanders become progressively more like the plain eschscholzii that we met in the zone of overlap at Camp Wallahi. This is why it is hard to treat eschscholzii and clauberi with confidence as separate species. They constitute a ring species. You'll recognize them as separate species if you only sample in the south. Move north, however, and they gradually turn into each other. Zoologists normally follow Stebbins' lead and place them all in the same species, but give them a range of subspecies names. Stebbins believes that the ancestors of Ensatina arrived at the north end of the Central Valley and evolved gradually down the two sides of the valley, diverging as they went. An alternative possibility is that they started in the south as, say, Eschscholtzii, then evolved their way up the west side of the valley, round the top, and down the other side, ending up as Cloudberi at the other end of the ring. Whatever the history, what happens today is that there is hybridization all round the ring except where the two ends of the line meet in the far south of California. As a complication, it seems that the Central Valley is not a total barrier to gene flow. Occasionally, salamanders seem to have made it across, for there are populations of one of the western subspecies on the eastern side of the valley where they hybridize with the eastern subspecies. Yet another complication is that there is a small break near the south end of the ring where there seem to be no salamanders at all. Presumably they used to be there, but have died out. Or maybe they are still there, but have not been found. The mountains in this area are rugged and hard to search. The ring is complicated, but a ring of continuous gene flow is, nevertheless, the predominant pattern in this genus, as it is with the better-known case of herring gulls and lesser black-backed gulls around the Arctic Circle. In Britain, the herring gull and the lesser black-backed gull are clearly distinct species. Anybody can tell the difference, most easily by the colour of the wingbacks. Herring gulls have silver-grey wingbacks, lesser blackbacks, dark grey, almost black. More to the point, the birds themselves can tell the difference too, for they don't hybridize, although they often meet and sometimes even breed alongside one another in mixed colonies. Zoologists therefore feel fully justified in giving them different names, Larus argentatus and Larus fuscus. But now here's the interesting observation and the point of resemblance to the salamanders. If you follow the population of herring gulls westward to North America, then on around the world, across Siberia and back to Europe again, you notice a curious fact. The herring gulls, as you move round the pole, gradually become less and less like herring gulls and more like lesser black-backed gulls, until it turns out that our Western European lesser black-backed gulls actually are the other end of a ring-shaped continuum which started with herring gulls. At every stage around the ring, the birds are sufficiently similar to their immediate neighbours in the ring to interbreed with them, until, that is, the ends of the continuum are reached and the ring bites itself in the tail. The herring gull and the lesser black-backed gull in Europe never interbreed, although they are linked by a continuous series of interbreeding colleagues all the way round the other side of the world. Ring species, like the salamanders and the gulls, are only showing us in the spatial dimension something that must always happen in the time dimension. Suppose we humans and the chimpanzees were a ring species. It could have happened, a ring perhaps moving up one side of the Rift Valley and down the other side, with two completely separate species coexisting at the southern end of the ring, but an unbroken continuum of interbreeding all the way up and back around the other side. If this were true, what would it do to our attitudes to other species, to apparent discontinuities generally? Many of our legal and ethical principles depend on the separation between Homo sapiens and all other species. Of the people who regard abortion as a sin, including the minority who go to the lengths of assassinating doctors and blowing up abortion clinics, many are unthinking meat-eaters and have no worries about chimpanzees being imprisoned in zoos and sacrificed in laboratories. Would they think again if we could lay out a living continuum of intermediates between ourselves and chimpanzees, linked in an unbroken chain of interbreeders, like the Californian salamanders? Surely they would. Yet it is the merest accident that the intermediates all happen to be dead. It is only because of this accident that we can comfortably and easily imagine a huge gulf between our two species, or between any two species for that matter.
I have previously recounted the case of the puzzled lawyer who questioned me after a public lecture. He brought the full weight of his legal acumen to bear on the following nice point. If species A evolves into species B, he reasoned closely, there must come a point when a child belongs to the new species B, but his parents still belong to the old species A. Members of different species cannot, by definition, interbreed with one another, yet surely a child would not be so different from its parents as to be incapable of interbreeding with their kind. Doesn't this, he wound up, wagging his metaphorical finger in the special way that lawyers, at least in courtroom dramas, have perfected as their own, undermine the whole idea of evolution? That is like saying, when you heat a kettle of cold water, there is no particular moment when the water ceases to be cold and becomes hot, therefore it is impossible to make a cup of tea. Since I always try to turn questions in a constructive direction, I told my lawyer about the herring gulls, and I think he was interested. He had insisted on placing individuals firmly in this species or that. He didn't allow for the possibility that an individual might lie halfway between two species, or a tenth of the way from species A to species B. Exactly the same limitation of thought hamstrings the endless debates about exactly when in the development of an embryo it becomes human, and when by implication abortion should be regarded as tantamount to murder. It is no use saying to these people that, depending upon the human characteristic that interests you, a fetus can be half human or 100th human. Human, to the qualitative, absolutist mind, is like diamond. There are no halfway houses. Absolutist minds can be a menace. They cause real misery, human misery. This is what I call the tyranny of the discontinuous mind, and it leads me to develop the moral of the salamander's tale. For certain purposes, names and discontinuous categories are exactly what we need. Indeed, lawyers need them all the time. Children are not allowed to drive, adults are. The law needs to impose a threshold, for example, the 17th birthday. Revealingly, insurance companies take a different view of the proper threshold age. Some discontinuities are real by any standards. You are a person, and I am another person, and our names are discontinuous labels that correctly signal our separateness. Carbon monoxide really is distinct from carbon dioxide. There is no overlap. A molecule consists of a carbon and 1 oxygen, or a carbon and 2 oxygens. None has a carbon and 1.5 oxygens. One gas is deadly poisonous, the other is needed by plants to make the organic substances that we all depend upon. Gold really is distinct from silver. Diamond crystals really are different from graphite crystals. Both are made of carbon, but the carbon atoms naturally arrange themselves in two quite distinct ways. There are no intermediates. But discontinuities are often far from so clear. My newspaper carried the following item during a recent flu epidemic. Or was it an epidemic? That question was the burden of the article.
Official statistics show there are 144 people in every 100,000 suffering from flu, said a spokeswoman for the Department of Health. As the usual gauge of an epidemic is 400 in every 100,000, it is not being officially treated as an epidemic by the government. But the spokeswoman added, Professor Donaldson is happy to stick by his version that this is an epidemic. He believes it is many more than 144 per 100,000. It's very confusing, and it depends on which definition you choose. Professor Donaldson has looked at his graph and said it is a serious epidemic.
What we know is that some particular number of people are suffering from flu. Doesn't that in itself tell us what we want to know? Yet for the spokeswoman, the important question is whether this counts as an epidemic. Has the proportion of sufferers crossed the Rubicon of 400 per 100,000? This is the great decision which Professor Donaldson had to make as he pored over his graph. You'd think he might have been better employed trying to do something about it, whether or not it counted officially as an epidemic. As it happens, in the case of epidemics, for once there really is a natural Rubicon, a critical mass of infections above which the virus or bacterium suddenly takes off and dramatically increases its rate of spreading. This is why public health officials try so hard to vaccinate more than a threshold proportion of the population against, say, whooping cough. The purpose is not just to protect the individuals vaccinated, it is also to deprive the pathogens of the opportunity to reach their own critical mass for takeoff. In the case of our flu epidemic, what should really worry the spokeswoman for the Ministry of Health is whether the flu virus has yet crossed its Rubicon for takeoff and leapt abruptly into high gear in its spread through the population. This should be decided by some means other than reference to magic numbers like 400 per 100,000. Concern with magic numbers is a mark of the discontinuous mind, or qualitative mind. The funny thing is that, in this case, the discontinuous mind overlooks a genuine discontinuity, the takeoff point for an epidemic. Usually, there isn't a genuine discontinuity to overlook. Many Western countries at present are suffering what is described as an epidemic of obesity. I seem to see evidence of this all around me, but I am not impressed by the preferred way of turning it into numbers. A percentage of the population is described as clinically obese. Once again, the discontinuous mind insists on separating people out into the obese on one side of a line, the non-obese on the other. That is not the way real life works. Obesity is continuously distributed. You can measure how obese each individual is, and you can compute group statistics from such measurements. Counts of numbers of people who lie above some arbitrarily defined threshold of obesity are not illuminating, if only because they immediately prompt a demand for the threshold to be specified and may be redefined. The same discontinuous mind also lurks behind all those official figures detailing the number of people below the poverty line. You can meaningfully express a family's poverty by telling us their income, preferably expressed in real terms of what they can buy, or you can say X is as poor as a church mouse, or Y is as rich as Croesus, and everybody will know what you mean. But spuriously precise counts or percentages of people said to fall above or below some arbitrarily defined poverty line are pernicious. They are pernicious because the precision implied by the percentage is instantly belied by the meaningless artificiality of the line. Lines are impositions of the discontinuous mind. Even more politically sensitive is the label black as opposed to white in the context of modern society, especially American society. This is the central issue in the grasshopper's tale, and I'll leave it for now, except to say that I believe race is yet another of the many cases where we don't need discontinuous categories, and where we should do without them, unless an extremely strong case in their favor is made. Here's another example. Universities in Britain award degrees that are classified into 3 distinct classes, first, second, and third class. Universities in other countries do something equivalent, if under different names like A, B, C, etc. Now, my point is this. Students do not really separate neatly into good, middling, and poor. There are not discrete and distinct classes of ability or diligence. Examiners go to some trouble to assess students on a finely continuous numerical scale, awarding marks or points that are designed to be added to other such marks, or otherwise manipulated in mathematically continuous ways. The score on such a continuous numerical scale conveys far more information than classification into one of 3 categories. Nevertheless, only the discontinuous categories are published. In a very large sample of students, the distribution of ability and prowess would normally be a bell curve, with a few doing very well, few doing very badly, and many in between. It might not actually be a symmetrical bell curve, but it would certainly be smoothly continuous, and it would become smoother as more and more students are added in. A few examiners, especially I hope I'll be forgiven for adding in non-scientific subjects, seem actually to believe that there really is a discrete entity called the first-class mind, or the alpha mind, and a student either definitely has it or definitely hasn't. The task of the examiner is to sort out the firsts from the seconds and the seconds from the thirds, just as one might sort sheep from goats. The likelihood that in reality there is a smooth continuum sliding from pure sheepiness through all intermediates to pure goatiness is a difficult one for some kinds of mind to grasp. If, against all my expectations, it should turn out that the more students you add in, the more the distribution of exam marks approximates to a discontinuous distribution with 3 peaks like the Eiger, the Mönch, and the Jungfrau, it would be a fascinating result. The awarding of first, second, and third class degrees might then actually be justifiable. But there is certainly no evidence for this, and it would be very surprising given everything we know about human variation. As things are, it is clearly unfair. There is far more difference between the top of one class and the bottom of the same class than there is between the bottom of one class and the top of the next class. It would be fairer to publish the actual marks obtained, or a rank order based upon those marks. But the discontinuous or qualitative mind insists on forcing people into one or other discrete category.
Returning to our topic of evolution, what about sheep and goats themselves? Are there sharp discontinuities between species, or do they merge into each other, like first-class and second-class exam performances? If we look only at surviving animals, the answer is normally yes, there are sharp discontinuities. Exceptions like the gulls and the Californian salamanders are rare, but revealing because they translate into the spatial domain, the continuity which is normally found only in the temporal domain. People and chimpanzees are certainly linked via a continuous chain of intermediates and a shared ancestor, but the intermediates are extinct. What remains is a discontinuous distribution. The same is true of people and monkeys and of people and kangaroos, except that the extinct intermediates lived longer ago. Because the intermediates are nearly always extinct, we can usually get away with assuming that there is a sharp discontinuity between every species and every other. But in this book, we're concerned with evolutionary history, with the dead as well as the living. When we are talking about all the animals that have ever lived, not just those that are living now, evolution tells us there are lines of gradual continuity linking literally every species to every other. When we're talking history, even apparently discontinuous modern species like sheep and dogs are linked via their common ancestor in unbroken lines of smooth continuity. Ernst Mayr, distinguished elder statesman of 20th century evolution, has blamed the delusion of discontinuity, under its philosophical name of essentialism, as the main reason why evolutionary understanding came so late in human history. Plato, whose philosophy can be seen as the inspiration for essentialism, believed that actual things are imperfect versions of an ideal archetype of their kind. Hanging somewhere in ideal space is an essential perfect rabbit, which bears the same relation to a real rabbit as a mathematician's perfect circle bears to a circle drawn in the dust. To this day, many people are deeply imbued with the idea that sheep are sheep and goats are goats, and no species can ever give rise to another, because to do so, they'd have to change their essence.
There is no such thing as essence. No evolutionist thinks that modern species change into other modern species. Cats don't turn into dogs or vice versa. Rather, cats and dogs have evolved from a common ancestor who lived tens of millions of years ago. If only all the intermediates were still alive, attempting to separate cats from dogs would be a doomed enterprise, as it is with the salamanders and the gulls. Far from being a question of ideal essences, separating cats from dogs turns out to be possible only because of the lucky, from the point of view of the essentialist, fact that the intermediates happen to be dead. Plato might find it ironic to learn that it is actually an imperfection, the sporadic ill fortune of death, that makes the separation of any one species from another possible. This, of course, applies to the separation of human beings from our nearest relatives, and indeed from our more distant relatives too. In a world of perfect and complete information, fossil information as well as recent, discrete names for animals would become impossible. Instead of discrete names, we would need sliding scales, just as the words hot, warm, cool, and cold are better replaced by a sliding scale such as Celsius or Fahrenheit. Evolution is now universally accepted as a fact by thinking people, so one might have hoped that essentialist intuitions in biology would have been finally overcome. Alas, this hasn't happened. Essentialism refuses to lie down. In practice, it is usually not a problem. Everyone agrees that Homo sapiens is a different species, and most would say a different genus, from Pan troglodytes, the chimpanzee. But everyone also agrees that if you follow human ancestry backward to the shared ancestor and then forward to chimpanzees, the intermediates all along the way will form a gradual continuum in which every generation would have been capable of mating with its parent or child of the opposite sex. By the interbreeding criterion, every individual is a member of the same species as its parents. This is an unsurprising, not to say platitudinously obvious conclusion, until you realize that it raises an intolerable paradox in the essentialist mind. Most of our ancestors throughout evolutionary history have belonged to different species from us by any criterion, and we certainly couldn't have interbred with them. In the Devonian period, our direct ancestors were fish. Yet, although we couldn't interbreed with them, we are linked by an unbroken chain of ancestral generations, every one of which could have interbred with their immediate predecessors and immediate successors in the chain. In the light of this, see how empty are most of those passionate arguments about the naming of particular hominid fossils. Homo ergaster is widely recognized as the predecessor species that gave rise to Homo sapiens, so I'll play along with that for what follows. To call Homo ergaster a separate species from Homo sapiens could have a precise meaning in principle, even if it is impossible to test in practice. It means that if we could go back in our time machine and meet our Homo ergaster ancestors, we could not interbreed with them.
But suppose that, instead of zooming directly to the time of Homo ergaster, or indeed any other extinct species in our ancestral lineage, we stopped our time machine every 1,000 years along the way and picked up a young and fertile passenger. We transport this passenger back to the next 1,000-year stop and release her, or him. Let's take a female and a male at alternate stops. Provided our one-stop time traveller could accommodate to local social and linguistic customs, quite a tall order, there would be no biological barrier to her interbreeding with a member of the opposite sex from 1,000 years earlier. Now we pick up a new passenger, say a male this time, and transport him back another 1,000 years. Once again, he too would be biologically capable of fertilizing a female from 1,000 years before his native time. The daisy chain would continue on back to when our ancestors were swimming in the sea. It could go back without a break to the fishes, and it would still be true that each and every passenger transported 1,000 years before its own time would be able to interbreed with its predecessors. Yet at some point, which might be 1,000,000 years back but might be longer or shorter, there would come a time when we moderns could not interbreed with an ancestor, even though our latest one-stop passenger could. At this point, we could say that we have traveled back to a different species. The barrier would not come suddenly. There would never be a generation in which it made sense to say of an individual that he is Homo sapiens, but his parents are Homo ergaster. You can think of it as a paradox if you like, but there's no reason to think that any child was ever a member of a different species from its parents, even though the daisy chain of parents and children stretches back from humans to fish and beyond. Actually, it isn't paradoxical to anybody but a dyed-in-the-wool essentialist. It is no more paradoxical than the statement that there is never a moment when a growing child ceases to be short and becomes tall, or a kettle ceases to be cold and becomes hot. The legal mind may find it necessary to impose a barrier between childhood and majority, the stroke of midnight on the 18th birthday, or whenever it is. But anyone can see that it is unnecessary for some purposes fiction. If only more people could see that the same applies to when, say, a developing embryo becomes human.
Creationists love gaps in the fossil record. Little do they know biologists have good reason to love them too. Without gaps in the fossil record, our whole system for naming species would break down. Fossils could not be given names, they'd have to be given numbers or positions on a graph. Or, instead of arguing heatedly over whether a fossil is really, say, an early Homo ergaster or a late Homo habilis, we might call it habigaster. There's a lot to be said for this. Nevertheless, perhaps because our brains evolved in a world where most things do fall into discrete categories, and in particular where most of the intermediates between living species are dead, we often feel more comfortable if we can use separate names for things when we talk about them. I'm no exception, and neither are you, so I shall not bend over backwards to avoid using discontinuous names for species in this book. But the salamander's tale explains why this is a human imposition rather than something deeply built into the natural world. Let us use names as if they really reflected a discontinuous reality, but let's privately remember that, at least in the world of evolution, it is no more than a convenient fiction, a pandering to our own limitations.
Rendezvous 18. Lungfish. At Rendezvous 18, around 417 million years ago, we are joined in the warm and shallow seas of the Devonian-Silurian boundary by a tiny trickle of pilgrims who have plodded a lonely course from the present. They are the lungfish, and they join us to look at the common ancestor we share with them, an experience that may seem less strange to them than to us, for they find they have much in common with Consistor 18. Approximately our 185 million great-grandparent, it was a Sarcopterygian, a lobe-fin fish, certainly much more like a lungfish than like a tetrapod. The lungfish's tale is about living fossils, creatures that haven't changed much since the time of their remote ancestors. A more famous living fossil joins the pilgrimage around the same time at Rendezvous 19.
Rendezvous 19. Coelacanths. Consistor 19, perhaps our 190 million great-grandparent, lived around 425 million years ago, just as plants were colonizing the land and coral reefs expanding in the sea. At this rendezvous we meet one of the sparsest, most tenuous band of pilgrims in this story. We know of only one genus of coelacanth alive today, and its discovery was a huge surprise when it happened. The coelacanths were well known in the fossil record, but thought to have gone extinct before the dinosaurs. Then, astoundingly, a living coelacanth turned up in the catch of a South African trawler in 1938. By good fortune, Captain Harry Goosen, skipper of the Narita, was friendly with Marjorie Courtney Latimer, the enthusiastic young curator of the East London Museum. It was Goosen's habit to put aside interesting finds for her, and on the 22nd of December 1938 he telephoned to tell her he had something. She went down to the quay, and an old Scotsman of the crew showed her a motley collection of discarded fish, which at first didn't seem of any interest. She was about to leave when
I saw a blue fin, and pushing off the fish, the most beautiful fish I had ever seen was revealed. It was 5 feet long and a pale mauve blue with iridescent silver markings.
She made a sketch of the fish, which she sent to South Africa's leading ichthyologist, Dr. J.L.B. Smith, and it knocked him out.
I would not have been more surprised if I'd seen a dinosaur walking down the street.
Unfortunately, Smith took his time going to the scene for reasons that are hard to fathom. By Keith Thompson's account in Living Fossil, Smith didn't trust his judgment until he had sent off for a particular reference book from Dr. Barnard, a colleague in Cape Town. Smith hesitantly confessed his secret to Barnard, who was immediately sceptical. It seems that it was weeks before Smith could bring himself to go to East London and actually see the fish. Meanwhile, poor Miss Courtney Latimer was coping with its noisome decay. Too large to fit in a formalin jar, she wrapped it in formalin-soaked cloths. These were inadequate to stave off decay, and eventually she had to have it stuffed. It was in this form that Smith finally saw it.
Coelacanth! Yes, by God! Although I had come prepared, that first sight hit me like a white-hot blast, and made me feel shaky and queer. My body tingled. I stood as if stricken to stone. I forgot everything else, and then, almost fearfully, went close up and touched and stroked, while my wife watched in silence. It was only then that speech came back. The exact words I have forgotten, but it was to tell them that it was true. It really was true. It was, unquestionably, a coelacanth. Not even I could doubt any more.
Smith named it Latimeria, after Marjorie. Since then, many more have been found in deep waters around the Comoros Islands near Madagascar, and a second species turned up on the other side of the Indian Ocean, off Sulawesi. The genus has now been studied in detail, although not without the acrimony and accusations of fakery that seem, regrettably, but I suppose understandably, to go with rare and very important discoveries.
Rendezvous 20. Ray-finned fish. Rendezvous 20 is a big one, 440 million years ago in the earliest Silurian, still with a southern ice cap left over from the cold Ordovician. Consistor 20, estimated to be our 195 million great-grandparent, is the one that unites us to the actinopterygian, or ray-finned fish, most of whom belong to the large and successful group known as teleosts. The teleost fish are the great success story among modern vertebrates. There are some 23,500 species of them. They're prominent at many levels of underwater food chains in both salt and freshwater. They have managed to invade hot springs at one extreme and the icy waters of the Arctic seas and high mountain lakes at the other. They thrive in acid streams, stinking marshes, and saline lakes. The teleost pilgrims arrive in a jostling crowd, brilliant in their variety. The magnitude of that variety is the inspiration for the next tale.
The Leafy Sea Dragon's Tale. When my daughter was tiny, she loved to ask adults to draw fish for her. She would rush up to me when I was trying to write a book, thrust a pencil in my hand and clamour,
"Draw a fish, Daddy, draw a fish!"
The cartoon fish that I would immediately draw to keep her quiet, and the only kind of fish she ever wanted me to draw, was always the same, a regulation issue fish like a herring or a perch, streamlined side view, pointed at the front end, triangular fin top and bottom, triangular tail at the back, finally dotted with an eye bracketed by the curve of a gill cover. I don't think I ran to pectoral or pelvic fins, which was remiss of me, because they all have them. The standard fish is indeed an extremely common shape, one that obviously works well over the full range of sizes, from minnow to tarpon. What would Juliet have said if I had possessed the skill to draw for her a leafy sea dragon, Phycoturus equus?
"No, Daddy, not seaweed. Draw a fish! Draw a fish!"
The message of the leafy sea dragon's tale is that animal shapes are malleable, like plasticine. A fish can change in evolutionary time to whatever unfishy shape is required for its way of life. Those fish that look like the standard issue Juliet fish do so only because it suits them. It is a good shape for swimming through open water. But if survival is a matter of hanging motionless in beds of gently swaying kelp, the standard fish shape can be twisted and kneaded, pulled out in fantastically branched projections, whose resemblance to the fronds of brown seaweed is so great that a botanist might be tempted to narrow it down to species, perhaps of the genus Fucus. Or what would Juliet have said if I'd drawn for her a snipe eel, Nemicthiidae, or a gulper eel, Euripharynx pelicanoides, two deep-sea eels with birds in their names? The snipe eel looks like a joke, ludicrously long and thin, with bird-like jaws that curve away from each other like a megaphone. So dysfunctional do these diverging jaws look, I can't help wondering how many of the fish have been seen alive. Could the megaphone jaws be a distortion in a dried-up museum specimen? The gulper looks like a nightmare. With jaws ludicrously too large for its body, or so it would seem, it is capable of swallowing whole prey larger than itself, one of several deep-sea fishes with this remarkable talent. It is not unusual, of course, for predators to kill prey larger than themselves, and then eat them in bits. Lions do it, so do spiders. But to swallow a larger animal than yourself whole is hard to imagine. The gulper eel, and other deep-sea fish, such as the closely related swallower eel and the unrelated black swallower, which is not an eel, achieve the trick. They do it by a combination of disproportionately oversized jaws and a slack, distensible stomach that hangs down only when full, looking rather like some gross external tumour. After the long digestion period, the stomach shrinks again. Why the trick of prodigious swallowing should be peculiar to snakes and deep-sea fish is not obvious to me. The gulper and the swallower eel lure prey into the vicinity of their mouth with a luminous lure at the tip of the tail. The teleost body plan seems almost indefinitely malleable over evolutionary time, tolerant of being pulled or squashed into any shape, however distantly removed from the standard fish shape. The oceanic sunfish's Latin name, mola mola, means millstone, and it is easy to see why. Seen from the side, it looks like a huge disk, up to an astonishing 4 meters in diameter and weighing up to 2 tons. The circularity of its outline is broken only by two gigantic fins on top and underneath, each one up to 2 meters long. The hippo's tail invoked, in explanation of its dramatic difference from its whale cousins, the liberation from gravity that whales must have enjoyed as soon as they severed all ties with the land. No doubt something similar explains the great variety of shapes that the teleost fish display. But in exploiting that liberation, teleosts have one other advantage over, for example, sharks. Teleosts cope with buoyancy in a very special way, and the pike will tell the tale.
The pike's tale. In the sad province of Ulster, where the mountains of Mourne sweep down to the sea, I know a beautiful lake. A party of children were swimming naked there one day, when somebody shouted that they'd seen a large pike. Instantly all the boys, but not the girls, fled to dry land. The northern pike, Esox lucius, is a formidable predator of small fish. It is beautifully camouflaged, not against predators, but to help it steal up on its prey. A stealth predator, and not particularly fast over a distance, it hangs almost motionless in the water, creeping imperceptibly forward until within striking distance. During the deadly creep, it propels itself with imperceptible movements of the rear-mounted dorsal fin. The whole hunting technique depends upon the ability to hang in the water at the desired level, like a drifting dirigible, without any effort, in perfect hydrostatic equilibrium. All locomotor work is concentrated on the clandestine business of creeping forwards. If a pike needed to swim in order to maintain its level as many sharks do, its ambush technique would not work. Effortless maintenance and adjustment of hydrostatic equilibrium is what teleost fish are supremely good at, and it may be the single most significant key to their success. How do they do it? By means of the swim bladder, a modified lung filled with gas which provides sensitive dynamic control of the animal's buoyancy. Except for some bottom dwellers who've secondarily lost the swim bladder, all teleosts have it, not just pike and not just their prey. The swim bladder is often explained as working like a Cartesian diver, but that is not quite correct. A Cartesian diver is a miniature diving bell containing a bubble of air which hangs at hydrostatic equilibrium in a bottle of water. When the pressure is increased, usually by squeezing down the cork in the neck of the bottle, the bubble is compressed and less water is displaced by the diver as a whole. Therefore, by Archimedes' principle, the diver sinks. If the cork is eased slightly upwards so that the pressure in the bottle decreases, the bubble in the diver expands, more water is displaced, and the diver floats a little higher. So, with your thumb on the cork, you can exert fine control over the level at which the diver finds its equilibrium. The key point about a Cartesian diver is that the number of air molecules in the bubble remains fixed, while the volume and the pressure are changed in inverse proportions following Boyle's law. If fish worked like Cartesian divers, they would use muscle power to squeeze or relax the swim bladder, thereby changing the pressure and volume, but leaving the number of molecules the same. That would work in theory, but it isn't what happens. Instead of keeping the number of molecules fixed and adjusting the pressure, a fish adjusts the number of molecules. To sink, the fish absorbs some molecules of gas from its swim bladder into the blood, thereby reducing the volume. To rise, it does the reverse, releasing molecules of gas into the swim bladder. In some teleosts, the swim bladder is also used to assist in hearing. The fish's body being mostly water, sound waves propagate through it pretty much as they did through the water before they hit the fish. When they strike the wall of the swim bladder, however, they suddenly reach a different medium, gas. The swim bladder therefore acts as a kind of eardrum. In some species it lies right against the inner ear. In others it is connected to the inner ear by a series of small bones called the Weberian ossicles. These do a similar job to our own hammer, anvil, and stirrup, but are completely different bones. The swim bladder seems to have evolved, been co-opted, from a primitive lung, and some surviving teleosts, such as bowfins, gars, and beshears, still use it for breathing. This perhaps comes as a little surprise to us, for whom breathing air seems like a significant advance that went with leaving the water for the land. One might have supposed the lung to be a modified swim bladder. On the contrary, it seems that the primitive breathing lung forked in evolution and went two ways. On the one hand, it carried its old breathing function out onto the land, and we use it still. The other branch of the fork was the new and exciting one. The old lung became modified to form a genuine innovation, the swim bladder.
The Flounder's Tale. An endearing quality of Chaucer is the naive perfectionism of his general prologue, where he introduces his pilgrims. It wasn't enough to have a doctor of physic on the pilgrimage. He had to be the finest doctor in the land.
In all this world, ne was there none him like, to speak of physic and of surgery.
The
very perfect gentle knight
was, it seemed, unmatched in Christendom for bravery, loyalty, and even temper. As for his squire and son, he was
a lover and a lusty bachelor, wonderly deliver and of great strength.
To top it all, he was
as fresh as is the month of May.
Even the knight's yeoman knew all there was to know of woodcraft. The reader comes to take it for granted that if a profession is mentioned, its practitioner will automatically turn out to be unrivalled in all England. Perfectionism is a vice of evolutionists. We are so used to the wonders of Darwinian adaptation, it is tempting to believe there could be nothing better. Actually, it is a temptation that I can almost recommend. A surprisingly strong case can be built for evolutionary perfection, but it must be done with circumspection and sophisticated attention. Here I shall give just one example of a historical constraint, the so-called jet engine effect. Imagine how imperfect a jet engine would be if, instead of being designed on a clean drawing board, it had to be changed one step at a time, screw by screw and rivet by rivet, from a propeller engine. A skate is a flat fish that might have been designed on a drawing board to be flat, resting on the belly with wide wings reaching symmetrically out to both sides. Teleost flatfish do it in a different way. They rest on one side, either the left, e.g. plaice, or the right, e.g. turbot and flounder. Whichever the side, the shape of the whole skull is distorted so that the eye on the lower side moves over to the upper side where it can see. Picasso would have loved them. But by the standards of any drawing board, they are revealingly imperfect. They have precisely the kind of imperfection you would expect from being evolved rather than designed.
The Mudskipper's Tale. On an evolutionary pilgrimage, it is fitting that some of the tales, though told by surviving pilgrims, should deal with recent reenactments of ancient evolutionary events. Teleost fish are so variable and so versatile, it is only to be expected that some of them might replay parts of the lobefin's history and come out onto the land. The mudskipper is just such a fish out of water, and it lives to tell the tale. A number of teleost fish species live in swampy water, poor in oxygen. Their gills cannot extract enough, and they need help from the air. Familiar aquarium fish from the swamps of Southeast Asia, such as the Siamese fighting fish Betta splendens, frequently come to the surface to gulp air, but they still use their gills to extract the oxygen. Since the gills are wet, you could say the gulping is equivalent to locally oxygenating their gill water, as you might bubble air through your aquarium. It goes further than that, however, because the gill chamber is furnished with an auxiliary air space richly supplied with blood vessels. This cavity is not a true lung. The true homologue of the lung in teleost fish is the swim bladder which, as the pike's tail has shown, they use for keeping their buoyancy neutral. Those fish that breathe air through their gill chambers have rediscovered air breathing by a completely different route. Perhaps the most advanced exponents of the air-breathing gill chamber are the climbing perches, anabas. These fish also live in poorly oxygenated water, and they have the habit of walking over the land looking for water when their previous home has dried up. They can survive out of water for days at a time.
Another group of walking teleost fish are the mudskippers, for example, Periophthalmus, whose tail this is. Some mudskippers actually spend more time out of water than in it. They eat insects and spiders, which are not normally found in the sea. It is possible that our Devonian ancestors enjoyed similar benefits when they first left the water, for they were preceded onto the land by both insects and spiders. A mudskipper flaps its body across the mudflats, and it can also crawl using its pectoral arm fins, whose muscles are so well developed that they can support the fish's weight. Indeed, mudskipper courtship takes place partly on land, and a male may do push-ups, as some male lizards do, to show off his golden chin and throat to females. The fin skeleton, too, has evolved convergently to resemble that of a tetrapod, such as a salamander. Mudskippers can jump more than half a metre by bending the body to one side and suddenly straightening it, hence some of their many vernacular names including mudhopper, johnny jumper, frogfish, and kangaroo fish. Another common name, climbing fish, comes from their habit of climbing mangrove trees looking for prey. They cling to the trees with the pectoral fins, aided by a kind of sucker which is made by bringing the pelvic fins together under the body. Like the swamp fish already mentioned, mudskippers breathe by taking air into their moist gill chambers. They also take in oxygen through the skin, which has to be kept moist. If a mudskipper is in danger of drying out, it will roll about in a puddle. Their eyes are especially vulnerable to dryness, and they sometimes wipe them with a wet fin. The eyes bulge close together near the top of the head, where, as with frogs and crocodiles, they can be used as periscopes to see above the surface when the fish is under water. When out on land, a mudskipper will frequently withdraw its bulging eyes into their sockets to moisten them. Before leaving the water on a land sortie, the fish will fill its gill cavities with water. In a popular book on the conquest of the land, the author mentions an account by an 18th century artist living in Indonesia who kept a frogfish alive for 3 days in his house.
It followed me everywhere with great familiarity, much like a little dog.
The book has a cartoon of a frogfish walking like a little dog, but what it actually depicts is clearly an anglerfish, a deep-sea fish with a lure on the end of a spine sticking up above the head, used to catch smaller fish. I suspect that the cartoonist has been the victim of a misunderstanding, an instructive one, because it shows what can happen if we rely on colloquial common names for animals rather than the scientific names that, whatever their faults, are designed to be unique. It is true that some people call anglerfish frogfish, but it is highly implausible that the fish that followed the artist around like a dog could have been a deep-sea anglerfish. It could easily have been a mudskipper, however. They do live in Indonesia, and frogfish is one of their colloquial names. A mudskipper looks, to my eyes at least, far more like a frog than an anglerfish ever could, and it leaps like a frog. I conjecture that the artist's pet frogfish, which followed him around like a little dog, was a mudskipper. I like the idea that we are descended from some creature which, even if it was different from a modern mudskipper in many other respects, was as adventurous and enterprising as a little dog, the nearest thing perhaps to a dog that the Devonian had to offer. A girlfriend of mine from long ago explained why she loved dogs. Dogs are such good sports. I think the first fish to venture out onto the land must have been an archetypal good sport, whom it would be a pleasure to call ancestor. Rendezvous 21. Sharks and their kin.
Out of the murderous innocence of the sea.
The context of Yeats's poem was completely other, but I can't help it. The phrase always makes me think of a shark. Murderous, but innocent of deliberate cruelty, just making a living as perhaps the world's most effective killing machine. I know people for whom the great white shark is their worst nightmare. If you are one of them, you may not wish to know that the Miocene shark, Carcharocles megalodon, was 3 times the size of a great white, with jaws and teeth to scale. My own recurrent nightmare, having grown up as an exact contemporary of the atomic bomb, is not a shark, but a huge, black, futuristic delta-winged aircraft, bristling with high-tech missile launchers, filling the sky with its shade and my heart with foreboding. almost exactly the shape of a manta ray, in fact. The dark shape that roars over the treetops of my dreams with its twin gun turrets so enigmatically menacing is a sort of technological cousin to Manta birostris. I always found it hard to accept that these 7-meter monsters are harmless filter feeders straining plankton through their gills. They're also extremely beautiful. A word association test that mentioned shark would very probably elicit the response jaws. So it is appropriate that Concestor 21, perhaps our 200 million great-grandparent, is the grand ancestor of all the vertebrates that have true jaws, the gnathostomes. Gnathos, in Greek, means lower jaw, and that is specifically what sharks and all the rest of us share. It was one of the triumphs of classical comparative anatomy to demonstrate that jaws evolved from modified parts of the gill skeleton. The next pilgrims to join us at Rendezvous 22 are the jawless vertebrates, the agnatha, well endowed with gills but with no lower jaw. Once numerous, diverse and heavily armoured, the agnatha are now reduced to the eel-shaped lampreys and hagfish. At Rendezvous 23 and 24 we are joined by lineages of vertebrate cousins, the amphioxuses and the sea squirts. They are not vertebrates, but they do belong to the same phylum, Chordata, as the vertebrates. Then, at Rendezvous 25, the echinoderms, starfish, sea urchins and their kin, join us. They are invertebrates, but closer to us than the great bulk of invertebrates whom we are to meet at the next rendezvous.
Rendezvous 26. Protostomes. In the deeps of geological time, and increasingly deprived of the hard support of fossils, we are now entirely reliant on the technique that I referred to in the general prologue as molecular range finding. The upside is that the technique is getting ever more sophisticated. Molecular range finding confirms a belief long held by comparative anatomists, or more strictly, comparative embryologists, that the greater part of the animal kingdom is deeply divided into two great subkingdoms, the Deuterostomia and the Protostomia. Here's how embryology comes in. Animals typically pass through a watershed event in their early life called gastrulation. The distinguished embryologist and scientific iconoclast Lewis Wolpert said,
"It is not birth, marriage or death, but gastrulation, which is truly the most important time in your life."
Gastrulation is something that all animals do early in their life. Typically, before gastrulation, an animal embryo consists of a hollow ball of cells, the blastula, whose wall is 1 cell thick. During gastrulation, the ball indents to form a cup with 2 layers. The opening of the cup closes in to form a small hole called the blastopore. Almost all animal embryos go through this stage, which presumably means it is a very ancient feature indeed. You might expect that so fundamental an opening would become one of the 2 deep holes in the body, and you'd be right. But now comes the big divide in the animal kingdom, between the Deuterostomia, every pilgrim who arrived before Rendezvous 26, including us, and the Protostomia, a huge throng who are now joining at Rendezvous 26. In deuterostome embryology, the eventual fate of the blastopore is to become the anus, or at least the anus develops close to the blastopore. The mouth appears later as a separate perforation at the other end of the gut. The protostomes do it differently. In some, the blastopore becomes the mouth, and the anus appears later. In others, the blastopore is a slit that subsequently zippers up in the middle, with the mouth at one end and the anus at the other. Protostome means mouth first. Deuterostome means mouth second. This traditional embryological classification of the animal kingdom has been upheld by modern molecular data. There are indeed 2 main kinds of animal, the deuterostomes, our lot, and the protostomes, them over there.
Rendezvous 26 is the biggest of all, more of a gigantic rally of pilgrims than a rendezvous. When does it happen? Such ancient dates are hard to estimate. My attempt of 590 million years is plus or minus a large margin of error. The same goes for the estimate that Concestor 26 is our 300 million great-grandparent. The protostomes constitute the great bulk of the pilgrimage of animals. Because our own species is of the deuterostome persuasion, I have given them special attention in this book, and I am portraying the protostomes as joining the pilgrimage altogether at one major rendezvous. Not only the protostomes themselves would see it the other way around, a dispassionate observer would too.
The protostomes have a much greater number of animal phyla than the deuterostomes, including the largest phyla of all. They include the mollusks, with twice as many species as the vertebrates. They include the 3 great worm phyla, flatworms, roundworms, and annelid worms, whose species together outnumber the mammal species perhaps 30-fold. Above all, the protostome pilgrims include the arthropods, insects, crustaceans, spiders, scorpions, centipedes, millipedes, and several other smaller groups. The insects alone constitute at least 3/4 of all animal species, and probably more. As Robert May, the current president of the Royal Society, has said, "To a first approximation, all species are insects."
The ragworm's tail. Any animal that moves in the sense of covering the ground from A to B, rather than just sitting in one place and waving its arms or pumping water through itself, is likely to need a specialized front end. It might as well have a name, so let's call it the head. The head hits novelty first. It makes sense to take in food at the end that encounters it first, and to concentrate the sense organs there too, eyes perhaps, some kind of feelers, organs of taste and smell. Then the main concentration of nervous tissue, the brain, had best be near the sense organs and near the action at the front end, where the food-catching apparatus is. So we can define the head end as the leading end, the one with the mouth, the main sense organs and the brain, if there is one. Another good idea is to avoid wastes somewhere near the back end, far from the mouth, to avoid re-imbibing what has just been passed out. By the way, although all this makes sense if we think worm, I should remind you that the argument evidently does not apply to radially symmetrical animals such as starfish. I am genuinely puzzled why starfish and their kind opt out of this argument. To return to our primeval worm, having dealt with its fore and aft asymmetry, how about up-down asymmetry? Why is there a dorsal side and a ventral side? The argument is similar, and this one applies to starfish just as much as to worms. Gravity being what it is, there are lots of inevitable differences between up and down. Down is where the sea bottom is, down is where the friction is, up is where the sunlight comes from, up is the direction from which things fall on you. It is unlikely that dangers will threaten equally from below and above, and in any case those dangers are likely to be qualitatively different. So our primitive worm should have a specialized upper or dorsal side and a specialized ventral or lower side, rather than simply not caring which side faces the sea bottom and which side faces the sky. Put the front-rear asymmetry together with the dorsal-ventral asymmetry, and we have automatically defined a left side and a right side. But, unlike the other two axes, we find no general reason to distinguish the left side from the right side, no reason why they should be anything other than mirror images. Danger is not more likely to threaten from the left than the right, or vice versa. Food is not more likely to be found on the left or the right side, though it may well be more likely above or below. Whatever is the best way for a left side to be, there is no general reason to expect any difference for the right. Limbs or muscles that were not mirrored left-right would have the unfortunate effect of driving the animal round in circles, instead of in direct pursuit of some goal. Perhaps revealingly the best exception I can think of is fictional. According to a Scottish legend, probably invented for the amusement of tourists and said to be believed by many of them, the haggis is a wild animal living in the Highlands. It has short legs on one side and long legs on the other, in accordance with its habit of running only one way round the sides of steep Highland hillsides. The prettiest real-life example I can think of is the wonky-eyed jewel squid of Australian waters, whose left eye is much larger than its right. It swims at a 45-degree angle, with the larger telescopic eye looking upwards for food, while the smaller right eye looks below for predators. The wrybill is a New Zealand sandpiper whose bill curves markedly to the right. The bird uses it to flick pebbles to the side and expose prey. Striking handedness is to be seen in fiddler crabs, who have one hugely enlarged claw for fighting, or more to the point, displaying their ability to fight. But perhaps the most intriguing story of asymmetry in the animal kingdom was told me by Sam Turvey. Trilobite fossils often display bite marks, indicating narrow escapes from predators. The fascinating thing is that about 70% of these bite marks are on the right-hand side. Either trilobites had an asymmetrical awareness of predators, like the wonky-eyed jewel squid, or their predators had handedness in their attack strategy.
But those are all exceptions, mentioned for their curiosity value and to make a revealing contrast with the symmetrical world of our primitive worm and its descendants. Our crawling archetype has a left and a right side which are mirror images of each other. Organs tend to arise in pairs, and where there are exceptions, such as the wonky-eyed jewel squid, we notice it and comment. The brain, sitting at the front end, for the reasons we have argued, needs to make nervous contact with the rest of the body. In a worm-shaped animal, it is sensible that it should do so via a main cable, a principal nerve trunk, running along the length of the body, probably with side branches at intervals along the body to exercise local control and take in local information. In a bilaterally symmetrical animal like a ragworm or a fish, the trunk nerve must run either dorsal or ventral of the digestive tract, and here we strike one of the main differences between us deuterostomes on the one hand and the protostomes, who've joined us in such strength, on the other. In us, the spinal nerve cord runs along the back. In a typical protostome, like a ragworm or a centipede, it is on the ventral side of the gut.
If Concestor 26 was indeed some kind of worm, it presumably followed either the dorsal nerve pattern or the ventral nerve pattern. I can't call them deuterostome and protostome patterns because the two separations don't quite coincide. Let me instead divide the animal kingdom into the dorsochords and the ventrichords. The dorsochords are all deuterostomes. The ventrichords are mostly protostomes, plus some early deuterostomes, perhaps including the acorn worms. Probably the deuterostomes were still ventrichords until some time later than Concestor 26. In 1820, the great French zoologist Geoffroy Saint-Hilaire suggested that a vertebrate could be thought of as an arthropod or an earthworm turned upside down. After Darwin and the acceptance of evolution, zoologists from time to time suggested that the vertebrate body plan actually had evolved through a worm-like ancestor literally turning upside down. That is the theory that I want to support here, on balance and with some caution. The alternative, which is that a worm-like ancestor gradually rearranged its internal anatomy while staying the same way up, seems to me less plausible, because it would have involved a greater amount of internal upheaval. I believe a change in behavior came first, suddenly by evolutionary standards, and it was followed by a whole lot of consequential evolutionary changes. As so often, there are modern equivalents to make the idea vivid for us today. The brine shrimp is one example, and we hear its tale next.
The brine shrimp's tale. Brine shrimps, Artemia, and the closely related fairy shrimps are crustaceans that swim on their backs and therefore have their nerve cord, the true zoological ventral side, on the side that now faces the sky. The upside-down catfish, Cynodontis nigriventris, is a deuterostome that does the same thing the other way round. It is a fish that swims on its back, and therefore has its main trunk nerve on the side facing the river bottom, which is the true zoological dorsal side.
I don't know why brine shrimps do it, but the catfish swim upside down because they take food from the water surface, or from the undersides of floating leaves. Presumably, individual fish discovered that this was a good source of food and learned to turn over. My conjecture is that, as the generations went by, natural selection favored those individuals who learned to perform the trick best. Their genes caught up with the learning, and now they never swim any other way. The brine shrimp's inversion is a recent reenactment of something that happened, in my view, more than 500 million years ago. an ancient, long-lost animal, some kind of worm with a ventral nerve cord and a dorsal heart like any protostome, turned over and swam, or crawled, upside down like a brine shrimp. A zoologist who happened to be present at the time would have died rather than relabel the main nerve trunk dorsal, just because it now ran along the side of the body facing the sky. Obviously, all his zoological training would have told him it was still a ventral nerve cord, corresponding to all the other organs and features that we expect to see on the ventral side of a protostome. Equally obvious to this Precambrian zoologist, the heart of our inverted worm was, in the deepest sense, a dorsal heart, even though it now beat under the skin nearest the sea bottom.
Given enough time, however, given enough millions of years of swimming or crawling upside down, natural selection would come to reshape all the organs and structures of the body to fit in with the upside-down habit. Eventually, unlike our modern brine shrimp, which has only recently turned over, the traces of the original dorsal-ventral homologies will become obliterated. Later generations of paleozoologists who encountered the descendants of this early maverick, after some tens of millions of years of upside-down habit, would start to redefine their concepts of dorsal and ventral. This is because so many anatomical details would have changed over evolutionary time. Other animals that swim on their backs are sea otters, especially when engaging in their remarkable habit of smashing shellfish with stones on the belly, and the aptly named backswimmers all the time. Backswimmers are a kind of bug, sometimes known as greater water boatmen, which row themselves underwater with their legs. The related lesser water boatmen do the same kind of thing, but they swim the right way up. Imagine that the descendants of our modern water boatmen or brine shrimps on the one hand, and the descendants of our modern upside-down catfish on the other, were to maintain their habits of swimming upside down for 100 million years into the future. Isn't it entirely likely that they might each give rise to a whole new sub-kingdom, each body plan so radically reshaped by the upside-down habit that zoologists who didn't know the history would define the brine shrimp's descendants as having a dorsal nerve cord and the descendants of the catfish as having a ventral nerve cord?
As we saw in the ragworm's tale, the world presents important practical differences between up and down, and these would start to imprint themselves by natural selection on the sky-pointing side and the floor-pointing side, respectively. What had once been the zoologically ventral side would start to look more and more like a zoologically dorsal side, and vice versa. I believe this is exactly what happened somewhere along the line leading to vertebrates, and that is why we now have a dorsal nerve cord and a ventral heart. There is nothing sacrosanct about dorsal and ventral. They can reverse, and I think they did reverse in the early ancestry of today's dorsochords. My bet is that Concestor 26 had its main trunk nerve running along the ventral side of the body like any protostome. We are modified worms swimming on our backs, descended from an early equivalent of a brine shrimp which, for some long-forgotten reason, turned over. The more general moral of the brine shrimp's tale is this. Major transitions in evolution may have begun as changes in behavioral habit, perhaps even non-genetic learned changes of habit, which only later were followed by genetic evolution. I fancy that a comparable tale could be told for the first bird ancestor to fly, the first fish to come out on the land, and the first whale ancestor to return to the water. A change of habit by an adventurous individual is later followed by a long evolutionary catch-up and clean-up. That is the most far-reaching lesson from the brine shrimp's tale.
The grasshopper's tale. The grasshopper's tale treats of the vexed and sensitive topic of race. There is a pair of European grasshopper species, Corthippus bruneus and C. bigutulus, which are so similar that even expert entomologists can't tell them apart, yet they never crossbreed in the wild, although they sometimes meet. This defines them to be good species. But experiments have shown that you need only allow a female to hear the mating call of a male of her own species caged nearby, and she will happily mate with a male of the wrong species, thinking, one is tempted to say, that he is the singer. When this happens, healthy and fertile hybrids are produced. It doesn't normally happen in the wild because a female doesn't normally find herself near but unable to reach a singing male of her own species at the same time as a male of the wrong species is courting her. It is quite common to find pairs of related species that never interbreed under natural conditions, but that can do so if humans interfere. The case of Corthippus bruneus and C. bigutulus is just one example. Biologists normally classify animals that mate under artificial conditions but refuse to mate in the wild as separate species, as has happened with the grasshoppers. But unlike, say, lions and tigers, which can hybridize in zoos to make sterile ligers and tigrons, those grasshoppers look identical. Apparently the only difference is in their songs. And it is this, and only this, that stops them crossbreeding and therefore leads us to recognize them as separate species. Human beings are the other way round. It requires an almost superhuman feat of political zeal to overlook the conspicuous differences between our own local populations or races. Yet we happily interbreed across races and are unequivocally and uncontroversially defined as members of the same species. The grasshopper's tale is about races and species, about the difficulties of defining both, and what all this has to say about human races.
Race is not a clearly defined word. Species, as we've seen, is different. There really is an agreed way to decide whether two animals belong in the same species. Can they interbreed? Obviously they can't if they're of the same sex, or are too young or too old, or one of them happens to be sterile. But those are pedantries easy to get around. In the case of fossils, too, which obviously can't breed, we apply the interbreeding criterion in our imaginations. Do we think it likely that, if these two animals were not fossils but alive, fertile and of opposite sex, they would be able to interbreed? The interbreeding criterion gives the species a unique status in the hierarchy of taxonomic levels. Above the species level, a genus is just a group of species that are pretty similar to each other. No objective criterion exists to decide how similar they have to be, and the same is true of all the higher levels: family, order, class, phylum, and the various sub- or super-names that intervene between them. Below the species level, race and subspecies are used more or less interchangeably, and again, no objective criterion exists that would enable us to decide whether two people should be considered part of the same race or not, nor to decide how many races there are. And of course there's the added complication, absent above the species level, that races interbreed, so there are lots of people of mixed race. Presumably species, on their way to becoming sufficiently separate to be incapable of interbreeding, usually pass through an intermediate stage of being separate races. Separate races might be regarded as species in the making, except that there's no necessary expectation that the making will continue to its end, to speciation. The interbreeding criterion works pretty well, and it delivers an unequivocal verdict on humans and their supposed races. All living human races interbreed with one another. We are all members of the same species, and no reputable biologist would say any different. But let me call your attention to an interesting, perhaps even slightly disturbing fact. While we happily interbreed with each other, producing a continuous spectrum of interraces, we are strangely reluctant to give up our divisive racial language. Wouldn't you expect that if all intermediates are on constant display, the urge to classify people as one or the other of two extremes would wither away, smothered by the absurdity of the attempt, which is continually manifested everywhere we look? Unfortunately, this is not what happens, and perhaps that very fact is revealing. People who are universally agreed by all Americans to be black may draw less than 1/8 of their ancestry from Africa, and often have a light skin color well within the normal range for people universally agreed to be white. Colin Powell is always described as black, but photographs of him together with his colleagues Bush and Rumsfeld suggest that his skin is not obviously darker than theirs. It's an interesting exercise to take such a photograph, cut a small rectangle from each forehead, and place the patches side by side. You will find that there's very little difference between Powell and the white men with whom he is standing. He may be lighter or darker, depending upon the particular cases. But now zoom out and look again at the original photograph. Immediately, Powell will look black. What cues are we picking up on? To ram home the point, do the same forehead patch exercise with Powell standing next to a genuinely black man such as Daniel Arap Moi, the recent president of Kenya. This time, the forehead patches will look dramatically different. But then, when we zoom out and look at the whole faces, we again see Mr. Powell as black. The news story that accompanied one such picture of Powell visiting Moi in May 2001 implied that the same conventions are understood in Africa.
As the first African-American Secretary of State, Powell has been accorded near messianic treatment in Africa. And perhaps because he is black, there was a profound resonance to Powell's harsh criticism.
Why do people so readily swallow the apparent contradiction, and there are numerous similar examples, between the verbal statement, "he is black," and the picture it accompanies? What is going on here? Various things. First, we are curiously eager to embrace racial classification, even when talking about individuals whose mixed parentage seems to make a nonsense of it, and even where it is irrelevant to anything that matters. Second, we tend not to describe people as of mixed race. Instead, we plump for one race or the other. Some American citizens are of pure African descent, and some are of pure European descent, leaving aside the fact that in the longer term we are all of African descent. Maybe it is convenient for some purposes to call them black and white respectively, and I'm not proposing any principled objection to these names. But many people, probably more than most of us realize, have both black and white ancestors. If we're going to use color terminology, many of us are presumably somewhere in between. Yet society insists on calling us one or the other. It is an example of the tyranny of the discontinuous mind, which was the subject of the salamander's tale. Americans are regularly asked to fill in forms in which they have to tick one of 5 boxes. Caucasian, whatever that might mean, it certainly doesn't mean from the Caucasus. African American, Hispanic, whatever that might mean, it certainly doesn't mean Spanish. Native American, or other. There are no boxes labeled half and half. But the very idea of ticking boxes is incompatible with the truth, which is that many, if not most people, are a complicated mixture of the offered categories and others. My inclination is irritably to refuse to tick any boxes, or to add my own box labeled human, especially when the rubric uses the mealy-mouthed euphemism ethnicity. Third, in the particular case of African Americans, there is something culturally equivalent to genetic dominance in our use of language. When Mendel crossed wrinkled peas with smooth peas, all the first-generation progeny were smooth. Smooth is dominant, wrinkled is recessive. The first-generation progeny all had one smooth allele and one wrinkled, yet the peas themselves were indistinguishable from peas with no wrinkled genes. When an Englishman marries an African, the progeny are intermediate in color and in most other characteristics. This is unlike the situation in peas, but we all know how society will call such children black every time. Blackness is not a true genetic dominant like smoothness in peas, but social perception of blackness behaves like a dominant. It is a cultural or mimetic dominant. That insightful anthropologist Lionel Tiger has attributed this to a racist contamination metaphor within white culture. And no doubt there is also a strong and understandable will on the part of descendants of slaves to identify with their African roots. Fourth, there is high inter-observer agreement in our racial categorizations. A man such as Colin Powell, of mixed race and intermediate physical characteristics, is not described as white by some observers and black by others. A small minority will describe him as mixed. All others will without fail describe Mr. Powell as black. And the same goes for anybody who shows the slightest trace of African ancestry, even if their percentage of European ancestors is overwhelming. Nobody describes Colin Powell as white. There is a useful technique called inter-observer correlation. It is a measure that's often used in science to establish that there really is a reliable basis for a judgment, even if nobody can pin down what that basis is. The rationale in the present case is this. We may not know how people decide whether somebody is black or white, and I hope I've just demonstrated that it isn't because they are black or white. But there must be some sort of reliable criterion lurking there, because any two randomly chosen judges will come to the same decision. The fact that the inter-observer correlation remains high, even over a huge spectrum of inter-races, is impressive testimony to something fairly deep-seated in human psychology. If it holds up cross-culturally, it will be reminiscent of the anthropologist's finding about perception of hue. Physicists tell us that the rainbow from red through orange, yellow, green, and blue to violet is a simple continuum of wavelength. It is biology and/or psychology, not physics, that singles out particular landmark wavelengths along the physical spectrum for special treatment and naming. Blue has a name, green has a name, blue-green does not. The interesting finding of anthropologists' experiments, as opposed to some influential anthropological theories, by the way, is that there is substantial agreement over such namings across different cultures. We seem to have the same kind of agreement over judgments of race. It may prove to be even stronger and clearer than for the rainbow. As I said, zoologists define a species as a group whose members breed with each other under natural conditions in the wild. It doesn't count if they breed only in zoos, or if we have to use artificial insemination, or if we fool female grasshoppers with cage-singing males, even if the offspring produced are fertile. We might dispute whether this is the only sensible definition of a species, but it is the definition that most biologists use.
If we wish to apply this definition to humans, however, there is a peculiar difficulty. How do we distinguish natural from artificial conditions for interbreeding? It's not an easy question to answer. Today, all surviving humans are firmly placed in the same species, and they do indeed happily interbreed. But the criterion, remember, is whether they choose to do so under natural conditions. What are natural conditions for humans? Do they even exist anymore? If, in ancestral times, as sometimes today, two neighboring tribes had different religions, different languages, different dietary customs, different cultural traditions, and were continually at war with one another, if the members of each tribe were brought up to believe that the other tribe were subhuman animals, as happens even today, if their religions taught that would-be sexual partners from the other tribe were taboo, shiksas, or unclean, there could well be no interbreeding between them. Yet anatomically and genetically, they could be completely the same as each other. And it would take only a change of religious or other customs to break down the barriers to interbreeding. How then might somebody try to apply the interbreeding criterion to humans? If Cortypus bruneus and C. bigutulus are separated as two distinct species of grasshoppers because they prefer not to interbreed although they physically could, might humans, at least in ancient times of tribal exclusivity, once have been separable in the same kind of way? Cortypus bruneus and C. bigutulus, remember, in all detectable respects except their song, are identical, and when they are easily persuaded to hybridize, their offspring are fully fertile. Whatever we may think as observers of superficial appearances, the human species today is, to a geneticist, especially uniform. Taking such genetic variation as the human population does possess, we can measure the fraction that is associated with the regional groupings that we call races. And it turns out to be a small percentage of the total, between 6 and 15%, depending on how you measure it, much smaller than many other species where races have been distinguished. Geneticists conclude, therefore, that race is not a very important aspect of a person. There are other ways to say this. If all humans were wiped out except for one local race, the great majority of the genetic variation in the human species would be preserved. This is not intuitively obvious and may be quite surprising to some people. If racial statements were as informative as most Victorians, for example, used to think, you would surely need to preserve a good spread of all the different races in order to preserve most of the variation in the human species. Yet this is not the case. We can explain this human uniformity by guessing that our ancestors passed through a genetic bottleneck not very long ago. the population was reduced to a small number, came close to going extinct, but just pulled through. Like the children of Noah in the myth, we are all descended from this small population, and that is why we are so genetically uniform. Similar evidence of even greater genetic uniformity suggests that cheetahs passed through an even narrower bottleneck more recently, around the end of the last ice age. Some people may find the evidence of biochemical genetics unsatisfying because it seems not to square with their everyday experience. Unlike cheetahs, we don't look uniform. Norwegians, Japanese, and Zulus really do look rather dramatically different from one another. With the best will in the world, it is intuitively hard to believe what is in fact the truth, that they are really more alike than three chimpanzees who look to our eyes much more similar.
This is, of course, a politically sensitive matter, a point I heard being amusingly lampooned by a West African medical researcher at a gathering of about 20 scientists. At the beginning of the conference, the chairman asked each of us around the table to introduce ourselves. The African, who was the only black person there, and he really was black, unlike many African Americans, happened to be wearing a red tie. He finished his self-introduction by laughingly saying, "You can easily remember me. I am the one with the red tie." He was genially mocking the way people bend over backwards to pretend not to notice racial differences. I think there was a Monty Python sketch along the same lines. Nevertheless, we can't write off the genetic evidence which suggests that, all appearances to the contrary, we really are an unusually uniform species. What is the resolution to the apparent conflict between appearance and measured reality? It is genuinely true that if you measure the total variation in the human species and then partition it into a between-race component and a within-race component, the between-race component is a small fraction of the total. Most of the variation among humans can be found within races as well as between them. Only a small admixture of extra variation distinguishes races from each other. That is all correct. What is not correct is the inference that race is therefore a meaningless concept. This point has been clearly made by the distinguished Cambridge geneticist A.W.F. Edwards in a recent paper called Human Genetic Diversity, Lewontin's Fallacy. R.C. Lewontin is an equally distinguished Cambridge, Massachusetts geneticist known for the strength of his political convictions and his weakness for dragging them into science at every possible opportunity. Lewontin's view of race has become near universal orthodoxy in scientific circles. He wrote, in a famous paper of 1972,
"It is clear that our perception of relatively large differences between human races and subgroups, as compared to the variation within these groups, is indeed a biased perception, and that, based on randomly chosen genetic differences, human races and populations are remarkably similar to each other, with the largest part by far of human variation being accounted for by the differences between individuals."
This is, of course, exactly the point I accepted above, not surprisingly, since what I wrote was largely based on Lewontin. But see how Lewontin goes on.
Human racial classification is of no social value and is positively destructive of social and human relations. Since such racial classification is now seen to be of virtually no genetic or taxonomic significance either, no justification can be offered for its continuance.
We can all happily agree that human racial classification is of no social value and is positively destructive of social and human relations. That is one reason why I object to ticking boxes in forms and why I object to positive discrimination in job selection. But that doesn't mean that race is of virtually no genetic or taxonomic significance. This is Edwards's point, and he reasons as follows. However small the racial partition of the total variation may be, if such racial characteristics as there are highly correlated with other racial characteristics, they are by definition informative, and therefore of taxonomic significance. Informative means something quite precise. An informative statement is one that tells you something you didn't know before. The information content of a statement is measured as reduction in prior uncertainty. Reduction in prior uncertainty, in turn, is measured as a change in probabilities. This provides a way to make the information content of a message mathematically precise, but we don't need to bother with that.
If I tell you Evelyn is male, you immediately know a whole lot of things about him. Your prior uncertainty about the shape of his genitals is reduced, though not obliterated. You now know facts you didn't know before about his chromosomes, his hormones, and other aspects of his biochemistry, and there is a quantitative reduction in your prior uncertainty about the depth of his voice and the distribution of his facial hair and of his body fat and musculature. Contrary to Victorian prejudices, your prior uncertainty about Evelyn's general intelligence or ability to learn remains unchanged by the news about his sex. Your prior uncertainty about his ability to lift weights or excel at most sports is quantitatively reduced, but only quantitatively. Plenty of females can beat plenty of males at any sport, although the best males can normally beat the best females. Your ability to bet on Evelyn's running speed, say, or the power of his tennis serve, has been slightly raised by my telling you his sex, but it has not reached certainty. Now to the question of race. What if I tell you Susie is Chinese? How much is your prior uncertainty reduced? You now are pretty certain that her hair is straight and black, or was black, that her eyes have an epicanthic fold, and one or two other things about her. If I tell you Colin is black, this does not, as we have seen, tell you he is black. Nevertheless, it's clearly not uninformative. The high inter-observer correlation suggests that there is a constellation of characteristics that most people recognize, such that the statement, Colin is black, really does reduce prior uncertainty about Colin. It works the other way around to some extent. If I tell you Carl is an Olympic sprinting champion, your prior uncertainty about his race is, as a matter of statistical fact, reduced. Indeed, you can have a fairly confident bet that he is black.
We got into this discussion through wondering whether the concept of race was, or ever had been, an information-rich way to classify people. How might we apply the criterion of inter-observer correlation to judging the question? Well, suppose we took standard full-face photographs of 20 randomly chosen natives of each of the following countries: Japan, Uganda, Iceland, Sri Lanka, Papua New Guinea, and Egypt. If we presented all 120 people with all 120 photographs, my guess is that every single one of them would achieve 100% success rates in sorting them into 6 different categories. What is more, if we told them the names of the 6 countries involved, all 120 subjects would correctly assign all 120 photographs to the correct countries. I haven't done the experiment, but I'm confident that you will agree with me on what the result would be. It may seem unscientific of me not to bother to do the experiment, but my confidence that you, being human, will agree without doing the experiment is the very point I'm trying to make. If the experiment were to be done, I do not think Lewontin would expect any other result than the one I have predicted. Yet an opposite prediction would seem to follow from his statement that racial classification has virtually no taxonomic or genetic significance. If there is no taxonomic or genetic significance, the only other way to get a high inter-observer correlation would be a worldwide similarity in cultural bias, and I do not think Lewontin would want to predict that either. In short, I think Edwards is right, and Lewontin, not for the first time, wrong. Lewontin did his sums right, of course. He's a brilliant mathematical geneticist. The proportion of the total variation in the human species that falls into the racial partition of variation is indeed low. But because the between-race variation, however low a percentage of the total variation, is correlated, it is informative in ways that could surely be demonstrated by measuring the inter-observer concordance of judgment. I must at this point reiterate my strong objection to being asked to fill in forms in which I have to tick a box labeling my race or ethnicity, and voice my strong support for Lewontin's statement that racial classification can be actively destructive of social and human relations, especially when people use racial classification as a way of treating people differently, whether through negative or positive discrimination. To tie a racial label to somebody is informative, in the sense that it tells you more than one thing about them. It might reduce your uncertainty about the color of their hair, the color of their skin, the straightness of their hair, the shape of their eye, the shape of their nose, and how tall they are. but there is no reason to suppose that it tells you anything about how well qualified they are for a job. And even in the unlikely event that it did reduce your statistical uncertainty about their likely suitability for some particular job, it would still be wicked to use racial labels as a basis for discrimination when hiring somebody. Choose on the basis of ability, and if, having done so, you end up with an all-black sprinting team, so be it. You have not practiced racial discrimination in arriving at this conclusion.
A great conductor, when auditioning instrumentalists for his orchestra, always had them perform behind a screen. They were told not to speak, and they even had to remove their shoes for fear the sound of high heels would betray the sex of the performer. Even if it was statistically the case that women tend to make better harpists, say, than men, this does not mean that you should actively discriminate against men when you choose a harpist.
Discriminating against individuals purely on the basis of a group to which they belong is, I'm inclined to think, always evil. There is near universal agreement today that the apartheid laws of South Africa were evil. Positive discrimination in favor of minority students on American campuses can fairly, in my opinion, be attacked on the same grounds as we used to attack apartheid. Both treat people as representative of groups rather than as individuals in their own right. Positive discrimination is sometimes justified as redressing centuries of injustice. But how can it be just to pay back a single individual today for the wrongs done by long-dead members of a plural group to which he belongs?
Interestingly, this kind of singular-plural confusion shows up in a form of words which is tellingly diagnostic of bigots. The Jew instead of Jews.
Your fuzzy-wuzzy is an excellent fighter, but he can't tell his left from his right. Now your Pathan...
People are individuals. They are individually different, far more different from other members of their group than their groups are from each other. In this, Lewontin is undoubtedly right. Inter-observer agreement suggests that racial classification is not totally uninformative, but what does it inform about? About no more than the characteristics used by the observers when they agree, things like eye shape and hair curliness. Nothing more, unless we are given further reasons to believe it. For some reason it seems to be the superficial, external, trivial characteristics that are correlated with race, perhaps especially facial characteristics. But why are human races so different in just these superficially conspicuous characteristics? Or is it just that we, as observers, are predisposed to notice them? Why do other species look comparatively uniform, whereas humans show differences that, if we encountered them elsewhere in the animal kingdom, might make us suspect we were dealing with a number of separate species?
The most politically acceptable explanation is that the members of any species have a heightened sensitivity to differences among their own kind. On this view, it is just that we notice human differences more readily than differences within other species. Chimpanzees, whom we find almost identical, look just as different in chimpanzee eyes as a Kikuyu is different from a Dutchman in our eyes. Expecting to confirm this kind of theory at the within-race level, the eminent American psychologist H.L. Teuber, an expert on the brain mechanisms of facial recognition, asked a Chinese graduate student to study the question, why do Westerners think Chinese people look more alike than Westerners? After 3 years' intensive research, the Chinese student reported his conclusion. Chinese people really do look more alike than Westerners. Teuber told the story with much twinkling and wiggling of eyebrows, a sure sign with him that a joke was on the way, so I don't know what the truth of the matter is, but I have no difficulty in believing it, and I certainly don't think it should upset anyone. Our relatively recent worldwide diaspora out of Africa has taken us to an extraordinarily wide variety of habitats, climates, and ways of life. It is plausible that the different conditions have exerted strong selection pressures, particularly on externally visible parts, such as the skin, which bear the brunt of the sun and the cold. It is hard to think of any other species that thrives so well from the tropics to the Arctic, from sea level to the high Andes, from parched deserts to dripping jungles, and through everything in between. Such different conditions would be bound to exert different natural selection pressures, and it would be positively surprising if local populations did not diverge as a result. Hunters in the deep forests of Africa, South America, and Southeast Asia have all independently become small, almost certainly because height is a handicap in dense vegetation. Peoples of high latitude, who, it has been surmised, need all the sun they can get to make vitamin D, tend to have lighter skins than those who face the opposite problem, the carcinogenic rays of the tropical sun. It is plausible that such regional selection would especially affect superficial characteristics like skin color, while leaving most of the genome intact and uniform. In theory, that could be the full explanation for our superficial and visible variety, covering deep similarity. But it doesn't seem enough to me. At the very least, I think it might be helped along by an additional suggestion, which I offer tentatively. It takes off from our earlier discussion about cultural barriers to interbreeding. We are indeed a very uniform species if you count the totality of genes, or if you take a truly random sample of genes. But perhaps there are special reasons for a disproportionate amount of variation in those very genes that make it easy for us to notice variation and to distinguish our own kind from others. This would include the genes responsible for externally visible labels like skin color. I want to suggest that this heightened discriminability has evolved by sexual selection, specifically in humans, because we are such a culture-bound species. Because our mating decisions are so heavily influenced by cultural tradition, and because our cultures, and sometimes our religions, encourage us to discriminate against outsiders, especially in choosing mates, those superficial differences that helped our ancestors to prefer insiders over outsiders have been enhanced out of all proportion to the real genetic differences between us. No less a thinker than Jared Diamond has supported a similar idea in The Rise and Fall of the Third Chimpanzee, and Darwin himself more generally invoked sexual selection in explanation of racial differences. I want to consider two versions of this theory, a strong and a weak one. The truth could be any combination of the two. The strong theory suggests that skin color and other conspicuous genetic badges evolved actively as discriminators in choosing mates. The weak theory, which can be thought of as leading into the strong version, places cultural differences such as language and religion in the same role as geographical separation in the incipient stages of speciation. Once cultural differences have achieved this initial separation, with the consequence that there is no gene flow to hold them together, the groups would subsequently evolve apart genetically, as if geographically separated. The general rule in evolution is that an ancestral population can split into two genetically distinct populations only if given a head start by an initial accidental separation, usually assumed to be geographical. A barrier such as a mountain range reduces gene flow between two populated valleys. So the gene pools in the two valleys are free to drift apart. The separation will normally be abetted by different selection pressures. One valley may be wetter than its neighbor on the other side of the mountains, for instance. But the initial accidental separation, which I have so far assumed to be geographical, is necessary. There is controversy here. Some people think the initial separation has to be geographical, while others, especially entomologists, emphasize so-called sympatric speciation. Many herbivorous insects eat only one species of plant. They meet their mates and lay their eggs on the preferred plants. Their larvae then apparently imprint on the plant that they grow up eating, and they choose, when adult, the same species of plant to lay their own eggs. So, if an adult female made a mistake and laid her eggs on the wrong species of plant, her daughter would imprint on that wrong plant and would, when the time came, lay her eggs on plants of the same wrong species. Her larvae would then imprint on the same wrong plant, hang around the wrong plant when adult, mate with others hanging around the wrong plant, and eventually lay their eggs on the wrong plant. In the case of these insects, you can see that in a single generation, gene flow with the parental type could be abruptly cut off. A new species is theoretically free to come into being without the need for geographical isolation. Or, another way of putting it, the difference between two kinds of food plant is, for these insects, equivalent to a mountain range or a river for other animals. It has been argued that this kind of sympatric speciation is commoner among insects than true geographical speciation, in which case since the majority of species are insects, it could even be that most speciation events are sympatric. Be that as it may, I am suggesting that human culture provides a special way in which gene flow can find itself blocked, which is somewhat analogous to the insect scenario I have just outlined. In the insect case, plant preferences are handed down from parent to offspring by the twin circumstances of larvae fixating on their food plant and adults mating and laying eggs on the same food plants. In effect, lineages establish traditions that travel longitudinally down generations. Human traditions are similar, if more elaborate. Examples are languages, religions, and social manners or conventions. Children usually adopt the language and the religion of their parents, although, just as with the insects and the food plants, there are enough mistakes to make life interesting. Again, as with the insects mating in the vicinity of their preferred food plants, people tend to mate with others speaking the same language and praying to the same gods. So, different languages and religions can play the role of food plants or of mountain ranges in traditional geographical speciation. Different languages, religions, and social customs can serve as barriers to gene flow. From here, according to the weak form of our theory, random genetic differences simply accumulate on opposite sides of a language or religion barrier, just as they might on opposite sides of a mountain range. Subsequently, according to the strong version of the theory, the genetic differences that build up are reinforced as people use conspicuous differences in appearance as additional labels of discrimination in mate choice, supplementing the cultural barriers that provided the original separation. I am certainly not suggesting that humans should be thought of as more than one species, very much the contrary. What I am suggesting is that human culture, the fact that we depart so strongly from random mating in directions determined by language, religion, and other cultural discriminators, has done very odd things to our genetics in the past.
Even though, if you take the totality of genes into account, we are a very uniform species, we're astonishingly variable in superficial features which are trivial but conspicuous. Discrimination fodder. The discrimination might apply not just to mate choice, but to choice of enemies and victims of xenophobic or religious prejudice.
The Rotifer's Tale. The brilliant theoretical physicist Richard Feynman is rumored to have said, "If you think you understand quantum theory, you don't understand quantum theory." I'm tempted by an evolutionist's equivalent. If you think you understand sex, you don't understand sex. The three modern Darwinians, from whom I believe we have the most to learn, John Maynard Smith, W.D. Hamilton, and George C. Williams, all devoted substantial parts of their long careers to wrestling with sex. Williams began his 1975 book, Sex and Evolution, with a challenge to himself.
This book is written from a conviction that the prevalence of sexual reproduction in higher plants and animals is inconsistent with current evolutionary theory. There is a kind of crisis at hand in evolutionary biology.
Maynard Smith and Hamilton said similar things. It is to resolve this crisis that all three Darwinian heroes, along with others of the rising generation, labored. I shall not attempt an account of their efforts, and certainly I have no rival solution to offer myself. Instead, the Rotifer's Tale displays an underexplored consequence of sexual reproduction for our view of evolution. Deloidea is a large class of the phylum Rotifera. The existence of the Deloid rotifers is an evolutionary scandal. Not my own bon mot, it rings with the unmistakable tones of John Maynard Smith. Many rotifers reproduce without sex. In this respect, they resemble aphids, stick insects, various beetles, and a few lizards, and are not particularly scandalous. What stuck in Maynard Smith's craw is that the Deloids as a whole reproduce only asexually, every last one of them, evidently descended from an asexual common ancestor that must have lived long enough ago to beget 18 genera and 360 species. Remains in amber suggest that this male-spurning matriarch lived at least 40 million years ago, very probably more. The Deloids are a highly successful group of animals, astonishingly numerous, and a dominant part of the freshwater faunas of the world. Not a single male has ever been found.
What is so scandalous about that? Well, suppose we take a family tree of the whole animal kingdom. The tips of the twigs all round the surface of the tree represent species. Major boughs represent classes or phyla. There are millions of species, which means that the evolutionary tree is far more intricately branched than any woody tree you will ever see. There are only a few tens of phyla, and not that many more classes. The phylum Rotifera is one branch of the tree, and it splits into 4 sub-branches, of which the class Deloidea is one. This class branch subdivides, and subdivides further, eventually to yield 360 twigs, each representing one species. The same kind of thing is going on for all the other phyla, each with its own classes, and so on. The outer twigs of the tree represent the present. Slightly inward from the outermost shell of twigs represents a short distance into the past, and so on to the main trunk, which represents, say, 1 billion years ago. That understood, we now take paint to our grey, wintry tree and colour the tips of certain twigs to label particular features. We might paint red all twigs that represent flying animals. If we now step back and contemplate the whole tree, we shall notice large areas of red separated by even larger areas of grey, representing whole major groups of animals that don't fly. Most of the insect twigs, the bird twigs and the bat twigs, are red, and they are neighbours of other red twigs. None of the others are. With a few exceptions, like fleas and ostriches, 3 entire classes are made up of flying animals. Redness is distributed in broad patches of uniform red against uniform grey. Think what this means for evolution. The 3 patches of red must have begun long ago with 3 ancestral animals, an early insect, an early bird, and an early bat, discovering how to fly. Flying obviously turned out to be a really good idea once it had been discovered, because it persisted and spread through all the descendant branches as the 3 species eventually gave rise to 3 large collections of descendant species, almost all of them persisting with their ancestors' ability to fly, the class Insecta, the class Aves, and the order Chiroptera. But now we do the same thing, not for flying, but for reproducing asexually without males. On our tree of life, we paint the twigs of all asexually reproducing species blue. Now we notice a completely different pattern. Where flying showed up as great swathes of red, asexual reproduction shows up as tiny sporadic dots of blue. An asexual beetle species shows up as a blue twig completely surrounded by grey. Maybe a group of 3 species in one genus are blue, but neighbouring genera are grey. You see what this means? Asexual reproduction arises from time to time, but it rapidly goes extinct before it has had time to grow into a stout branch with many blue twigs. Unlike flying, the habit of reproducing asexually doesn't persist long enough to give rise to a whole family, order, or class of asexual creatures.
With one scandalous exception. Unlike all the other little blips of blue, the Deloid rotifers are a hefty patch of uninterrupted blue, enough to make the proverbial sailor's trousers. In evolution, what this seems to mean is that an ancestral Deloid discovered asexual reproduction, just like the odd beetle we talked about. But whereas asexual beetles and hundreds of other asexual species dotted around the tree go extinct long before they can evolve into a larger grouping, such as a family or order, let alone a class, the Deloids seem to have stuck with asexuality and flourished with it through enough evolutionary time to generate an entire asexual class, now numbering 360 species. For the Deloid rotifers, but not convincingly for any other kind of animal, asexual reproduction is like flying. It seems to be a good and successful innovation for the Deloids, while in all other parts of the tree it constitutes a fast track to extinction.
The statement that there are 360 species raises a problem. The biological definition of a species is a group of individuals that interbreed with each other and not with others. The Deloids, being asexual, don't interbreed with anybody. Every one is an isolated female, each of whose descendants goes her own sweet way in genetic isolation from every other individual. So when we say there are 360 species, we can only mean there are 360 types, which we humans recognize as looking sufficiently different from each other that we would expect, if they bred sexually at all, to see them shunning the other types as sexual partners. Not everyone accepted that the Deloid rotifers really were asexual. There is a large gap in logic between the negative statement that males have never been seen and a positive conclusion that there aren't any. As Olivia Judson recounts in her winningly sophisticated zoological comedy, Dr. Tatiana's Sex Guide to All Creation, naturalists have been caught like that before. Asexual species often turn out to have concealed males. The males of certain anglerfish are tiny dwarfs who ride about as parasites on the bodies of females. Had they been even smaller, we might have overlooked them altogether, as nearly happened in the case of certain scale insects, where the males are, in the words of Lawrence Hirst, "wee things that stick to the female's legs." Hirst went on to quote his mentor Bill Hamilton's astute remark,
"How often do you see humans having sex? If you were a Martian looking around, you'd be pretty sure we were asexual."
So it would be nice to come up with some more positive evidence that the Deloid rotifers really are anciently asexual. Geneticists are becoming increasingly ingenious in the art of reading the patterns of gene distribution in modern animals and making inferences about their evolutionary history by picking up signals in the genes of living animals. The logic is not deductive. We don't deduce from modern genes that the course of history must have been so-and-so. Instead, we say, if the course of history was so-and-so, we should expect to see such and such a pattern of gene distribution today. That is what David Mark Welch and Matthew Mieselson of Harvard University did with rotifers. They reasoned that if the Deloids had been purely asexual for many millions of years, we should expect the genes of living Deloids to show a certain pattern. Omitting the details here, Mark Welch and Mieselson found exactly the pattern predicted and concluded that the Deloid rotifers really are anciently, continuously, universally, and successfully asexual. They really are an evolutionary scandal. For perhaps 80 million years they have flourished by doing something that no other group of animals can get away with, except for very short periods, before going extinct.
Why would we normally expect asexual reproduction to lead to extinction? Well, that is a big question, because it amounts to the question of what is good about sex itself, the question that better scientists than I have spent book after book failing to answer. I shall just point out that the Deloid rotifers are a paradox within a paradox. In one way, they're like the soldier in the marching platoon whose mother cried out, "There goes my boy, he's the only one in step." Maynard Smith called them an evolutionary scandal, but he was the one mainly responsible for pointing out that sex itself, on the face of it, is the evolutionary scandal. At least a naive view of Darwinian theory would predict that sex should be heavily disfavored by natural selection, outcompeted twofold by asexual reproduction. In that sense, the Deloidia, far from being a scandal, appear to be the only soldier in step. Here's why. The problem is the one Maynard Smith dubbed the twofold cost of sex. Darwinism, in its modern form, expects that individuals will strive to pass on as many of their genes as possible. So isn't it just daft to throw away half your genes with every egg or sperm you make, in order to mix the other half with the genes of somebody else? Wouldn't a mutant female who behaves like a Deloid rotifer and passes on 100% of her genes to every offspring instead of 50% do twice as well?
Maynard Smith added that the reasoning breaks down if the male partner works hard or contributes economic goods in such a way that a couple can rear twice as many offspring as an asexual loner. In that case, the twofold cost of sex is cancelled out by a doubling in the number of offspring. In a species such as an emperor penguin, with male and female parent contributing approximately equally towards the labour and other costs of child rearing, the twofold cost of sex is abolished or at least mitigated. In species where economic and labour contributions are unequal, it is nearly always the father who shirks, devoting his energies instead to duffing up other males, This magnifies the cost of sex up to the full twofold penalty of the original reasoning. This is why Maynard Smith's alternative name, the twofold cost of males, is preferable. In this light, which Maynard Smith himself was largely responsible for shining, it isn't the Deloid rotifers who are the evolutionary scandal, but everything else. More pertinently, the male sex is an evolutionary scandal, except that it does exist, and indeed is almost universal throughout the animal kingdom. What is going on? As Maynard Smith wrote,
one is left with the feeling that some essential feature of the situation is being overlooked.
The twofold cost is the starting point for masses of theorizing by Maynard Smith, Williams, Hamilton, and many younger colleagues. The widespread existence of males who don't earn their keep as fathers must mean that there really are very substantial Darwinian benefits to sexual recombination itself. It is not too difficult to think of what they might be in qualitative terms, and lots of possible benefits, some obvious, some esoteric, have been proposed. The problem is to think of a benefit of sufficient quantitative magnitude to counteract the massive twofold cost.
To do justice to all the theories would take a book. It has already taken several, including the seminal works I've previously mentioned by Williams and Maynard Smith, and Graham Bell's beautifully written tour de force, The Masterpiece of Nature. Yet no definitive verdict has emerged. A nice book aimed at a non-specialist audience is Matt Ridley's The Red Queen. Though primarily favouring one of the theories on offer, W.D. Hamilton's theory that sex serves an unceasing arms race against parasites, Ridley does not neglect to explain the problem itself and the other answers to it. As for me, I shall swiftly recommend Ridley's book and the others before going straight to the main purpose of this tale, which is to draw attention to an underappreciated consequence of the evolutionary invention of sex. Sex brought into existence the gene pool, made meaningful the species, and changed the whole ballgame of evolution itself. Think what evolution must look like to a Deloid rotifer. Think how different the evolutionary history of those 360 species must have been from the normal pattern of evolution. We portray sex as raising diversity, and so in a sense it does. That is the basis of most theories of how sex overcomes its twofold cost. But, paradoxically, it also has a seemingly opposite effect. Sex normally acts as a kind of barrier to evolutionary divergence. In a population of mice, say, any tendency to strike out in some enterprising new evolutionary direction is held in check by the swamping effect of sexual mixing. The genes of the would-be enterprising diverger are swamped into conformity by the inertial mass of the rest of the gene pool. That is why geographical isolation is so important to speciation. It takes a mountain range or a difficult sea crossing to allow a newly striking out lineage to evolve its own way without being dragged back to the inertial norm. Think how different evolution must be for the Deloid rotifers. Far from being swamped into normalcy by the gene pool, they don't even have a gene pool. The very idea of a gene pool has no meaning if there is no sex. Gene pool is a persuasive metaphor because the genes of a sexual population are being continually mixed and diffused, as if in a liquid. Bring in the time dimension, and the pool becomes a river, flowing through geological time, the river out of Eden, as I called it in the title of another book. It is the binding effect of sex that provides the river with its limiting banks, channeling the species into some kind of evolutionary direction. Without sex, there would be no coherently channeled flow, but a shapeless, outward diffusion, less like a river than a smell wafting out in all directions from some point of origin. Natural selection presumably takes place among the Deloids, but it must be a very different kind of natural selection from the one the rest of the animal kingdom is accustomed to. Where there is sexual mixing of genes, the entity that is carved into shape by natural selection is the gene pool. Good genes tend statistically to help the individual bodies in which they find themselves to survive. Bad genes tend to make them die. In sexually reproducing animals, it is the deaths and reproductions of individual animals that constitute the immediate selective events, but the long-term consequence is a change in the statistical profile of genes in the gene pool. So it is the gene pool, as I say, that is the object of the Darwinian sculptor's attention. Moreover, genes are favored for their capacity to cooperate with other genes in building bodies. That is why bodies are such harmonious engines of survival. The right way to look at this, given sex, is that genes are continually being tried out against different genetic backgrounds. In every generation, a gene is shuffled into a new team of companions, meaning the other genes with which it shares a body on any particular occasion. Genes that are habitually good companions, fitting in well with others and cooperating well with them, tend to be in winning teams, meaning successful individual bodies that pass them on to offspring. Genes that are not good cooperators tend to make the teams in which they find themselves become losing teams, meaning unsuccessful bodies that die before reproducing. The proximal set of genes with which a gene has to cooperate are the ones with which it shares a body, this body. But in the long term, the set of genes with which it has to cooperate are all the genes of the gene pool, for they are the ones that it repeatedly encounters as it hops from body to body down the generations. This is why I say it is the gene pool of a species that is the entity sculpted into shape by the chisels of natural selection. Proximally, natural selection is the differential survival and reproduction of whole individuals, the individuals that the gene pool throws up as samples of what it can do. Once again, none of this could be said of the Deloid rotifers. Nothing like the sculpting of the gene pool goes on, for there is no gene pool to sculpt. A Deloid rotifer has just one big gene. The message of the rotifer's tale is a consequence of sex, not a theory for the benefit of sex, nor a theory of why sex arose in the first place. But if I ever were to attempt a theory for the benefit of sex, if I were ever to essay a serious assault on the essential feature of the situation that is being overlooked, it is hereabouts that I would start.
And we should listen again and again to the rotifer's tale. These tiny, obscure denizens of puddles and mossy moisture may hold the key to the outstanding paradox of evolution. What's wrong with asexual reproduction if the Deloid rotifers have run with it for so long? Or if it's right for them, why don't the rest of us do it and save the massive twofold cost of sex?
The Barnacle's Tale. When I was at boarding school, it was occasionally necessary to apologize to the housemaster for being late for dinner.
Sorry I'm late, sir. Orchestra practice.
Or whatever the excuse might be. On those occasions when there really was no good excuse and we had something to hide, we formed the habit of murmuring,
Sorry I'm late, sir. Barnacles.
He always nodded kindly, and I don't know whether he ever wondered what this mysterious out-of-school activity might be. It is possible that we were inspired by the example of Darwin, who devoted years of his life to barnacles so single-mindedly that his children were moved to ask, in innocent puzzlement, after being shown round the house of some friends,
But then where does your father do his barnacles?
I'm not sure that we knew the Darwin story then, and I suspect that we invented the excuse because there is something about barnacles that seems too implausible to be a bluff. Barnacles are not what they seem. That applies to other animals too, and it is the theme of the Barnacle's Tale.
Contrary to all appearances, barnacles are crustaceans. The ordinary acorn barnacles, which encrust the rocks like miniature limpets, helping your shoes not to slip if you have them, and hurting your feet if you don't, are completely unlike limpets internally. Inside the shell, they are distorted shrimps, lying on their backs, kicking their legs in the air. Their feet bear feathery combs or baskets with which they filter particles of food out of the water. Goose barnacles do the same thing, but instead of sheltering under a conical shell like an acorn barnacle, they sit on the end of a stout stalk. They get their name from yet another misunderstanding of the true nature of barnacles. Their wet filtering feathers give them the appearance of a baby bird in its egg. In the days when people believed in spontaneous generation, a folk belief grew that goose barnacles hatched into geese, specifically Brantoleucopsis, the barnacle goose. Most deceptive of all, indeed probably holding the record for animals not looking remotely like the thing that zoologists know them to be, are the parasitic barnacles, such as Saculina. Saculina is not what it seems with a vengeance. Zoologists would never have realized that it is in fact a barnacle, but for its larva. The adult is a soft sac that clings to the underside of a crab and sends long, branching, plant-like roots inside to absorb nourishment from the crab's tissues. The parasite not only doesn't look like a barnacle, it doesn't look like a crustacean of any kind. It has completely lost all trace of the armor plating and all trace of the bodily segmentation that nearly all other arthropods have. It might as well be a parasitic plant or fungus. Yet in terms of its evolutionary relationships, it is a crustacean, and not just a crustacean, but specifically a barnacle. Barnacles are indeed not what they seem. By the way, Saculina's branching root system is not indiscriminate in its invasion of the crab's tissues. It heads first for the crab's reproductive organs, which has the effect of castrating the crab. Is this just an accidental byproduct? Probably not. Castration not only sterilizes the crab. Like a fat bullock, the castrated crab, instead of concentrating on becoming a lean, mean, reproducing machine, diverts resources towards getting larger. More food for the parasite. To lead into the final tale of this cluster, here's a little fable set in the future. 500 million years after vertebrate and arthropod life completely perished in the mother of all comet collisions, intelligent life has eventually re-evolved in remote descendants of octopuses. Octopoid paleontologists come upon a rich fossil bed dating from the 21st century AD. Not a fair cross-section of contemporary life, this bounteous shale nevertheless impresses the paleontologists with its variety and diversity. Carefully weighing the fossils up with 8-arm balanced judgment and expertly sucking the details, one octopodon scholar goes so far as to suggest that life, during this pre-catastrophe dawn age, was more extravagantly profligate in its diversity than it ever would be again, throwing up weird and wonderful new body plans in gleeful experimentation. You can see what he means by thinking of your own animal contemporaries and imagining that a tiny sampling of them fossilizes. Think of the Herculean task facing our future paleontologist and empathize with his difficulties in trying to discern their affinities from imperfect and sporadic fossil traces. The moral is that of the barnacle again. But the parable of the paleontologist of the future and his seduction by the rhetoric of weird wonders gleefully carousing in morphospace was not idly spun. It was intended as a softening up for the next tale, which is all about the Cambrian explosion.
The Velvet Worm's Tale. If modern zoology admits of anything approaching a full-blown origin myth, it is the Cambrian explosion. The Cambrian is the first period of the Phanerozoic Eon, the last 545 million years, during which animal and plant life as we know it suddenly became manifest in fossils. Before the Cambrian, fossils were either tiny traces or enigmatic mysteries. From the Cambrian onwards, there has been a clamorous menagerie of multicellular life more or less plausibly presaging our own. It is the suddenness with which multicellular fossils appear at the base of the Cambrian that prompts the metaphor of explosion. Creationists love the Cambrian explosion because it seems, to their carefully impoverished imaginations, to conjure a sort of paleontological orphanage inhabited by parentless phyla, animals without antecedents, as if they had suddenly materialized overnight from nothing, complete with holes in their socks, to borrow Bertrand Russell's image from a different context. At the other extreme, romantically overheated zoologists love the Cambrian explosion for its aura of Arcadian dreamtime, a zoological age of innocence in which life danced to a frenzied and radically different evolutionary tempo, a prelapsarian bacchanalia of leaping improvisation, before a fall into the earnest utilitarianism that has prevailed since. In Unweaving the Rainbow, I quoted the following words of a distinguished biologist who may now have thought better of it.
Soon after multicelled forms were invented, a grand burst of evolutionary novelty thrust itself outward. One almost gets the sense of multicellular life gleefully trying out all its possible ramifications in a kind of wild dance of heedless exploration.
If there is one animal more than any other that stands for this feverish vision of the Cambrian, it is hallucigenia. Stands? It seems that hallucigenia, and Simon Conway Morris chose its name advisedly, was originally reconstructed upside down. It is now tentatively placed in the phylum Lobopodia, which has modern representatives in the form of Peripatus and the other onychophorans or velvet worms. Hallucigenia's connection with the modern onychophora is still controversial, and we must remember what a lot of imagination necessarily intervenes between a blurred and squashed fossil in a rock and the reconstruction that is eventually drawn, often in daring colour, on the page. It has even been suggested that hallucigenia might not be a whole animal at all, but a part of some unknown animal. It would not be the first time such a mistake had been made. Some early artists' reconstructions of Cambrian scenes included a swimming jellyfish-like creature, seemingly inspired by tin pineapple rings, which turned out to be part of the jaw apparatus of the mysteriously predatory animal Anomalocaris. Other Cambrian fossils, for example Aschia, certainly seem quite like marine versions of Peripatus, and this reinforces Peripatus's entitlement to tell this Cambrian tale. Most fossils, in any era, are the remains of hard parts of animals, vertebrate bones, the carapaces of arthropods, or the shells of mollusks or brachiopods. But there are 3 Cambrian fossil beds, one in Canada, one in Greenland, and one in China, where freak conditions, with almost miraculous good fortune for us, preserved soft parts as well. These are the Burgess Shale of British Columbia, Sirius Passet of northern Greenland, and the Chengjiang site of southern China. Chengjiang has fossils that appear to be true vertebrates, vastly earlier than traditional zoological knowledge expected. Milocunmingia, of which more than 500 specimens have now been discovered at Chengjiang, looks pretty much like a good jawless fish, such as previously had been thought not to arise until 50 million years later in the middle of the Ordovician. The pushing of the vertebrates back into the middle of the Cambrian only strengthens the idea of sudden explosion that is the basis of the myth. It really does appear that most of today's major animal phyla first appear as fossils in a narrow span within the Cambrian. This doesn't mean that there were no representatives of those phyla before the Cambrian, but they have mostly not fossilized. How should we interpret this? We can distinguish various combinations of 3 hypotheses. First, no real explosion. On this view, there was only an explosion of fossilizability, not of actual evolution. The phyla actually go back a long way before the Cambrian, with concestors spread out through hundreds of millions of years in the pre-Cambrian. This view is supported by some molecular biologists who have used molecular clock techniques to date key concestors. Molecular clock estimates have in general tended to push deep branchings between major phyla way back into the pre-Cambrian, far further than most paleontologists are happy with. On this view, fossils were, for unknown reasons, not readily formed before the Cambrian. Perhaps they lacked readily fossilizable hard parts, such as shells, carapaces, and bones. After all, the Burgess Shale and the Chengjiang beds are extremely unusual among all geological layers in recording soft parts as fossils. Perhaps Precambrian animals, although long existing in a wide range of complex body plans, were simply too small to fossilize. In favor of this idea, there are some small animal phyla that have left no fossils at all after the Cambrian, until they appear today as live orphans. Why then should we feel entitled to expect fossils before the Cambrian? Second, medium fuse explosion. The concestors uniting the various phyla really did live reasonably close to each other in time, but still spread out over several tens of millions of years before the observed explosion of fossils. From the great distance of the present, Chengjiang at 525 million years seems at first sight rather close to a putative concestor at, say, 590 million. But a full 65 million years separates them, which is the same time as has elapsed today since the death of the dinosaurs, the entire time during which modern mammals have radiated and radiated again to produce the spectacularly diverse ranges that we see today. Even 10 million years is a long time in the light of the extremely rapid evolutionary bursts of the Galapagos finch's tail. It is all too easy with hindsight to think that because we recognize two ancient fossils as belonging to different modern phyla, those two fossils must have been as different from each other as modern representatives of the two phyla are. It is too easy to forget that the modern representatives have had 500 million years in which to diverge. There is no good reason to believe that a Cambrian taxonomist, blessedly free of 500 million years' worth of zoological hindsight, would have placed the two fossils in separate phyla. He might have placed them only in separate orders, notwithstanding the then unknowable fact that their descendants were destined eventually to diverge so far as to warrant separate phylum status. Third, overnight explosion. This third school of thought is, in my opinion, bonkers, or to use more parliamentary language, wildly and irresponsibly unrealistic. But I must spend some time on it because it has recently become unaccountably popular, following the rhetoric I attributed to romantically overheated zoologists. This third school believes that new phyla sprang into existence overnight in a single macromutational leap. Here are some quotations I've used before in Unweaving the Rainbow from otherwise reputable scientists.
It was as if the facility for making evolutionary leaps that produced major functional novelties, the basis of new phyla, had somehow been lost when the Cambrian period came to an end. It was as if the mainspring of evolution had lost some of its power. Hence, evolution in Cambrian organisms could take bigger leaps, including phylum-level leaps, while later on it would be more constrained, making only modest jumps up to the class level.
Or this from the same distinguished scientist from whom we heard at the beginning of the tale.
Early on in the branching process, we find a variety of long-jump mutations that differ from the stem and from one another quite dramatically. These species have sufficient morphological differences to be categorized as founders of distinct phyla. These founders also branch, but do so via slightly closer long-jump variants, yielding branches from each founder of a phylum to dissimilar daughter species, the founders of classes. As the process continues, fitter variants are found in progressively more nearby neighborhoods, so founders of orders, families, and genera emerge in succession.
Those preposterous quotations move me to retort that it is as though a gardener looked at an old oak tree and remarked wonderingly,
"Isn't it strange that no major new boughs have appeared on this tree for many years? These days, all the new growth appears to be at the twig level."
Here's another quotation, which this time I will attribute because it was published after Unweaving the Rainbow, and I have therefore not used it before. Andrew Parker's In the Blink of an Eye is mainly concerned with advocating his interesting and original theory that the Cambrian explosion was triggered by animals' sudden discovery of eyes. But before coming to his theory itself, Parker begins by falling hook, line, and sinker for the wild and irresponsible version of the Cambrian explosion myth. He first expresses the myth itself in the most frankly explosive version I have read.
544 million years ago, there were indeed 3 animal phyla with their variety of external forms, but at 538 million years ago, there were 38, the same number that exists today.
He goes on to make it clear that he is not talking about extremely rapid gradualistic evolution compressed into a period of 6 million years, which would be an extreme version of our hypothesis too, and just barely acceptable. Nor is he saying, as I would, that near the initial divergence of a pair of what are destined to become phyla, they would not have been very different, would indeed have passed through successive stages of being a pair of species, then genera, and so on until eventually their separation warranted recognition at the phylum level. No, Parker gives every appearance of regarding his 38 phyla at 538 million years as fully fledged phyla that sprang into existence overnight at the drop of a macromutational hat.
38 animal phyla have evolved on Earth, so only 38 monumental genetic events have taken place, resulting in 38 different internal organizations.
Monumental genetic events are not utterly out of the question. Control genes can certainly mutate in dramatic ways. But there's monumental and monumental. A fruit fly with a pair of legs where the antennae should be is about as monumental as it gets, and even then there is a big question mark over survival. There's a powerful general reason for this, which I shall briefly explain. A mutant animal has a certain probability of being better off as a consequence of its new mutation. Better off means better when compared to the pre-mutated parental type. The parent must have been at least good enough to survive and reproduce, otherwise it wouldn't be a parent. It is easy to see that the smaller the mutation, the more likely it is to be an improvement. It is easy to see was a favorite phrase of the great statistician and biologist R.A. Fisher, and he sometimes used it when it was anything but easy for ordinary mortals to see. In this particular case, however, I think it is genuinely easy to follow Fisher's argument for the case of a simple metric feature, something such as thigh length, which varies in one dimension, some number of millimeters that could grow larger or could grow smaller. Imagine a set of mutations of increasing magnitude. At one extreme, a mutation of 0 magnitude is by definition exactly as good as the parent's copy of the gene, which, as we've seen, must have been at least good enough to survive childhood and reproduce. Now imagine a random mutation of small magnitude. The leg, say, gets 1 millimeter longer or 1 millimeter shorter. Assuming that the parental gene is not perfect, a mutation that is infinitesimally different from the parent version has a 50% chance of being better and a 50% chance of being worse. It'll be better if it is a step in the right direction, worse if it is a step in the opposite direction relative to the parental condition. But a very large mutation will probably be worse than the parental version, even if it is a step in the right direction, because it will overshoot. To push to the extreme, imagine an otherwise normal man with thighs 2 meters long. Fisher's argument was more general than this. When we are talking about macromutational leaps into new phylum territory, we are no longer dealing with simple metric characters like leg length, and we need another version of the argument. The essential point, as I put it before, is that there are many more ways of being dead than of being alive. Imagine a mathematical landscape of all possible animals. I have to call it mathematical because it is a landscape in hundreds of dimensions, and it includes an almost infinitely large range of conceivable monstrosities, as well as the relatively small number of animals that have actually ever lived. What Parker calls a monumental genetic event would be equivalent to a macromutation of huge effect, not just in one dimension, as with our thigh example, but in hundreds of dimensions simultaneously. That is the scale of change we are talking about if we imagine, as Parker seems to, an abrupt and immediate change from one phylum to another. In the multidimensional landscape of all possible animals, living creatures are islands of viability, separated from other islands by gigantic oceans of grotesque deformity. Starting from any one island, you can evolve away from it one step at a time, here inching out a leg, there shaving the tip of a horn, or darkening a feather. Evolution is a trajectory through multidimensional space, in which every step of the way has to represent a body capable of surviving and reproducing about as well as the parental type reached by the preceding step of the trajectory. Given enough time, a sufficiently long trajectory leads from a viable starting point to a viable destination so remote that we recognize it as a different phylum, say mollusks. And a different step-by-step trajectory from the same starting point can lead, through continuously viable intermediates, to another viable destination, which we recognize as yet another phylum, say annelids. Something like this must have happened for each of the forks leading to each pair of animal phyla from their respective concestors. The point we are leading up to is this. A random change of sufficient magnitude to initiate a new phylum at one fell swoop will be so large, in hundreds of dimensions simultaneously, that it would have to be preposterously lucky to land on another island of viability. Almost inevitably, a mega mutation of that magnitude will land in the middle of the ocean of inviability, probably unrecognizable as an animal at all. Creationists foolishly liken Darwinian natural selection to a hurricane blowing through a junkyard and having the luck to assemble a Boeing 747. They are wrong, of course, for they completely miss the gradual, cumulative nature of natural selection. But the junkyard metaphor is entirely apt to the hypothetical overnight invention of a new phylum. An evolutionary step of the same magnitude as, say, the overnight transition from earthworm to snail really would have to be as lucky as the hurricane in the junkyard. We can then, with complete confidence, reject the third of our three hypotheses, the bonkers one. That leaves the other two, or some compromise between them, and here I find myself agnostic and eager for more data. It seems to be increasingly accepted that the early molecular clock estimates were exaggerating when they pushed the major branch points hundreds of millions of years back into the Precambrian. On the other hand, the mere fact that there are few, if any, fossils of most animal phyla before the Cambrian should not stampede us into assuming that those phyla must have evolved extremely rapidly. The hurricane in a junkyard argument tells us that all those Cambrian fossils must have had continuously evolving antecedents. Those antecedents had to be there, but they have not been discovered. Whatever the reason, and whatever the timescale, they failed to fossilize, but they must have been there. On the face of it, it is harder to believe that a whole lot of animals could be invisible for 100 million years than that they could be invisible for only 10 million years. This leads some people to prefer the short-fuse Cambrian explosion theory. On the other hand, the shorter you make the fuse, the harder it is to believe all that diversification could be crammed into the time available. So this argument cuts both ways and doesn't decisively choose between our two surviving hypotheses. The fossil record is not completely void of metazoan life before Chengjiang and Sirius Passet. Around 20 million years earlier, almost plumb on the Cambrian-Precambrian boundary, start to appear a variety of microscopic fossils that look rather like tiny shells. Together they are known as the small shelly fauna. It came as a surprise to most paleontologists when some of these were identified as armor plating from lobopods, relatives of the velvet worm. That means that the divergences between different groups of protostomes must have occurred in the Precambrian before the visible explosion. And there are hints of older animal diversity. 20 million years before the start of the Cambrian, in the Ediacaran period of the late Precambrian, there was a worldwide flourishing of a mysterious group of animals called the Ediacaran fauna, named after the Ediacara Hills in South Australia, where they were first found. It is hard to know quite what most of them were, but they were among the first large animals to be fossilized. Some of them are probably sponges, some are a bit like jellyfish, others somewhat resemble sea anemones or sea pens, feather-like relatives of sea anemones. Some look a bit worm-like or slug-like, and could conceivably represent true bilateria. Others are just plain mysterious. There is even one tadpole-like fossil from Australia, still not formally described, that is suspected of being a chordate. That's the phylum, remember, to which the vertebrates belong. If this turns out to be right, it would be very exciting, but we must wait and see. In spite of such tantalizing straws in the wind, the consensus among zoologists is that the Ediacaran fauna, though intriguing, doesn't help us much one way or the other in tracing the ancestry of most modern animals. There are also fossil imprints that appear to be the trails or burrows of Precambrian animals. These traces tell us of the early existence of crawling animals large enough to make them. Unfortunately, they don't tell us much about what those animals looked like. There are also some even older, mostly microscopic fossils found at Dushantuo in China, which appear to be embryos, though it is not clear what kind of animal they might have grown into. Older still are small disc-shaped impressions from northwest Canada, dated between about 600 and 610 million years ago, but these animals are, if anything, even more enigmatic than the Ediacaran forms. I wouldn't be surprised to see either of the first two hypotheses vindicated. I'm not sticking my neck out, but I'll eat my hat if any evidence is ever found in favor of hypothesis 3. there is every reason to suppose that evolution in the Cambrian was essentially the same kind of process as evolution today. All that overexcited rhetoric about the mainspring of evolution running down after the Cambrian, all that euphoric shouting about wild, heedless dances of extravagant invention, with new phyla leaping into existence in a blissful dawn of zoological irresponsibility. Now here's something I am prepared to stick my neck out for. All that stuff is just plain dotty.
The epilogue to the velvet worm's tale explains the ins and outs of the so-called molecular clock, which enables biologists to put a date on the last common ancestor of any two living creatures by comparing their DNA or its products. But now on to the next rendezvous.
Rendezvous 27 to 33. The next 6 rendezvous occur so far into the past that we are very uncertain of their timing or even, in some cases, their order. In the course of them we greet an obscure group of worms called the aceals, the large and important group of cnidarians, which includes jellyfish and sea anemones, and the superficially jellyfish-like ctenophores, or comb jellies. The jellyfish's tale is about the vertical migrations of plankton. The polypifer's tale extols coral reefs as the largest biological structures on Earth. Next, at Rendezvous 30, our pilgrimage is joined by Trichoplax, a tiny mat-like creature that would not be noticed but for its pivotal position in the tree of life. The sponges, who join us at Rendezvous 31, and the choanoflagellates at Rendezvous 32, are important because of what they may be telling us about the origin of the metazoa, animals constructed of many cells. The sponge's tale introduces the story, and the choanoflagellate's tale completes it, so far as our ignorance of such ancient events allows. Rendezvous 33 sees the arrival of another group of tiny and obscure distant cousins, the so-called drips, known today only as parasites of fish and other freshwater animals. Then, at Rendezvous 34, the pilgrimage is joined by the fungi, one of the great kingdoms of life, followed at Rendezvous 35 by the amoebozoans, one of the groups once lumped under the catch-all name protozoa, and including the famous amoeba.
Rendezvous 36. Plants. Rendezvous 36 is where we meet the true lords of life, the plants. Life could get along without animals and without fungi, but abolish the plants and life would rapidly cease. Plants sit indispensably at the base, the very foundation of nearly every food chain. They are the most noticeable creatures on our planet, the first living things any visiting Martian would remark. By far the largest single organisms that ever lived are plants, and an impressive percentage of the world's biomass is locked up in plants. This doesn't just happen to be so. Some such high proportion follows necessarily from the fact that almost all biomass comes ultimately from the sun via photosynthesis, most of it in green plants, and the transaction at every link of the food chain is only about 10% efficient. The surface of the land is green because of plants, and the surface of the sea would be green too if its floating carpet of photosynthesizers were macroscopic plants instead of microorganisms too small to reflect noticeable quantities of green light. It is as though plants are going out of their way to cover every square centimetre with green, leaving none uncovered. And that is pretty much what they are doing, for a very sensible reason. A finite number of photons reaches the planet's surface from the sun, and every last photon is precious. The total number of photons that can be garnered from its star by a planet is limited by its surface area, with the complication that only one side is facing its star at any one time. From a plant's point of view, a square centimetre of the Earth's surface that is anything but green amounts to a negligently wasted opportunity to sweep up photons. Leaves are solar panels, as flat as possible to maximize photons caught per unit expenditure. There is a premium on placing your leaves in such a position that they are not overshadowed by other leaves, especially somebody else's leaves. This is why forest trees grow so tall. Tall trees that are not in the forest are out of place, probably because of human interference. It is a complete waste of effort to grow tall if you're the only tree around, It's much better to spread out sideways, like grasses, because that way you trap more photons per unit of effort put into growing. As for forests, it is no accident that they are so dark. Every photon that makes it to the ground represents failure on the part of the leaves above. With few exceptions, such as Venus flytraps, plants don't move. With few exceptions, such as sponges, animals do. Why the difference? It must have to do with the fact that plants eat photons, while animals ultimately eat plants. We need that ultimately, of course, because the plants are sometimes eaten at second or third hand via animals eating other animals. But what is it about eating photons that makes it a good idea to sit still with roots in the ground? What is it about eating plants, as opposed to being a plant, that makes it a good idea to move? Well, given that plants stay still, animals have got to move in order to eat them. But why do plants stay still? Maybe it has something to do with the need to be rooted in order to suck nutrients out of the soil. Maybe there is too unbridgeable a distance between the best shape to be if you want to move, solid and compact, and the best shape to be if you want to expose yourself to lots of photons, high surface area, hence straggly and unwieldy. Whatever the reason, of the three great groups of mega-life that have evolved on this planet, two of them, the fungi and the plants, stay mostly still as statues, while the third group, the animals, do most of the scurrying about, most of the active go-getting. Plants even make use of animals to do their scurrying for them, and flowers, with their beauteous colours, shapes and scents, are the instruments of this manipulation.
The cauliflower's tail tells of a brilliant piece of recent theory which explains the previously mysterious Kleiber's law, one of the most precisely and widely obeyed mathematical laws in all of biology. The redwood's tail explains the various methods of dating ancient relics, from tree rings through carbon dating to potassium-argon dating, which are so important throughout the book. Rendezvous 37 is called Uncertain, to dramatize that we are here even more ignorant about the exact branchings of the tree of life than usual at this great age. The pilgrims who join us hereabouts include a great variety of microbes, which would once have been lumped under protozoa or algae. One of them, the mixotrich, which lives in termite guts, tells a revealing tale.
The mixotrich's tale. Mixotricha paradoxa means unexpected combination of hairs, and we shall see why in a moment. It is a microorganism that lives in the gut of an Australian termite, Darwin's termite, Mastotermes darwiniensis. Pleasingly, though not necessarily for the human inhabitants, one of the main places where it flourishes is the town of Darwin in northern Australia. Termites bestride the tropics like a distributed colossus. In tropical savannas and forests, they reach population densities of 10,000 per square metre, and are estimated to consume up to a third of the total annual production of dead wood, leaves and grass. Their biomass per unit area is double that of migrating herds of wildebeest on the Serengeti and Masai Mara, but is spread across the entire tropics. If you ask the source of the termites' alarming success, it is twofold. First, they can eat wood, which includes cellulose, lignin and other matter that animal guts normally can't digest. Second, they are highly social and gain great economies from division of labour among specialists. A termite mound has many of the attributes of a single large and voracious organism, with its own anatomy, its own physiology, and its own mud-fashioned organs, including an ingenious ventilation and cooling system. The mound itself stays in one place, but it has a myriad mouths and 6 myriad legs, and these range over a foraging area the size of a football pitch. Termites' legendary feats of cooperation are possible in a Darwinian world only because the majority of individuals are sterile but closely related to a minority who are very fertile indeed. Sterile workers act like parents towards their younger siblings, thereby freeing the queen to become a specialized egg factory, and a grotesquely efficient one at that. Genes for worker behavior are passed to future generations via the minority of the workers' siblings who are destined to reproduce, helped by the majority of their siblings who are destined to be sterile. You will appreciate that the system works only because it is a strictly non-genetic decision whether a young termite shall become a worker or a reproducer. The analogy of insect colony to human body is often made, and it's not a bad one. The majority of our cells subjugate their individuality, devoting themselves to assisting the reproduction of the minority that are capable of it. Germ-line cells in the testes or ovaries, whose genes are destined to travel, via sperms or eggs, into the distant future. But genetic relatedness is not the only basis for subjugation of individuality in fruitful division of labour. Any sort of mutual assistance, where each side corrects a deficiency in the other, can be favoured by natural selection on both. To see an extreme example, we dive inside the gut of an individual termite, that seething and, as we may assume, noisome chemostat which is the world of the mixer trick. Termites, as we have seen, enjoy an additional advantage over bees, wasps, and ants: their prodigious feats of digestion. There is almost nothing that termites can't eat, from houses to billiard balls to priceless first folios. Wood is potentially a rich food source, but it is denied to almost all animals because cellulose and lignin are so indigestible. Termites and certain cockroaches are the outstanding exception. Termites are indeed related to cockroaches, and Darwin's termite, like other so-called lower termites, is a sort of living fossil. One could imagine it halfway between cockroaches and advanced termites. In order to digest cellulose, you need enzymes called cellulases. Most animals can't make cellulases, but some microorganisms can. As Taxtail will explain, bacteria and archaea are biochemically more versatile than the rest of the living kingdoms put together. Animals and plants perform a fraction of the biochemical mix of tricks available to bacteria. For digesting cellulose, herbivorous mammals all rely upon microbes in their guts. Over evolutionary time, they have entered into a partnership in which they make use of chemicals such as acetic acid, which to the microbes are waste products. The microbes themselves gain a safe haven with plenty of raw materials for their own biochemistries, pre-processed and ready chopped into small, manageable pieces. All herbivorous mammals have bacteria in the lower gut, which the food reaches after the mammal's own digestive juices have had a go at it. Sloths, kangaroos, colobus monkeys, and especially cud-chewing ruminants have independently evolved the trick of also keeping bacteria in the upper portion of the gut, which precedes the mammal's own main digestive efforts. Unlike mammals, termites are capable of manufacturing their own cellulases, at least in the case of the so-called advanced termites. But up to 1/3 of the net weight of a more primitive, in other words more cockroach-like termite, such as Darwin's termite, consist of its rich gut fauna of microbes, including eukaryotic protozoa as well as bacteria. The termites locate and chew the wood into small pieces. The microbes live on the wood fragments, digesting them with enzymes unavailable to the termite's own biochemical toolkit. Or you could say the microbes have become tools in the termite's toolkit. As with the cattle, it is the waste products of the microbes that the termites live on. We could say that Darwin's termite and the other primitive termites farm microorganisms in their guts. And this brings us finally to the mixer trick, whose tale this is.
Mixotricha paradoxa is not a bacterium. Like many of the microbes in termite guts, it is a large protozoan, 0.5 millimetre long or more, and large enough to contain hundreds of thousands of bacteria inside itself, as we shall see. It lives nowhere except in the gut of Darwin's termite, where it is a member of the mixed community of microbes that thrive on the wood chips milled by the termite's jaws. Microorganisms populate the termite's gut as richly as the termites themselves populate the mound, and as termite mounds populate the savanna. If the mound is a town of termites, each termite gut is a town of microorganisms. We have here a two-level community. But, and now we come to the crux of the tale, there is a third level, and the details are utterly remarkable. Mixotricha itself is a town of bacteria. The full story was revealed by the work of L.R. Cleveland and A.V. Grimstone, but it is especially the American biologist Lynn Margulis who has drawn our attention to Mixotricha's significance for evolution. Mixotricha's surface appears to be covered with cilia, a carpet of tiny beating hairs. But Cleveland and Grimstone demonstrated with the electron microscope that Mixotricha's cilia are not cilia at all. They are bacteria. Each one of the hundreds of thousands of tiny hairs is a single spirochete, a bacterium whose entire body is a long, wiggling hair. Some important diseases, such as syphilis, are caused by spirochetes. They normally swim freely, but Mixotricha's spirochetes are stuck to its body wall, exactly as though they were cilia. They don't move like cilia, however. They move like spirochetes. Cilia move with an actively propulsive rowing stroke, followed by a recovery stroke in which they bend so as to present less resistance to the water. Spirochetes undulate in a completely different and very characteristic manner, and that is just what Mixotricha's hairs do. Amazingly, they seem to be coordinated with each other, moving in waves that begin at the front end of the body and travel backwards. Cleveland and Grimstone measured the wavelength, the distance between wave crests, as about 1/100th of a millimeter. This suggests that the spirochetes are somehow in touch with each other. Probably they are literally in touch, responding directly to the movements of neighbors with a delay that determines the wavelength. I don't think it is known why the waves pass from front to back. What is known is that the spirochetes are not just jammed haphazardly into the Mixotrich skin. On the contrary, the Mixotrich has, in a repeat pattern all over its surface, a complicated apparatus for holding spirochetes and, what's more, pointing them in a posterior direction, so that their undulating movements drive the Mixotrich forwards. If these spirochetes are parasites, it is hard to think of a more remarkable example of a host being friendly to its parasites. Each spirochete has its own little emplacement, called a bracket by Cleveland and Grimstone. Each bracket is tailor-made to hold one spirochete, or sometimes more than one. No cilium could ask for more. It becomes quite tricky to draw the line between own body and alien body in such cases, and that, to anticipate, is one of the main messages of this tale. The resemblance to cilia goes further. If you look with a powerful microscope into the very fabric of a typical ciliate protozoan, such as Paramecium, you'll find that every cilium has a so-called basal body at its root. Now, amazingly, although the cilia of Mixotricha are not cilia at all, they do appear to have basal bodies. Each spirochete-toting bracket has at its base one basal body, shaped rather like a vitamin pill. Except that, well, having learned about Mixotricha's idiosyncratic way of doing things, what would you guess those basal bodies actually are? Yes, they too are bacteria. A completely different kind of bacteria, not spirochetes but oval, pill-shaped bacteria. Over large parts of the body wall there is a one-to-one relationship between bracket, spirochete, and basal bacterium. Each bracket has one spirochete stuck through it and one pill bacterium at its base. Looking at this, it is easy to understand why J.L. Sutherland, who first examined them in the 1930s, saw cilia. She naturally expected to see basal bodies wherever there are cilia, and when she looked, lo and behold, there were the basal bodies. Little could she know, both cilia and basal bodies were hitchhiking bacteria. As for the four large flagella at the front end, they seem to be used not for propulsion at all, but as rudders for steering the craft as it is propelled by the thousands of spirochete galley slaves. Much as I'd like to claim it, by the way, that evocative phrase is not my own. It was coined by S.L. Tam, who found, after Cleveland and Grimstone's work on Mixotricha, that other termite gut protozoa do the same trick, but instead of spirochetes, their galley slaves are ordinary bacteria with flagella. Now for the other bacteria in the Mixotrich, the pill-shaped ones that look like basal bodies. What are they doing? Are they contributing to the economy of their host? Are they getting something out of the relationship themselves? Probably yes, but it has not been shown definitely. They may well be making cellulases that digest wood, for of course the mixotrichs subsist on the tiny chips of wood in the termite's gut, originally broken up by the powerful jaws of the termite. We have here a triple-decker dependency, reminiscent of Jonathan Swift's verse.
So, naturalists observe, a flea has smaller fleas that on him prey, and these have smaller still to bite 'em, and so proceed ad infinitum. Thus every poet in his kind is bit by him that comes behind.
Augustus de Morgan came behind for another bite, giving us the rhyme in the form that most of us know today.
Great fleas have little fleas upon their backs to bite 'em, and little fleas have lesser fleas, and so ad infinitum. And the great fleas themselves, in turn, have greater fleas to go on, while these again have greater still, and so on.
Finally we come to the strangest part of the mixotrich's tale, the climax towards which the narrative has been directed. This whole story of vicarious biochemistry, the borrowing by greater creatures of the biochemical talents of lesser ones inside them, is charged with evolutionary déjà vu. The message of the mixotrich to the rest of the pilgrims is this: this has all happened before.
The Great Historic Rendezvous. Rendezvous in this book has a special meaning, following the central metaphor of a backwards pilgrimage. But there is one cataclysmic event, arguably the most decisive event in the history of life, which really was a rendezvous, literally, a historic rendezvous that actually took place in the true forward direction of history. This was the origin of the eukaryotic nucleated cell, the high-tech miniature machine that is the micro-foundation of all large-scale and complex life on this planet. To distinguish it from all the other metaphorical backwards rendezvous points, we may call it the Great Historic Rendezvous. The word historic has here a double meaning. It means of major importance, and it also means forward chronology, as opposed to backward.
I have referred to the Great Historic Rendezvous as one event because of what now appears to be its single momentous consequence, the evolution of the eukaryotic cell, with its nucleus to contain the chromosomes, its complicated ultrastructure of membranes, and its self-reproducing miniature organelles, such as mitochondria, and in plants, chloroplasts. But it was actually 2 or 3 events, perhaps widely spaced in time. Each one of these historic rendezvous events was a merging with bacterial cells to form a larger cell. The mixotrich's tale, as a recent reenactment, has prepared us to see the kind of thing that happened. Perhaps 2 billion years ago, an ancient single-celled organism, some kind of proto-protozoan, entered into a strange relationship with a bacterium, a relationship similar to that between Mixotricha and its bacteria. As with Mixotricha, the same thing happened more than once with different bacteria, the events possibly separated by hundreds of millions of years. All our cells are like individual mixotrichs, stuffed with bacteria, which have become so transformed by generations of cooperation with the host cell that their bacterial origins are almost lost to sight. As with the mixotrich, only more so, the bacteria have become so intimately enmeshed in the life of the eukaryotic cell that it was a major scientific triumph to detect that they were there at all. I'm fond of the Cheshire cat simile used by Sir David Smith, one of our leading experts on symbiosis, for the cooperative living together of once distinct elements in cells.
In the cell habitat, an invading organism can progressively lose pieces of itself, slowly blending into the general background, its former existence betrayed only by some relic. Indeed, one is reminded of Alice in Wonderland's encounter with the Cheshire cat. As she watched it, it vanished quite slowly, beginning with the tail and ending with the grin, which remained some time after the rest of it had gone. There are a number of objects in a cell like the grin of the Cheshire cat. For those who try to trace their origin, the grin is challenging and truly enigmatic. What are the biochemical tricks that these once free bacteria brought into our lives,
tricks that they perform to this day, tricks without which all life would instantly cease. Two most important ones are photosynthesis, which uses solar power to synthesize organic compounds and oxygenates the air as a byproduct, and oxidative metabolism, which uses oxygen, ultimately from plants, to slow burn the organic compounds and redeploy the energy that originally came from the sun. These chemical technologies were developed before the great historic rendezvous by different bacteria, and in a sense, bacteria are still the only game in town. All that has changed is that they now practice their biochemical arts in the purpose-built factories called eukaryotic cells. Photosynthetic bacteria used to be called blue-green algae, a terrible name, since most of them aren't blue-green and none of them are algae. Most of them are green, and it is better to call them green bacteria, although some are reddish, yellowish, brownish, blackish, or yes, in some cases, bluish-green. Green also is sometimes used as a word for photosynthetic, and in that sense, too, green bacteria is a good name. Their scientific name is cyanobacteria. They are true bacteria, and they seem to be a good monophyletic group. In other words, all of them, and nothing else, are descended from a single ancestor which would, itself, have been classified as a cyanobacterium.
The green colour of algae and of cabbages, pine trees and grasses all comes from small green bodies called chloroplasts within their cells. Chloroplasts are distant descendants of once free-living green bacteria. They still have their own DNA, and they still reproduce by asexual division, building up to a substantial population within each plant cell. As far as a chloroplast is concerned, it is a member of a reproducing population of green bacteria. The world in which it lives and reproduces is the interior of a plant cell. From time to time, its world suffers a minor upheaval when the plant cell divides into two daughter cells. Roughly half the chloroplasts find themselves in each daughter cell, and they soon resume their normal existence of reproducing to populate their new world with chloroplasts. All the while, the chloroplasts use their green pigment to trap photons from the sun and channel the sun's energy in the useful direction of synthesizing organic compounds from carbon dioxide and water supplied by the host plant. The oxygen wastes are partly used by the plant and partly exhaled into the atmosphere through holes in the leaves, called stomata, singular stoma. The organic compounds synthesized by the chloroplasts are ultimately made available to the host plant cell. Interestingly reminiscent of the Mixotrix tale, some chloroplasts show evidence of having entered plant cells indirectly by piggybacking inside other eukaryotic cells, which would presumably have been called algae. The evidence is that some chloroplasts have a double membrane. Presumably the inner one is the wall of the original bacterium, the outer one the wall of the alga. As with Mixotricha, we can see recent reenactments in the many examples of single-celled green algae being incorporated in the cells or tissues of fungi and animals, for example the green algae that inhabit corals. Those chloroplasts that have a single membrane presumably entered directly, not on the coattails of algae. All the free oxygen in the atmosphere comes from green bacteria, whether free-living or in the form of chloroplasts. When it first appeared in the atmosphere, oxygen was a poison. Indeed, some people colourfully say it still is a poison, which is why doctors advise us to eat antioxidants. It was a brilliant chemical coup to discover how to use oxygen to extract originally solar energy from organic compounds. This discovery, which can be seen as a sort of reverse photosynthesis, was entirely made by bacteria, but a different kind of bacteria. As with photosynthesis itself, bacteria still have a monopoly on the technology, except that again, as with photosynthesis, eukaryotic cells like ours give house room to these oxygen-loving bacteria, who now travel under the name of mitochondria. We have become so dependent on oxygen via the biochemical wizardry of mitochondria that the statement that it is a poison makes sense only when uttered in a tone of self-conscious paradox. Carbon monoxide, the deadly poison in car exhausts, kills us by competing with oxygen for the favours of our oxygen-carrying hemoglobin molecules. Depriving somebody of oxygen is a swift way to kill them. Yet our own cells, unaided, wouldn't know what to do with oxygen. It is only mitochondria and their bacterial cousins that do. As with chloroplasts, molecular comparison tells us the particular group of bacteria from which mitochondria are drawn. Mitochondria sprang from the so-called alpha-proteobacteria, and they are therefore related to the rickettsias which cause typhus and other nasty diseases. Mitochondria themselves have lost much of their original genome and have become completely adapted to life inside eukaryotic cells. But like chloroplasts, they still reproduce autonomously by division. Although mitochondria have lost most of their genes, they haven't lost all of them, and this is very fortunate for molecular geneticists, as we've seen in many places throughout this book. Because the great historic rendezvous is a true rendezvous in the forward historical direction, our pilgrimage from now on should strictly be a split pilgrimage. We should follow the separate backward pilgrimages of the various participants in the eukaryotic compact until they are finally reunited in the deep past, but that would make for a gratuitously complicated journey.
Rendezvous 38 and 39. The remaining pilgrims yet to join are all prokaryotes. At one time they would all have been called bacteria, but molecular studies have shown that there are two great prokaryotic kingdoms, the true bacteria, or eubacteria, and the archaebacteria, or archaea. Current opinion places us eukaryotes slightly closer to the archaea than to the eubacteria. In pilgrimage terminology, this means that the archaean pilgrims join us at Rendezvous 38 and the eubacteria at Rendezvous 39. Two of the eubacteria tell tales.
The rhizobium's tale. The wheel is the proverbial human invention. Take apart any machine of more than rudimentary complexity, and you'll find wheels. Ship and aeroplane propellers, spinning drills, lathes, potter's wheels. Our technology runs on the wheel and would seize up without it. The wheel may have been invented in Mesopotamia during the 4th millennium BC. We know it was elusive enough to need inventing because the New World civilization still lacked it by the time of the Spanish conquest. The alleged exception here, children's toys, seems so bizarre as to prompt suspicion. Could it be one of those myths that spreads purely because it is so memorable, like the Inuit having 50 words for snow? Whenever humans have a good idea, zoologists have grown accustomed to finding it anticipated in the animal kingdom. Examples pervade this book, including echo-ranging bats, electrolocation, the duckbill's tail, the dam, the beaver's tail, the parabolic reflector, limpets, the infrared heat-seeking sensor, some snakes, the hypodermic syringe, wasps, snakes and scorpions, the harpoon, cnidarians, and jet propulsion, squids. Why not the wheel? It is possible that the wheel impresses us only by contrast with our rather undistinguished legs. Before we had engines driven by fuels, fossilized solar energy, we were easily outpaced by animal legs. No wonder Richard III offered his kingdom for four-footed transportation out of his predicament. Perhaps most animals wouldn't benefit from wheels because they can already run so fast on legs. After all, until very recently, all our wheeled vehicles have been pulled by leg power. To a horse, a wheel is something that slows you down. Here's another way in which we risk overrating the wheel. It is dependent for maximum efficiency on a prior invention, the road, or other smooth, hard surface. A car's powerful engine enables it to beat a horse or a dog or a cheetah on a hard, flat road, but run the race over wild country or ploughed fields, perhaps with hedges or ditches in the way, and it is a rout. The horse will leave the car wallowing. Well then, perhaps we should change our question. Why haven't animals developed the road? There's no great technical difficulty. The road should be child's play compared with the beaver dam or the bowerbird's ornamented arena. There are even some digger wasps that tamp soil hard, picking up a stone tool to do so. Presumably the same skills could be used by larger animals to flatten a road. But it raises an unexpected problem. Even if road building is technically feasible, it is a dangerously altruistic activity. If I, as an individual, build a good road from A to B, you may benefit from the road just as much as I do. Why should this matter? Because Darwinism is a selfish game. Building a road that might help others will be penalized by natural selection. A rival individual benefits from my road just as much as I do, but he does not pay the cost of building. Freeloaders who use my road and don't bother to build their own will be free to concentrate their energy on out-reproducing me while I slave away on the road. Unless special measures are taken, genetic tendencies towards lazy, selfish exploitation will thrive at the expense of industrious road building. The upshot will be that no roads get built. With the benefit of foresight, we can see that everybody will be worse off. But natural selection, unlike us humans with our big, recently evolved brains, has no foresight. What is so special about humans that we have managed to overcome our antisocial instincts and build roads that we all share? Oh, there is so much. No other species comes remotely close to a welfare state, to an organization that takes care of the old, that looks after the sick and the orphaned, that gives to charity. On the face of it, these things present a challenge to Darwinism, but this is not the place to go into that. We have governments, police, taxation, public works to which we all subscribe, whether we like it or not. The man who wrote, "Sir, you are very kind, but I'd prefer not to join your income tax scheme," heard back, we may be sure, from the Inland Revenue. Unfortunately, no other species has invented the tax. They have, however, invented the virtual fence. An individual can secure his exclusive use of a resource if he actively defends it against rivals. Many species of animals are territorial, not just birds and mammals, but fish and insects too. They defend an area against rivals of the same species, often so as to sequester a private feeding ground or a private courtship bower or nesting area. An animal with a large territory might benefit by building a network of good flat roads across the territory from which rivals were excluded. This is not impossible, but such animal roads would be too local for long-distance, high-speed travelling. Roads of any quality would be limited to the small area that an individual can defend against genetic rivals. Not an auspicious beginning for the evolution of the wheel. But now, finally, we come to the teller of this tale. There is one revealing exception to the premise. Some very small creatures have evolved the wheel in the fullest sense of the word. The wheel may even have been the first locomotor device ever evolved, given that for most of its first 2 billion years, life consisted of nothing but bacteria. Many bacteria, of which rhizobium is typical, swim using thread-like spiral propellers, each driven by its own continuously rotating propeller shaft. It used to be thought that these flagella were wagged like tails, the appearance of spiral rotation resulting from a wave of motion passing along the length of the flagellum, as in a wriggling snake. The truth is much more remarkable. The bacterial flagellum is attached to a shaft that rotates freely and indefinitely in a hole that runs through the cell wall. This is a true axle, a freely rotating hub. It is driven by a tiny molecular motor, which uses the same biophysical principles as a muscle, but a muscle is a reciprocating engine, which after contracting has to lengthen again to prepare for a new power stroke. The bacterial motor just keeps on going in the same direction, a molecular turbine. The fact that only very small creatures have evolved the wheel suggests what may be the most plausible reason why larger creatures have not. It's a rather mundane, practical reason, but nonetheless important. A large creature would need big wheels, which, unlike man-made ones, would have to grow in situ, rather than being separately fashioned out of dead materials and then mounted. For a large living organ, growth in situ demands blood, or something equivalent, and probably something equivalent to nerves, too. The problem of supplying a freely rotating organ with blood vessels, not to mention nerves, that don't tie themselves in knots, is too vivid to need spelling out. There might be a solution, but we need not be surprised that it has not been found. Human engineers might suggest running concentric ducts to carry blood through the middle of the axle into the middle of the wheel. But what would the evolutionary intermediates have looked like? Evolutionary improvement is like climbing a mountain. You can't jump from the bottom of a cliff to the top in a single leap. Sudden, precipitous change is an option for engineers, but in nature the summit of the evolutionary mountain can be reached only via a gradual ramp upwards from the starting point. The wheel may be one of those cases where the engineering solution can be seen in plain view, yet be unattainable in evolution because it lies on the other side of a deep valley, unevolvable by large animals, but within the reach of bacteria because of their small size. In an imaginative piece of lateral thinking, Philip Pullman, in his epic of childhood fiction, His Dark Materials, solves the problem for big animals in a completely unexpected but very biological way. He invents a species of benevolent trunked animal, the mulefa, who have evolved symbiotically with a species of gigantic tree that sheds hard, circular, wheel-like seed pods. The feet of the mulefa have a horny, polished spur, which fits into a hole in the center of a seed pod, which then works as a wheel. The trees gain from the arrangement because whenever, as eventually must happen, a wheel wears out and has to be discarded, the mulefa disperse the seeds inside. The trees have evolved to return the favor by making the pods perfectly circular, with a suitable hole for the mulefan axle right in the center, into which they secrete a high-grade lubricating oil. Pullman cleverly notes that the whole system is made possible only by a geological peculiarity of the world on which these creatures live. Basalt happens to form in long ribbon-like lines over the savanna, which serve as unmade but hard roads. Short of Pullman's ingenious symbiosis, we may provisionally accept the wheel as one of those inventions that, even if it were a good idea in the first place, cannot evolve in large animals, either because of the prior need for a road, or because the problem of the twisted blood vessels could never be solved, or because the intermediates to a final solution would never be good for anything. Bacteria were able to evolve the wheel because the world of the very small is so very different and presents such different technical problems.
As it happens, the bacterial flagellar motor itself has recently, in the hands of a species of creationists who call themselves intelligent design theorists, been elevated to the status of icon of alleged unevolvability. Since it manifestly exists, the conclusion of their argument is different. Whereas I proposed unevolvability as an explanation for why large animals like mammals don't grow wheels, creationists have seized upon the bacterial flagellar wheel as something that cannot exist, and yet does. So it must have come about by supernatural means. This is the ancient argument from design, also called the argument from Paley's watchmaker, or the argument from irreducible complexity. I have less kindly called it the argument from personal incredulity, because it always has the form,
I personally cannot imagine a natural sequence of events whereby X could have come about. Therefore it must have come about by supernatural means.
Time and again, scientists have retorted that if you make this argument, it says less about nature than about the poverty of your imagination. The argument from personal incredulity would lead us to invoke the supernatural every time we see a good conjurer whose tricks we cannot fathom. It is perfectly legitimate to propose the argument from irreducible complexity as a possible explanation for the lack of something that doesn't exist, as I did for the absence of wheeled mammals. That is very different from evading the scientist's responsibility to explain something that does exist, such as wheeled bacteria. Nevertheless, to be fair, it is possible to imagine validly using some version of the argument from design or the argument from irreducible complexity. Future visitors from outer space who mount archaeological digs of our planet will surely find ways to distinguish designed machines such as planes and microphones from evolved machines such as bat wings and ears. It is an interesting exercise to think about how they will make the distinction. They may face some tricky judgments in the messy overlap between natural evolution and human design. If the alien scientists can study living specimens, not just archaeological relics, what will they make of fragile, highly strung racehorses and greyhounds, of snuffling bulldogs who can scarcely breathe and can't be born without caesarean assistance, of blear-eyed Pekingese baby surrogates, of walking udders such as Frisian cows, walking rashers such as landrace pigs, or walking woolly jumpers such as merino sheep? Molecular machines, nanotechnology, crafted for human benefit on the same scale as the bacterial flagellar motor, may pose the alien scientists even harder problems. Francis Crick, no less, has speculated semi-seriously in Life Itself that bacteria might not have originated on this planet, but been seeded from elsewhere. In Crick's fantasy, they were sent in the nose cone of a rocket by alien beings who wanted to propagate their form of life, but shrank from the technically harder problem of transporting themselves and relied, instead, upon natural evolution to finish the job once the bacterial infection had taken root. Crick and his colleague Leslie Orgel, who originally suggested the idea with him, supposed that the bacteria had originally evolved by natural processes on the home planet, but they could equally, while in the mood for science fiction, have added a touch of nanotechnological artifice to the mix. Crick himself, whether with regret or relief it is hard to say, finds little good evidence to support his own theory of directed panspermia. But the hinterland between science and science fiction constitutes a useful mental gymnasium in which to wrestle with a genuinely important question. Given that the illusion of design conjured by Darwinian natural selection is so breathtakingly powerful, how do we, in practice, distinguish its products from deliberately designed artifacts? Another great molecular biologist, Jacques Monod, began his Chance and Necessity in similar terms. Could there be genuinely persuasive examples of irreducible complexity in nature, complex organization made of parts, the loss of any one of which would be fatal to the whole? If so, might this suggest genuine design by a superior intelligence, say from an older and more highly evolved civilization on another planet? It is possible that an example of such a thing might eventually be discovered. But the bacterial flagellar motor, alas, is not it. Like so many previous allegations of irreducible complexity from the eye on, the bacterial flagellum turns out to be eminently reducible. Kenneth Miller of Brown University deals with the whole question in a tour de force of clear exposition. As Miller shows, the allegation that the component parts of the flagellar motor have no other function is simply false. As one example, many parasitic bacteria have a mechanism for injecting chemicals into host cells called the TTSS, type 3 secretory system. The TTSS makes use of a subset of the very same proteins that are used in the flagellar motor. In this case, they are used not for providing rotatory motion of a circular hub, but for making a hole in a cell wall. Miller summarizes.
Stated directly, the TTSS does its dirty work using a handful of proteins from the base of the flagellum. From the evolutionary point of view, this relationship is hardly surprising. In fact, it's to be expected that the opportunism of evolutionary processes would mix and match proteins to produce new functions. According to the doctrine of irreducible complexity, however, this should not be possible. If the flagellum is indeed irreducibly complex, then removing just one part, let alone 10 or 15, should render what remains, by definition, non-functional. Yet the TTSS is indeed fully functional, even though it is missing most of the parts of the flagellum. The TTSS may be bad news for us, but for the bacteria that possess it, it is a truly valuable biochemical machine. The existence of the TTSS in a wide variety of bacteria demonstrates that a small portion of the irreducibly complex flagella mechanism can indeed carry out an important biological function. Since such a function is clearly favored by natural selection, the contention that the flagellum must be fully assembled before any of its component parts can be useful is obviously incorrect. What this means is that the argument for intelligent design of the flagellum has failed.
Miller's indignation at intelligent design theory receives a boost from an interesting source, his deep religious convictions, which are more fully articulated in Finding Darwin's God. Miller's God, if not Darwin's, is the God revealed in, or perhaps synonymous with, the deep lawfulness of nature. The creationist quest to demonstrate God through the negative route of the argument from personal incredulity turns out, as Miller shows, to assume that God capriciously violates his own laws, and this, to those like Miller of a thoughtfully religious disposition, is a cheap and demeaning sacrilege. As a non-religious person, I can sympathetically buttress Miller's argument with a parallel one of my own. If not sacrilegious, the intelligent design style of argument from personal incredulity is lazy. I satirized it in an imagined conversation between Sir Andrew Huxley and Sir Alan Hodgkin, both sometime presidents of the Royal Society, who shared the Nobel Prize for working out the molecular biophysics of the nerve impulse.
I say, Huxley, this is a terribly difficult problem. I can't see how the nerve impulse works, can you?
No, Hodgkin, I can't, and these differential equations are fiendishly hard to solve. Why don't we just give up and say that the nerve impulse propagates by nervous energy?
Excellent idea, Huxley. Let's write the letter to nature now. It'll only take one line, then we can turn to something easier.
Andrew Huxley's elder brother Julian made a similar point when, long ago, he satirized vitalism, then usually epitomized by Henri Bergson's name of élan vital, as tantamount to explaining that a railway engine was propelled by élan locomotif. My censure of laziness and Miller's of sacrilege do not apply to the hypothesis of directed panspermia. Crick was talking about superhuman, not supernatural design. The difference really matters. On Crick's worldview, superhuman designers of bacteria, or of the means to seed Earth with them, would themselves have originally evolved by some local equivalent of Darwinian selection on their own planet. Crucially, Crick would always seek what Daniel Dennett calls a crane, would never resort, as Henri Bergson would, to a skyhook. The main objection to the irreducible complexity argument amounts to a demonstration that the allegedly irreducible complex entity, the flagellar motor, the blood-clotting cascade, the Krebs cycle, or whatever it might be, is actually reducible. The personal incredulity was simply wrong. To this we add the reminder that, even if we can't think of a step-by-step pathway by which the complexity might have evolved, the eager slide to assuming that it is therefore supernatural is either sacrilegious or lazy, according to taste.
But there is another objection that needs to be mentioned, the arch and scaffolding of Graham Cairns Smith. Cairns Smith was writing in a different context, but his point works here too. An arch is irreducible in the sense that if you remove part of it, the whole collapses. Yet it is possible to build it gradually by means of scaffolding. The subsequent removal of the scaffolding, so that it no longer appears in the visible picture, does not entitle us to a mystified and obscurantist attribution of supernatural powers to the masons. The flagellar motor is common among bacteria. Rhizobium was chosen to tell the tale because of the second claim to impress us with the versatility of bacteria. Farmers sow plants of the pea family, Leguminosae, as a part of most good crop rotation schemes for one very good reason. Leguminous plants can use raw nitrogen straight out of the air. It is by far the most abundant gas in our atmosphere, rather than having to suck up nitrogen compounds from the soil. But it isn't the plants themselves that fix atmospheric nitrogen and turn it into usable compounds. It is symbiotic bacteria, specifically Rhizobium, housed for the purpose in special nodules provided for them, with every indication of inadvertent solicitude on the roots of the plants. Such contracting out of ingenious chemical tricks to chemically much more versatile bacteria is an extremely common pattern throughout animals and plants. It is the main message of Tack's tale. Tack is the nickname of Thermus aquaticus, the heat-loving bacterium who tells the last tale in the book about the enormous versatility of bacteria as biochemists compared to us eukaryotes.
Tack's tale. Although our tales are not in the main told in the first person, let us make an exception for the last word of all our tales and give it to Thermus aquaticus.
Look at life from our perspective, and you eukaryotes will soon cease giving yourselves such airs. You bipedal apes, you stump-tailed tree shrews, you desiccated lobe-fins, you vertebrated worms, you hoxed-up sponges, you newcomers on the block, you eukaryotes, you barely distinguishable members of a monotonously narrow parish, you are little more than fancy froth on the surface of bacterial life. Why, the very cells that build you are themselves colonies of bacteria, replaying the same old tricks we bacteria discovered 1 billion years ago. We were here before you arrived, and we shall be here after you are gone.
Canterbury. As befits the destination of a 4 billion-year pilgrimage, our Canterbury has a patina of mystery. It is the singularity known as the origin of life, but we could better call it the origin of heredity. Life itself is not clearly defined, a fact that contradicts intuition and traditional wisdom. Ezekiel chapter 37, in which the prophet was commanded down into the valley of the bones, identifies life with breath. I cannot resist quoting the passage, bone to his bone, such wonderful economy of language.
So I prophesied as I was commanded, and as I prophesied there was a noise, and behold a shaking, and the bones came together, bone to his bone. And when I beheld, lo, the sinews and the flesh came up upon them, and the skin covered them above, but there was no breath in them. Then said he unto me, Prophesy unto the wind, prophesy, Son of man, and say to the wind, Thus saith the Lord God, Come from the 4 winds, O breath, and breathe upon these slain, that they may live.
And of course the winds did. A great army breathed and stood up. Breath, for Ezekiel, defines the difference between dead and alive. Darwin himself implied the same in one of his more eloquent passages, the concluding words of The Origin of Species.
Thus, from the war of nature, from famine and death, the most exalted object which we are capable of conceiving, namely the production of the higher animals, directly follows. There is grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been, and are being, evolved.
Darwin rightly reversed Ezekiel's order of events. The breath of life came first and created the conditions under which bones and sinews, flesh and skin would eventually evolve. Incidentally, the phrase by the Creator is not present in the first edition of The Origin. It was added in the second edition, probably as a sop to the religious lobby. Darwin later regretted this in a letter to his friend Hooker.
I have long since regretted that I truckled to public opinion and used the Pentateuchal term of creation, by which I really meant appeared by some wholly unknown process. It is mere rubbish thinking at present of the origin of life. One might as well think of the origin of matter.
Darwin probably, and in my view rightly, saw the origin of primitive life as a relatively, and I stress relatively, easy problem, compared with the one he solved, how life, once begun, developed its amazing diversity, complexity, and powerful illusion of good design. Nevertheless, Darwin did later, in another letter to Hooker, venture a guess about the wholly unknown process that started it all. He was led to it through wondering why we don't see life originating again and again.
It is often said that all the conditions for the first production of a living organism are now present which could ever have been present. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes, at the present day such matter would be instantly absorbed, which would not have been the case before living creatures were found.
The doctrine of spontaneous generation had only lately been experimentally attacked by Pasteur. It had long been believed that rotting meat spontaneously generated maggots, that goose barnacles spontaneously generated goslings, and even that dirty laundry placed with wheat generated mice. Perversely, the spontaneous generation theory was supported by the Church, following Aristotle in this, as in so much else. I say perversely because, at least with hindsight, spontaneous generation was as direct a challenge to divine creation as evolution would ever be. The idea that flies or mice could spring spontaneously into existence hugely underestimates the stupendous achievement that the creation of flies or mice would be, an insult to the Creator, one might have thought. But the science-free mindset fails to grasp how complex and inherently improbable a fly or a mouse is. Darwin was perhaps the first to appreciate the full magnitude of this mistake. As late as 1872, in a letter to Wallace, the co-discoverer of natural selection, Darwin could still find it necessary to express his skepticism about rotifers and tardigrades being spontaneously generated, as had been suggested in a book, The Beginnings of Life, which he otherwise admired. His skepticism was on target as usual. Rotifers and tardigrades are complex life forms beautifully fitted to their respective ways of life. For them to be spontaneously generated would imply that they became fit and complex
by a happy accident, and this I cannot believe.
Happy accidents of such magnitude were anathema to Darwin, as they should have been to the Church, for a different reason. The whole rationale of Darwin's theory was, and is, that adaptive complexity comes about by slow and gradual degrees, step by step, no single step making too large a demand on blind chance as explanation. The Darwinian theory, by rationing chance to the small steps needed to supply variation for selection, provides the only realistic escape from sheer luck as the explanation for life. If rotifers could spring into existence just like that, Darwin's life work was unnecessary. But natural selection itself had to have a beginning. In this sense alone, some kind of spontaneous generation must have happened, if only once. The beauty of Darwin's contribution was that the single spontaneous generation we must postulate did not have to synthesize anything complicated like a maggot or a mouse. It only had to make, well, now we approach the heart of the problem. If not breath, what was the vital ingredient that first enabled natural selection to get going and lead eventually, after epochs of cumulative evolution, to maggots, mice, and men? The details lie buried, perhaps beyond recovery, at our ancient Canterbury. But we can give the key ingredient a minimalist name to express the kind of thing it must have been. That name is heredity. We should be seeking not the origin of life, which is vague and undefined, but the origin of heredity, true heredity, and that means something very precise. I have previously invoked fire to help explain it. Fire rivals breath as imagery for life. When we die, the fire of life goes out. Our ancestors who first tamed it probably thought fire a living thing, a god even. Staring into flames or embers, especially at night when the campfire warmed and protected them, did they commune in imagination with a glowing, dancing soul? Fire stays alive as long as you feed it. Fire breathes air. You can suffocate it by cutting off its oxygen supply. You can drown it with water. Wildfire devours the forest, driving animal prey before it with the speed and ruthlessness of a pack of wolves in literally hot pursuit. As with wolves, our ancestors could capture a fire cub as a useful pet, tame it, feed it regularly, and clear away its ashy excreta. Before the art of fire-making was discovered, society would have prized the lesser art of husbanding a captured fire. Perhaps a live scion of the home fire was carried in a pot for barter to a neighbouring group whose own fire had unfortunately died. Wildfires would have been observed giving birth to daughter fires, spitting sparks and live cinders up on the wind, like dandelion puffs, to land and seed the dry grass at a distance. Did ergastrine philosophers theorize that fire cannot spontaneously generate, but must always be born of a parent fire, either wildfire out on the plains, or domestic fire fenced in by hearthstones? And did the first fire-making sticks therefore rub out a world view? Our ancestors might even have imagined a population of reproducing wildfires, or a pedigree of descent among domestic fires, traced from a glowing ancestor, bought from a distant clan, and traded on to others. But still there was no true heredity. Why not? How can you have reproduction and a pedigree, yet no heredity? This is the lesson fire has for us here. True heredity would mean the inheritance not of fire itself, but of variations among fires. Some fires are yellower than others, some redder, some roar, some crackle, some hiss, some smoke, some spit. Some have tinges of blue or green amongst the flames. Our ancestors, if they had studied their domesticated wolves, would have noticed a telling difference between dog pedigrees and fire pedigrees. With dogs, like begets like. At least some of what distinguishes one dog from another is handed down by its parents. Of course, some comes in sideways too, from food, disease, and accident. With fires, all the variation comes from the environment. None descends from the progenitive spark. It comes from the quality and dampness of the fuel, from the lie and strength of the wind, from the drawing qualities of the hearth, from the soil, from traces of copper and potassium that add touches of blue-green and lilac to sodium's yellow flame. Unlike a dog, nothing about the quality of an adult fire arrives via the spark that gave it birth. Blue fires don't beget blue fires. Crackling fires don't inherit their crackle from the parent fire that threw up their initiating spark. Fires exhibit reproduction without heredity. The origin of life was the origin of true heredity. We might even say the origin of the first gene. By first gene, I hasten to insist, I don't mean first DNA molecule. Nobody knows whether the first gene was made of DNA, and I bet it wasn't. By first gene, I mean first replicator. A replicator is an entity, for example a molecule, that forms lineages of copies of itself. There will always be errors in copying, so the population will acquire variety. The key to true heredity is that each replicator resembles the one from which it was copied more than it resembles a random member of the population. The origin of the first such replicator was not a probable event, but it only had to happen once. Thereafter, its consequences were automatically self-sustaining, and they eventually gave rise, by Darwinian evolution, to all of life.
A length of DNA, or under certain conditions the related molecule RNA, is a true replicator. So is a computer virus. So is a chain letter. But all these replicators need a complicated apparatus to assist them. DNA needs a cell richly equipped with pre-existing biochemical machinery, highly adapted to read and copy the DNA code. A computer virus needs a computer with some sort of data link to other computers, all designed by human engineers to obey coded instructions. A chain letter needs a good supply of idiots with evolved brains educated at least enough to read. What is unique about the first replicator, the one that sparked life, is that it had no ready supply of anything evolved, designed, or educated. The first replicator worked de novo, ab initio, without precedent and without help other than from the ordinary laws of chemistry. A powerful source of help to a chemical reaction is a catalyst, and catalysis in some form was surely involved in the origin of replication. A catalyst is an agent that speeds up a chemical reaction while not being consumed by it. All biological chemistry consists of catalyzed reactions, the catalysts usually being the large protein molecules called enzymes. A typical enzyme offers the shaped cavities of its three-dimensional form as receptacles for the ingredients of one chemical reaction. It lines them up for each other, enters into temporary chemical liaison with them, matchmakes with an aimed precision that they would be unlikely to discover in open diffusion. Catalysts, by definition, are not consumed in the chemical reaction they boost, but they may be produced. An autocatalytic reaction is a reaction that manufactures its own catalyst. As you can imagine, an autocatalytic reaction is reluctant to start, but once started, it takes off on its own. Like wildfire, indeed, for fire has some of the properties of an autocatalytic reaction. Fire is not strictly a catalyst, but it is self-generating. Chemically, it is an oxidation process that gives off heat and needs heat to push it over a threshold to start. Once started, it continues and spreads as a chain reaction because it generates the heat needed to restart itself. Another famous chain reaction is an atomic explosion, in this case not a chemical reaction but a nuclear one. Heredity began as a lucky initiation of an autocatalytic or otherwise self-regenerating process. It immediately took off and spread like a fire, eventually leading to natural selection and all that was to follow. We too oxidize carbonaceous fuel to generate heat, but we don't burst into flames because we do our oxidation in a controlled way, step by step, trickling the energy into useful channels instead of dissipating it as undisciplined heat. Such controlled chemistry, or metabolism, is as universal a feature of life as heredity. Theories of the origin of life need to account for both heredity and metabolism, but some writers have mistaken the priority. They have sought a theory of metabolism's spontaneous origin and somehow hoped that heredity would follow, like other useful devices. But heredity, as we shall see, is not to be thought of as a useful device. Heredity has to be first on the scene, because before heredity, usefulness itself has no meaning. Without heredity, and hence natural selection, there would have been nothing to be useful for. The very idea of usefulness cannot begin until the natural selection of hereditary information does.
The earliest theories of the origin of life that are taken seriously today are those of A.I. Oparin in Russia and J.B.S. Haldane in England, writing in the 1920s in ignorance of each other. Both emphasized metabolism rather than heredity. Both tumbled to the important fact that the atmosphere of Earth before life would have to be reducing if life were to arise. This rather unhelpful technical term means that the atmosphere lacked free oxygen. Organic compounds, compounds of carbon, when there is free oxygen around, are vulnerable to being burned or otherwise oxidized to carbon dioxide. It seems odd to us, who die within minutes if deprived of oxygen, but life could not originate on any planet with free oxygen in its atmosphere. As I've already explained, oxygen would have been a deadly poison to our earliest ancestors. Everything we know about other planets makes it almost certain that Earth's original atmosphere was a reducing one. Free oxygen came late. It was a polluting waste product of green bacteria, at first swimming free and later incorporated into plant cells. At some point, our ancestors evolved the ability to cope with oxygen, and later came to depend upon it. Incidentally, having said that oxygen is produced by green plants and algae, it is an oversimplification to leave it at that. It is true that plants give off oxygen, but when a plant dies, its decay in chemical reactions equivalent to burning all its carbonaceous materials would use up an amount of oxygen equal to all the oxygen released by that plant during its lifetime. There would therefore be no net gain in atmospheric oxygen, but for one thing. Not all dead plants decay. Some of them are laid down as coal, or equivalents, where they are removed from circulation. if all the fossil fuels in the world were burned by humanity, much of the oxygen in the atmosphere would be replaced by carbon dioxide, restoring the ancient status quo. This is not likely to happen in the near future, but we should not forget that the only reason we have oxygen to breathe is that most of the carbon in the world is tied up underground. We burn it all at our peril. Oxygen atoms were always present in that early atmosphere, but they were not free as oxygen gas. They were tied up in compounds, such as carbon dioxide and water. Carbon today is mostly locked up in living bodies or, a far greater proportion, in rocks such as chalk, limestone and coal, which come from the remains of once living bodies. In Canterbury times, those same carbon atoms would mostly have been in the atmosphere as compound gases, such as carbon dioxide and methane. Nitrogen, today's main atmospheric gas, would in a reducing atmosphere have been compounded with hydrogen as ammonia. Oparin and Haldane realized that a reducing atmosphere would have been friendly to the spontaneous synthesis of simple organic compounds. Here are Haldane's own words, which I quote for the sake of his famous concluding phrase.
Now, when ultraviolet light acts on a mixture of water, carbon dioxide and ammonia, a vast variety of organic substances are made, including sugars and apparently some of the materials from which proteins are built. This fact has been demonstrated in the laboratory by Bailey of Liverpool and his colleagues. In this present world, such substances, if left about, decay. That is to say, they are destroyed by microorganisms. But before the origin of life, they must have accumulated till the primitive oceans reached the consistency of hot, dilute soup.
This was written in 1929, more than 20 years before the much-cited experiment of Miller and Urey, which one would think, from Haldane's account, to have been a kind of repeat of Bailey's. However, E.C.C. Bailey was not concerned with the origin of life. His interest was photosynthesis, and his achievement was to synthesize sugars by beaming ultraviolet rays into water containing dissolved carbon dioxide in the presence of a catalyst such as iron or nickel. It was Haldane, rather than Bailey himself, who, with characteristic brilliance, was anticipating something remarkably like the Miller-Urey experiment, and reading it backwards into Bailey's work. What Miller, under Urey's direction, did was to take two flasks, one above the other, connected by two tubes. The lower flask contained heated water to represent the primeval ocean. The upper flask housed the mocked-up primordial atmosphere, methane, ammonia, water vapor, and hydrogen. Through one of the two tubes, vapor rose from above the heated ocean in the lower flask and was fed into the top of the atmosphere in the upper flask. The other tube returned downwards from atmosphere to ocean. On the way, it passed through a spark chamber, lightning, and a cooling chamber, where vapor condensed to form rain, which replenished the ocean. After only a week in this recycling simulacrum, the ocean had turned yellow-brown, and Miller analyzed its contents. As Haldane would have predicted, it had become a soup of organic compounds, including no fewer than 7 amino acids, the essential building blocks of proteins. Among the 7 were 3, glycine, aspartic acid, and alanine, from the list of 20 found in living things. Later experiments, along Miller's lines but substituting carbon dioxide or carbon monoxide for methane, have achieved similar results. We can draw the robust conclusion that biologically important small molecules, including amino acids, sugars, and significantly the building blocks of DNA and RNA, spontaneously form when various versions of the Oparin-Haldane primitive Earth are simulated in the laboratory. Before Oparin and Haldane, speculators about the origin of life had assumed that the first organisms must have been plants of some kind, perhaps green bacteria. People were used to the idea that life depends upon photosynthesis, the sunlight-driven manufacture of organic compounds accompanied by a release of oxygen. Oparin and Haldane, with their reducing atmosphere, saw plants as arriving on the scene later. Early life arose in a sea of pre-existing organic compounds. There was soup to eat, and no need to photosynthesize, at least until the soup ran out. For Oparin, the vital step was the origin of the first cell. And to be sure, cells, like organisms, have the important property that they never arise spontaneously, but always from other cells. It was pardonable to see the origin of the first cell, metabolizer, as a synonym for the origin of life, rather than the first gene, replicator, as I would. Among more modern theorists with the same bias, the distinguished theoretical physicist Freeman Dyson is aware of it and defends it. The majority of recent theorists, including Leslie Orgel in California, Manfred Eigen and his colleagues in Germany, and Graham Cairn Smith in Scotland, more of a lone maverick but by no means to be written off, give priority to self-replication, both chronologically and in terms of centrality. Rightly so, in my opinion. What would heredity without a cell look like? Don't we have a chicken and egg problem? Certainly so if we take heredity to demand DNA, for DNA can't replicate without a large supporting cast of molecules, including proteins, that can only be made by DNA-coded information. But just because DNA is the main self-replicating molecule we know, it doesn't follow that it is the only one we could imagine, or the only one that has ever existed in nature. Graham Cairn Smith has persuasively argued that the original replicators were inorganic mineral crystals, with DNA a late usurper, stepping into the starring role when life had evolved to the point where such a genetic takeover became possible. I won't expound his case here, partly because I've already given it my best shot in The Blind Watchmaker, but also for a larger reason. Cairn Smith makes the clearest case I have read that replication was primary, and that DNA must have had a forerunner of some kind whose nature is unknown, save that it exhibited true heredity. I think it is a shame if this unassailable part of his case becomes tied in people's minds to his more controversial and speculative case for mineral crystals as the forerunner. I have nothing against the mineral crystals theory, which is why I expounded it before, but what I really want to emphasize is the primacy of replication and the strong likelihood that there was a late takeover by DNA from some forerunner. I can make the point most forcefully by deliberately switching in this book to a different particular theory of what that forerunner might have been. Whatever its ultimate merits as the original replicator, RNA is certainly a better candidate than DNA, and it has been cast as forerunner by a number of theorists in their so-called RNA world. To introduce the RNA world theory, I need to digress on enzymes. If the replicator is the star of life's show, the enzyme is the co-star, more than just supporting cast. Life depends utterly on the virtuoso ability of enzymes to catalyze biochemical reactions in a very fussy way. When I first learned about enzymes at school, the conventional and in my view mistaken wisdom that science should be taught by homely example meant that we spat into water to demonstrate the power of the salivary enzyme amylase to digest starch and make sugar. From this we gained the impression that an enzyme is like a corrosive acid. Biological washing powders, which use enzymes to digest dirt out of clothes, give the same impression. But these are destructive enzymes, working to dismember large molecules into their smaller components. Constructive enzymes are involved in synthesizing large molecules from smaller ingredients, and they do so by behaving as robotic matchmakers, as follows.
The interior of a cell contains a solution of thousands of molecules, atoms, and ions of many different kinds. Pairs of these could combine with each other in almost infinitely varied ways, but on the whole they do not. So there is a huge repertoire of potential chemistry waiting to happen in a cell, but most of it doesn't happen. Hold that in mind while reflecting on the following. A chemistry lab has hundreds of bottles on its shelves, all securely stoppered so their contents don't meet each other unless a chemist desires it, in which case a sample from one bottle is added to a sample from another. You could say that the shelves in a chemistry lab also house a huge repertoire of potential chemistry waiting to happen. And again, most of it doesn't happen. But imagine taking all the bottles off all the shelves and tipping them into a single vat full of water. A preposterous act of scientific vandalism, yet such a vat is pretty much what a living cell is. The hundreds of ingredients of thousands of potential chemical reactions are not kept in separate bottles until required to react together. Instead, they are all mixed up together in the same shared space all the time. But still they wait, largely unreactive, until required to react, as though separated in virtual bottles. There are no virtual bottles, but there are enzymes working as robotic matchmakers, or we might even call them robotic lab assistants. Enzymes discriminate, much as a radio tuner does when it puts a particular wireless set in touch with a particular transmitter, while ignoring the hundreds of other signals simultaneously bombarding its aerial with a babel of carrier frequencies. Suppose there is an important chemical reaction in which ingredient A combines with ingredient B to yield product Z. In a chemistry lab, we achieve this by taking the bottle labelled A off the shelf and the bottle labelled B from another shelf, mixing their contents in a clean flask and providing other necessary conditions, such as heat or stirring. We achieve the specific reaction we want by taking only 2 bottles off the shelf. In the living cell, lots of A molecules and lots of B molecules are among the huge variety of molecules floating around in the water, where they may meet but seldom combine, even if they do. In any case, they are no more likely to meet than thousands of other possible combinations. Now we introduce an enzyme called abscase, which is specifically shaped to catalyze the A plus B equals Z reaction. There are millions of abscase molecules in the cell, each one acting as a robotic lab assistant. Each abscase lab assistant grabs one A molecule, not off a shelf, but floating free in the cell. It then grabs a B molecule as it drifts by. It holds the A firmly in its grip so that it faces in a particular direction, and it holds the B equally firmly so that it abuts the A in just the right position and orientation to bond with the A and make a Z. The enzyme may do other things too, the equivalent of the human lab assistant wielding a stirrer or lighting a Bunsen burner. It may form a temporary chemical alliance with A or B, exchanging atoms or ions that will eventually be paid back, so the enzyme ends up as it started, thus qualifying as a catalyst. The result of all this is that a new Z molecule forms in the shaped grip of the enzyme molecule. The lab assistant then releases the new Z into the water and waits for another A to come by, whereupon it grabs it, and the cycle resumes. If there were no robotic lab assistant, a drifting A would occasionally bump into a drifting B under the right conditions to bond. But this lucky occurrence would be rare, no more common than the occasional chance-matched encounters that either A or B might make with lots of other potential partners. A might bump into C and make Y, or B might bump into D and make X. Small amounts of Y and X are being made all the time by lucky drift. But it is the presence of the lab assistant enzyme abscase that makes all the difference. In the presence of abscase, Z is churned out in, from the cell's point of view, industrial quantities. An enzyme typically multiplies the spontaneous rate of reaction by a factor varying between 1,000,000 and 1,000,000,000,000. If a different enzyme, ACase, were introduced, A would be combined with C instead of B, again at racing conveyor belt speed, to make a lavish supply of Y. It is the very same A molecules we're talking about, not confined to a bottle, but free to combine with either B or C, depending on which enzyme is present to grab them. The production rates of Z and Y will therefore depend on, among other things, how many of each of the 2 rival lab assistants, abscase and ACase, are floating about in the cell. And that depends on which of 2 genes in the nucleus of the cell is turned on. It is, however, a little more complicated than that. Even if a molecule of abscase is present, it may be inactivated. One way that this can happen is that another molecule comes and sits in the active cavity of the enzyme. It is as though the lab assistant's robotic arms were temporarily handcuffed. The handcuffs remind us, by the way, to issue the ritual warning that, as always with metaphors, there is a risk that robotic lab assistant might mislead. An enzyme molecule doesn't actually have arms to reach out and seize ingredients such as A, let alone submit to handcuffs. Instead, it has special zones in its own surface for which A, say, has an affinity, either because of a snug physical fit to a shaped cavity or due to some recondite chemical property. And this affinity can be temporally negated in ways that resemble the calculated throwing of an off switch. Most enzyme molecules are special-purpose machines which make only one product, a sugar, say, or a fat, a purine or a pyrimidine, building blocks of DNA and RNA, or an amino acid. 20 of them are building blocks of natural proteins. But some enzymes are more like programmable machine tools that take in a punched paper tape to determine what they do. Outstanding among these is the ribosome, a large and complicated machine tool constructed from both protein and RNA, which makes proteins themselves. Amino acids, the building blocks of proteins, have already been made by special-purpose enzymes and are floating around in the cell, available to be picked up by the ribosome. The punched paper tape is RNA, specifically messenger RNA, mRNA. The messenger tape, which itself has copied its message from DNA in the genome, feeds into the ribosome and, as it passes through the reading head, the appropriate amino acids are assembled into a protein chain in the order specified by the tape using the genetic code. The coded sequence of DNA, therefore, controls what goes on in the cell. It specifies the sequence of amino acids in each protein, which determines the protein's three-dimensional shape, which in turn gives that protein its particular enzymatic properties. Importantly, the control may be indirect in that, as we saw in the mouse's tail, genes determine which other genes shall be turned on and when. Most genes in any one cell are not switched on. This is why, of all the reactions that could be going on in the vat full of mixed ingredients, only one or two actually do go on at any one time, the ones whose specific lab assistants are active in the cell. After that digression on catalysis and enzymes, we now turn from ordinary catalysis to the special case of autocatalysis, some version of which probably played a key role in the origin of life. Think back to our hypothetical example of molecules A and B combining to make Z under the influence of the enzyme abscase. What if Z itself is its own abscase? I mean, what if the Z molecule happens to have just the right shape and chemical properties to seize one A and one B, bring them together in the correct orientation, and combine them to make a new Z just like itself? In our previous example, we could say that the amount of abscase in the solution would influence the amount of Z produced. But now, if Z actually is one and the same molecule as abscase, we need only a single molecule of Z to seed a chain reaction. The first Z grabs A's and B's and combines them to make more Z's. Then these new Z's grab more A's and B's to make still more Z's, and so on. This is autocatalysis. Under the right conditions, the population of Z molecules will grow exponentially, explosively. This is the kind of thing that sounds promising as an ingredient for the origin of life. But it is all hypothetical. Julius Riebeck and his colleagues at the Scripps Institute in California made it real. They explored some fascinating examples of autocatalysis in real chemistry. In one of their examples, Z was a substance called AATE. The details don't matter. What matters is that the product of the chemical reaction is its own catalyst. The first molecule of AATE is reluctant to form, but once formed, an immediate chain reaction is set in train as more and more AATE synthesizes itself by serving as its own catalyst. As if that weren't enough, this brilliant series of experiments went on to demonstrate true heredity in the sense defined here. Riebeck and his team found a system in which more than one variant of the autocatalyzed substance existed, Each variant catalyzed the synthesis of itself, using its preferred variant of one of the ingredients. This raised the possibility of true competition in a population of entities showing true heredity and an instructively rudimentary form of Darwinian selection. Riebeck's chemistry is highly artificial. Nevertheless, his story beautifully illustrates the principle of autocatalysis, according to which the product of a chemical reaction serves as its own catalyst. It is something like autocatalysis that we need for the origin of life. Could RNA, or something like RNA under the conditions of the early Earth, have autocatalyzed its own synthesis, Riebeck style, and in water instead of chloroform? The problem is a formidable one, as explained by the German Nobel Prize-winning chemist Manfred Eigen. He pointed out that any self-replication process is subject to degradation by copying error, mutation. Imagine a population of replicating entities in which there is a high probability of error in every copying event. If a coded message is to hold its own against the ravages of mutation, at least one member of the population in any one generation must be identical to its parent. If there are 10 code units, letters, in an RNA chain, for example, the average error rate per letter must be less than 1 in 10. We can then expect that at least some members of the offspring generation will have the full complement of 10 correct code letters. But if the error rate is greater, there will be a relentless degradation as the generations go by, simply because of mutation alone, no matter how strong the selection pressure. This is called an error catastrophe. Short chains of RNA, and indeed DNA, can spontaneously self-replicate without an enzyme, but the error rate per letter is far higher than when an enzyme is present. And this means that long before a sufficient length of gene could be built up to make the protein for a working enzyme, the fledgling gene would have been destroyed by mutation. That is the catch-22 of the origin of life. A gene big enough to specify an enzyme would be too big to replicate accurately without the aid of an enzyme of the very kind that it's trying to specify. So the system apparently cannot get started. The solution to the catch-22 that Eigen offers is the theory of the hypercycle. It uses the old principle of divide and rule. The coded information is subdivided into subunits small enough to lie below the threshold of an error catastrophe. Each subunit is a mini-replicator in its own right, and it is small enough for at least one copy to survive in each generation. All the subunits cooperate in some important larger function, large enough to suffer an error catastrophe if catalyzed by a single large chemical rather than being subdivided.
As I have so far described the theory, there is a danger that the whole system would be unstable because some subunits would self-replicate faster than others. This is where the clever part of the theory kicks in. Each subunit flourishes in the presence of the others. More specifically, the production of each is catalyzed by the presence of another, such that they form a cycle of dependency, a hypercycle. This automatically prevents any one element from racing ahead. It cannot do so because it depends on its predecessor in the hypercycle. I'm going to leave the hypercycle theory at this point and return to the suggestion, which is fully compatible with it, that RNA, in the early days when life was just beginning and proteins did not yet exist, might have served as its own catalyst. This is the RNA world theory. To see how plausible it is, we need to look at why proteins are good at being enzymes but bad at being replicators, at why DNA is good at replicating but bad at being an enzyme, and finally, why RNA might just be good enough at both roles to break out of the catch-22. Three-dimensional shape is largely what matters for enzyme activity. Proteins are good at being enzymes because they can assume almost any shape you want in 3 dimensions as an automatic consequence of their amino acid sequence in 1 dimension. It is the chemical affinities of amino acids for other amino acids in different parts of the chain that determines the particular knot into which the protein chain ties itself. So, the 3-dimensional shape of a protein molecule is specified by the one-dimensional sequence of amino acids, and that is itself specified by the one-dimensional sequence of code letters in a gene. In principle, practice is a different matter and formidably difficult. It should be possible to write down a sequence of amino acids that would spontaneously coil itself up into almost any shape you like, not just shapes that make good enzymes, but any arbitrary shape you choose to specify. It is this protean talent that qualifies proteins to act as enzymes. There is a protein capable of selecting any one out of the hundreds of potential chemical reactions that could go on in a cell full of jumbled ingredients. Proteins then make wonderful enzymes capable of tying themselves into knots of any desired shape, but they are lousy replicators. Unlike DNA and RNA, whose component elements have specific pairing rules, the Watson-Crick pairing rules, discovered by those two inspired young men, amino acids have no such rules. DNA, by contrast, is a splendid replicator, but a lousy candidate for the enzyme role in life. This is because, unlike proteins with their near infinite variety of three-dimensional shapes, DNA has only one shape, the famous double helix itself. The double helix is ideally suited to replication because the two sides of the stairway peel easily away from one another, each being then exposed as a template for new letters to join following the Watson-Crick pairing rules. It is not much good for anything else. RNA has some of the virtues of DNA as a replicator and some of the virtues of protein as a versatile shaper of enzymes. The four letters of RNA are sufficiently similar to the four letters of DNA that either set can serve as a template for the other. On the other hand, RNA does not easily form a long double helix, which means that it is somewhat inferior to DNA as a replicator. This is partly because the double helix system lends itself to proof correction. When the DNA double helix splits and each single helix immediately serves as a template for its complement, errors can instantly be spotted and corrected. Each daughter chain remains attached to its parent, and comparison between the two permits instant error detection. Proofreading based on this principle reduces mutation rates to the order of 1 in a billion, which is what makes large genomes like ours possible. RNA, lacking this kind of proofreading, has mutation rates that are thousands of times greater than DNA. This means that only simple organisms with small genomes, such as some viruses, can use RNA as their primary replicator. But the lack of a double helix structure has its upside as well as its downside. Because the RNA chain doesn't spend all its time paired with its complementary chain, but breaks away from the complement as soon as it is formed, it is free to tie itself in knots like a protein. Just as the protein does it by virtue of the chemical affinities of amino acids for other amino acids in different parts of the same chain, RNA does it using the ordinary Watson-Crick base pairing rules, the same ones as are used to make copies of RNA. Putting it another way, lacking a partner chain to pair with in a double helix like DNA, RNA is free to pair with odd bits of itself. RNA finds small stretches of itself with which it can pair, either in a miniature double helix or in some other shape. The pairing rules insist that these stretches have to be going in opposite directions. An RNA chain, therefore, has a tendency to fall into a series of hairpin bends. The repertoire of three-dimensional shapes into which an RNA molecule is capable of throwing itself may not be as great as the repertoire of large protein molecules, but it is large enough to encourage the thought that RNA might furnish a versatile armory of enzymes. And to be sure, many RNA enzymes called ribozymes have been discovered. The conclusion is that RNA has some of the replicator virtues of DNA and some of the enzyme virtues of proteins. Maybe before the coming of DNA, the arch replicator, and before the coming of proteins, the arch catalysts, there was a world in which RNA alone had enough of both virtues to stand in for both experts. Perhaps an RNA fire ignited itself in the original world, and then later started to make proteins that turned around and helped synthesize RNA, and later DNA too, which took over as the dominant replicator. That is the hope of the RNA world theory. Research continues, and I expect exciting results. But now I want to switch to a newly fashionable line of thought, fully compatible with the RNA world and with many others among the current theories of the origin of life. What is new is the suggested location in which the crucial events first took place. Not warm little pond, but hot deep rock, an exciting theory which amounts to this. Our pilgrims, to complete their journey and locate their Canterbury, are now going to have to bore deep underground into the primordial rock. The main inspirer of the theory is another maverick, Thomas Gold, originally an astronomer, but versatile enough to deserve the now rare accolade general scientist, and distinguished enough to have been elected to both the Royal Society of London and the American National Academy of Sciences.
Gold believes that our emphasis on the sun as energetic prime mover of life may be misplaced. Perhaps we have yet again been misled by what happens to be familiar, yet again assigned to ourselves and our kind of life a centrality in the scheme of things that we do not deserve. There was a time when textbooks asserted that all life depended ultimately on sunlight. Then, in 1977, the startling discovery was made that volcanic vents on the floor of deep oceans support a strange community of creatures living without benefit of sunlight. Heat from red-hot lava raises the water temperature to more than 100 degrees Celsius, still well below boiling point at the colossal pressures of those depths. The surrounding water is very cold, and the temperature gradient drives various kinds of bacterial metabolism. These thermophile bacteria, including sulfur bacteria, who make use of hydrogen sulfide streaming from the volcanic vents, constitute the base of elaborate food chains, higher links of which include blood-red tube worms up to 3 meters long, limpets, mussels, starfish, barnacles, white crabs, prawns, and fish capable of thriving at 80 degrees Celsius. There are bacteria, as we have seen, which can take such Hadean temperatures in their stride, but no other animal is known to do so, and these polychaete worms have accordingly been dubbed Pompeii worms. Some of the sulfur bacteria are given house room by animals, for example by mussels, and by the huge tube worms, who take special biochemical steps using hemoglobin, hence their blood-red colour, to feed sulfide to their own bacteria. These colonies of life, based on bacterial extraction of energy from hot volcanic vents, astonished everybody, first by their very existence, and then by their abundant richness, which contrasted startlingly with the near-desert conditions of the surrounding sea bottom. Even after this sensational discovery, most biologists continue to believe that life is centered on the sun. The creatures of the deep-sea smoker communities, fascinating though they might be, are assumed by most of us to be a rare and unrepresentative aberration. Gold believes otherwise. He thinks hot, dark, high-pressure depths are where life fundamentally belongs and where it originated, not necessarily in the sea, but perhaps in the rocks deep underground. We who live at the surface, in the light and the cool and the fresh air, we are the anomalous aberrations. He points out that hopanoids, organic molecules made in bacterial cell walls, are ubiquitous in rocks, and quotes an authoritative estimate of between 10 trillion and 100 trillion tons of hopanoids in the rocks of the world. This comfortably exceeds the trillion tons or so of organic carbon in surface-dwelling life. Gold notes that the rocks are seamed with cracks and fissures, which, though small to our eyes, provide more than 1 billion trillion cubic centimeters of hot, wet space suitable for life on the bacterial scale of existence. Heat energy and the chemicals of the rocks themselves will be enough to sustain bacteria in huge numbers. Gold notes that many bacteria thrive at temperatures up to 110 degrees Celsius, and this would permit them to live down to depths of between 5 and 10 kilometers, a distance that would take them less than 1,000 years to travel. It is impossible to verify his estimate, but he thinks the biomass of bacteria in the hot, deep rocks might exceed the biomass of the surface sun-based life with which we are familiar.
Turning to the question of the origin of life, Gould and others have pointed out that thermophily, love of high temperatures, is not a rare oddity among bacteria and archaeans. It is common, so common and so widely distributed around bacterial family trees that it might well be the primitive state from which our familiar cool forms of life have evolved. With respect to both chemistry and temperature, the conditions on the surface of the primitive Earth, some scientists call it the Hadean Age, were more like those in Gould's hot deep rocks than they were like today's surface conditions. A persuasive case can indeed be made that when we dig down into the rocks, we are digging backwards in time and rediscovering something like the conditions of life's scalding Canterbury. The idea has been further championed recently by the Anglo-Australian physicist Paul Davies, whose book, The Fifth Miracle, summarizes new evidence discovered since Gould's paper of 1992. Various drilling samples have been found to contain hyperthermophile bacteria, alive and reproducing, amid scrupulous precautions to preclude contamination from the surface. Some of these bacteria have been successfully cultured in a modified pressure cooker. Davies, like Gould, believes life may have originated deep underground, and that the bacteria which still live there may be relatively unchanged relics of our remote ancestors. This idea is especially appealing for our pilgrimage because it offers us the hope of meeting something like the earliest bacteria, rather than the more familiar bacteria modified for modern conditions of light, cold, and oxygen. Having endured ridicule at first, the hot, deep rock theory of the origin of life is now verging on the positively fashionable. Whether it will turn out to be right must await more research, but I confess to hoping that it will. There are many other theories that I have not gone into. Maybe one day we shall reach some sort of definite consensus on the origin of life. If so, I doubt if it will be supported by direct evidence, because I suspect that it has all been obliterated. Rather, it will be accepted because somebody produces a theory so elegant that, as the great American physicist John Archibald Wheeler said in another context,
"We will grasp the central idea of it all as so simple, so beautiful, so compelling, that we will say to each other, 'Oh, how could it have been otherwise? How could we all have been so blind for so long?'"
If that isn't how we finally realize we know the answer to the riddle of life's origin, I don't think we shall ever know it.
The host's return. The genial host, having guided Chaucer and the other pilgrims from London to Canterbury, and stood impresario to their tales, turned around and led them straight back to London.
If I now return to the present, it must be alone. For to presume upon evolution's following the same forward course twice would be to deny the rationale of our backward journey. Evolution was never aimed at any particular end point. Our backwards pilgrimage has been a series of swelling mergers as we were swallowed up in ever more inclusive groupings, the apes, the primates, the mammals, the vertebrates, the deuterostomes, the animals, and so on back to the arch-ancestor of all life. If we turn around and move forward now, we cannot retrace our steps. That would imply that evolution, were it to be rerun, would follow the same course, putting those same mergers into reverse gear in the form of splits. The stream of life would branch in all the right places. Photosynthesis and an oxygen-based metabolism would be rediscovered. The eukaryotic cell would reconstitute itself. Cells would club together in neo-metazoan bodies. There would be a new split between plants on the one hand and animals plus fungi on the other, a new split between protostomes and deuterostomes. The backbone would be rediscovered, and so would eyes, ears, limbs, nervous systems. Eventually, a swollen-brained biped would emerge, with skilled hands guided by forward-looking eyes, culminating in the proverbial cricket team to beat the Australians. My disavowal of aimed evolution underlay my original choice to do history backwards. And yet, in my opening lines, I confessed to an ear for a rhyme that would lead me into cautious flirtation with recurring patterns, with lawfulness and forward directionality in evolution. So, although my return as host will not be a retracing of steps, I shall be publicly wondering whether something a little bit like a retracing might not be appropriate. The American theoretical biologist Stuart Kauffman put the question well in a 1985 article.
One way to underline our current ignorance is to ask, if evolution were to recur from the Precambrian, when early eukaryotic cells had already been formed, what organisms in 1 or 2 billion years might be like. And if the experiment were repeated myriads of times, what properties of organisms would arise repeatedly, what properties would be rare, which properties were easy for evolution to happen upon, which were hard. A central failure of our current thinking about evolution is that it has not led us to pose such questions, although the answers might in fact yield deep insight into the expected character of organisms.
I especially like Kauffman's statistical proviso. He envisages not just one thought experiment, but a statistical sample of thought experiments in quest of general laws of life, as opposed to local manifestations of particular lives. The Kauffman question is akin to the science fiction question of what life on other planets might be like, except that on other planets the starting and prevailing conditions would be different. On a large planet, gravity would impose a whole new set of selection pressures. Animals the size of spiders could not have spidery limbs, they'd break under the weight, but would need the support of stout vertical columns like the tree trunks on which our elephants stand. Conversely, on a smaller planet, animals the size of elephants but of gossamer build could skitter and leap over the surface like jumping spiders. Those expectations about body build will apply to the whole statistical sample of high-gravity worlds and the whole statistical sample of low-gravity worlds.
Gravity is a given condition of a planet which life cannot influence. So is its distance from its central star. So is its speed of rotation, which determines day length. So is the tilt of its axis, which on a planet like ours, with its near-circular orbit, is the main determinant of seasons. On a planet with a far-from-circular orbit like Pluto, the dramatically changing distance from the central star will be a much more significant determiner of seasonality. The presence, distance, mass and orbit of a moon or moons exert a subtle but strong influence on life via the tides. All these factors are givens, uninfluenced by life, and therefore to be treated as constant in successive reruns of the Kauffman thought experiment. How shall we set about answering the Kauffman question? What would life be like if the tape were rerun a statistical number of times? Immediately we can recognize a whole family of Kauffman questions of steadily increasing difficulty. Kauffman chose to reset the clock at the moment when the eukaryotic cell was assembled from its bacterial components. But we could imagine restarting the process 2 or 3 eons earlier with the origin of life itself. Or at the other extreme, we can restart the clock much later, say at Concestor 1, our split from the chimpanzees, and ask whether the hominids would, in a statistically significant number of reruns, given that life had reached Concestor 1, have evolved bipedality, brain enlargement, language, civilization, and baseball. In between, there is a Kauffman question for the origin of the mammals, for the origin of the vertebrates, and any number of other Kauffman questions. Short of pure speculation, does the history of life, as it actually happened, provide anything approaching a natural Kauffman experiment to guide us?
Yes, it does. We met several natural experiments throughout our pilgrimage. By happy accidents of prolonged geographical isolation, Australia, Madagascar, South America, even Africa, furnish us with approximate reruns of major episodes of evolution. These land masses were isolated from each other and from the rest of the world for significant parts of the period after the dinosaurs disappeared, when the mammal group displayed most of its evolutionary creativity. The isolation was not total, but was sufficient to foster the lemurs in Madagascar and the ancient and diverse radiation of Afrotheria in Africa. In the case of South America, we have distinguished 3 separate foundations of mammals, with long periods of isolation in between. Australia provides the most perfect conditions for this kind of natural experiment. Its isolation was nearly perfect for much of the period in question, and it began with a very small, possibly single, inoculum of marsupials. As I look at these natural experiments, mostly I'm impressed by how similarly evolution turns out when it is allowed to run twice. Among marsupials, Thylacinus is uncannily similar to a dog, Notorictes to a mole, Petaurus to flying squirrels, Thylacis milis to the saber-tooths, and to various false saber-tooths among the placental carnivores. The differences are instructive too. Kangaroos are hopping antelope substitutes. Bipedal hopping, when perfected at the end of a line of evolutionary progression, may be as impressively fast as quadrupedal galloping. But the two gaits are radically different from each other in ways that have wrought major changes in the whole anatomy. Presumably, at some ancestral parting of the ways, either of the two experimental lineages could have followed the route of perfecting bipedal hopping, and either could have perfected quadrupedal galloping. As it happens, possibly for almost accidental reasons originally, the kangaroos hopped one way and the antelopes galloped the other. We now marvel at the downstream divergences between the end products. The mammals underwent their disparate evolutionary radiations at roughly the same time as each other on different land masses. The vacuum left by the dinosaurs freed them to do so. But the dinosaurs in their time had similar evolutionary radiations, although with notable omissions. For example, I can't get an answer to my question of why there seem to have been no dinosaur moles. And before the dinosaurs there were yet other multiple parallels, notably among the mammal-like reptiles, and these too culminated in similar ranges of types. When I give public lectures, I always try to answer questions at the end. The commonest question by far is, what might humans evolve into next? My interlocutor always seems touchingly to imagine it is a fresh and original question, and my heart sinks every time. For it is a question that any prudent evolutionist will evade. You cannot, in detail, forecast the future evolution of any species except to say that statistically the great majority of species have gone extinct. But although we cannot forecast the future of any species, say 20 million years hence, we can forecast the general range of ecological types that will be around. There will be herbivores and carnivores, grazers and browsers, meat-eaters, fish-eaters, and insect-eaters. These dietary forecasts themselves presuppose that in 20 million years there will still be foods corresponding to the definitions. Browsers presuppose the continued existence of trees. Insectivores presuppose insects, or anyway small leggy invertebrates, doo-doos, to employ that useful technical term from Africa. Within each category, herbivores, carnivores, and so on, there will be a range of sizes. There will be runners, flyers, swimmers, climbers, and burrowers. The species won't be exactly the same as the ones we see today, or the parallel ones that evolved in Australia or South America, or the dinosaur equivalents, or the mammal-like reptile equivalents. But there will be a similar range of types, making their livings in a similar range of ways. If, during the next 20 million years, there is a major catastrophe and a mass extinction comparable to the end of the dinosaurs, we can expect the range of ecotypes to be drawn from the new ancestral starting points and, notwithstanding my speculation about rodents at Rendezvous 10, it might be quite hard to guess which of today's animals will provide those starting points. Admittedly, all this concerns quite recent evolution, not so prolonged a rerun as Kaufmann imagined. But these recent reruns can surely teach us some lessons about the inherent reproducibility of evolution. If early evolution ran along similar lines to later evolution, those lessons might amount to general principles. My hunch is that the principles we learn from recent evolution since the decease of the dinosaurs probably hold good at least back to the Cambrian, and probably back to the origin of the eukaryotic cell. I have a hunch that the parallelism of mammal radiations in Australia, Madagascar, South America, Africa, and Asia may provide a sort of template for answering Kaufmann questions for much older starting points, such as the one he chose, the origin of the eukaryotic cell. Earlier than that landmark event, confidence evaporates. My colleague Mark Ridley in Mendel's Demon suspects that the origin of eukaryotic complexity was a massively improbable event, perhaps even more improbable than the origin of life itself. Influenced by Ridley, my bet is that most rerun thought experiments that start with the origin of life will not make it into the eukaryocracy. We don't have to rely on geographical separation, as in the Australian natural experiment, to study convergence. We can think of the experiment of evolution being rerun not from the same starting point in different geographical areas, but from different starting points, very possibly in the same geographical area. Convergence in animals so unrelated to each other that what they tell us has nothing to do with geographical separation. It has been estimated that the eye has evolved independently between 40 and 60 times around the animal kingdom. This inspired my chapter called The 40-Fold Path to Enlightenment in Climbing Mount Improbable, so I won't repeat myself here, except to say that Professor Michael Land of Sussex University, our leading expert on the comparative zoology of eyes, recognizes 9 independent principles of optical mechanism, each of which has evolved more than once.
It seems that life, at least as we know it on this planet, is almost indecently eager to evolve eyes. We can confidently predict that a statistical sample of Kaufman reruns would culminate in eyes, and not just eyes, but compound eyes like those of an insect, a prawn, or a trilobite, and camera eyes like ours or a squid's with color vision and with mechanisms for fine-tuning the focus and the aperture. Also, very probably, parabolic reflector eyes like those of a limpet, and pinhole eyes like those of Nautilus, the latter-day ammonite-like mollusk in its floating coiled shell, whom we met at Rendezvous 26. And if there is life on other planets around the universe, it is a good bet that there will also be eyes, based on the same range of optical principles as we know on this planet. There are only so many ways to make an eye, and life as we know it may well have found them all. We can do the same kind of count for other adaptations. Echolocation, the trick of emitting sound pulses and navigating by accurate timing of the echoes, has evolved at least 4 times, in bats, toothed whales, oilbirds, and cave swiftlets. Not as many times as the eye, but still often enough to make us think it not too unlikely that, if the conditions are right, it will evolve. Very probably, too, reruns of evolution would rediscover the same specific principles, the same tricks for confronting difficulties. How do we know that something like the eye or echolocation has evolved independently? By looking at the family tree. Relatives of oilbirds and of cave swiftlets don't do echolocation. Oilbirds and cave swiftlets have separately taken up life in caves. We know they have evolved the technology independently of bats and whales, since nothing else in the surrounding family tree does it. Different groups of bats may have evolved echolocation more than once independently. We don't know how many more times echolocation evolved. Some shrews and seals have a rudimentary form of the skill, and some blind human individuals have learned it. Did pterodactyls do it? Since there is a good living to be made flying at night, and since bats weren't around in those days, it's not unlikely. The same goes for ichthyosaurs. They looked very like dolphins and presumably made their living in a similar way. Since dolphins make heavy use of echolocation, it is reasonable to wonder whether ichthyosaurs did too in the days before dolphins. There is no direct evidence, and we must remain open-minded. One point against, ichthyosaurs had extraordinarily big eyes. It is one of their most conspicuous features, which might suggest that they relied upon vision instead of echolocation. Dolphins have relatively small eyes, and one of their most conspicuous features, the rounded bump or melon above the beak, acts as an acoustic lens, focusing sound into a narrow beam projected in front of the animal like a searchlight.
Like any zoologist, I can search my mental database of the animal kingdom and come up with an estimated answer to questions of the form, how many times has X evolved independently? It would make a good research project to do the counts more systematically. Presumably some X's will come up with a many times answer, as with eyes, or several times, as with echolocation. others only once, or even never, although I have to say it is surprisingly difficult to find examples of these. And the differences could be interesting. I suspect that we'd find certain potential evolutionary pathways which life is eager to go down. Other pathways have more resistance. In Climbing Mount Improbable, I developed the analogy of a huge museum of all life, both real and conceivable, with corridors going off in many dimensions and representing evolutionary change, again, both real and conceivable. Some of these corridors were wide open, almost beckoning. Others were blocked off by barriers that were hard or even impossible to surmount. Evolution repeatedly races down the easy corridors, and just occasionally and unexpectedly leaps one of the hard barriers. Let's now go quickly through some more examples where it might be worth making a systematic count of how many times X has evolved. The venomous sting, injecting poison hypodermically through a sharp-pointed tube, has evolved at least 10 times independently, in jellyfish and their relatives, spiders, scorpions, centipedes, insects, mollusks, cone shells, snakes, the shark group, stingrays, bony fish, stonefish, mammals, male platypus, and plants, stinging nettles. It's a good bet that venom, including hypodermic injection, would evolve in reruns. Sound production for social purposes has evolved independently in birds, mammals, crickets and grasshoppers, cicadas, fish and frogs. Electrolocation, the use of weak electric fields for navigation, has evolved several times, as we saw in the duckbill's tail. So has the probably subsequent use of electric currents as weapons. The physics of electricity is the same on all worlds, and we could bet with some confidence on repeated evolution of creatures that exploit electricity for both navigational and offensive purposes. True flapping flight, as opposed to passive gliding or parachuting, seems to have evolved 4 times in insects, pterosaurs, bats and birds. Parachuting and gliding of various kinds have evolved many times, maybe hundreds of times independently, and may well be the evolutionary precursor to true flight. Examples include lizards, frogs, snakes, flying fish, squids, cologos, marsupials, and rodents twice. I'd put a lot of money on gliders turning up in Kaufmannian reruns, and a reasonable sum on true flapping flyers. Jet propulsion may have evolved twice. Cephalopod mollusks do it at high speed in the case of squids. The other example I can think of is also a mollusk, but it is not high speed. Scallops mostly live on the sea bottom, but occasionally they swim. They rhythmically open and close their 2 shells, like a pair of snapping castanets. You'd think, I would, that this would propel them backwards in a direction opposite to the snapping. In fact, they move forwards, as though biting their way into the water. How can this be? The answer is that the snapping movements pump water through a pair of apertures behind the hinge. These 2 jets propel the animal forwards. The effect is so counterintuitive, it is almost comical. How about things that have evolved only once, or not at all? As we learn from the rhizobium's tail, the wheel, with a true freely rotating bearing, seems to have evolved only once, in bacteria, before being finally invented in human technology. Language, too, has apparently evolved only in us, that is to say, at least 40 times less often than eyes. It is surprisingly hard to think of good ideas that have evolved only once. I put the challenge to my Oxford colleague, the entomologist and naturalist George McGavin, and he came up with a nice list, but still a short one compared with the list of things that have evolved many times. Bombardier beetles of the genus Brachinus are unique, in Dr. McGavin's experience, in mixing chemicals to make an explosion. The ingredients are made and held in separate, obviously, glands. When danger threatens, they are squirted into a chamber near the rear end of the beetle, where they explode, forcing noxious, caustic and boiling hot liquid out through a directed nozzle at the enemy. The case is well known to creationists who love it. They think it is self-evidently impossible to evolve by gradual degrees, because the intermediate stages would all explode. I enjoyed demonstrating the error of this argument during my Royal Institution Christmas lectures for children, shown on BBC television in 1991. Donning a World War II helmet and inviting nervous members of the audience to leave, I mixed hydroquinone and hydrogen peroxide, the two ingredients of the Bombardier explosion. Nothing happened. It didn't even get warm. The explosion requires a catalyst. I raised the concentration of catalyst gradually, which steadily increased the whoosh to a satisfactory climax. In nature, the beetle provides the catalyst, and would have had no difficulty in gradually increasing the dose over evolutionary time.
Next in the McGavin list is the archerfish, which may be unique in shooting a missile to knock prey down from a distance. It comes to the surface of the water and spits a mouthful of water at a perched insect, knocking it down into the water where it eats it. The other possible candidate for a knocking down predator might be an antlion. Antlions are insect larvae of the order Neuroptera. Like many larvae, they look nothing like their adults. With their huge jaws, they would be good casting for a horror film. Each antlion lurks in the sand just below the surface at the base of a conical pit trap, which it digs itself. It digs by flicking sand vigorously outwards from the centre. This causes miniature landslides down the sides of the pit, and the laws of physics do the rest, neatly shaping the cone. Prey, usually ants, fall into the pit and slide down the steep sides into the antlion's jaws. The possible point of resemblance to the archerfish is that prey don't fall only passively. They're sometimes knocked down into the pit by the particles of sand. These are not, however, aimed with the precision of an archerfish's spit, which is guided with devastating accuracy by binocularly focused eyes. Spitting spiders are a bit different again. Lacking the fleetness of a wolf spider or the net of a web spider, the spitting spider chucks a venomous glue some distance towards its prey, pinning it to the ground until the spider arrives and bites it to death. This is different from the archerfish technique of knocking prey down. Various animals, for example, venom-spitting cobras, spit defensively, not to catch prey. The bolus spider, Mastophora, is different again, and is probably another unique case. It could be said to throw a missile at prey, moths attracted by the fake sexual scent of a female moth which the spider synthesizes. But the missile, a blob of silk, is attached to a thread of silk which the spider whirls around like a lasso or bola and reels in. McGavin's next candidate for an evolutionary one-off is a beauty. It is the diving bell spider, Agarineter aquatica. This spider lives and hunts entirely underwater, but like dolphins, dugongs, turtles, freshwater snails, and other land animals that have returned to water, it needs to breathe air. Unlike all those other exiles, however, Agarineter constructs its own diving bell. It spins it of silk. Silk is the universal solution to any spider problem, attached to an underwater plant. The spider goes to the surface to collect air, which it carries in the same way as some water bugs in a layer trapped by body hairs. But unlike the bugs, which just carry the air like a scuba cylinder wherever they go, the spider takes it to its diving bell, where it unloads it to replenish the supply. The spider sits in the diving bell watching for prey, and it stores and eats prey there once caught. But George McGavin's champion example of a one-off is the larva of an African horsefly called Tabanus. Predictably, in Africa, the pools of water in which the larvae live and feed eventually dry up. Each larva buries itself in the mud and pupates. The adult fly emerges from the baked mud and flies off to feed on blood, eventually to complete the cycle by laying eggs in pools of water when the rains return. The buried larva is vulnerable to a predictable danger. As mud dries out, it cracks, and there is a risk that a crack will tear right across the grub's refuge. It could theoretically save itself if it could somehow engineer a way for any crack that approaches it to be diverted around it instead. And it does indeed achieve this in a truly wonderful and probably unique manner. Before burying itself in its own pupation chamber, It first corkscrews its way down into the mud in a spiral. It then corkscrews its way back to the surface in an opposite spiral. Finally, it dives into the mud straight down the centre between the two spirals, and that is its resting place through the bad times until the water returns.
Now, you see what this means. The lava is encased in a cylinder of mud whose circular boundary has been weakened in advance by the preliminary spiral burrowing. This means that when a crack snakes across the drying mud, if it hits the edge of the cylindrical column, instead of cutting straight across the middle, it goes instead in a curved bypass around the edge of the cylinder, and the lava is spared. It is just like the perforations around a stamp that stop you tearing the stamp across. Dr. McGavin believes this ingenious trick is literally unique to this one genus of horsefly. But are there any good ideas that have never evolved by natural selection? As far as I know, no animal on this planet has ever evolved an organ for the transmission or reception of radio waves for long-distance communication. The use of fire is another example. Human experience shows how powerfully useful it can be. There are some plants whose seeds need fire to germinate, but I don't think that constitutes using it in the same sense as, for example, electric eels use electricity. The use of metal for skeletal purposes is another example of a good idea that has never evolved except in human artifacts. Presumably, it is hard to achieve without fire. This kind of comparative exercise, counting up which things evolve often, which seldom, when we do it alongside the geographical comparisons discussed earlier, might enable us to predict things about life outside this planet, as well as to guess the likely outcome of Kaufman-style thought experiments in rerunning evolution. We positively expect eyes, ears, wings, and electric organs, but perhaps not bombardier beetle explosions or archerfish water bullets. Those biologists who could be said to take their lead from the late Stephen Jay Gould regard all of evolution, including post-Cambrian evolution, as massively contingent, lucky, unlikely to be repeated in a Kaufman rerun. Calling it rewinding the tape of evolution, Gould independently evolved Kaufman's thought experiment. The chance of anything remotely resembling humans on a second rerun is widely seen as vanishingly small, and Gould voiced it persuasively in Wonderful Life. It was this orthodoxy that led me to the cautious self-denying ordinance of my opening chapter, led me indeed to undertake my backwards pilgrimage, and now leads me to forsake my pilgrim companions at Canterbury and return alone. And yet, I have long wondered whether the hectoring orthodoxy of contingency might have gone too far. My review of Gould's Full House, reprinted in A Devil's Chaplain, defended the unpopular notion of progress in evolution, not progress towards humanity, Darwin forfend, but progress in directions that are at least predictable enough to justify the word. As I shall argue in a moment, the cumulative build-up of complex adaptations like eyes strongly suggests a version of progress, especially when coupled in imagination with some of the wonderful products of convergent evolution. Convergent evolution also inspired the Cambridge geologist Simon Conway Morris, whose provocative book, Life's Solution: Inevitable Humans in a Lonely Universe, presents exactly the opposite case to Gould's contingency. Conway Morris means his subtitle in a sense which is not far from literal. He really thinks that a rerun of evolution would result in a second coming of man, or something extremely close to man. And for such an unpopular thesis, he mounts a defiantly courageous case. The two witnesses that he repeatedly calls are convergence and constraint. Convergence we have met again and again through this book, including in this chapter. Similar problems call forth similar solutions, not just twice or three times, but in many cases dozens of times. I thought I was pretty extreme in my enthusiasm for convergent evolution, but I've met my match in Conway Morris, who presents a stunning array of examples, many of which I had not met before. But whereas I usually explain convergence by invoking similar selection pressures, Conway Morris adds the testimony of his second witness, constraint. The materials of life and the processes of embryonic development allow only a limited range of solutions to a particular problem. Given any particular evolutionary starting situation, there is only a limited number of ways out of the box. So, if two reruns of a Kaufman experiment encounter anything like similar selection pressures, developmental constraints will enhance the tendency to arrive at the same solution. You can see how a skilled advocate could deploy these two witnesses in defense of the daring belief that a rerun of evolution would be positively likely to converge on a large-brained biped with two skilled hands, forward-pointing camera eyes, and other human features. Unfortunately, it has only happened once on this planet, but I suppose there has to be a first time. I admit that I was impressed by Conway Morris's parallel case for the predictability of the evolution of insects. Among the defining features of insects are the following: an articulated exoskeleton, compound eyes, a characteristic six-legged gait whereby 3 of the 6 walking legs are always on the ground, and thereby define a triangle, 2 legs on one side, 1 leg on the other, which keeps the animal stable. Respiratory tubes, known as tracheae, that serve to bring oxygen into the interior of the animal via special openings, spiracles, along the side of the body, and to complete the list of evolutionary peculiarities, the repeated, 11 times independently, evolution of complex eusocial colonies, as in the honeybees. All pretty strange, all one-offs in the great lottery of life? On the contrary, all are convergent. Conway Morris goes through his list, showing that each item has evolved more than once in different parts of the animal kingdom, in many cases several times, including several times independently in insects themselves. If nature finds it so easy to evolve the component parts of insecthood separately, it is not all that implausible that the whole collection should evolve twice. I am tempted by Conway Morris's belief that we should stop thinking of convergent evolution as a colourful rarity to be remarked and marvelled at when we find it. Perhaps we should come to see it as a norm, exceptions to which are occasions for surprise. For example, true syntactic language seems to be unique to one species, our own. Perhaps this is one thing that a re-evolved brainy biped would lack. In my opening chapter, The Conceit of Hindsight, I listened to warnings against seeking patterns, rhymes, or reasons in evolution, but said that I would cautiously flirt with them. The host's return has provided an opportunity to sweep over the whole course of evolution in the forward direction and see what patterns we can descry. The idea that all evolution was aimed at producing Homo sapiens was certainly well rejected, and nothing we have seen on our backwards journey reinstates it. Even Conway Morris claims only that something approximately similar to our kind of animal is one of several outcomes, others being insects, for example, that we would expect to see recurring if evolution were rerun again and again.
What other patterns or rhymes do we discern if we look over our long pilgrimage? Is evolution progressive?
There is at least one reasonable definition of progress under which I would defend it. I need to work up to this. To begin with, progress can be defined in a weak, minimalist sense, with no value judgment, as the predictable continuation into the future of trends from the past. The growth of a child is progressive in that whatever trends we observe in weight, height, and other measurements over 1 year continue in the next. There is no value judgment in this weak definition of progress. Growth of a cancer is progressive in exactly the same weak sense. So also is shrinkage of a cancer under therapy. What then would not be progressive in the weak sense? Random, aimless fluctuation. The tumour grows a little, shrinks somewhat, grows a lot, shrinks a bit, grows a little, shrinks a lot, and so on. A progressive trend is one in which there are no reversals, or if there are reversals, they are outnumbered and outweighed by movement in the dominant direction. In a sequence of dated fossils, progress in this value-neutral sense would mean simply that whatever anatomical trend you see as you go from early to intermediate is continued as a trend from intermediate to late. I now need to clarify the distinction between value-neutral progress and value-laden progress. Progress in the weak sense just defined is value-neutral, but most people think of progress as value-laden. The doctor, reporting shrinkage of the tumour in response to chemotherapy, announces with satisfaction, "We are making progress." Doctors do not, on the whole, look at an X-ray of a swelling tumour with numerous secondaries and announce that the tumour is making progress, though they easily could. That would be value-laden, but with negative value. Progressive in human political or social affairs usually refers to a trend in a direction which the speaker considers desirable. We look over human history and regard the following trends as progressive: abolition of slavery, widening of the franchise, reduction of discrimination by sex or race, reduction of disease and poverty, increase in public hygiene, reduction in atmospheric pollution, increase in education. A person of certain political views might see at least some of these trends as negatively value-laden and yearn nostalgically for the days before women had the vote or were allowed into the club dining room. But the trends are still progressive in more than just the weak, minimalist, value-neutral sense we first defined. They are progressive according to some specified value system, even if it is not a value system you or I would share.
Astonishingly, it is only 100 years since the Wright brothers first achieved powered flight in a heavier-than-air machine. The history of aviation since 1903 has been unmistakably progressive and at amazing speed. Only 66 years later, men walked on the moon. The fact that they no longer do so, and the fact that the only supersonic passenger service has just been discontinued, are economically dictated, temporary reversals in an overall trend that is unquestionably progressive. Aircraft are getting faster, and they are progressing in all sorts of other ways at the same time. Much of this progress does not chime with everyone's values, for example those unlucky enough to live under a flight path. And much of the progress in aviation is driven by military needs. But nobody would deny the existence of a coherently expressible set of values which at least some sane people might hold, according to which even fighters, bombers, and guided missiles have progressively improved over the whole century since the Wrights. The same could be said of all other forms of transport, indeed other forms of technology, including, more than anything else, computers.
Evolution exhibits progress not just in the weak value-free sense. There are episodes of progress that are value-laden according to at least some entirely plausible value systems. Since we're talking armaments, it is a good moment to note that the most familiar examples come out of arms races between predators and prey. The first use of the arms race listed in the Oxford English Dictionary is from Hansard, written Proceedings of the House of Commons, for 1936.
This House cannot agree to a policy which in fact seeks security in national armaments alone and intensifies the ruinous arms race between the nations, inevitably leading to war.
The Daily Express in 1937, under the headline Arms Race Worry, said, "All were worried at the armament race." It was not long before the theme found its way into the literature of evolutionary biology. Hugh Cott, in his classic Adaptive Coloration in Animals, published in 1940, deep in the Second World War, wrote,
"Before asserting that the deceptive appearance of a grasshopper or butterfly is unnecessarily detailed, we must first ascertain what are the powers of perception and discrimination of the insect's natural enemies. Not to do so is like asserting that the armor of a battle-cruiser is too heavy, or the range of her guns too great, without inquiring into the nature and effectiveness of the enemy's armament. The fact is that in the primeval struggle of the jungle, as in the refinements of civilized warfare, we see in progress a great evolutionary armament race, whose results for defense are manifested in such devices as speed, alertness, armor, spinescence, burrowing habits, nocturnal habits, poisonous secretions, nauseous taste, and procryptic, aposematic, and mimetic coloration, and for offense in such counter-attributes as speed, surprise, ambush, allurement, visual acuity, claws, teeth, stings, poison fangs, and anticryptic and alluring coloration. Just as greater speed in the pursued has developed in relation to increased speed in the pursuer, or defensive armor in relation to aggressive weapons, so the perfection of concealing devices has evolved in response to increased powers of perception.
My Oxford colleague John Krebs and I took up the whole matter of evolutionary arms races in a paper given at the Royal Society in 1979. We pointed out that the improvements to be seen in an animal arms race are improvements in equipment to survive, not generally improvements in survival itself, and for an interesting reason. In an arms race between attack and defense, there may be episodes during which one side or the other temporarily pulls ahead, but in general, improvements on one side cancel out improvements on the other. There is even something a bit paradoxical about arms races. They are economically costly for both sides, yet there is no net benefit to either, because potential gains on one side are neutralized by gains on the other. From an economic point of view, both sides would be better off coming to an agreement to call off the arms race. As a ludicrous extreme, prey species might sacrifice a tithe of their number in exchange for secure and untroubled grazing for the rest. Neither predators nor prey need to divert valuable resources into muscles for fast running, sensory systems for detection of enemies, vigilance and prolonged hunts that are time-wasting and stressful for both sides. Both sides would benefit if such a trades union agreement could be reached. Unfortunately, Darwinian theory knows no route by which this could happen. Instead, both sides pour resources into competing with their own side to outrun the other, and individuals of both are forced into difficult economic trade-offs within their own bodily economies. If there were no predators, rabbits could devote all their economic resources and all their valuable time to feeding and reproducing more rabbits. Instead, they are forced to devote substantial time to looking out for predators and substantial economic resources into building up escape equipment. In turn, this forces predators to shift the balance of their economic investment away from the central business of reproducing and into improving their weaponry for catching prey. Arms races in animal evolution and human technology alike show themselves not in improved performance, but in increased shifting of economic investment away from alternative aspects of life and into servicing the arms race itself. Krebs and I recognized asymmetries in arms races that might result in one side shifting more economic resources into the arms race than the other. One such imbalance we dubbed the life-dinner principle. It takes its name from the Aesop fable, in which the rabbit runs faster than the fox, because the rabbit is running for his life, while the fox is only running for his dinner. There is an asymmetry in the cost of failure. In the arms race between cuckoos and hosts, every individual cuckoo can confidently look back on an unbroken line of ancestors who literally never failed to fool a foster parent. An individual of the host species, on the other hand, can look back on ancestors, many of whom never met a cuckoo, and many of whom met one and were fooled by it. Plenty of genes for failing to detect and kill cuckoos have passed successfully down the generations of the host species, but genes that cause cuckoos to fail in fooling hosts have a much more hazardous ride down the generations. This asymmetry of risk fosters another, an asymmetry in resources devoted to the arms race as opposed to other parts of life's economy. To repeat this important point, the cost of failure is harder on the cuckoos than on the hosts. This leads to asymmetries in how the two sides set their balance between competing calls on their time and other economic resources. Arms races are deeply and inescapably progressive, in a way that, for example, evolutionary accommodation to the weather is not. For an individual of any one generation, predators and parasites just make life harder in pretty much the same way as bad weather does. But over evolutionary time, there is a crucial difference. Unlike the weather, which fluctuates aimlessly, predators and parasites, and prey and hosts, are themselves evolving in a systematic direction, getting systematically worse from their victim's point of view. Unlike evolutionary tracking of ice ages and droughts, arms race trends from the past can be extrapolated into the future, and those trends are value-laden in the same kind of way as technological improvements in planes and weapons. Predators' eyes get sharper, though not necessarily more effective, because prey get harder to see. Running speeds increase progressively on both sides. though again the benefits are in general cancelled out by parallel improvements on the other side. Sabre teeth get sharper and longer as hides get tougher. Toxins get nastier as biochemical tricks for neutralizing them improve. With the passing of evolutionary time, the arms race progresses. All the features of life that a human engineer would admire as complex and elegant become more complex, more elegant, and more redolent of the illusion of design. In Climbing Mount Improbable, I distinguish designed from designoid. Spectacular feats of designoid engineering, such as the eye of a buzzard, the ear of a bat, the musculoskeletal apparatus of a cheetah or a gazelle, are all climactic products of evolutionary arms races between predators and prey. Parasite-host arms races culminate in even more finely meshed, co-adaptive designoid climaxes. And now for an important point. The evolution of any complex designoid organ in an arms race must have come about in a large number of steps of progressive evolution. Such evolution qualifies as progressive by our definition because each change tends to continue the direction of its predecessors. How do we know there are many steps rather than just one or two? By elementary probability theory. The parts of a complex machine, such as a bat's ear, could be rearranged at random in 1,000,000 ways before you hit another arrangement that could hear as well as the real thing. It is statistically improbable, not just in the boring sense that any particular arrangement of parts is as improbable with hindsight as any other. Very few permutations of atoms are precision auditory instruments. A real bat's ear is 1 in 1,000,000. It works. Something so statistically improbable cannot sensibly be explained as the result of a single stroke of luck. It has to be constructed by some sort of improbability-generating process, ratcheted up by what the philosopher Daniel Dennett calls a crane, as opposed to a skyhook. The analogy is to the man-made lifting machine, not the bird. The only cranes known to science, and I would bet the only cranes there ever have been or ever will be in the universe, are design and selection. Design explains the efficient complexity of microphones. Natural selection explains the efficient complexity of bat ears. Ultimately, selection explains microphones and everything design too, because the designers of microphones are themselves evolved engineers generated by natural selection. Ultimately, design cannot explain anything because there is an inevitable regression to the problem of the origin of the designer.
Design and natural selection are both processes of gradual, step-by-step, progressive improvement. Natural selection, at least, could not be anything else. In the case of design, it may or may not be a matter of principle, but it is an observed fact. The Wright brothers did not have a blinding flash of inspiration and promptly build a Concorde or a stealth bomber. They built a creaking, rickety crate that barely lifted off the ground and lurched into a neighbouring field. From Kitty Hawk to Cape Canaveral, every step of the way was built on its predecessors. Improvement is gradual, step by step in the same continued direction, fulfilling our definition of progressive. We could, with difficulty, conceive of a Victorian genius designing a Sidewinder missile fully formed within his Zeusian side-whiskered head. The notion defies all common sense and all history, but it does not instantly fall foul of the laws of probability in the way we would have to say of the spontaneous evolution of a flying, echolocating modern bat. A single macromutational leap from ground-dwelling ancestral shrew to flying, echolocating bat is ruled out just as safely as we can rule out luck when a conjurer successfully guesses the complete order of a shuffled pack of cards. Luck is not literally impossible in either case, but no good scientist would advance such prodigious luck as an explanation. The card-guessing feat has to be a trick. We've all seen tricks that appear just as baffling to the uninitiated. Nature does not set out to fool us as a conjurer does, but we can still rule out luck, and it was the genius of Darwin to rumble nature's sleight of hand. The echo-ranging bat is the result of an inching series of minor improvements, each adding cumulatively to its predecessors as it propels the evolutionary trend on in the same direction. That is progress, by definition. The argument applies to all complex biological objects that project the illusion of design and are therefore statistically improbable in a specified direction. All must have evolved progressively. The returning host, now unabashedly sensitive to major themes in evolution, notes progress as one of them. But progress of this kind is not a uniform, inexorable trend from the start of evolution all the way to the present. Rather, to take up the initial quotation from Mark Twain on history, it rhymes. We notice an episode of progress during the course of an arms race, but that particular arms race comes to an end. Perhaps one side is driven extinct by the other, or both sides go extinct, maybe in the course of a mass catastrophe of the kind that did for the dinosaurs. Then the whole process starts again, not from scratch, but from some discernibly earlier part of the arms race. Progress in evolution is not a single upward climb, but has a rhyming trajectory, more like the teeth of a saw. A sawtooth plunged deeply at the end of the Cretaceous when the last of the dinosaurs abruptly gave way to the mammal's new and spectacular climb of progressive evolution. But there had been lots of smaller sawteeth during the long reign of the dinosaurs, and since their immediate post-dinosaur rise, the mammals too have had smaller arms races, followed by extinctions, followed by renewed arms races. Arms races rhyme with earlier arms races in periodic spurts of many-stepped, progressive evolution.
That's all I want to say about arms races as drivers of progress. What other messages from the past does the returning host carry back to the present? Well, I must mention the alleged distinction between macroevolution and microevolution. I say alleged because my own view is that macroevolution, evolution on the grand scale of millions of years, is simply what you get when microevolution, evolution on the scale of individual lifetimes, is allowed to go on for millions of years. The contrary view is that macroevolution is something qualitatively different from microevolution. Neither view is self-evidently silly, nor are they necessarily contradictory. As so often, it depends on what you mean. We can use the parallel of the growth of a child. Imagine an argument about an alleged distinction between macro growth and micro growth. To study macro growth, we weigh the child every few months. Every birthday we stand her up against a white doorpost and draw a pencil line to record her height. More scientifically, we could measure various parts of the body, for example, the diameter of the head, the width of the shoulders, the length of the major limb bones, and plot them against each other. We also note significant events in development, such as the first appearance of pubic hair, or the first sign of breast and menstruation in girls, and of facial hair in boys. These are the changes that constitute macro growth, and we measure them on a timescale of years or months. Our instruments are not sensitive enough to pick up the daily and hourly changes in the body, micro growth, which, when summed over months, constitute macro growth. Or oddly, they may be too sensitive. A very accurate weighing machine could in theory pick up hourly growth, but the delicate signal is swamped by blundering increases in weight with every meal and decreases with every act of elimination. The acts of microgrowth itself, which all consist of cell divisions, make no immediate impact on weight at all, and an undetectable impact on gross body measurements. So, is macrogrowth the sum of lots of small episodes of microgrowth? Yes. But it is also true that the different timescales impose completely different methods of study and habits of thought. Microscopes looking at cells are not appropriate for the study of child development at the whole body level, and weighing machines and measuring tapes are not suitable for the study of cell multiplication. The two timescales in practice demand radically different methods of study and habits of thought. The same could be said of macroevolution and microevolution. If the terms are used to signify differences in how best to study them, I have no quarrel with a working distinction between microevolution and macroevolution. I do have a quarrel with those people who elevate this rather mundane practical distinction into one of almost, or more than almost, mystical import. There are those who think Darwin's theory of evolution by natural selection explains microevolution, but is in principle impotent to explain macroevolution, which consequently needs an extra ingredient, in extreme cases a divine extra ingredient. Unfortunately, this hankering after skyhooks has been given aid and comfort by real scientists whose intentions are innocent of any such thing. I have discussed the theory of punctuated equilibrium before, too often and too thoroughly to repeat myself in this book, so I shall only add that its advocates usually go on to propose a fundamental decoupling between microevolution and macroevolution. This is an unwarranted inference. No extra ingredient needs to be added at the micro level to explain the macro level. Rather, an extra level of explanation emerges at the macro level as a consequence of events at the micro level, extrapolated over unimaginable time spans. The working distinction between micro and macroevolution is similar to one that we meet in many other situations. The changes in the map of the world over geological time are due to the effects summed over millions of years of plate tectonic events occurring on a timescale of minutes, days, and years. But, as with the growth of a child, there is practically no overlap between the methods of study that serve the two timescales. The language of voltage fluctuations is not useful for discussing how a large computer program, such as Microsoft Excel, works. No sensible person denies that computer programs, however complicated, are entirely executed by temporal and spatial patterns of changes between two voltages. But no sensible person attends to that fact while writing, debugging, or using a large computer program. I have never seen any good reason to doubt the following proposition. Macroevolution is lots of little bits of microevolution joined end-to-end over geological time, and detected by fossils instead of by genetic sampling. Nevertheless, there could be, and I believe are, major events in evolutionary history after which the very nature of evolution itself changes. Evolution itself might be said to evolve. So far in this chapter, progress has meant individual organisms becoming better over evolutionary time at doing what individuals do, which is survive and reproduce. But we can also countenance changes in the phenomenon of evolution itself. Might evolution itself become better at doing something, what evolution does, as history goes by? Is late evolution some kind of improvement on early evolution? Do creatures evolve to improve not just their capacity to survive and reproduce, but the lineage's capacity to evolve? Is there an evolution of evolvability? I coined the phrase evolution of evolvability in 1987, when it was a somewhat heretical idea, especially for me as the alleged ultra-Darwinist. I was placed in the odd situation of advocating an idea at the same time as apologizing for it to people who couldn't see why it needed any apology. It is now a much-discussed topic, and others have taken it further than I ever envisaged, for example, the cell biologists Mark Kirshner and John Gerhardt, and the evolutionary entomologist Mary Jane West Eberhard in her magisterial book Developmental Plasticity and Evolution. What makes an organism good at evolving over and above being good at surviving and reproducing? An example first. We have already met the idea that island archipelagos are workshops of speciation. If the islands are near enough to each other to allow occasional immigrations, but far enough apart to allow time for evolutionary divergence between immigrations, we have a recipe for speciation, which is the first step towards evolutionary radiation. But how near is near enough? How far is far enough? That depends on the locomotor powers of the animals. For woodlice, a separation of a few yards is equivalent to a separation of many miles for a flying bird or bat. The Galapagos Islands are spaced just right for the divergent evolution of small birds, such as Darwin's finches, not necessarily for divergent evolution generally. For this purpose, separation of islands should be measured not in absolute units, but in units of travelability, calibrated to the kind of animal we are talking about. As with the Irish boatman who, when my parents asked him the distance of the great Blasket Island, replied, "About 3 miles in fine weather."
It follows that a Galapagos finch which either decreased or increased its flight range in evolution might thereby decrease its evolvability. Shortening the range lowers the chance of initiating a new race of descendants on another island. That way round it is easily understood. Lengthening the range has a less obvious effect in the same direction. Descendants are seeded onto new islands so frequently that there's no time for separate evolution before the next immigrant arrives. To push to the extreme, birds whose flight range is large enough to render the distance between islands trivial no longer see the islands as separate at all. As far as gene flow is concerned, the whole archipelago counts as one continent. So once again, speciation is not fostered. High evolvability, if we choose to measure evolvability as speciation rate, is an inadvertent consequence of intermediate locomotor range, where what counts as intermediate, as opposed to too short or too long, depends upon the spacing of the islands concerned. Of course, island in this sort of argument does not have to mean land surrounded by water. As the cichlid's tale showed, lakes are islands for aquatic animals, and reefs can be islands within lakes. Mountain tops are islands for land-bound animals that cannot easily tolerate low altitudes. A tree can be an island for an animal with a short range. For the AIDS virus, every man is an island.
If an increase or a decrease in travelling range results in an increase in evolvability, would we want to call this an evolved improvement? My ultra-Darwinist hackles start to quiver at this point. My heresy litmus starts to blush. It sounds uncomfortably like evolutionary foresight. Birds evolve an increase or a decrease to their flying range because of natural selection for individual survival. Future effects on evolution are an irrelevant consequence. Nevertheless, we might find with hindsight that the species that fill the world tend to be descended from ancestral species with a talent for evolution. You could say, therefore, that there is a kind of high level between lineage selection in favor of evolvability, an example of what the great American evolutionist George C. Williams called clade selection. Conventional Darwinian selection leads to individual organisms being finely tuned survival machines. Could it be that, as a consequence of clade selection, life itself has increasingly become a set of finely tuned evolving machines? If this is so, we might expect that in Kaufmannian reruns of evolution, the same progressive improvements in evolvability might be rediscovered. When I first wrote about the evolution of evolvability, I proposed a number of watershed events in evolution, after which evolvability suddenly improved. The most promising example of a watershed event I could think of was segmentation. Segmentation is the train-like modularization of the body in which parts and systems are repeated serially down the body. It seems to have been independently invented in its full form in arthropods, vertebrates, and annelid worms. The origin of segmentation is one of those evolutionary events that cannot have been gradual. Bony fish typically have about 50 vertebrae, but eels have as many as 200. Caecilians, worm-like amphibians, vary between 95 and 285 vertebrae. Snakes differ hugely in vertebral number. The record known to me is 565 for an extinct snake. Every vertebra of a snake represents one segment with its own pair of ribs, its own muscle blocks, its own nerves sprouting from the spinal cord. You can't have fractional numbers of segments, and the evolution of variable segment numbers must include numerous instances in which a mutant snake differed from its parents in some whole number of segments, at least one, possibly more, in one fell swoop. Similarly, when segmentation originated, there must have been a mutational transition straight from unsegmented parents to a child with 2, at least, segments. It is hard to imagine such a freak surviving, let alone finding a mate and reproducing, but it evidently happened because segmented animals are all around us. Very probably the mutation involved Hox genes, like those of the fruit fly's tail. In my 1987 evolvability paper, I guessed that the individual success or otherwise of the first segmented animal during its own lifetime is relatively unimportant. No doubt many other mutants have been more successful as individuals. What is important about the first segmented animal is that its descendant lineages were champion evolvers. They radiated, speciated, gave rise eventually to whole new phyla. Whether or not segmentation was a beneficial adaptation during the individual lifetime of the first segmented animal, segmentation represented a change in embryology that was pregnant with evolutionary potential.
The ease with which whole segments can be added or subtracted from the body is one thing that contributes to enhanced evolvability. So is differentiation among segments. In animals such as millipedes and earthworms, most of the segments are the same as each other. But there is a recurrent tendency, especially among arthropods and vertebrates, for particular segments to become specialized for particular purposes, and hence different from other segments. Compare a lobster with a centipede. A lineage that manages to evolve a segmented body plan is immediately able to evolve a whole range of new animals by altering segmental modules all along the body. Segmentation is an example of modularity, and modularity in general is a main ingredient in the thinking of more recent writers on the evolution of evolvability. Of the many meanings of module listed in the Oxford English Dictionary, the relevant one is
one of a series of production units or component parts that are standardized to facilitate assembly or replacement and are usually prefabricated as self-contained structures.
Modular is the adjective describing an assemblage of modules, and modularity is the corresponding abstract noun, the property of being modular. Other examples of modular construction include many plants. Leaves and flowers are modules. But perhaps the best examples of modularity are to be found at the cellular and biochemical level. Cells themselves are modules par excellence, and within cells, so are protein molecules, and of course, DNA itself. The invention of multicellularity is another important watershed event that almost certainly enhanced evolvability. It preceded segmentation by hundreds of millions of years, and segmentation is itself a kind of large-scale reenactment of it, another leap in modularity. What other watersheds have there been? The dedicatee of this book, John Maynard Smith, collaborated with his Hungarian colleague Eös Szatmári on the major transitions in evolution. Most of their major transitions would fit under my heading of watershed events, major improvements in evolvability. This obviously includes the origin of replicating molecules, for without them there could be no evolution at all. If, as Cairn Smith and others have suggested, DNA usurped the key role of replicator from some less proficient predecessor, bridged by intermediate stages, each one of those stages would constitute a leap forward in evolvability. If we accept the RNA world theory, there would have been a major transition or watershed when a world of RNA serving as both replicator and enzyme gave over to a separation between DNA in the replicator role and proteins in the enzyme role. Then there was the clubbing together of replicating entities, genes, in cells with walls, which prevented the gene products leaking away and kept them together with the products of other genes with which they could collaborate in cellular chemistry. A very major transition, and very probably a watershed of evolvability, was the birth of the eukaryotic cell by the commingling of several prokaryotic cells. So was the origin of sexual reproduction, which coincided with the origin of the species itself, with its own gene pool and all that implied for future evolution. Maynard Smith and Szatmári go on to list the origin of multicellularity, the origin of colonies such as ant and termite nests, and the origin of human societies with language. There is a rhyming similarity between at least several of these major transitions. They often involve the coming together of previously independent units in a larger grouping at a higher level, with concomitant loss of independence at the lower level.
To their list I have already added segmentation, and I would stress another which I call bottlenecking. Once again, to spell it out in full would be to repeat from previous books, especially the final chapter, Rediscovering the Organism, of the extended phenotype. Bottlenecking refers to a type of life history in multicellular organisms. In bottlenecking, the life cycle regularly returns to a single cell, from which a multicellular body is grown anew. The alternative to a bottlenecked life cycle might be a hypothetical straggling water plant that reproduces by breaking off small multicellular chunks of itself, which drift off, grow, and then break off more small chunks. Bottlenecking has three important consequences, all of which are certainly good candidates for improvements in evolvability. First, evolutionary innovations can be reinvented from the bottom up rather than as remouldings of existing structures, the equivalent of beating swords into plowshares. An improvement in, say, a heart has a better chance of being a clean improvement if genetic changes can alter the whole course of development from a single cell. Imagine the alternative. Take the existing heart and modify it by differential tissue growth within its continuously beating fabric. This on-the-trot remodeling would impair the working of the heart and compromise the would-be improvement. Second, by continually resetting to a consistent starting point in a recurrent life cycle, bottlenecking provides a calendar by which embryological events may be timed. Genes may be turned on or off at key points in the growth cycle. Our hypothetical straggling chunk extruder lacks a recognizable timetable to regulate such switchings on and off. Third, without bottlenecking, different mutations would accumulate in different parts of the straggling chunk extruder. The incentive among cells to cooperate would be reduced. In effect, subpopulations of cells would be tempted to behave as cancers to increase their chance of contributing genes to the extruded chunks. With bottlenecking, since every generation starts out as a single cell, the whole body has a good chance of being made of a uniform genetic population of cooperating cells, all descended from that single cell. Without bottlenecking, the cells of the body might have, from a genetic point of view, divided loyalties.
Related to bottlenecking is another important landmark event in evolution, and one that may well have contributed to evolvability and might be rediscovered in Kaufman reruns. This is the separation of the germ line from the soma, first clearly understood by the great German biologist August Weismann. As we saw at Rendezvous 31, what happens in the developing embryo is that a portion of the cells are set aside for reproduction, germline cells, while the rest are destined to make the body, somatic cells. Germline genes are potentially immortal, with a prospect of direct descendants millions of years into the future. Somatic genes are destined for a finite, if not always predictable, number of cell divisions to make the body tissues, after which their line will come to an end and the organism will die. Plants often violate the separation, most obviously when they practice vegetative reproduction. This could constitute an important difference between the ways plants and animals evolve. Before the evolutionary invention of the separate soma, all living cells were potentially the ancestors of an indefinite line of descendants, as sponge cells still are. The invention of sex is a major watershed, which is superficially confusable both with bottlenecking and with the separation of the germline, but is logically distinct from both. In its most general form, sex is the partial mixing of genomes. We are familiar with a particular, highly regimented version of it in which every individual gets 50% of its genome from each of two parents. We are used to the idea that there are two kinds of parent, female and male, but this is not a necessary part of sexual reproduction. Isogamy is a system in which two individuals, not distinguished as male and female, combine half their genes to make a new individual. The male-female divide is best seen as a further watershed event, which came after the origin of sex itself. Regimented sex of this kind is accompanied in every generation by a reduction division, in which each individual donates 50% of its genome to each offspring. Without this reduction, genomes would double in size with every generation. Bacteria practice a haphazard form of sexual donation that is sometimes described as sex, but which is really very different, having more in common with the cut and paste or copy and paste functions of a word processor. Fragments of a genome are copied or cut from one bacterium and pasted into another, which does not have to be a member of the same species, though the very meaning of species is in doubt for bacteria. Because genes are software subroutines that perform cellular operations, a pasted gene can immediately go to work in its new environment doing the same task as it did before. What is in it for the donating bacterium? That may be the wrong question. The right question might be, what is in it for the donated gene? And the answer is that genes that successfully get themselves donated and then successfully help the recipient bacterium to survive and pass them on, thereby increase the number of copies of themselves in the world. It is not clear whether our regimented eukaryotic sex has evolved from bacterial cut-and-paste sex, or whether it was an entirely new watershed event. Both must have had a huge impact on subsequent evolution, and are candidates for discussion under the heading of the evolution of evolvability. Regimented sex, as we saw in the rotifer's tale, has a dramatic effect upon future evolution because it makes possible the very existence of species with their gene pools.
The host's farewell. If, as returning host, I reflect on the whole pilgrimage of which I have been a grateful part, my overwhelming reaction is one of amazement. Amazement not only at the extravaganza of details that we have seen, amazement, too, at the very fact that there are any such details to be had at all on any planet. The universe could so easily have remained lifeless and simple, just physics and chemistry, just the scattered dust of the cosmic explosion that gave birth to time and space. The fact that it did not, the fact that life evolved out of nearly nothing, some 10 billion years after the universe evolved, out of literally nothing, is a fact so staggering that I would be mad to attempt words to do it justice. And even that is not the end of the matter. Not only did evolution happen, it eventually led to beings capable of comprehending the process, and even of comprehending the process by which they comprehend it. This pilgrimage has been a trip, not just in the literal sense, but in the countercultural sense I met when a young man in California in the 1960s. The most potent hallucinogen on sale in Haight or Ashbury or Telegraph Avenue would be tame by comparison. If it's amazement you want, the real world has it all. Not to stray outside the covers of the book, think of Venus's girdle, migrating jellyfish and tiny harpoons. Think of the platypus's radar and the electric fish, of the horsefly larva, with the apparent foresight to preempt cracks in the mud. Think redwood, think peacock, think starfish, with its piped hydraulic power. Think cichlids of Lake Victoria. It is not pride in my book, but reverence for life itself that encourages me to say, if you want a justification for the latter, open the former anywhere at random, and reflect on the fact that although this book has been written from a human point of view, another book could have been written in parallel for any of 10 million starting pilgrims. Not only is life on this planet amazing and deeply satisfying to all whose senses have not become dulled by familiarity, the very fact that we have evolved the brain power to understand our evolutionary genesis redoubles the amazement and compounds the satisfaction. Pilgrimage implies piety and reverence. I have not had occasion here to mention my impatience with traditional piety and my disdain for reverence where the object is anything supernatural, but I make no secret of them. It is not because I wish to limit or circumscribe reverence, not because I want to reduce or downgrade the true reverence with which we are moved to celebrate the universe once we understand it properly. On the contrary would be an understatement. My objection to supernatural beliefs is precisely that they miserably fail to do justice to the sublime grandeur of the real world. They represent a narrowing down from reality, an impoverishment of what the real world has to offer. I suspect that many who call themselves religious would find themselves agreeing with me. To them, I would only quote a favorite remark that I overheard at a scientific conference. A distinguished elder statesman of my subject was having a long argument with a colleague. As the altercation came to an end, he twinkled and said, "You know, we really do agree. It's just that you say it wrong." I feel I have returned from a true pilgrimage.
And grasses all comes from small green bodies called chloroplasts within their cells. Chloroplasts are distant descendants of once free-living green bacteria. They still have their own DNA, and they still reproduce by asexual division, building up to a substantial population within each plant cell. As far as a chloroplast is concerned, it is a member of a reproducing population of green bacteria. The world in which it lives and reproduces is the interior of a plant cell. From time to time, its world suffers a minor upheaval when the plant cell divides into two daughter cells. Roughly half the chloroplasts find themselves in each daughter cell, and they soon resume their normal existence of reproducing to populate their new world with chloroplasts. All the while, the chloroplasts use their green pigment to trap photons from the sun and channel the sun's energy in the useful direction of synthesizing organic compounds from carbon dioxide and water supplied by the host plant. The oxygen wastes are partly used by the plant and partly exhaled into the atmosphere through holes in the leaves, called stomata, singular stoma. The organic compounds synthesized by the chloroplasts are ultimately made available to the host plant cell. Interestingly reminiscent of the Mixotrix tale, some chloroplasts show evidence of having entered plant cells indirectly by piggybacking inside other eukaryotic cells, which would presumably have been called algae. The evidence is that some chloroplasts have a double membrane. Presumably the inner one is the wall of the original bacterium, the outer one the wall of the alga. As with Mixotricha, we can see recent reenactments in the many examples of single-celled green algae being incorporated in the cells or tissues of fungi and animals, for example the green algae that inhabit corals. Those chloroplasts that have a single membrane presumably entered directly, not on the coattails of algae. All the free oxygen in the atmosphere comes from green bacteria, whether free-living or in the form of chloroplasts. When it first appeared in the atmosphere, oxygen was a poison. Indeed, some people colourfully say it still is a poison, which is why doctors advise us to eat antioxidants. It was a brilliant chemical coup to discover how to use oxygen to extract originally solar energy from organic compounds. This discovery, which can be seen as a sort of reverse photosynthesis, was entirely made by bacteria, but a different kind of bacteria. As with photosynthesis itself, bacteria still have a monopoly on the technology, except that again, as with photosynthesis, eukaryotic cells like ours give house room to these oxygen-loving bacteria, who now travel under the name of mitochondria. We have become so dependent on oxygen via the biochemical wizardry of mitochondria that the statement that it is a poison makes sense only when uttered in a tone of self-conscious paradox. Carbon monoxide, the deadly poison in car exhausts, kills us by competing with oxygen for the favours of our oxygen-carrying hemoglobin molecules. Depriving somebody of oxygen is a swift way to kill them. Yet our own cells, unaided, wouldn't know what to do with oxygen. It is only mitochondria, and their bacterial cousins that do. As with chloroplasts, molecular comparison tells us the particular group of bacteria from which mitochondria are drawn. Mitochondria sprang from the so-called alpha-proteobacteria, and they are therefore related to the rickettsias which cause typhus and other nasty diseases. Mitochondria themselves have lost much of their original genome and have become completely adapted to life inside eukaryotic cells. But like chloroplasts, they still reproduce autonomously by division. Although mitochondria have lost most of their genes, they haven't lost all of them, and this is very fortunate for molecular geneticists, as we've seen in many places throughout this book. Because the great historic rendezvous is a true rendezvous in the forward historical direction, our pilgrimage from now on should strictly be a split pilgrimage. We should follow the separate backward pilgrimages of the various participants in the eukaryotic compact until they are finally reunited in the deep past, but that would make for a gratuitously complicated journey.
Rendezvous 38 and 39. The remaining pilgrims yet to join are all prokaryotes. At one time they would all have been called bacteria, but molecular studies have shown that there are two great prokaryotic kingdoms, the true bacteria, or eubacteria, and the archaebacteria, or archaea. Current opinion places us eukaryotes slightly closer to the archaea than to the eubacteria. In pilgrimage terminology, this means that the archaean pilgrims join us at rendezvous 38 and the eubacteria at rendezvous 39. Two of the eubacteria tell tales.
The rhizobium's tale. The wheel is the proverbial human invention. Take apart any machine of more than rudimentary complexity, and you'll find wheels. Ship and aeroplane propellers, spinning drills, lathes, potter's wheels. Our technology runs on the wheel and would seize up without it. The wheel may have been invented in Mesopotamia during the 4th millennium BC. We know it was elusive enough to need inventing because the New World civilization still lacked it by the time of the Spanish conquest. The alleged exception here, children's toys, seems so bizarre as to prompt suspicion. Could it be one of those myths that spreads purely because it is so memorable, like the Inuit having 50 words for snow? Whenever humans have a good idea, zoologists have grown accustomed to finding it anticipated in the animal kingdom. Examples pervade this book, including echo-ranging bats, electrolocation, the duckbill's tail, the dam, the beaver's tail, the parabolic reflector, limpets, the infrared heat-seeking sensor, some snakes, the hypodermic syringe, wasps, snakes and scorpions, the harpoon, cnidarians, and jet propulsion, squids. Why not the wheel? It is possible that the wheel impresses us only by contrast with our rather undistinguished legs. Before we had engines driven by fuels, fossilized solar energy, we were easily outpaced by animal legs. No wonder Richard III offered his kingdom for four-footed transportation out of his predicament. Perhaps most animals wouldn't benefit from wheels because they can already run so fast on legs. After all, until very recently, all our wheeled vehicles have been pulled by leg power. To a horse, a wheel is something that slows you down. Here's another way in which we risk overrating the wheel. It is dependent for maximum efficiency on a prior invention, the road, or other smooth, hard surface. A car's powerful engine enables it to beat a horse or a dog or a cheetah on a hard, flat road, but run the race over wild country or ploughed fields, perhaps with hedges or ditches in the way, and it is a rout. The horse will leave the car wallowing. Well then, perhaps we should change our question. Why haven't animals developed the road? There's no great technical difficulty. The road should be child's play compared with the beaver dam or the bowerbird's ornamented arena. There are even some digger wasps that tamp soil hard, picking up a stone tool to do so. Presumably the same skills could be used by larger animals to flatten a road. But it raises an unexpected problem. Even if road building is technically feasible, it is a dangerously altruistic activity. If I, as an individual, build a good road from A to B, you may benefit from the road just as much as I do. Why should this matter? Because Darwinism is a selfish game. Building a road that might help others will be penalized by natural selection. A rival individual benefits from my road just as much as I do, but he does not pay the cost of building. Freeloaders who use my road and don't bother to build their own will be free to concentrate their energy on out-reproducing me while I slave away on the road. Unless special measures are taken, genetic tendencies towards lazy, selfish exploitation will thrive at the expense of industrious road building. The upshot will be that no roads get built. With the benefit of foresight, we can see that everybody will be worse off. But natural selection, unlike us humans with our big, recently evolved brains, has no foresight. What is so special about humans that we have managed to overcome our antisocial instincts and build roads that we all share? Oh, there is so much. No other species comes remotely close to a welfare state, to an organization that takes care of the old, that looks after the sick and the orphaned, that gives to charity. On the face of it, these things present a challenge to Darwinism, but this is not the place to go into that. We have governments, police, taxation, public works to which we all subscribe, whether we like it or not. The man who wrote, "Sir, you are very kind, but I'd prefer not to join your income tax scheme," heard back, we may be sure, from the Inland Revenue. Unfortunately, no other species has invented the tax. They have, however, invented the virtual fence. An individual can secure his exclusive use of a resource if he actively defends it against rivals. Many species of animals are territorial, not just birds and mammals, but fish and insects too. They defend an area against rivals of the same species, often so as to sequester a private feeding ground or a private courtship bower or nesting area. An animal with a large territory might benefit by building a network of good flat roads across the territory from which rivals were excluded. This is not impossible, but such animal roads would be too local for long-distance, high-speed travelling. Roads of any quality would be limited to the small area that an individual can defend against genetic rivals. Not an auspicious beginning for the evolution of the wheel. But now, finally, we come to the teller of this tale. There is one revealing exception to the premise. Some very small creatures have evolved the wheel in the fullest sense of the word. The wheel may even have been the first locomotor device ever evolved, given that for most of its first 2 billion years, life consisted of nothing but bacteria. Many bacteria, of which rhizobium is typical, swim using thread-like spiral propellers, each driven by its own continuously rotating propeller shaft. It used to be thought that these flagella were wagged like tails, the appearance of spiral rotation resulting from a wave of motion passing along the length of the flagellum, as in a wriggling snake. The truth is much more remarkable. The bacterial flagellum is attached to a shaft that rotates freely and indefinitely in a hole that runs through the cell wall. This is a true axle, a freely rotating hub. It is driven by a tiny molecular motor, which uses the same biophysical principles as a muscle, but a muscle is a reciprocating engine, which after contracting has to lengthen again to prepare for a new power stroke. The bacterial motor just keeps on going in the same direction, a molecular turbine. The fact that only very small creatures have evolved the wheel suggests what may be the most plausible reason why larger creatures have not. It's a rather mundane, practical reason, but nonetheless important. A large creature would need big wheels, which, unlike man-made ones, would have to grow in situ, rather than being separately fashioned out of dead materials and then mounted. For a large living organ, growth in situ demands blood, or something equivalent, and probably something equivalent to nerves, too. The problem of supplying a freely rotating organ with blood vessels, not to mention nerves, that don't tie themselves in knots, is too vivid to need spelling out. There might be a solution, but we need not be surprised that it has not been found. Human engineers might suggest running concentric ducts to carry blood through the middle of the axle into the middle of the wheel. But what would the evolutionary intermediates have looked like? Evolutionary improvement is like climbing a mountain. You can't jump from the bottom of a cliff to the top in a single leap. Sudden, precipitous change is an option for engineers, but in nature the summit of the evolutionary mountain can be reached only via a gradual ramp upwards from the starting point. The wheel may be one of those cases where the engineering solution can be seen in plain view, yet be unattainable in evolution because it lies on the other side of a deep valley, unevolvable by large animals, but within the reach of bacteria because of their small size. In an imaginative piece of lateral thinking, Philip Pullman, in his epic of childhood fiction, His Dark Materials, solves the problem for big animals in a completely unexpected but very biological way. He invents a species of benevolent trunked animal, the mulefa, who have evolved symbiotically with a species of gigantic tree that sheds hard, circular, wheel-like seed pods. The feet of the mulefa have a horny, polished spur, which fits into a hole in the center of a seed pod, which then works as a wheel. The trees gain from the arrangement because whenever, as eventually must happen, a wheel wears out and has to be discarded, the mulefa disperse the seeds inside. The trees have evolved to return the favor by making the pods perfectly circular, with a suitable hole for the mulefan axle right in the center, into which they secrete a high-grade lubricating oil. Pullman cleverly notes that the whole system is made possible only by a geological peculiarity of the world on which these creatures live. Basalt happens to form in long ribbon-like lines over the savanna, which serve as unmade but hard roads. Short of Pullman's ingenious symbiosis, we may provisionally accept the wheel as one of those inventions that, even if it were a good idea in the first place, cannot evolve in large animals, either because of the prior need for a road, or because the problem of the twisted blood vessels could never be solved, or because the intermediates to a final solution would never be good for anything. Bacteria were able to evolve the wheel because the world of the very small is so very different and presents such different technical problems.
As it happens, the bacterial flagella motor itself has recently, in the hands of a species of creationists who call themselves intelligent design theorists, been elevated to the status of icon of alleged unevolvability. Since it manifestly exists, the conclusion of their argument is different. Whereas I proposed unevolvability as an explanation for why large animals like mammals don't grow wheels, creationists have seized upon the bacterial flagella wheel as something that cannot exist, and yet does. So it must have come about by supernatural means. This is the ancient argument from design, also called the argument from Paley's watchmaker, or the argument from irreducible complexity. I have less kindly called it the argument from personal incredulity, because it always has the form,
I personally cannot imagine a natural sequence of events whereby X could have come about. Therefore it must have come about by supernatural means.
Time and again, scientists have retorted that if you make this argument, it says less about nature than about the poverty of your imagination. The argument from personal incredulity would lead us to invoke the supernatural every time we see a good conjurer whose tricks we cannot fathom. It is perfectly legitimate to propose the argument from irreducible complexity as a possible explanation for the lack of something that doesn't exist, as I did for the absence of wheeled mammals. That is very different from evading the scientist's responsibility to explain something that does exist, such as wheeled bacteria. Nevertheless, to be fair, it is possible to imagine validly using some version of the argument from design or the argument from irreducible complexity. Future visitors from outer space who mount archaeological digs of our planet will surely find ways to distinguish designed machines such as planes and microphones from evolved machines such as bat wings and ears. It is an interesting exercise to think about how they will make the distinction. They may face some tricky judgments in the messy overlap between natural evolution and human design. If the alien scientists can study living specimens, not just archaeological relics, what will they make of fragile, highly strung racehorses and greyhounds, of snuffling bulldogs who can scarcely breathe and can't be born without caesarean assistance, of blear-eyed Pekingese baby surrogates, of walking udders such as Frisian cows, walking rashers such as landrace pigs, or walking woolly jumpers such as merino sheep? Molecular machines, nanotechnology, crafted for human benefit on the same scale as the bacterial flagellar motor, may pose the alien scientists even harder problems. Francis Crick, no less, has speculated semi-seriously in Life Itself that bacteria might not have originated on this planet, but been seeded from elsewhere. In Crick's fantasy, they were sent in the nose cone of a rocket by alien beings who wanted to propagate their form of life, but shrank from the technically harder problem of transporting themselves and relied, instead, upon natural evolution to finish the job once the bacterial infection had taken root. Crick and his colleague Leslie Orgel, who originally suggested the idea with him, supposed that the bacteria had originally evolved by natural processes on the home planet, but they could equally, while in the mood for science fiction, have added a touch of nanotechnological artifice to the mix. Crick himself, whether with regret or relief it is hard to say, finds little good evidence to support his own theory of directed panspermia. But the hinterland between science and science fiction constitutes a useful mental gymnasium in which to wrestle with a genuinely important question. Given that the illusion of design conjured by Darwinian natural selection is so breathtakingly powerful, how do we, in practice, distinguish its products from deliberately designed artifacts? Another great molecular biologist, Jacques Monod, began his Chance and Necessity in similar terms. Could there be genuinely persuasive examples of irreducible complexity in nature, complex organization made of parts, the loss of any one of which would be fatal to the whole? If so, might this suggest genuine design by a superior intelligence, say from an older and more highly evolved civilization on another planet? It is possible that an example of such a thing might eventually be discovered, but the bacterial flagellar motor, alas, is not it. Like so many previous allegations of irreducible complexity from the eye on, the bacterial flagellum turns out to be eminently reducible. Kenneth Miller of Brown University deals with the whole question in a tour de force of clear exposition. As Miller shows, the allegation that the component parts of the flagellar motor have no other function is simply false. As one example, many parasitic bacteria have a mechanism for injecting chemicals into host cells called the TTSS, type 3 secretory system. The TTSS makes use of a subset of the very same proteins that are used in the flagellar motor. In this case, they are used not for providing rotatory motion of a circular hub, but for making a hole in a cell wall. Miller summarizes.
Stated directly, the TTSS does its dirty work using a handful of proteins from the base of the flagellum. From the evolutionary point of view, this relationship is hardly surprising. In fact, it's to be expected that the opportunism of evolutionary processes would mix and match proteins to produce new functions. According to the doctrine of irreducible complexity, however, this should not be possible. If the flagellum is indeed irreducibly complex, then removing just one part, let alone 10 or 15, should render what remains, by definition, non-functional. Yet the TTSS is indeed fully functional, even though it is missing most of the parts of the flagellum. The TTSS may be bad news for us, but for the bacteria that possess it, it is a truly valuable biochemical machine. The existence of the TTSS in a wide variety of bacteria demonstrates that a small portion of the irreducibly complex flagellar mechanism can indeed carry out an important biological function. since such a function is clearly favoured by natural selection, the contention that the flagellum must be fully assembled before any of its component parts can be useful is obviously incorrect. What this means is that the argument for intelligent design of the flagellum has failed.
Miller's indignation at intelligent design theory receives a boost from an interesting source, his deep religious convictions, which are more fully articulated in Finding Darwin's God. Miller's God, if not Darwin's, is the God revealed in, or perhaps synonymous with, the deep lawfulness of nature. The creationist quest to demonstrate God through the negative route of the argument from personal incredulity turns out, as Miller shows, to assume that God capriciously violates his own laws, and this, to those like Miller of a thoughtfully religious disposition, is a cheap and demeaning sacrilege. As a non-religious person, I can sympathetically buttress Miller's argument with a parallel one of my own. If not sacrilegious, the intelligent design style of argument from personal incredulity is lazy. I've satirized it in an imagined conversation between Sir Andrew Huxley and Sir Alan Hodgkin, both sometime presidents of the Royal Society, who shared the Nobel Prize for working out the molecular biophysics of the nerve impulse.
I say, Huxley, this is a terribly difficult problem. I can't see how the nerve impulse works, can you?
No, Hodgkin, I can't, and these differential equations are fiendishly hard to solve. Why don't we just give up and say that the nerve impulse propagates by nervous energy?
Excellent idea, Huxley. Let's write the letter to nature now. It'll only take one line, then we can turn to something easier.
Andrew Huxley's elder brother Julian made a similar point when, long ago, he satirized vitalism, then usually epitomized by Henri Bergson's name of élan vital, as tantamount to explaining that a railway engine was propelled by élan locomotif. My censure of laziness and Miller's of sacrilege do not apply to the hypothesis of directed panspermia. Crick was talking about superhuman, not supernatural design. The difference really matters. On Crick's worldview, superhuman designers of bacteria, or of the means to seed Earth with them, would themselves have originally evolved by some local equivalent of Darwinian selection on their own planet. Crucially, Crick would always seek what Daniel Dennett calls a crane, would never resort, as Henri Bergson would, to a skyhook. The main objection to the irreducible complexity argument amounts to a demonstration that the allegedly irreducible complex entity, the flagellar motor, the blood-clotting cascade, the Krebs cycle, or whatever it might be, is actually reducible. The personal incredulity was simply wrong. To this we add the reminder that, even if we can't think of a step-by-step pathway by which the complexity might have evolved, the eager slide to assuming that it is therefore supernatural is either sacrilegious or lazy, according to taste.
But there is another objection that needs to be mentioned, the arch and scaffolding of Graham Cairns Smith. Cairns Smith was writing in a different context, but his point works here too. An arch is irreducible in the sense that if you remove part of it, the whole collapses. Yet it is possible to build it gradually by means of scaffolding. The subsequent removal of the scaffolding, so that it no longer appears in the visible picture, does not entitle us to a mystified and obscurantist attribution of supernatural powers to the masons. The flagellar motor is common among bacteria. Rhizobium was chosen to tell the tale because of the second claim to impress us with the versatility of bacteria. Farmers sow plants of the pea family, Leguminosae, as a part of most good crop rotation schemes for one very good reason. Leguminous plants can use raw nitrogen straight out of the air. It is by far the most abundant gas in our atmosphere, rather than having to suck up nitrogen compounds from the soil. But it isn't the plants themselves that fix atmospheric nitrogen and turn it into usable compounds. It is symbiotic bacteria, specifically Rhizobium, housed for the purpose in special nodules provided for them, with every indication of inadvertent solicitude on the roots of the plants. Such contracting out of ingenious chemical tricks to chemically much more versatile bacteria is an extremely common pattern throughout animals and plants. It is the main message of Tack's tale. Tack is the nickname of Thermus aquaticus, the heat-loving bacterium who tells the last tale in the book about the enormous versatility of bacteria as biochemists compared to us eukaryotes.
Tack's tale. Although our tales are not in the main told in the first person, let us make an exception for the last word of all our tales and give it to Thermus aquaticus.
Look at life from our perspective, and you eukaryotes will soon cease giving yourselves such airs. You bipedal apes, you stump-tailed tree shrews, you desiccated lobe-fins, you vertebrated worms, you hoxed-up sponges, you newcomers on the block, you eukaryotes, you barely distinguishable members of a monotonously narrow parish, you are little more than fancy froth on the surface of bacterial life. Why, the very cells that build you are themselves colonies of bacteria, replaying the same old tricks we bacteria discovered 1 billion years ago. We were here before you arrived, and we shall be here after you are gone.
Canterbury. As befits the destination of a 4 billion-year pilgrimage, our Canterbury has a patina of mystery. It is the singularity known as the origin of life, but we could better call it the origin of heredity. Life itself is not clearly defined, a fact that contradicts intuition and traditional wisdom. Ezekiel chapter 37, in which the prophet was commanded down into the valley of the bones, identifies life with breath. I cannot resist quoting the passage, bone to his bone, such wonderful economy of language.
So I prophesied as I was commanded, and as I prophesied there was a noise, and behold a shaking, and the bones came together, bone to his bone. And when I beheld, lo, the sinews and the flesh came up upon them, and the skin covered them above, but there was no breath in them. Then said he unto me, Prophesy unto the wind, prophesy, Son of man, and say to the wind, Thus saith the Lord God, Come from the 4 winds, O breath, and breathe upon these slain, that they may live.
And of course the winds did. A great army breathed and stood up. Breath, for Ezekiel, defines the difference between dead and alive. Darwin himself implied the same in one of his more eloquent passages, the concluding words of The Origin of Species.
Thus, from the war of nature, from famine and death, the most exalted object which we are capable of conceiving, namely the production of the higher animals, directly follows. There is grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been, and are being, evolved.
Darwin rightly reversed Ezekiel's order of events. The breath of life came first and created the conditions under which bones and sinews, flesh and skin would eventually evolve. Incidentally, the phrase by the Creator is not present in the first edition of The Origin. It was added in the second edition, probably as a sop to the religious lobby. Darwin later regretted this in a letter to his friend Hooker.
I have long since regretted that I truckled to public opinion and used the Pentateuchal term of creation, by which I really meant appeared by some wholly unknown process. It is mere rubbish thinking at present of the origin of life. One might as well think of the origin of matter.
Darwin probably, and in my view rightly, saw the origin of primitive life as a relatively, and I stress relatively, easy problem, compared with the one he solved: how life, once begun, developed its amazing diversity, complexity, and powerful illusion of good design. Nevertheless, Darwin did later, in another letter to Hooker, venture a guess about the wholly unknown process that started it all. He was led to it through wondering why we don't see life originating again and again.
It is often said that all the conditions for the first production of a living organism are now present which could ever have been present. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed ready to undergo still more complex changes, at the present day such matter would be instantly absorbed, which would not have been the case before living creatures were found.
The doctrine of spontaneous generation had only lately been experimentally attacked by Pasteur. It had long been believed that rotting meat spontaneously generated maggots, that goose barnacles spontaneously generated goslings, and even that dirty laundry placed with wheat generated mice. Perversely, the spontaneous generation theory was supported by the Church, following Aristotle in this as in so much else. I say perversely because, at least with hindsight, spontaneous generation was as direct a challenge to divine creation as evolution would ever be. The idea that flies or mice could spring spontaneously into existence hugely underestimates the stupendous achievement that the creation of flies or mice would be, an insult to the Creator, one might have thought. But the science-free mindset fails to grasp how complex and inherently improbable a fly or a mouse is. Darwin was perhaps the first to appreciate the full magnitude of this mistake. As late as 1872, in a letter to Wallace, the co-discoverer of natural selection, Darwin could still find it necessary to express his skepticism about rotifers and tardigrades being spontaneously generated, as had been suggested in a book, The Beginnings of Life, which he otherwise admired. His skepticism was on target as usual. Rotifers and tardigrades are complex life forms beautifully fitted to their respective ways of life. For them to be spontaneously generated would imply that they became fit and complex
by a happy accident, and this I cannot believe.
Happy accidents of such magnitude were anathema to Darwin, as they should have been to the Church, for a different reason. The whole rationale of Darwin's theory was, and is, that adaptive complexity comes about by slow and gradual degrees, step by step, no single step making too large a demand on blind chance as explanation. The Darwinian theory, by rationing chance to the small steps needed to supply variation for selection, provides the only realistic escape from sheer luck as the explanation for life. If rotifers could spring into existence just like that, Darwin's life work was unnecessary. But natural selection itself had to have a beginning. In this sense alone, some kind of spontaneous generation must have happened, if only once. The beauty of Darwin's contribution was that the single spontaneous generation we must postulate did not have to synthesize anything complicated like a maggot or a mouse. It only had to make... Well, now we approach the heart of the problem. If not breath, what was the vital ingredient that first enabled natural selection to get going and lead eventually, after epochs of cumulative evolution, to maggots, mice, and men? The details lie buried, perhaps beyond recovery, at our ancient Canterbury. But we can give the key ingredient a minimalist name to express the kind of thing it must have been. That name is heredity. We should be seeking not the origin of life, which is vague and undefined, but the origin of heredity, true heredity, and that means something very precise. I have previously invoked fire to help explain it. Fire rivals breath as imagery for life. When we die, the fire of life goes out. Our ancestors who first tamed it probably thought fire a living thing, a god even. Staring into flames or embers, especially at night when the campfire warmed and protected them, did they commune in imagination with a glowing, dancing soul? Fire stays alive as long as you feed it. Fire breathes air. You can suffocate it by cutting off its oxygen supply. You can drown it with water. Wildfire devours the forest, driving animal prey before it with the speed and ruthlessness of a pack of wolves in, literally, hot pursuit. As with wolves, our ancestors could capture a fire cub as a useful pet, tame it, feed it regularly, and clear away its ashy excreta. Before the art of fire-making was discovered, society would have prized the lesser art of husbanding a captured fire. Perhaps a live scion of the home fire was carried in a pot for barter to a neighbouring group whose own fire had unfortunately died. Wildfires would have been observed giving birth to daughter fires, spitting sparks and live cinders up on the wind, like dandelion puffs, to land and seed the dry grass at a distance. Did ergastrine philosophers theorize that fire cannot spontaneously generate, but must always be born of a parent fire, either wildfire out on the plains, or domestic fire fenced in by hearthstones? And did the first fire-making sticks therefore rub out a world view? Our ancestors might even have imagined a population of reproducing wildfires, or a pedigree of descent among domestic fires, traced from a glowing ancestor, bought from a distant clan, and traded on to others. But still there was no true heredity. Why not? How can you have reproduction and a pedigree, yet no heredity? This is the lesson fire has for us here. True heredity would mean the inheritance not of fire itself, but of variations among fires. Some fires are yellower than others, some redder, some roar, some crackle, some hiss, some smoke, some spit. Some have tinges of blue or green amongst the flames. Our ancestors, if they had studied their domesticated wolves, would have noticed a telling difference between dog pedigrees and fire pedigrees. With dogs, like begets like. At least some of what distinguishes one dog from another is handed down by its parents. Of course, some comes in sideways too, from food, disease, and accident. With fires, all the variation comes from the environment. None descends from the progenitive spark. It comes from the quality and dampness of the fuel, from the lie and strength of the wind, from the drawing qualities of the hearth, from the soil, from traces of copper and potassium that add touches of blue-green and lilac to sodium's yellow flame. Unlike a dog, nothing about the quality of an adult fire arrives via the spark that gave it birth. Blue fires don't beget blue fires. Crackling fires don't inherit their crackle from the parent fire that threw up their initiating spark. Fires exhibit reproduction without heredity. The origin of life was the origin of true heredity. We might even say the origin of the first gene. By first gene, I hasten to insist, I don't mean first DNA molecule. Nobody knows whether the first gene was made of DNA, and I bet it wasn't. By first gene, I mean first replicator. A replicator is an entity, for example a molecule, that forms lineages of copies of itself. There will always be errors in copying, so the population will acquire variety. The key to true heredity is that each replicator resembles the one from which it was copied more than it resembles a random member of the population. The origin of the first such replicator was not a probable event, but it only had to happen once. Thereafter, its consequences were automatically self-sustaining, and they eventually gave rise, by Darwinian evolution, to all of life.
A length of DNA, or under certain conditions the related molecule RNA, is a true replicator. So is a computer virus. So is a chain letter. But all these replicators need a complicated apparatus to assist them. DNA needs a cell richly equipped with pre-existing biochemical machinery, highly adapted to read and copy the DNA code. A computer virus needs a computer with some sort of data link to other computers, all designed by human engineers to obey coded instructions. A chain letter needs a good supply of idiots with evolved brains educated at least enough to read. What is unique about the first replicator, the one that sparked life, is that it had no ready supply of anything evolved, designed, or educated. The first replicator worked de novo, ab initio, without precedent and without help other than from the ordinary laws of chemistry. A powerful source of help to a chemical reaction is a catalyst, and catalysis in some form was surely involved in the origin of replication. A catalyst is an agent that speeds up a chemical reaction while not being consumed by it. All biological chemistry consists of catalyzed reactions, the catalysts usually being the large protein molecules called enzymes. A typical enzyme offers the shaped cavities of its three-dimensional form as receptacles for the ingredients of one chemical reaction. It lines them up for each other, enters into temporary chemical liaison with them, matchmakes with an aimed precision that they would be unlikely to discover in open diffusion. Catalysts, by definition, are not consumed in the chemical reaction they boost, but they may be produced. An autocatalytic reaction is a reaction that manufactures its own catalyst. As you can imagine, an autocatalytic reaction is reluctant to start, but once started, it takes off on its own. Like wildfire, indeed, for fire has some of the properties of an autocatalytic reaction. Fire is not strictly a catalyst, but it is self-generating. Chemically, it is an oxidation process that gives off heat and needs heat to push it over a threshold to start. Once started, it continues and spreads as a chain reaction because it generates the heat needed to restart itself. Another famous chain reaction is an atomic explosion, in this case not a chemical reaction but a nuclear one. Heredity began as a lucky initiation of an autocatalytic or otherwise self-regenerating process. It immediately took off and spread like a fire, eventually leading to natural selection and all that was to follow. We too oxidize carbonaceous fuel to generate heat, but we don't burst into flames because we do our oxidation in a controlled way, step by step, trickling the energy into useful channels instead of dissipating it as undisciplined heat. Such controlled chemistry, or metabolism, is as universal a feature of life as heredity. Theories of the origin of life need to account for both heredity and metabolism, but some writers have mistaken the priority. They have sought a theory of metabolism's spontaneous origin and somehow hoped that heredity would follow, like other useful devices. But heredity, as we shall see, is not to be thought of as a useful device. Heredity has to be first on the scene, because before heredity, usefulness itself has no meaning. Without heredity, and hence natural selection, there would have been nothing to be useful for. The very idea of usefulness cannot begin until the natural selection of hereditary information does.
The earliest theories of the origin of life that are taken seriously today are those of A.I. Oparin in Russia and J.B.S. Haldane in England, writing in the 1920s in ignorance of each other. Both emphasized metabolism rather than heredity. Both tumbled to the important fact that the atmosphere of Earth before life would have to be reducing if life were to arise. This rather unhelpful technical term means that the atmosphere lacked free oxygen. Organic compounds, compounds of carbon, when there is free oxygen around, are vulnerable to being burned or otherwise oxidized to carbon dioxide. It seems odd to us, who die within minutes if deprived of oxygen, but life could not originate on any planet with free oxygen in its atmosphere. As I've already explained, oxygen would have been a deadly poison to our earliest ancestors. Everything we know about other planets makes it almost certain that Earth's original atmosphere was a reducing one. Free oxygen came late. It was a polluting waste product of green bacteria, at first swimming free and later incorporated into plant cells. At some point, our ancestors evolved the ability to cope with oxygen, and later came to depend upon it. Incidentally, having said that oxygen is produced by green plants and algae, it is an oversimplification to leave it at that. It is true that plants give off oxygen, but when a plant dies, its decay in chemical reactions equivalent to burning all its carbonaceous materials would use up an amount of oxygen equal to all the oxygen released by that plant during its lifetime. There would therefore be no net gain in atmospheric oxygen, but for one thing. Not all dead plants decay. Some of them are laid down as coal, or equivalents, where they are removed from circulation. If all the fossil fuels in the world were burned by humanity, much of the oxygen in the atmosphere would be replaced by carbon dioxide, restoring the ancient status quo. This is not likely to happen in the near future, but we should not forget that the only reason we have oxygen to breathe is that most of the carbon in the world is tied up underground. We burn it all at our peril. Oxygen atoms were always present in that early atmosphere, but they were not free as oxygen gas. They were tied up in compounds, such as carbon dioxide and water. Carbon today is mostly locked up in living bodies or, a far greater proportion, in rocks such as chalk, limestone and coal, which come from the remains of once living bodies. In Canterbury times, those same carbon atoms would mostly have been in the atmosphere as compound gases, such as carbon dioxide and methane. Nitrogen, today's main atmospheric gas, would in a reducing atmosphere have been compounded with hydrogen as ammonia. Oparin and Haldane realized that a reducing atmosphere would have been friendly to the spontaneous synthesis of simple organic compounds. Here are Haldane's own words, which I quote for the sake of his famous concluding phrase.
Now, when ultraviolet light acts on a mixture of water, carbon dioxide and ammonia, a vast variety of organic substances are made, including sugars and apparently some of the materials from which proteins are built. This fact has been demonstrated in the laboratory by Bailey of Liverpool and his colleagues. In this present world, such substances, if left about, decay. That is to say, they are destroyed by microorganisms. But before the origin of life, they must have accumulated till the primitive oceans reached the consistency of hot, dilute soup.
This was written in 1929, more than 20 years before the much-cited experiment of Miller and Urey, which one would think, from Haldane's account, to have been a kind of repeat of Bailey's. However, E.C.C. Bailey was not concerned with the origin of life. His interest was photosynthesis, and his achievement was to synthesize sugars by beaming ultraviolet rays into water containing dissolved carbon dioxide in the presence of a catalyst such as iron or nickel. It was Haldane, rather than Bailey himself, who, with characteristic brilliance, was anticipating something remarkably like the Miller-Urey experiment, and reading it backwards into Bailey's work. What Miller, under Urey's direction, did was to take two flasks, one above the other, connected by two tubes. The lower flask contained heated water to represent the primeval ocean. The upper flask housed the mocked-up primordial atmosphere, methane, ammonia, water vapor, and hydrogen. Through one of the two tubes, vapor rose from above the heated ocean in the lower flask and was fed into the top of the atmosphere in the upper flask. The other tube returned downwards from atmosphere to ocean. On the way, it passed through a spark chamber, lightning, and a cooling chamber, where vapor condensed to form rain, which replenished the ocean. After only a week in this recycling simulacrum, the ocean had turned yellow-brown, and Miller analyzed its contents. As Haldane would have predicted, it had become a soup of organic compounds, including no fewer than 7 amino acids, the essential building blocks of proteins. Among the 7 were 3, glycine, aspartic acid, and alanine, from the list of 20 found in living things. Later experiments along Miller's lines with substituting carbon dioxide or carbon monoxide for methane have achieved similar results. We can draw the robust conclusion that biologically important small molecules, including amino acids, sugars, and significantly the building blocks of DNA and RNA, spontaneously form when various versions of the Oparin-Haldane primitive Earth are simulated in the laboratory. Before Oparin and Haldane, speculators about the origin of life had assumed that the first organisms must have been plants of some kind, perhaps green bacteria. People were used to the idea that life depends upon photosynthesis, the sunlight-driven manufacture of organic compounds accompanied by a release of oxygen. Oparin and Haldane, with their reducing atmosphere, saw plants as arriving on the scene later. Early life arose in a sea of pre-existing organic compounds. There was soup to eat, and no need to photosynthesize, at least until the soup ran out. For Oparin, the vital step was the origin of the first cell. And to be sure, cells, like organisms, have the important property that they never arise spontaneously, but always from other cells. It was pardonable to see the origin of the first cell, metabolizer, as a synonym for the origin of life, rather than the first gene, replicator, as I would. Among more modern theorists with the same bias, the distinguished theoretical physicist Freeman Dyson is aware of it and defends it. The majority of recent theorists, including Leslie Orgel in California, Manfred Eigen and his colleagues in Germany, and Graham Cairns-Smith in Scotland, more of a lone maverick but by no means to be written off, give priority to self-replication, both chronologically and in terms of centrality. Rightly so, in my opinion. What would heredity without a cell look like? Don't we have a chicken and egg problem? Certainly so if we take heredity to demand DNA, for DNA can't replicate without a large supporting cast of molecules, including proteins, that can only be made by DNA-coded information. But just because DNA is the main self-replicating molecule we know, it doesn't follow that it is the only one we could imagine, or the only one that has ever existed in nature. Graham Cairns-Smith has persuasively argued that the original replicators were inorganic mineral crystals, with DNA a late usurper, stepping into the starring role when life had evolved to the point where such a genetic takeover became possible. I won't expound his case here, partly because I've already given it my best shot in The Blind Watchmaker, but also for a larger reason. Cairns-Smith makes the clearest case I have read that replication was primary, and that DNA must have had a forerunner of some kind whose nature is unknown, save that it exhibited true heredity. I think it is a shame if this unassailable part of his case becomes tied in people's minds to his more controversial and speculative case for mineral crystals as the forerunner. I have nothing against the mineral crystals theory, which is why I expounded it before, but what I really want to emphasize is the primacy of replication and the strong likelihood that there was a late takeover by DNA from some forerunner. I can make the point most forcefully by deliberately switching in this book to a different particular theory of what that forerunner might have been. Whatever its ultimate merits as the original replicator, RNA is certainly a better candidate than DNA, and it has been cast as forerunner by a number of theorists in their so-called RNA world. To introduce the RNA world theory, I need to digress on enzymes. If the replicator is the star of life's show, the enzyme is the co-star, more than just supporting cast. Life depends utterly on the virtuoso ability of enzymes to catalyze biochemical reactions in a very fussy way. When I first learned about enzymes at school, the conventional and in my view mistaken wisdom that science should be taught by homely example meant that we spat into water to demonstrate the power of the salivary enzyme amylase to digest starch and make sugar. From this we gained the impression that an enzyme is like a corrosive acid. Biological washing powders, which use enzymes to digest dirt out of clothes, give the same impression. But these are destructive enzymes, working to dismember large molecules into their smaller components. Constructive enzymes are involved in synthesizing large molecules from smaller ingredients, and they do so by behaving as robotic matchmakers, as follows.
The interior of a cell contains a solution of thousands of molecules, atoms, and ions of many different kinds. Pairs of these could combine with each other in almost infinitely varied ways, but on the whole they do not. So there is a huge repertoire of potential chemistry waiting to happen in a cell, but most of it doesn't happen. Hold that in mind while reflecting on the following. A chemistry lab has hundreds of bottles on its shelves, all securely stoppered so their contents don't meet each other unless a chemist desires it, in which case a sample from one bottle is added to a sample from another. You could say that the shelves in a chemistry lab also house a huge repertoire of potential chemistry waiting to happen. And again, most of it doesn't happen. But imagine taking all the bottles off all the shelves and tipping them into a single vat full of water. A preposterous act of scientific vandalism, yet such a vat is pretty much what a living cell is. The hundreds of ingredients of thousands of potential chemical reactions are not kept in separate bottles until required to react together. Instead, they are all mixed up together in the same shared space all the time. but still they wait, largely unreactive, until required to react, as though separated in virtual bottles. There are no virtual bottles, but there are enzymes working as robotic matchmakers, or we might even call them robotic lab assistants. Enzymes discriminate, much as a radio tuner does when it puts a particular wireless set in touch with a particular transmitter, while ignoring the hundreds of other signals simultaneously bombarding its aerial with a babel of carrier frequencies. Suppose there is an important chemical reaction in which ingredient A combines with ingredient B to yield product Z. In a chemistry lab, we achieve this by taking the bottle labelled A off the shelf and the bottle labelled B from another shelf, mixing their contents in a clean flask and providing other necessary conditions, such as heat or stirring. We achieve the specific reaction we want by taking only 2 bottles off the shelf. In the living cell, lots of A molecules and lots of B molecules are among the huge variety of molecules floating around in the water, where they may meet but seldom combine, even if they do. In any case, they are no more likely to meet than thousands of other possible combinations. Now we introduce an enzyme called abscase, which is specifically shaped to catalyze the A plus B equals Z reaction. There are millions of abscase molecules in the cell, each one acting as a robotic lab assistant. Each abscase lab assistant grabs one A molecule, not off a shelf, but floating free in the cell. It then grabs a B molecule as it drifts by. It holds the A firmly in its grip so that it faces in a particular direction, and it holds the B equally firmly so that it abuts the A in just the right position and orientation to bond with the A and make a Z. The enzyme may do other things too, the equivalent of the human lab assistant wielding a stirrer or lighting a Bunsen burner. It may form a temporary chemical alliance with A or B, exchanging atoms or ions that will eventually be paid back, so the enzyme ends up as it started, thus qualifying as a catalyst. The result of all this is that a new Z molecule forms in the shaped grip of the enzyme molecule. The lab assistant then releases the new Z into the water and waits for another A to come by, whereupon it grabs it, and the cycle resumes. If there were no robotic lab assistant, a drifting A would occasionally bump into a drifting B under the right conditions to bond. But this lucky occurrence would be rare, no more common than the occasional chance-matched encounters that either A or B might make with lots of other potential partners. A might bump into C and make Y, or B might bump into D and make X. Small amounts of Y and X are being made all the time by lucky drift. But it is the presence of the lab assistant enzyme abscase that makes all the difference. In the presence of abscase, Z is churned out in, from the cell's point of view, industrial quantities. An enzyme typically multiplies the spontaneous rate of reaction by a factor varying between 1,000,000 and 1,000,000,000,000. If a different enzyme, ACase, were introduced, A would be combined with C instead of B, again at racing conveyor belt speed, to make a lavish supply of Y. It is the very same A molecules we're talking about, not confined to a bottle, but free to combine with either B or C, depending on which enzyme is present to grab them. The production rates of Z and Y will therefore depend on, among other things, how many of each of the 2 rival lab assistants, abscase and ACase, are floating about in the cell. And that depends on which of 2 genes in the nucleus of the cell is turned on. It is, however, a little more complicated than that. Even if a molecule of abscase is present, it may be inactivated. One way that this can happen is that another molecule comes and sits in the active cavity of the enzyme. It is as though the lab assistant's robotic arms were temporarily handcuffed. The handcuffs remind us, by the way, to issue the ritual warning that, as always with metaphors, there is a risk that robotic lab assistant might mislead. An enzyme molecule doesn't actually have arms to reach out and seize ingredients such as A, let alone submit to handcuffs. Instead, it has special zones in its own surface for which A, say, has an affinity, either because of a snug physical fit to a shaped cavity or due to some recondite chemical property. And this affinity can be temporarily negated in ways that resemble the calculated throwing of an off switch. Most enzyme molecules are special-purpose machines which make only one product, a sugar, say, or a fat, a purine or a pyrimidine, building blocks of DNA and RNA, or an amino acid. 20 of them are building blocks of natural proteins. But some enzymes are more like programmable machine tools that take in a punched paper tape to determine what they do. Outstanding among these is the ribosome, a large and complicated machine tool constructed from both protein and RNA, which makes proteins themselves. Amino acids, the building blocks of proteins, have already been made by special-purpose enzymes and are floating around in the cell, available to be picked up by the ribosome. The punched paper tape is RNA, specifically messenger RNA, mRNA. The messenger tape, which itself has copied its message from DNA in the genome, feeds into the ribosome and, as it passes through the reading head, the appropriate amino acids are assembled into a protein chain in the order specified by the tape using the genetic code. The coded sequence of DNA, therefore, controls what goes on in the cell. It specifies the sequence of amino acids in each protein, which determines the protein's three-dimensional shape, which in turn gives that protein its particular enzymatic properties. Importantly, the control may be indirect in that, as we saw in the mouse's tail, genes determine which other genes shall be turned on and when. Most genes in any one cell are not switched on. This is why, of all the reactions that could be going on in the vat full of mixed ingredients, only one or two actually do go on at any one time, the ones whose specific lab assistants are active in the cell. After that digression on catalysis and enzymes, we now turn from ordinary catalysis to the special case of autocatalysis, some version of which probably played a key role in the origin of life. Think back to our hypothetical example of molecules A and B combining to make Z under the influence of the enzyme abscase. What if Z itself is its own abscase? I mean, what if the Z molecule happens to have just the right shape and chemical properties to seize one A and one B, bring them together in the correct orientation, and combine them to make a new Z just like itself? In our previous example, we could say that the amount of abscase in the solution would influence the amount of Z produced. But now, if Z actually is one and the same molecule as abscase, we need only a single molecule of Z to seed a chain reaction. The first Z grabs A's and B's and combines them to make more Z's. Then these new Z's grab more A's and B's to make still more Z's, and so on. This is autocatalysis. Under the right conditions, the population of Z molecules will grow exponentially, explosively. This is the kind of thing that sounds promising as an ingredient for the origin of life. But it is all hypothetical. Julius Riebeck and his colleagues at the Scripps Institute in California made it real. They explored some fascinating examples of autocatalysis in real chemistry. In one of their examples, Z was a substance called AATE. The details don't matter. What matters is that the product of the chemical reaction is its own catalyst. The first molecule of AATE is reluctant to form, but once formed, an immediate chain reaction is set in train as more and more AATE synthesizes itself by serving as its own catalyst. As if that weren't enough, this brilliant series of experiments went on to demonstrate true heredity in the sense defined here. Riebeck and his team found a system in which more than one variant of the autocatalyzed substance existed. Each variant catalyzed the synthesis of itself, using its preferred variant of one of the ingredients. This raised the possibility of true competition in a population of entities showing true heredity, and an instructively rudimentary form of Darwinian selection. Riebeck's chemistry is highly artificial. Nevertheless, his story beautifully illustrates the principle of autocatalysis, according to which the product of a chemical reaction serves as its own catalyst. It is something like autocatalysis that we need for the origin of life. Could RNA, or something like RNA under the conditions of the early Earth, have autocatalyzed its own synthesis, Riebeck style, and in water instead of chloroform? The problem is a formidable one, as explained by the German Nobel Prize-winning chemist Manfred Eigen. He pointed out that any self-replication process is subject to degradation by copying error, mutation. Imagine a population of replicating entities in which there is a high probability of error in every copying event. If a coded message is to hold its own against the ravages of mutation, at least one member of the population in any one generation must be identical to its parent. If there are 10 code units, letters, in an RNA chain, for example, the average error rate per letter must be less than 1 in 10. We can then expect that at least some members of the offspring generation will have the full complement of 10 correct code letters. But if the error rate is greater, there will be a relentless degradation as the generations go by, simply because of mutation alone, no matter how strong the selection pressure. This is called an error catastrophe. Short chains of RNA, and indeed DNA, can spontaneously self-replicate without an enzyme, but the error rate per letter is far higher than when an enzyme is present. And this means that long before a sufficient length of gene could be built up to make the protein for a working enzyme, the fledgling gene would have been destroyed by mutation. That is the catch-22 of the origin of life. A gene big enough to specify an enzyme would be too big to replicate accurately without the aid of an enzyme of the very kind that it's trying to specify. So the system apparently cannot get started. The solution to the catch-22 that Eigen offers is the theory of the hypercycle. It uses the old principle of divide and rule. The coded information is subdivided into subunits small enough to lie below the threshold of an error catastrophe. Each subunit is a mini-replicator in its own right, and it is small enough for at least one copy to survive in each generation. All the subunits cooperate in some important larger function, large enough to suffer an error catastrophe if catalyzed by a single large chemical rather than being subdivided.
As I have so far described the theory, there is a danger that the whole system would be unstable because some subunits would self-replicate faster than others. This is where the clever part of the theory kicks in. Each subunit flourishes in the presence of the others. More specifically, the production of each is catalyzed by the presence of another, such that they form a cycle of dependency, a hypercycle. This automatically prevents any one element from racing ahead. It cannot do so because it depends on its predecessor in the hypercycle. I'm going to leave the hypercycle theory at this point and return to the suggestion, which is fully compatible with it, that RNA, in the early days when life was just beginning and proteins did not yet exist, might have served as its own catalyst. This is the RNA world theory. To see how plausible it is, we need to look at why proteins are good at being enzymes but bad at being replicators, at why DNA is good at replicating but bad at being an enzyme, and finally, why RNA might just be good enough at both roles to break out of the catch-22. Three-dimensional shape is largely what matters for enzyme activity. Proteins are good at being enzymes because they can assume almost any shape you want in 3 dimensions as an automatic consequence of their amino acid sequence in 1 dimension. It is the chemical affinities of amino acids for other amino acids in different parts of the chain that determines the particular knot into which the protein chain ties itself. So, the 3-dimensional shape of a protein molecule is specified by the 1-dimensional sequence of amino acids, and that is itself specified by the 1-dimensional sequence of code letters in a gene. In principle, practice is a different matter and formidably difficult. it should be possible to write down a sequence of amino acids that would spontaneously coil itself up into almost any shape you like, not just shapes that make good enzymes, but any arbitrary shape you choose to specify. It is this protean talent that qualifies proteins to act as enzymes. There is a protein capable of selecting any one out of the hundreds of potential chemical reactions that could go on in a cell full of jumbled ingredients. Proteins then make wonderful enzymes capable of tying themselves into knots of any desired shape, but they are lousy replicators. Unlike DNA and RNA, whose component elements have specific pairing rules, the Watson-Crick pairing rules, discovered by those two inspired young men, amino acids have no such rules. DNA, by contrast, is a splendid replicator, but a lousy candidate for the enzyme role in life. This is because, unlike proteins with their near infinite variety of three-dimensional shapes, DNA has only one shape, the famous double helix itself. The double helix is ideally suited to replication because the two sides of the stairway peel easily away from one another, each being then exposed as a template for new letters to join following the Watson-Crick pairing rules. It is not much good for anything else. RNA has some of the virtues of DNA as a replicator and some of the virtues of protein as a versatile shaper of enzymes. The 4 letters of RNA are sufficiently similar to the 4 letters of DNA that either set can serve as a template for the other. On the other hand, RNA does not easily form a long double helix, which means that it is somewhat inferior to DNA as a replicator. This is partly because the double helix system lends itself to proof correction. When the DNA double helix splits and each single helix immediately serves as a template for its complement, errors can instantly be spotted and corrected. Each daughter chain remains attached to its parent, and comparison between the two permits instant error detection. Proofreading based on this principle reduces mutation rates to the order of 1 in a billion, which is what makes large genomes like ours possible. RNA, lacking this kind of proofreading, has mutation rates that are thousands of times greater than DNA. This means that only simple organisms with small genomes, such as some viruses, can use RNA as their primary replicator. But the lack of a double helix structure has its upside as well as its downside. Because the RNA chain doesn't spend all its time paired with its complementary chain, but breaks away from the complement as soon as it is formed, it is free to tie itself in knots like a protein. Just as the protein does it by virtue of the chemical affinities of amino acids for other amino acids in different parts of the same chain, RNA does it using the ordinary Watson-Crick base pairing rules, the same ones as are used to make copies of RNA. Putting it another way, lacking a partner chain to pair with in a double helix like DNA, RNA is free to pair with odd bits of itself. RNA finds small stretches of itself with which it can pair, either in a miniature double helix or in some other shape. The pairing rules insist that these stretches have to be going in opposite directions. An RNA chain, therefore, has a tendency to fall into a series of hairpin bends. The repertoire of three-dimensional shapes into which an RNA molecule is capable of throwing itself may not be as great as the repertoire of large protein molecules, but it is large enough to encourage the thought that RNA might furnish a versatile armory of enzymes. And to be sure, many RNA enzymes called ribozymes have been discovered. The conclusion is that RNA has some of the replicator virtues of DNA and some of the enzyme virtues of proteins. Maybe before the coming of DNA, the arch replicator, and before the coming of proteins, the arch catalysts, there was a world in which RNA alone had enough of both virtues to stand in for both experts. Perhaps an RNA fire ignited itself in the original world, and then later started to make proteins that turned around and helped synthesize RNA, and later DNA too, which took over as the dominant replicator. That is the hope of the RNA world theory. Research continues, and I expect exciting results. But now I want to switch to a newly fashionable line of thought fully compatible with the RNA world and with many others among the current theories of the origin of life. What is new is the suggested location in which the crucial events first took place. Not warm little pond, but hot deep rock, an exciting theory which amounts to this: our pilgrims, to complete their journey and locate their Canterbury, are now going to have to bore deep underground into the primordial rock. The main inspirer of the theory is another maverick, Thomas Gould, originally an astronomer, but versatile enough to deserve the now rare accolade general scientist, and distinguished enough to have been elected to both the Royal Society of London and the American National Academy of Sciences.
Gould believes that our emphasis on the sun as energetic prime mover of life may be misplaced. Perhaps we have yet again been misled by what happens to be familiar, yet again assigned to ourselves and our kind of life a centrality in the scheme of things that we do not deserve. There was a time when textbooks asserted that all life depended ultimately on sunlight. Then, in 1977, the startling discovery was made that volcanic vents on the floor of deep oceans support a strange community of creatures living without benefit of sunlight. Heat from red-hot lava raises the water temperature to more than 100 degrees Celsius, still well below boiling point at the colossal pressures of those depths. The surrounding water is very cold, and the temperature gradient drives various kinds of bacterial metabolism. These thermophile bacteria, including sulfur bacteria, who make use of hydrogen sulfide streaming from the volcanic vents, constitute the base of elaborate food chains, higher links of which include blood-red tube worms up to 3 meters long, limpets, mussels, starfish, barnacles, white crabs, prawns, and fish capable of thriving at 80 degrees Celsius. There are bacteria, as we have seen, which can take such Hadean temperatures in their stride, but no other animal is known to do so, and these polychaete worms have accordingly been dubbed Pompeii worms. Some of the sulfur bacteria are given house room by animals, for example by mussels, and by the huge tube worms, who take special biochemical steps using hemoglobin, hence their blood-red colour, to feed sulfide to their own bacteria. These colonies of life, based on bacterial extraction of energy from hot volcanic vents, astonished everybody, first by their very existence, and then by their abundant richness, which contrasted startlingly with the near-desert conditions of the surrounding sea bottom. Even after this sensational discovery, most biologists continue to believe that life is centered on the sun. The creatures of the deep-sea smoker communities, fascinating though they might be, are assumed by most of us to be a rare and unrepresentative aberration. Gould believes otherwise. He thinks hot, dark, high-pressure depths are where life fundamentally belongs and where it originated, not necessarily in the sea, but perhaps in the rocks deep underground. We who live at the surface, in the light and the cool and the fresh air, we are the anomalous aberrations. He points out that hopanoids, organic molecules made in bacterial cell walls, are ubiquitous in rocks, and quotes an authoritative estimate of between 10 trillion and 100 trillion tons of hopanoids in the rocks of the world. This comfortably exceeds the trillion tons or so of organic carbon in surface-dwelling life. Gould notes that the rocks are seamed with cracks and fissures, which, though small to our eyes, provide more than 1 billion trillion cubic centimeters of hot, wet space suitable for life on the bacterial scale of existence. Heat energy and the chemicals of the rocks themselves will be enough to sustain bacteria in huge numbers. Gould notes that many bacteria thrive at temperatures up to 110 degrees Celsius, and this would permit them to live down to depths of between 5 and 10 kilometers, a distance that would take them less than 1,000 years to travel. It is impossible to verify his estimate, but he thinks the biomass of bacteria in the hot, deep rocks might exceed the biomass of the surface sun-based life with which we are familiar.
Turning to the question of the origin of life, Gould and others have pointed out that thermophily, love of high temperatures, is not a rare oddity among bacteria and archaeans. It is common, so common and so widely distributed around bacterial family trees that it might well be the primitive state from which our familiar cool forms of life have evolved. With respect to both chemistry and temperature, the conditions on the surface of the primitive Earth, some scientists call it the Hadean Age, were more like those in Gould's hot deep rocks than they were like today's surface conditions. A persuasive case can indeed be made that when we dig down into the rocks, we are digging backwards in time and rediscovering something like the conditions of life's scalding Canterbury. The idea has been further championed recently by the Anglo-Australian physicist Paul Davies, whose book, The Fifth Miracle, summarizes new evidence discovered since Gould's paper of 1992. Various drilling samples have been found to contain hyperthermophile bacteria, alive and reproducing, amid scrupulous precautions to preclude contamination from the surface. Some of these bacteria have been successfully cultured in a modified pressure cooker. Davies, like Gould, believes life may have originated deep underground, and that the bacteria which still live there may be relatively unchanged relics of our remote ancestors. This idea is especially appealing for our pilgrimage because it offers us the hope of meeting something like the earliest bacteria, rather than the more familiar bacteria modified for modern conditions of light, cold, and oxygen. Having endured ridicule at first, the hot, deep rock theory of the origin of life is now verging on the positively fashionable. Whether it will turn out to be right must await more research, but I confess to hoping that it will. There are many other theories that I have not gone into. Maybe one day we shall reach some sort of definite consensus on the origin of life. If so, I doubt if it will be supported by direct evidence, because I suspect that it has all been obliterated. Rather, it will be accepted because somebody produces a theory so elegant that, as the great American physicist John Archibald Wheeler said in another context,
"We will grasp the central idea of it all as so simple, so beautiful, so compelling, that we will say to each other, 'Oh, how could it have been otherwise? How could we all have been so blind for so long?'"
If that isn't how we finally realize we know the answer to the riddle of life's origin, I don't think we shall ever know it.
The host's return. The genial host, having guided Chaucer and the other pilgrims from London to Canterbury, and stood impresario to their tales, turned around and led them straight back to London.
If I now return to the present, it must be alone. For to presume upon evolution's following the same forward course twice would be to deny the rationale of our backward journey. Evolution was never aimed at any particular end point. Our backwards pilgrimage has been a series of swelling mergers as we were swallowed up in ever more inclusive groupings, the apes, the primates, the mammals, the vertebrates, the deuterostomes, the animals, and so on back to the arch-ancestor of all life. If we turn around and move forward now, we cannot retrace our steps. That would imply that evolution, were it to be rerun, would follow the same course, putting those same mergers into reverse gear in the form of splits.